Section 1 of Gray's Anatomy, Part 3. This is a Librivox recording. All Librivox recordings are in the public domain. For more information or to volunteer, please visit librivox.org. Recorded by Laurie Anne Walden. Anatomy of the Human Body, Part 3, by Henry Gray. Section 1. Angiology, Introduction. The vascular system is divided for descriptive purposes into a. The blood vascular system, which comprises the heart and blood vessels for the circulation of the blood, and b. The lymph vascular system, consisting of lymph glands and lymphatic vessels, through which a colorless fluid, the lymph, circulates. It must be noted, however, that the two systems communicate with each other and are intimately associated developmentally. The heart is the central organ of the blood vascular system and consists of a hollow muscle. By its contraction the blood is pumped to all parts of the body through a complicated series of tubes termed arteries. The arteries undergo enormous ramification in their course throughout the body, and end in minute vessels called arterioles, which in their turn open into a close meshed network of microscopic vessels termed capillaries. After the blood has passed through the capillaries, it is collected into a series of larger vessels called veins, by which it is returned to the heart. The passage of the blood through the heart and blood vessels constitutes what is termed the circulation of the blood, of which the following is an outline. The human heart is divided by septa into right and left halves, and each half is further divided into two cavities, an upper termed the atrium, and a lower, the ventricle. The heart therefore consists of 4 chambers, 2, the right atrium and right ventricle, forming the right half, and 2, the left atrium and left ventricle, the left half. The right half of the heart contains venous or impure blood, the left, arterial or pure blood. The atria are receiving chambers, and the ventricles distributing ones. From the cavity of the left ventricle the pure blood is carried into a large artery, the aorta, through the numerous branches of which it is distributed to all parts of the body, with the exception of the lungs. In its passage through the capillaries of the body, the blood gives up to the tissues the materials necessary for their growth and nourishment, and at the same time receives from the tissues the waste products resulting from their metabolism. In so doing it is changed from arterial into venous blood, which is collected by the veins, and through them returned to the right atrium of the heart. From this cavity the impure blood passes into the right ventricle, and is thence conveyed through the pulmonary arteries to the lungs. In the capillaries of the lungs it again becomes arterialized, and is then carried to the left atrium by the pulmonary veins. From the left atrium it passes into the left ventricle, from which the cycle once more begins. The course of the blood from the left ventricle through the body, generally to the right side of the heart, constitutes the greater or systemic circulation, while its passage from the right ventricle through the lungs to the left side of the heart is termed the lesser or pulmonary circulation. It is necessary, however, to state that the blood which circulates through the spleen, pancreas, stomach, small intestine, and the greater part of the large intestine, is not returned directly from these organs to the heart, but is conveyed by the portal vein to the liver. In the liver this vein divides, like an artery, and ultimately ends in capillary-like vessels, or sinusoids, from which the rootlets of a series of veins, called the hepatic veins, arise. These carry the blood into the inferior vena cava, whence it is conveyed to the right atrium. From this it will be seen that the blood contained in the portal vein passes through 2 sets of vessels: 1, the capillaries in the spleen, pancreas, stomach, etc., and 2, the sinusoids in the liver. The blood in the portal vein carries certain of the products of digestion, the carbohydrates, which are mostly taken up by the liver cells and stored as glycogen, and the protein products, which remain in solution and are carried into the general circulation to the various tissues and organs of the body. Speaking generally, the arteries may be said to contain pure, and the veins impure blood. This is true of the systemic, but not of the pulmonary vessels, since it has been seen that the impure blood is conveyed from the heart to the lungs by the pulmonary arteries, and the pure blood returned from the lungs to the heart by the pulmonary veins. Arteries, therefore, must be defined as vessels which convey blood from the heart, and veins as vessels which return blood to the heart. Structure of arteries. The arteries are composed of three coats, an internal or endothelial coat, tunica intima of Kuliker, a middle or muscular coat, tunica media, and an external or connective tissue coat, tunica adventitia. The two inner coats together are very easily separated from the external, as by the ordinary operation of tying a ligature around an artery. If a fine string be tied forcibly upon an artery, and then taken off, the external coat will be found undivided, but the two inner coats are divided in the track of the ligature, and can easily be further dissected from the outer coat. The inner coat, tunica intima, can be separated from the middle by a little maceration, or it may be stripped off in small pieces. But on account of its friability, it cannot be separated as a complete membrane. It is a fine, transparent, colorless structure which is highly elastic, and, after death, is commonly corrugated into longitudinal wrinkles. The inner coat consists of 1. A layer of pavement endothelium, the cells of which are polygonal, oval, or fusiform, and have very distinct round or oval nuclei. This endothelium is brought into view most distinctly by staining with nitrate of silver. 2. A subendothelial layer, consisting of delicate connective tissue with branched cells lying in the interspaces of the tissue. In arteries of less than 2 mm in diameter, the subendothelial layer consists of a single stratum of stellate cells, and the connective tissue is only largely developed in vessels of a considerable size. 3. An elastic or fenestrated layer, which consists of a membrane containing a network of elastic fibers, having principally a longitudinal direction, and in which, under the microscope, small elongated apertures or perforations may be seen, giving it a fenestrated appearance. It was therefore called by Henle the fenestrated membrane. This membrane forms the chief thickness of the inner coat, and can be separated into several layers, some of which present the appearance of a network of longitudinal elastic fibers, and others a more membranous character, marked by pale lines having a longitudinal direction. In minute arteries the fenestrated membrane is a very thin layer, but in the larger arteries, and especially the aorta, it has a very considerable thickness. The middle coat, tunica media, is distinguished from the inner by its color and by the transverse arrangement of its fibers. In the smaller arteries it consists principally of plain muscle fibers in fine bundles, arranged in lamellae and disposed circularly around the vessel. These lamellae vary in number according to the size of the vessel, the smallest arteries having only a single layer, and those slightly larger 3 or 4 layers. It is to this coat that the thickness of the wall of the artery is mainly due. In the larger arteries, as the iliac, femoral, and carotid, elastic fibers unite to form lamellae which alternate with the layers of muscular fibers. These lamellae are united to one another by elastic fibers which pass between the muscular bundles, and are connected with the fenestrated membrane of the inner coat. In the largest arteries, as the aorta and innominate, the amount of elastic tissue is very considerable. In these vessels a few bundles of white connective tissue have also been found in the middle coat. The muscle fiber cells are about 50 microns in length, and contain well-marked rod-shaped nuclei, which are often slightly curved. The external coat, tunica adventitia, consists mainly of fine and closely felted bundles of white connective tissue, but also contains elastic fibers in all but the smallest arteries. The elastic tissue is much more abundant next the tunica media, and it is sometimes described as forming here, between the adventitia and media, a special layer, the tunica elastica externa of Henle. This layer is most marked in arteries of medium size. In the largest vessels the external coat is relatively thin, but in small arteries it is of greater proportionate thickness. In the smaller arteries it consists of a single layer of white connective tissue and elastic fibers, while in the smallest arteries, just above the capillaries, the elastic fibers are wanting, and the connective tissue of which the coat is composed becomes more nearly homogeneous the nearer it approaches the capillaries, and is gradually reduced to a thin membranous envelope which finally disappears. Some arteries have extremely thin walls in proportion to their size. This is especially the case in those situated in the cavity of the cranium and vertebral canal, the difference depending on the thinness of the external and middle coats. The arteries, in their distribution throughout the body, are included in thin fibro-areolar investments, which form their sheaths. The vessel is loosely connected with its sheath by delicate areolar tissue, and the sheath usually encloses the accompanying veins, and sometimes a nerve. Some arteries, as those in the cranium, are not included in sheaths. All the larger arteries, like the other organs of the body, are supplied with blood vessels. These nutrient vessels, called the vasa vasorum, arise from a branch of the artery, or from a neighboring vessel at some considerable distance from the point at which they are distributed. They ramify in the loose areolar tissue connecting the artery with its sheath, and are distributed to the external coat, but do not, in man, penetrate the other coats. In some of the larger mammals a few vessels have been traced into the middle coat. Minute veins return the blood from these vessels. They empty themselves into the vein or veins accompanying the artery. Lymphatic vessels are also present in the outer coat. Arteries are also supplied with nerves, which are derived from the sympathetic, but may pass through the cerebrospinal nerves. They form intricate plexuses upon the surfaces of the larger trunks, and run along the smaller arteries as single filaments, or bundles of filaments which twist around the vessel and unite with each other in a plexiform manner. The branches derived from these plexuses penetrate the external coat and are distributed principally to the muscular tissue of the middle coat, and thus regulate, by causing the contraction and relaxation of this tissue, the amount of blood sent to any part. The capillaries. The smaller arterial branches, excepting those of the cavernous structure of the sexual organs, of the splenic pulp, and of the placenta, terminate in networks of vessels which pervade nearly every tissue of the body. These vessels, from their minute size, are termed capillaries. They are interposed between the smallest branches of the arteries and the commencing veins, constituting a network, the branches of which maintain the same diameter throughout. The meshes of the network are more uniform in shape and size than those formed by the anastomoses of the small arteries and veins. The diameters of the capillaries vary in the different tissues of the body, the usual size being about 8 microns. The smallest are those of the brain and the mucous membrane of the intestines, and the largest those of the skin and the marrow of bone, where they are stated to be as large as 20 microns in diameter. The form of the capillary net varies in the different tissues, the meshes being generally rounded or elongated. The rounded form of mesh is most common, and prevails where there is a dense network, as in the lungs, in most glands and mucous membranes, and in the cutis. The meshes are not of an absolutely circular outline, but more or less angular, sometimes nearly quadrangular or polygonal, or more often irregular. Elongated meshes are observed in the muscles and nerves, the meshes resembling parallelograms in form, the long axis of the mesh running parallel with the long axis of the nerve or muscle. Sometimes the capillaries have a looped arrangement, a single vessel projecting from the common network, and returning after forming one or more loops, as in the papillae of the tongue and skin. The number of the capillaries and the size of the meshes determine the degree of vascularity of a part. The closest network and the smallest interspaces are found in the lungs and in the choroid coat of the eye. In these situations the interspaces are smaller than the capillary vessels themselves. In the intertubular plexus of the kidney, in the conjunctiva, and in the cutis, the interspaces are from 3 to 4 times as large as the capillaries which form them, and in the brain from 8 to 10 times as large as the capillaries in their long diameters, and from 4 to 6 times as large in their transverse diameters. In the adventitia of arteries, the width of the meshes is 10 times that of the capillary vessels. As a general rule, the more active the function of the organ, the closer is its capillary net, and the larger its supply of blood. The meshes of the network are very narrow in all growing parts, in the glands, and in the mucous membranes, wider in bones and ligaments which are comparatively inactive. Blood vessels are nearly altogether absent in tendons, in which very little organic change occurs after their formation. In the liver the capillaries take a more or less radial course toward the intralobular vein, and their walls are incomplete, so that the blood comes into direct contact with the liver cells. These vessels in the liver are not true capillaries, but sinusoids. They are developed by the growth of columns of liver cells into the blood spaces of the embryonic organ. Structure. The wall of a capillary consists of a fine transparent endothelial layer composed of cells joined edge to edge by an interstitial cement substance and continuous with the endothelial cells which line the arteries and veins. When stained with nitrate of silver, the edges which bound the epithelial cells are brought into view. These cells are of large size and of an irregular polygonal or lanceolate shape, each containing an oval nucleus which may be displayed by carmine or hematoxylin. Between their edges, at various points of their meeting, roundish dark spots are sometimes seen, which have been described as stomata, though they are closed by intercellular substance. They have been believed to be the situations through which the colorless corpuscles of the blood, when migrating from the blood vessels, emerge. But this view, though probable, is not universally accepted. Collisal describes these cells as having a rather more complex structure. He states that each consists of two parts, of hyaline ground plates, and of a protoplasmic granular part, in which is embedded the nucleus, on the outside of the ground plates. The hyaline internal coat of the capillaries does not form a complete membrane, but consists of plates which are inelastic, and though in contact with each other are not continuous. When, therefore, the capillaries are subjected to intravascular pressure, the plates become separated from each other. The protoplasmic portions of the cells, on the other hand, are united together. In some organs, for example the glomeruli of the kidneys, intercellular cement cannot be demonstrated in the capillary wall, and the cells are believed to form a syncytium. In many situations a delicate sheath or envelope of branched nucleated connective tissue cells is found around the simple capillary tube, particularly in the larger ones, and in other places, especially in the glands, the capillaries are invested with retiform connective tissue. Sinusoids. In certain organs, for example, the heart, the liver, the suprarenal and parathyroid glands, the glomus caroticum and glomus coccygeum, the smallest blood vessels present various differences from true capillaries. They are wider, with an irregular lumen, and have no connective tissue covering, their endothelial cells being in direct contact with the cells of the organ. Moreover, they are either arterial or venous, and not intermediate, as are the true capillaries. These vessels have been called sinusoids by Minot. They are formed by columns of cells or trabeculae pushing their way into a large blood vessel or blood space, and carrying its endothelium before them. At the same time, the wall of the vessel or space grows out between the cell columns. Structure of veins. The veins, like the arteries, are composed of three coats, internal, middle, and external. And these coats are, with the necessary modifications, analogous to the coats of the arteries, the internal being the endothelial, the middle the muscular, and the external the connective tissue or areolar. The main difference between the veins and the arteries is in the comparative weakness of the middle coat in the former. In the smallest veins the three coats are hardly to be distinguished. The endothelium is supported on a membrane separable into two layers, the outer of which is the thicker, and consists of a delicate nucleated membrane, adventitia, while the inner is composed of a network of longitudinal elastic fibers, media. In the veins next above these in size (.4 millimeters in diameter), according to Kuliker, a connective tissue layer containing numerous muscle fibers circularly disposed can be traced, forming the middle coat, while the elastic and connective tissue elements of the outer coat become more distinctly perceptible. In the middle-sized veins the typical structure of these vessels becomes clear. The endothelium is of the same character as in the arteries, but its cells are more oval and less fusiform. It is supported by a connective tissue layer consisting of a delicate network of branched cells, and external to this is a layer of elastic fibers disposed in the form of a network in place of the definite fenestrated membrane seen in the arteries. This constitutes the internal coat. The middle coat is composed of a thick layer of connective tissue with elastic fibers intermixed in some veins with a transverse layer of muscular tissue. The white fibrous element is in considerable excess, and the elastic fibers are in much smaller proportion in the veins than in the arteries. The outer coat consists, as in the arteries, of areolar tissue, with longitudinal elastic fibers. In the largest veins the outer coat is from 2 to 5 times thicker than the middle coat, and contains a large number of longitudinal muscle fibers. These are most distinct in the inferior vena cava, especially at the termination of this vein in the heart, in the trunks of the hepatic veins, in all the large trunks of the portal vein, and in the external iliac, renal, and azygous veins. In the renal and portal veins they extend through the whole thickness of the outer coat. But in the other veins mentioned, a layer of connective and elastic tissue is found external to the muscular fibers. All the large veins which open into the heart are covered for a short distance with a layer of striped muscular tissue continued onto them from the heart. Muscular tissue is wanting: 1. In the veins of the maternal part of the placenta; 2. In the venous sinuses of the dura mater and the veins of the pia mater of the brain and medulla spinalis; 3. In the veins of the retina; 4. In the veins of the cancellous tissue of bones; 5. In the venous spaces of the corpora cavernosa. The veins of the above-mentioned parts consist of an internal endothelial lining supported on one or more layers of areolar tissue. Most veins are provided with valves which serve to prevent the reflux of the blood. Each valve is formed by a reduplication of the inner coat, strengthened by connective tissue and elastic fibers, and is covered on both surfaces with endothelium, the arrangement of which differs on the two surfaces. On the surface of the valve next the wall of the vein the cells are arranged transversely, while on the other surface, over which the current of blood flows, the cells are arranged longitudinally in the direction of the current. Most commonly two such valves are found placed opposite one another, more especially in the smaller veins or in the larger trunks at the point where they are joined by smaller branches. Occasionally there are 3, and sometimes only 1. The valves are semilunar. They are attached by their convex edges to the wall of the vein. The concave margins are free, directed in the course of the venous current, and lie in close apposition with the wall of the vein as long as the current of blood takes its natural course. If, however, any regurgitation takes place, the valves become distended, their opposed edges are brought into contact, and the current is interrupted. The wall of the vein on the cardiac side of the point of attachment of each valve is expanded into a pouch or sinus, which gives to the vessel, when injected or distended with blood, a knotted appearance. The valves are very numerous in the veins of the extremities, especially of the lower extremities, these vessels having to conduct the blood against the force of gravity. They are absent in the very small veins, that is, those less than 2 millimeters in diameter, also in the vena cavae, hepatic, renal, uterine, and ovarian veins. A few valves are found in each spermatic vein, and one also at its point of junction with the renal vein or inferior vena cava, respectively. The cerebral and spinal veins, the veins of the cancellated tissue of bone, the pulmonary veins, and the umbilical vein and its branches, are also destitute of valves. A few valves are occasionally found in the azygous and intercostal veins. Rudimentary valves are found in the tributaries of the portal venous system. The veins, like the arteries, are supplied with nutrient vessels, vasa vasorum. Nerves also are distributed to them in the same manner as to the arteries, but in much less abundance. End of section 1.
Section 2 of Gray's Anatomy, Part 3. This is a Librivox recording. All Librivox recordings are in the public domain. For more information or to volunteer, please visit LibriVox.org. Anatomy of the Human Body, Part 3, by Henry Gray. The blood. The blood is an opaque, rather viscous fluid of a bright red or scarlet color when it flows from the arteries, of a dark red or purple color when it flows from the veins. It is salt to the taste and has a peculiar faint odor and an alkaline reaction. Its specific gravity is about 1.06, and its temperature is generally about 37 degrees, though varying slightly in different parts of the body. General composition of the blood. Blood consists of a fairly yellow fluid, the plasma or liquor sanguinis, in which are suspended numerous minute particles, the blood corpuscles, the majority of which are colored and give the blood its red tint. If a drop of blood be placed on a thin layer on a glass slide and examined under the microscope, a number of these corpuscles will be seen floating in the plasma. The blood corpuscles are of three kinds: 1. Colored corpuscles or erythrocytes, 2. Colorless corpuscles or leukocytes, and 3. Blood platelets. 1. Colored or red corpuscles, erythrocytes, when examined under the microscope, are seen to be circular discs, biconcave in profile. The disc has no nucleus, but in consequence of its biconcave shape, presents, according to the alterations of focus under an ordinary high power, a central part, sometimes bright, sometimes dark, which has the appearance of a nucleus. It is to the aggregation of the red corpuscles that the blood owes its red hue, although when examined by transmitted light, their color appears to be only a faintly reddish yellow. The corpuscles vary slightly in size even in the same drop of blood, but the average diameter is about 7.5 microns, and the thickness about 2 microns. Footnote: a micromillimeter, or micron, is 1/1000th of a millimeter, or 1/25,000th of an inch. End note. Besides these, there are found certain smaller corpuscles of about 1/2 of the size just indicated. These are termed microcytes, and are very scarce in normal blood. In disease conditions, for example anemia, however, they are more numerous. The number of red corpuscles in the blood is enormous. Between 4 million and 5 million are contained in a cubic millimeter. Power states that the red corpuscles of an adult would present an aggregate surface of about 3,000 square yards. If the web of a living frog's foot be spread out and examined under the microscope, the blood is seen to flow in a continuous stream through the vessels, and the corpuscles show no tendency to adhere to each other or to the wall of the vessel. Doubtless the same is the case in the human body, but when human blood is drawn and examined on a slide without reagents, the corpuscles tend to collect into heaps, like rouleaux of coins. It has been suggested that this phenomenon may be explained by alteration in surface tension. During life, the red corpuscles may be seen to change their shape under pressure so as to adapt themselves, to some extent, to the size of the vessel. They are, however, highly elastic, and speedily recover their shape when the pressure is removed. They are readily influenced by the medium in which they are placed. In water they swell up, lose their shape, and become globular, endosmosis. Subsequently, the hemoglobin is dissolved out, and the envelope can barely be distinguished as a faint circular outline. Solutions of salt or sugar, denser than the plasma, give them a stellate or crenated appearance, exosmosis. But the usual shape may be restored by diluting the solution to the same tonicity as the plasma. The crenated outline may be produced as the first effect of the passage of an electric shock. Subsequently, if sufficiently strong, the shock ruptures the envelope. A solution of salt, isotonic with the plasma, merely separates the blood corpuscles mechanically without changing their shape. Two views are held with regard to the structure of the erythrocytes. The older view, that of Rolette, supposes that the corpuscle consists of a sponge work or stroma permeated by a solution of hemoglobin. Schaefer, on the other hand, believes that the hemoglobin solution is contained within an envelope or membrane, and the facts stated above with regard to the osmotic behavior of the erythrocyte support this belief. The envelope consists mainly of lecithin, cholesterin, and nucleoprotein. The colorless corpuscles or leukocytes are of various sizes, some no larger, others smaller than the red corpuscles. In human blood, however, the majority are rather larger than the red corpuscles and measure about 10 microns in diameter. On the average, from 7,000 to 12,000 leukocytes are found in each cubic millimeter of blood. They consist of minute masses of nucleated protoplasm and exhibit several varieties, which are differentiated from each other chiefly by the occurrence or non-occurrence of granules in their protoplasm and by the staining reactions of these granules when present. The most numerous, 60%, and important are irregular in shape, possessed of the power of amoeboid movement, and are characterized by nuclei which often consist of 2 or 3 parts, multipartite, connected together by fine threads of chromatin. The protoplasm is clear and contains a number of very fine granules, which stain with acid dyes such as eosin or with neutral dyes, and are therefore called oxyphil or neutrophil. These cells are termed the polymorphonuclear leukocytes. 2. A second variety comprises from 1 to 4% of the leukocytes. They are larger than the previous kind and are made up of coarsely granular protoplasm, the granules being highly refractile and grouped around single nuclei of horseshoe shape. The granules stain deeply with eosin, and the cells are therefore often termed eosinophil corpuscles. 3. The third variety is called the hyaline cell or macrocyte. This is usually about the same size as the eosinophil cell, and when at rest is spherical in shape and contains a single round or oval nucleus. The protoplasm is free from granules, but it is not quite transparent, having the appearance of ground glass. 4. The fourth kind of colorless corpuscle is designated the lymphocyte, because it is identical with the cell derived from the lymph glands or other lymphoid tissue. It is the smallest of the leukocytes and consists chiefly of a spheroidal nucleus with a very little surrounding protoplasm of a homogeneous nature. It is regarded as the immature form of the hyaline cell. The third and fourth varieties together constitute from 20 to 30% of the colorless corpuscles, but of these 2 varieties the lymphocytes are by far the more numerous. Leukocytes having in their protoplasm granules which stain with basic dyes, basophil, have been described as occurring in human blood, but they are rarely found except in disease. The colorless corpuscles are very various in shape in living blood, because many of them have the power of constantly changing their form by protruding finger-shaped or filamentous processes of their substance, by which they move and take up granules from the surrounding medium. In locomotion, the corpuscle pushes out a process of its substance, a pseudopodium as it is called, and then shifts the rest of the body into it. In the same way, when any granule or particle comes in its way, the corpuscle wraps a pseudopodium around it, and then withdraws the pseudopodium with the contained particle into its own substance. By means of these amoeboid properties, the cells have the power of wandering or emigrating from the blood vessels by penetrating their walls, and thus finding their way into the extravascular spaces. A chemical investigation of the protoplasm of the leukocytes shows the presence of nucleoprotein and of globulin. The occurrence of small amounts of fat, lecithin, and glycogen may also be demonstrated. The blood platelets are discoid or irregularly shaped, colorless, refractile bodies, much smaller than the red corpuscles. Each contains a central chromatin mass resembling a nucleus. Blood platelets possess the power of amoeboid movement. When blood is shed, they rapidly disintegrate and form granular masses, setting free prothrombin and the substance called by Howell thromboplastin. It is doubtful whether they exist normally in circulating blood. End of section 2.
Section 3 of Gray's Anatomy, Part 3. This is a Librivox recording. All Librivox recordings are in the public domain. For more information or to volunteer, please visit librivox.org. Recording by Julie van Mallegem. Anatomy of the Human Body, Part 3, by Henry Gray. Development of the Vascular System, Part 1. Blood vessels first make their appearance in several scattered vascular areas, which are developed simultaneously between the endoderm and the mesoderm of the yolk sac, i.e., outside the body of the embryo. Here a new type of cell, The angioblast, or vasoformative cell, is differentiated from the mesoderm. These cells, as they divide, form small, dense, syncytial masses, which soon join with similar masses by means of fine processes to form plexuses. These plexuses increase both by division and growth of its cells and by the addition of new angioblasts, which differentiate from the mesoderm. Within these solid plexuses, and also within the isolated masses of angioblasts, vacuoles appear, through liquefaction of the central part of the syncytium into plasma. The lumen of the blood vessels, thus formed, is probably intracellular. The flattened cells at the periphery form the endothelium. The nucleated red blood corpuscles develop either from small masses of the original angioblast, left attached to the inner wall of the lumen, or directly from the flat endothelial cells. In either case, the syncytial mass thus formed projects from and is attached to the wall of the vessel. Such a mass is known as a blood island, and hemoglobin gradually accumulates within it. Later, the cells on the surface round up, giving the mass a mulberry-like appearance. Then the red blood cells break loose and are carried away in the plasma. Such free blood cells continue to divide. The term blood island was originally used for the syncytial masses of angioblasts found in the area vasculosa, but it is probably best to limit the term to the masses within the lumen from which the red blood cells arise, as Sabin has done. Blood islands have been seen in the area vasculosa, in the omphalocement vein and arteries, and in the dorsal aorta. The differentiation of angioblasts from the mesoderm occurs not only in the area vasculosa, but within the embryo, and probably most of the larger blood vessels are developed in situ in this manner. This process of the differentiation of angioblasts from the mesoderm probably ceases in different regions of the embryo at different periods, and after its cessation new vessels are formed by sprouts from vessel already laid down in the form of capillary plexuses. The first rudiment of the heart appears as a pair of tubular vessels which are developed in the splanchnople of the pericardial area. These are named the primitive aortae, and a direct continuity is soon established between them and the vessels of the yolk sac. Each receives anteriorly a vein, the vitelline vein, from the yolk sac, and is prolonged backward on the lateral aspect of the notochord under the name of the dorsal aorta. The dorsal aortae give branches to the yolk sac, and are continued backward through the body stalk, as the umbilical arteries to the villi of the chorion. Ethernet describes the circulation in an embryo, which he estimates to be about 13 days old. The rudiment of the heart is situated immediately below the foregut, and consists of a short stem. It gives off two vessels, the primitive aortae, which run backward, one on either side of the notochord, and then pass into the body stalk, along which they are carried to the chorion. From the chorionic villi the blood is returned by a pair of umbilical veins, which unite in the body stalk to form a single vessel and subsequently encircle the mouth of the yolk sac and open into the heart. At the junction of the yolk sac and body stalk, each vein is joined by a branch from the vascular plexus of the yolk sac. From his observations, it seems that in the human embryo the chorionic circulation is established before that on the yolk sac. By the forward growth and flexure of the head, the pericardial area and the anterior portions of the primitive aortae are folded backward on the ventral aspect of the foregut, and the original relation of the somatoplar and splanchnoplare layers of the pericardial area is reversed. Each primitive aorta now consists of a ventral and a dorsal part connected anteriorly by an arch. These three parts are named respectively the anterior ventral aorta, the dorsal aorta, and the first cephalic arch. The vitelline veins, which enter the embryo through the anterior wall of the umbilical orifice, are now continuous with the posterior ends of the anterior ventral aorta. With the formation of the tail fold, the posterior parts of the primitive aortae are carried forward in a ventral direction to form the posterior ventral aortae and primary caudal arches. In the pericardial region, the two primitive aortae grow together and fuse to form a single tubular heart, the posterior end of which receives the two vitelline veins, while from its anterior end the two anterior ventral aortae emerge. The first cephalic arches pass through the mandibular arches, and behind them 5 additional pairs subsequently develop, so that altogether 6 pairs of aortic arches are formed. The 5th arches are very transitory vessels connecting the ventral aortae with the dorsal ends of the 6 arches. By the rhythmical contraction of the tubular heart, the blood is forced through the aortae and blood vessels of the vascular area, from which it is returned to the heart by the vitelline veins. This constitutes the vitelline circulation, and by means of it nutriment is absorbed from the org vitalis. The vitelline veins at first open separately into the posterior end of the tubular heart, but after a time their terminal portions fuse to form a single vessel. The vitelline veins ultimately drain the blood from the digestive tube and are modified to form the portal vein. This is caused by the growth of the liver, which interrupts their direct continuity with the heart, and the blood returned by them circulates through the liver before reaching the heart. With the atrophy of the yolk sac, the vitelline circulation diminishes and ultimately ceases, while an increasing amount of blood is carried through the umbilical arteries to the villi of the chorion. Subsequently, as the non-placental chorionic villi atrophy, their vessels disappear, and then the umbilical arteries convey the whole of their contents to the placenta, whence it is returned to the heart by the umbilical veins. In this manner the placental circulation is established, and by means of it nutritive materials are absorbed from, and waste products given up to the maternal blood. The umbilical veins, like the vitelline, undergo interruption in the developing liver, and the blood returned by them passes through this organ before reaching the heart. Ultimately, the right umbilical vein shrivels up and disappears. During the occurrence of these changes, great alterations take place in the primitive heart and blood vessels. Further development of the heart. Between the endothelial lining and the outer wall of the heart, there exists for a time an intricate trabecular network of mesodermal tissue from which, at a later stage, the musculi papillaris, chordae tenonii, and trabeculae are developed. The simple tubular heart, already described, becomes elongated and bends on itself so as to form an S-shaped loop, the anterior part bending to the right and the posterior part to the left. The intermediate portion arches transversely from left to right, and then turns sharply forward into the anterior part of the loop. Slight contractions make their appearance in the tube, and divide it from behind forward into 5 parts, viz. 1. The sinus venosus, 2. The primitive atrium, 3. The primitive ventricle, 4. The bulbus cordis, and 5. The truncus arteriosus. The constriction between the atrium and ventricle constitutes the arterial canal and indicates the site of the future atrioventricular valves. The sinus venosus is at first situated in the septum transversum, a layer of mesoderm in which the liver and the central tendon of the diaphragm are developed, behind the primitive atrium, and is formed by the union of the vitelline veins. The veins or ducts of Cuvier from the body of the embryo and the umbilical veins from the placenta subsequently open into it. The sinus is at first transversely and opens by a median aperture into the primitive atrium. Soon, however, it assumes an oblique position and becomes crescentic in form. Its right half or horn increases more rapidly than the left, and the opening into the atrium now communicates with the right portion of the atrial cavity. The right horn and transverse portion of the sinus ultimately become incorporated with and form a part of the adult right atrium, the line of union between it and the auricula being indicated in the interior of the atrium by a vertical crest, the crista terminalis of His. The left horn, which ultimately receives only the left duct of Cuvier, persists as the coronary sinus. The vitelline and umbilical veins are soon replaced by a single vessel, the inferior vena cava, and the three veins, inferior vena cava and right and left Cuvierian ducts, open into the dorsal aspect of the atrium by a common slit-like aperture. The upper part of this aperture represents the opening of the permanent superior vena cava, the lower that of the inferior vena cava, and the intermediate part the orifice of the coronary sinus. The slit-like aperture lies obliquely and is guarded by two halves, the right and left venous valves. Above the opening these unite with each other and are continuous with the fold named the septum spurium. Below the opening they fuse to form a triangular thickening, the spina vestibuli. The right venous valve is retained. A small septum, the sinus septum, grows from the posterior wall of the sinus venosus, diffused with the valve, and divided into two parts, an upper, the valve of the inferior vena cava, and a lower, the valve of the coronary sinus. The extreme upper portion of the right venous valve, together with the septum spurium, form the crista terminalis already mentioned. The upper and middle thirds of the left venous valve disappear. The lower third is continued into the spina vestibuli, and later fuses with the septum secundum of the atria, and takes part in the formation of the limbus fossae ovalis. The atrial canal is at first a short straight tube, connecting the atrial with the ventricular portion of the heart, but its growth is relatively slow, and it becomes overlapped by the atria and ventricles, so that its position on the surface of the heart is indicated only by an annular constriction. Its lumen is reduced to a transverse slit, and two thickenings appear, one on its dorsal and another on its ventral wall, or endocardial cushions, as they are termed, project into the canal, and, meeting in the middle-line, unite to form the septum intermedium, which divides the canal into two channels, the future right and left atrioventricular orifices. The primitive atrium grows rapidly and partially encircles the bulbus cordis. The groove against which the bulbus cordis lies is the first indication of a division into right and left atria. The cavity of the primitive atrium becomes subdivided into right and left chambers by a septum, the septum primum, which grows downward into the cavity. For a time the atria communicate with each other by an opening, the ostium primum of Born, below the free margin of the septum. This opening is closed by the union of the septum primum with the septum intermedium, and the communication between the atria is re-established through an opening which is developed in the upper part of the septum primum. This opening is known as a foramen ovale, ostium secundum of Born, and persists until birth. A second septum, the septum secundum, semilunar in shape, grows downward from the upper wall of the atrium immediately to the right of the primary septum and foramen ovale. Shortly after birth it fuses with the primary septum, and by this means the foramen ovale is closed, but sometimes the fusion is incomplete, and the upper part of the foramen remains patent. The limbus fossae ovalis denotes the free margin of the septum secundum. Issuing from each lung is a pair of pulmonary veins. Each pair unites to form a single vessel, and these in turn join in a common trunk which opens into the left atrium. Subsequently, the common trunk and the two vessels forming it expand and form the vestibule, or greater part of the atrium, the expansion reaching as far as the openings of the four vessels, so that in the adult all four veins open separately into the left atrium. The primitive ventricle becomes divided by a septum, the septum inferius or ventricular septum, which grows upward from the lower part of the ventricle, its position being indicated on the surface of the heart by a furrow. Its dorsal part increases more rapidly than its ventral portion, and fuses with the dorsal part of the septum intermedium. For a time an interventricular foramen exists above its ventral portion, but this foramen is ultimately closed by the fusion of the aortic septum with the ventricular septum. When the heart assumes its S-shaped form, the bulbus cordis lies ventral to and in front of the primitive ventricle. The adjacent walls of the bulbus cordis and ventricle approximate, fuse, and finally disappear, and the bulbus cordis now communicates freely with the right ventricle, while the junction of the bulbus with the truncus arteriosus is brought directly ventral to and applied to the atrial canal. By the upgrowth of the ventricular septum, the bulbus cordis is in great measure separated from the left ventricle, but remains an integral part of the right ventricle, of which it forms the infundibulum. The truncus arteriosus and bulbus cordis are divided by the aortic septum. This makes its appearance in three portions. 1. Two distal ridge-like thickenings project into the lumen of the tube. These increase in size and ultimately meet and fuse to form a septum, which takes a spiral course toward the proximal end of the truncus arteriosus. It divides the distal part of the truncus into two vessels, the aorta and pulmonary artery, which lie side by side above, but near the heart. The pulmonary artery is in front of the aorta. 2. 4 endocardial cushions appear in the proximal part of the truncus arteriosus in the region of the future semilunar valves. The manner in which these are related to the aortic septum is described below. 3. 2 endocardial thickenings, anterior and posterior, develop in the bulbus cordis and unite to form a short septum. This joins above with the aortic septum and below with the ventricular septum. The septum grows down into the ventricle as an oblique partition, which ultimately blends with the ventricular septum in such a way as to bring the bulbus cordis into communication with the pulmonary artery, and through the latter with the sixth pair of aortic arches, while the left ventricle is brought into continuity with the aorta, which communicates with the remaining aortic arches. End of section 3. Section 4 of Gray's Anatomy, Part 3. This is a Librivox recording. All Librivox recordings are in the public domain. For more information or to volunteer, please visit librivox.org. Recording by Julie van Wallichem. Anatomy of the Human Body, Part 3, by Henry Gray. Development of the Vascular System, Part 2. The valves of the heart. The atrioventricular valves are developed in relation to the atrial canal. By the upward expansion of the bases of the ventricles, the canal becomes invaginated into the ventricular cavities. The invaginated margin forms the rudiments of the lateral cusps of the atrioventricular valves. The medial or septal cusps of the valves are developed as downward prolongations of the septum intermedium. The aortic and pulmonary semilunar valves are formed from 4 endocardial thickenings, an anterior, a posterior, and 2 lateral, which appear at the proximal end of the truncus arteriosus. As the aortic septum grows downward, it divides each of the lateral thickenings into 2, thus giving rise to 6 thickenings, the rudiments of the semilunar valves, 3 at the aortic and 3 at pulmonary orifice. Further development of the arteries. Recent observations show that practically none of the main vessels of the adult arise as such in the embryo. In the site of each vessel a capillary network forms, and by the enlargement of definite paths, in this the larger arteries and veins are developed. The branches of the main arteries are not always simple modifications of the vessels of the capillary network, but may arise as new outgrowths from the enlarged stem. It has been seen that each primitive aorta consists of a ventral and a dorsal part, which are continuous through the first aortic arch. The dorsal aortae at first run backward, separately on either side of the notochord, but about the 3rd week they fuse from about the level of the 4th thoracic to that of the 4th lumbar segment to form a single trunk, the descending aorta. The first aortic arches run through the mandibular arches, and behind them 5 additional pairs are developed within the visceral arches, so that, in all, 6 pairs of aortic arches are formed. The first and second arches pass between the ventral and dorsal aortae, while the others arise at first by a common trunk from the truncus arteriosus, but end separately in the dorsal aortae. As the neck elongates, the ventral aortae are drawn out, and the 3rd and 4th arches arise directly from these vessels. In fishes, these arches persist, and give off branches to the gills, in which the blood is oxygenated. In mammals some of them remain as permanent structures, while others disappear or become obliterated. The anterior ventral aortae. These persist on both sides. The right forms a. the innominate artery, b. the right common and external carotid arteries. The left gives rise to a. the short portion of the aortic arch, which reaches from the origin of the innominate artery to that of the left common carotid artery, b. the left common and external carotid arteries. The aortic arches. The first and second arches disappear early, but the dorsal end of the second gives origin to the stapedial artery, a vessel which atrophies in man but persists in some mammals. It passes through the ring of the stapes and divides into supraorbital, infraorbital, and mandibular branches, which follow the 3 divisions of the trigeminal nerve. The infraorbital and mandibular arise from a common stem, the terminal part of which anastomoses with the external carotid. On the obliteration of the stapedial artery, this anastomosis enlarges and forms the internal maxillary artery, and the branches of the stapedial artery are now branches of this vessel. The common stem of the infraorbital and mandibular branches passes between the 2 roots of the auriculotemporal nerve and becomes the middle meningeal artery. The original supraorbital branch of the stapedial is represented by the orbital twigs of the middle meningeal. The 3rd aortic arch constitutes the commencement of the internal carotid artery, and is, therefore, named the carotid arch. The 4th right arch forms the right subclavian as far as the origin of its internal mammary branch, while the 4th left arch constitutes the arch of the aorta between the origin of the left carotid artery and the termination of the ductus arteriosus. The 5th arch disappears on both sides. The 6th right arch disappears. The 6th left arch gives off the pulmonary arteries and forms the ductus arteriosus. This duct remains pervious during the whole of fetal life, but is obliterated a few days after birth. His showed that in the early embryo the right and left arches each gives a branch to the lungs, but that later both pulmonary arteries take origin from the left arch. The dorsal aortae persist and form the continuations of the internal carotid arteries. These arteries pass to the brain, and each divides into an anterior and a posterior branch, the former giving off the ophthalmic and the anterior and middle cerebral arteries, while the latter turns back and joins the cerebral part of the vertebral artery. Behind the third arch, the right dorsal aorta disappears as far as the point where the two dorsal aortae fuse to form the descending aorta. The part of the left dorsal aorta between the third and fourth arches disappears, while the remainder persists to form the descending part of the arch of the aorta. A constriction, the aortic isthmus, is sometimes seen in the aorta between the origin of the left subclavian and the attachment of the ductus arteriosus. Sometimes the right subclavian artery arises from the aortic arch distal to the origin of the left subclavian and passes upward and to the right behind the trachea and esophagus. This condition may be explained by the persistence of the right dorsal aorta and the obliteration of the fourth right arch. In birds, the fourth right arch forms the arch of the aorta. In reptiles, the fourth arch on both sides persists and gives rise to the double aortic arch in these animals. The heart originally lies on the ventral aspect of the pharynx, immediately behind the stomacheum. With the elongation of the neck and the development of the lungs, it recedes within the thorax, and as a consequence, the anterior ventral aortae are drawn out, and the original position of the fourth and fifth arches is greatly modified. Thus, on the right side, the fourth recedes to the root of the neck, while on the left side it is withdrawn within the thorax. The recurrent nerves originally pass to the larynx under the sixth pair of arches, and are therefore pulled backward with the descent of these structures, so that in the adult the left nerve hooks around the ligamentum arteriosum. Owing to the disappearance of the fifth and the sixth right arches, the right nerve hooks around that immediately above them, i.e., the commencement of the subclavian artery. Segmental arteries arise from the primitive dorsal aortae, and course between successive segments. The seventh segmental artery is of special interest, since it forms the lower end of the vertebral artery, and, when the forelimb bud appears, sends a branch to it, the subclavian artery. From the seventh segmental arteries, the entire left subclavian and the greater part of the right subclavian are formed. The second pair of segmental arteries accompany the hypoglossal nerves to the brain, and are named the hypoglossal arteries. Each sends forward a branch which forms the cerebral part of the vertebral artery and anastomoses with the posterior branch of the internal carotid. The two vertebrals unite on the ventral surface of the hindbrain to form the basilar artery. Later, the hypoglossal artery atrophies, and the vertebral is connected with the first segmental artery. The cervical part of the vertebral is developed from a longitudinal anastomosis between the first 7 segmental arteries, so that the seventh of these ultimately becomes the source of the artery. As a result of the growth of the upper limb, the subclavian artery increases greatly in size, and the vertebral then appears to spring from it. Recent observations show that several segmental arteries contribute branches to the upper limb bud, and form in it a free capillary anastomosis. Of these branches, only one, viz., that derived from the seventh segmental artery, persists to form the subclavian artery. The subclavian artery is prolonged into the limb under the names of the axillary and brachial arteries, and these together constitute the arterial stem for the upper arm. The direct continuation of this stem in the forearm is a volar interosseous artery. A branch which accompanies the median nerve soon increases in size and forms the main vessel, median artery, of the forearm, while the volar interosseous diminishes. Later, the radial and ulnar arteries are developed as branches of the brachial part of the stem, and coincidentally with their enlargement the median artery recedes. Occasionally it persists as a vessel of some considerable size, and then accompanies the median nerve into the palm of the hand. The primary arterial stem for the lower limb is formed by the inferior gluteal sciatic artery, which accompanies the sciatic nerve along the posterior aspect of the thigh to the back of the knee, whence it is continued at a perineal artery. This arrangement exists in reptiles and amphibians. The femoral artery arises later as a branch of the common iliac, and passing down the front and medial side of the thigh to the bend of the knee, joins the inferior gluteal artery. The femoral quickly enlarges, and coincidentally with this, the part of the inferior gluteal immediately above the knee undergoes atrophy. The anterior and posterior tibial arteries are branches of the main arterial stem. Further development of the veins. The formation of the great veins of the embryo may be best considered by dividing them into two groups, visceral and parietal. The visceral veins. The visceral veins are the two vitelline or omphallomesenteric veins bringing the blood from the yolk sac, and the two umbilical veins returning the blood from the placenta. These four veins open close together into the sinus venosus. The vitelline veins run upward, at first in front, and subsequently on either side of the intestinal canal. They unite on the ventral aspect of the canal, and beyond this are connected to one another by two anastomotic branches, one on the dorsal and the other on the ventral aspect of the duodenal portion of the intestine, which is thus encircled by two venous rings. Into the middle or dorsal anastomosis the superior mesenteric vein opens. The portions of the veins above the upper ring become interrupted by the developing liver and broken up by it into a plexus of small capillary-like vessels termed sinusoids, Minot. The branches conveying the blood to this plexus are named the venae advenientes, and become the branches of the portal vein, while the vessels draining the plexus into the sinus venosus are termed the venae revehentes, and form the future hepatic veins. Ultimately, the left vena revehens no longer communicates directly with the sinus venosus, but opens into the right vena revehens. The persistent part of the upper venous ring, above the opening of the superior mesenteric vein, forms the trunk of the portal vein. The two umbilical veins fuse early to form a single trunk in the body stalk, but remain separate within the embryo, and pass forward to the sinus venosus in the side walls of the body. Like the vitelline veins, their direct connection with the sinus venosus becomes interrupted by the developing liver, and thus, at this stage, the whole of the blood from the yolk sac and placenta passes through the substance of the liver before it reaches the heart. The right umbilical and right vitelline veins shrivel and disappear. The left umbilical, on the other hand, becomes enlarged, and opens into the upper venous ring of the vitelline veins. With the atrophy of the yolk sac, the left vitelline vein also undergoes atrophy and disappears. Finally, a direct branch is established between this ring and the right hepatic vein. This branch is named the ductus venosus, and enlarging rapidly, it forms a wide channel through which most of the blood, returned from the placenta, is carried direct to the heart, without passing through the liver. A small proportion of the blood from the placenta is, however, conveyed from the left umbilical vein to the liver through the left vena advehens. The left umbilical vein and the ductus venosus undergo atrophy and obliteration after birth, and form respectively the ligamentum teres and ligamentum venosum of the liver. The parietal veins. The first indication of a parietal system consists in the appearance of two short transverse veins, the ducts of Cuvier, which open one on either side into the sinus venosus. Each of these ducts receives an ascending and descending vein. The ascending veins return the blood from the parietes of the trunk and from the Wolffian bodies, and are called cardial veins. The descending veins return the blood from the head, and are called primitive jugular veins. The blood from the lower limbs is collected by the right and left iliac and hypogastric veins, which in the earlier stages of development open into the corresponding right and left cardial veins. Later, a transverse branch, the left common iliac vein, is developed between the lower parts of the two cardial veins, and through this the blood is carried into the right cardinal vein. The portion of the left cardial vein below the left renal vein atrophies and disappears up to the point of entrance of the left spermatic vein. The portion above the left renal vein persists as a hemiazygous and accessory hemiazygous veins and the lower portion of the highest left intercostal vein. The right cardinal vein, which now receives the blood from both lower extremities, forms a large venous trunk along the posterior abdominal wall. Up to the level of the renal veins, it forms the lower part of the inferior vena cava. Above the level of the renal veins, the right cardinal vein persists as an azygous vein and receives the right intercostal veins, while the hemiazygous veins are brought into communication with it by the development of transverse branches in front of the vertebral column. Inferior vena cava. The development of the inferior vena cava is associated with the formation of two veins, the subcardinal veins. These lie parallel to and on the ventral aspect of the cardinal veins, and originate as longitudinal anastomosing channels which link up the tributaries from the mesentery to the cardinal veins. They communicate with the cardinal veins above and below, and also by a series of transverse branches. The two subcardinals are for a time connected with each other in front of the aorta by cross branches, but these disappear and are replaced by a single transverse channel at a level where the renal veins join the cardinals, and at the same level a cross-communication is established on either side between the cardinal and subcardinal. The portion of the left subcardinal behind this cross-communication disappears, while that in front, i.e. the pre-renal part, forms a connection with the ductus venosus at a point of opening of the hepatic veins, and rapidly enlarging, receives the blood from the post-renal part of the right cardinal through the cross-communication referred to. In this manner, a single trunk, the inferior vena cava, is formed, and consists of the proximal part of the ductus venosus, the pre-renal part of the right subcardinal vein, the post-renal part of the right cardinal vein, and the cross branch which joins these two veins. The left subcardinal disappears, except the part immediately in front of the renal vein, which is retained as a left suprarenal vein. The spermatic or ovarian vein opens into the post-renal of the corresponding cardinal vein. This portion of the right cardinal, as already explained, forms the lower part of the inferior vena cava, so that the right spermatic opens directly into that vessel. The post-renal segment of the left cardinal disappears, with the exception of the portion between the spermatic and renal vein, which is retained as a terminal part of the left spermatic vein. In consequence of the atrophy of the Wolffian bodies, the cardinal veins diminish in size. The primitive jugular veins, on the other hand, become enlarged, owing to the rapid development of the head and brain. They are further augmented by receiving the veins subclavian from the upper extremities, and so come to form the chief veins of the Cuvierian ducts. These ducts gradually assume an almost vertical position in consequence of the descent of the heart into the thorax. The right and left Cuvierian ducts are originally of the same diameter, and are frequently termed the right and left superior venae cavae. By the development of a transverse branch, the left innominate vein, between the two primitive jugular veins, the blood is carried across from the left to the right primitive jugular. The portion of the right primitive jugular vein between the left innominate and the azygous vein forms the upper part of the superior vena cava of the adult. The lower part of this vessel, i.e., below the entrance of the azygous vein, is formed by the right Cuvierian duct. Below the origin of the transverse branch, the left primitive jugular vein and left Cuvierian duct atrophy, the former constituting the upper part of the highest left intercostal vein, while the latter is represented by the ligament of the left vena cava, vestigial fold of Marshall, and the oblique vein of the left atrium, oblique vein of Marshall. Both right and left superior venae cavae are present in some animals, and are occasionally found in the adult human being. The oblique vein of the left atrium passes downward across the back of the left atrium to open into the coronary sinus, which, as already indicated, represents the persistent left horn of the sinus venosus. Venous sinuses of the dura mater. The primary arrangement for drainage of the capillaries of the head consists of a primary head vein which starts in the region of the midbrain and runs caudalward along the side of the brain tube to terminate at the duct of Cuvier. The primary head vein drains three plexuses of capillaries, the anterior dural plexus, the middle dural plexus, and the posterior dural plexus. The growth of the cartilaginous capsule of the ear and the growth and alteration in form of the brain bring about changes in this primary arrangement. Owing to the growth of the otic capsule and middle ear, the course of the primary head vein becomes unfavorable, and a segment of it becomes obliterated. To make the necessary adjustment, an anastomosis is established above the otic capsule, and the middle plexus drains into the posterior plexus. Then the anterior plexus fuses with the middle plexus, and drains through it and the newly established channel dorsal to the otic capsule. All that remains of the primary head vein is a cardinal portion or internal jugular, and the part in the region of the trigeminal nerve which may be called the cavernous sinus. Into it drain the orbital veins. The drainage from the cavernous sinus is now upward through the original trunk of the middle plexus, which is now the superior petrosal sinus, into the newly established dorsal channel. This dorsal channel is a transverse sinus. The inferior petrosal sinus appears later. From the anterior plexus a sagittal plexus extends forward, from which develops the superior sagittal sinus. The straight sinus is formed in the ventral part of the sagittal plexus. As the hemispheres extend backward, these sinuses elongate by incorporating the more caudal loops of the plexus. The anterior part of the sinus is completed first. The external jugular vein at first drains the region behind the ear, posterior auricular, and enters the primitive jugular as a lateral tributary. A group of veins from the face and lingual region converge to form a common vein, the linguofacial, which also terminates in the primitive jugular. Later, cross communication developed between the external jugular and the linguofacial, with the result that a posterior group of facial veins is transferred to the external jugular. End of section 4.
Section 5 of Gray's Anatomy, Part 3. This is a Librivox recording. All Librivox recordings are in the public domain. For more information or to volunteer, please visit Librivox.org. Recording by Leanne Howlett. Anatomy of the Human Body, Part 3, by Henry Gray. The Thoracic Cavity. The heart and lungs are situated in the thorax, the walls of which afford them protection. The heart lies between the two lungs, and is enclosed within a fibrous bag, the pericardium, while each lung is invested by a serous membrane, the pleura. The skeleton of the thorax and the shape and boundaries of the cavity have already been described. The cavity of the thorax. The capacity of the cavity of the thorax does not correspond with its apparent size externally, because 1. The space enclosed by the lower ribs is occupied by some of the abdominal viscera, and 2. The cavity extends above the anterior parts of the first ribs into the neck. The size of the thoracic cavity is constantly varying during life with the movements of the ribs and diaphragm, and with the degree of distension of the abdominal viscera. From the collapsed state of the lungs, as seen when the thorax is opened in the dead body, it would appear as if the viscera only partly filled the cavity, but during life there is no vacant space, that which is seen after death being filled up by the expanded lungs. The upper opening of the thorax. The parts which pass through the upper opening of the thorax are, from before backward, in or near the middle line, the sternohyoideus and sternothyroideus muscles, the remains of the thymus, the inferior thyroid veins, the trachea, esophagus, thoracic duct, and the longus colli muscles. At the sides, the innominate artery, the left common carotid, left subclavian and internal mammary arteries, and the costo-cervical trunks, the innominate veins, the vagus, cardiac, phrenic, and sympathetic nerves, the greater parts of the anterior divisions of the first thoracic nerves, and the recurrent nerve of the left side. The apex of each lung, covered by the pleura, also projects through this aperture, a little above the level of the sternal end of the first rib. The lower opening of the thorax. The lower opening of the thorax is wider transversely than from before backward. It slopes obliquely downward and backward, so that the thoracic cavity is much deeper behind than in front. The diaphragm closes the opening and forms the floor of the thorax. The floor is flatter at the center than at the sides, and higher on the right side than on the left. In the dead body the right side reaches the level of the upper border of the fifth costal cartilage, while the left extends only to the corresponding part of the sixth costal cartilage. From the highest point on each side the floor slopes suddenly downward to the costal and vertebral attachments of the diaphragm. This slope is more marked behind than in front, so that only a narrow space is left between the diaphragm and the posterior wall of the thorax. 4a. The pericardium. The pericardium is a conical fibroserous sac in which the heart and the roots of the great vessels are contained. It is placed behind the sternum and the cartilages of the third, fourth, fifth, sixth, and seventh ribs of the left side in the mediastinal cavity. In front, it is separated from the anterior wall of the thorax, in the greater part of its extent, by the lungs and pleurae, but a small area, somewhat variable in size, and usually corresponding with the left half of the lower portion of the body of the sternum and the medial ends of the cartilages of the fourth and fifth ribs of the left side, comes into direct relationship with the chest wall. The lower extremity of the thymus, in the child, is in contact with the front of the upper part of the pericardium. Behind, it rests upon the bronchi, the esophagus, the descending thoracic aorta, and the posterior part of the mediastinal surface of each lung. Laterally it is covered by the pleurae, and is in relation with the mediastinal surfaces of the lungs. The phrenic nerve, with its accompanying vessels, descends between the pericardium and pleura on either side. Structure of the pericardium. Although the pericardium is usually described as a single sac, an examination of its structure shows that it consists essentially of two sacs intimately connected with one another, but totally different in structure. The outer sac, known as the fibrous pericardium, consists of fibrous tissue. The inner sac, or serous pericardium, is a delicate membrane which lies within the fibrous sac and lines its walls. It is composed of a single layer of flattened cells resting on loose connective tissue. The heart invaginates the wall of the serous sac from above and behind, and practically obliterates its cavity, the space being merely a potential one. The fibrous pericardium forms a flask-shaped bag, the neck of which is closed by its fusion with the external coats of the great vessels, while its base is attached to the central tendon and to the muscular fibers of the left side of the diaphragm. In some of the lower mammals the base is either completely separated from the diaphragm or joined to it by some loose areolar tissue. In man, much of its diaphragmatic attachment consists of loose fibrous tissue which can be readily broken down, but over a small area the central tendon of the diaphragm and the pericardium are completely fused. Above, the fibrous pericardium not only blends with the external coats of the great vessels, but is continuous with the pretracheal layer of the deep cervical fascia. By means of these upper and lower connections it is securely anchored within the thoracic cavity. It is also attached to the posterior surface of the sternum by the superior and inferior sterno-pericardiac ligaments, the upper passing to the manubrium, and the lower to the xiphoid process. The vessels receiving fibrous prolongations from this membrane are the aorta, the superior vena cava, the right and left pulmonary arteries, and the four pulmonary veins. The inferior vena cava enters the pericardium through the central tendon of the diaphragm and receives no covering from the fibrous layer. The serous pericardium is, as already stated, a closed sac which lines the fibrous pericardium and is invaginated by the heart. It therefore consists of a visceral and a parietal portion. The visceral portion, or epicardium, covers the heart and the great vessels, and from the latter is continuous with the parietal layer which lines the fibrous pericardium. The portion which covers the vessels is arranged in the form of two tubes. The aorta and pulmonary artery are enclosed in one tube, the arterial mesocardium. The superior and inferior venae cavae and the four pulmonary veins are enclosed in a second tube, the venous mesocardium, the attachment of which to the parietal layer presents the shape of an inverted U. The cul-de-sac enclosed between the limbs of the U lies behind the left atrium and is known as the oblique sinus, while the passage between the venous and arterial mesocardia, that is, between the aorta and pulmonary artery in front and the atria behind, is termed the transverse sinus. The ligament of the left vena cava. Between the left pulmonary artery and subjacent pulmonary vein is a triangular fold of the serous pericardium. It is known as the ligament of the left vena cava, vestigial fold of Marshall. It is formed by the duplicature of the serous layer over the remnant of the lower part of the left superior vena cava, duct of Cuvier, which becomes obliterated during fetal life and remains as a fibrous band stretching from the highest left intercostal vein to the left atrium, where it is continuous with a small vein, the vein of the left atrium, oblique vein of Marshall, which opens into the coronary sinus. The arteries of the pericardium are derived from the internal mammary and its musculophrenic branch, and from the descending thoracic aorta. The nerves of the pericardium are derived from the vagus and phrenic nerves and the sympathetic trunks. End of section 5. Recording by Leanne Howlett.
Section 6 of Gray's Anatomy, Part 3. This is a Librivox recording. All Librivox recordings are in the public domain. For more information or to volunteer, please visit Librivox.org. Recorded by Laurie Anne Walden. Anatomy of the Human Body, Part 3, by Henry Gray. The Heart, Core. The heart is a hollow muscular organ of a somewhat conical form. It lies between the lungs and the middle mediastinum, and is enclosed in the pericardium. It is placed obliquely in the chest behind the body of the sternum and adjoining parts of the rib cartilages, and projects farther into the left than into the right half of the thoracic cavity, so that about 1/3 of it is situated on the right and 2/3 on the left of the median plane. Size. The heart in the adult measures about 12 cm in length, 8 to 9 cm in breadth at the broadest point, and 6 cm in thickness. Its weight in the male varies from 280 to 340 grams, in the female from 230 to 280 grams. The heart continues to increase in weight and size up to an advanced period of life. This increase is more marked in men than in women. Component parts. As has already been stated, the heart is subdivided by septa into right and left halves, and a constriction subdivides each half of the organ into 2 cavities, the upper cavity being called the atrium, the lower the ventricle. The heart, therefore, consists of 4 chambers, that is, right and left atria, and right and left ventricles. The division of the heart into 4 cavities is indicated on its surface by grooves. The atria are separated from the ventricles by the coronary sulcus, auriculoventricular groove. This contains the trunks of the nutrient vessels of the heart, and is deficient in front, where it is crossed by the root of the pulmonary artery. The interatrial groove, separating the 2 atria, is scarcely marked on the posterior surface, while anteriorly it is hidden by the pulmonary artery and aorta. The ventricles are separated by 2 grooves, one of which, the anterior longitudinal sulcus, is situated on the sternocostal surface of the heart, close to its left margin. The other, posterior longitudinal sulcus, on the diaphragmatic surface near the right margin. These grooves extend from the base of the ventricular portion to a notch, the incisura apicis cordis, on the acute margin of the heart, just to the right of the apex. The base, basis cordis, directed upward, backward, and to the right, is separated from the 5th, 6th, 7th, and 8th thoracic vertebrae by the esophagus, aorta, and thoracic duct. It is formed mainly by the left atrium, and, to a small extent, by the back part of the right atrium. Somewhat quadrilateral in form, it is in relation above with the bifurcation of the pulmonary artery, and is bounded below by the posterior part of the coronary sulcus, containing the coronary sinus. On the right it is limited by the sulcus terminalis of the right atrium, and on the left by the ligament of the left vena cava and the oblique vein of the left atrium. The 4 pulmonary veins, 2 on either side, open into the left atrium, while the superior vena cava opens into the upper, and the anterior vena cava into the lower, part of the right atrium. The apex, apex cordis. The apex is directed downward, forward, and to the left, and is overlapped by the left lung and pleura. It lies behind the 5th left intercostal space, 8 to 9 cm from the mid-sternal line, or about 4 cm below and 2 mm to the medial side of the left mammary papilla. The sternocostal surface is directed forward, upward, and to the left. Its lower part is convex, formed chiefly by the right ventricle, and traversed near its left margin by the anterior longitudinal sulcus. Its upper part is separated from the lower by the coronary sulcus, and is formed by the atria. It presents a deep concavity, occupied by the ascending aorta and the pulmonary artery. The diaphragmatic surface, directed downward and slightly backward, is formed by the ventricles, and rests upon the central tendon and a small part of the left muscular portion of the diaphragm. It is separated from the base by the posterior part of the coronary sulcus, and is traversed obliquely by the posterior longitudinal sulcus. The right margin of the heart is long, and is formed by the right atrium above and the right ventricle below. The atrial portion is rounded and almost vertical. It is situated behind the third, fourth, and fifth right costal cartilages, about 1.25 centimeters from the margin of the sternum. The ventricular portion, thin and sharp, is named the acute margin. It is nearly horizontal and extends from the sternal end of the sixth right costal cartilage to the apex of the heart. The left or obtuse margin is shorter, full, and rounded. It is formed mainly by the left ventricle, but to a slight extent above by the left atrium. It extends from a point in the second left intercostal space, about 2.5 millimeters from the sternal margin, obliquely downward, with a convexity to the left, to the apex of the heart. Right atrium, atrium dextrum, right auricle. The right atrium is larger than the left, but its walls are somewhat thinner, measuring about 2 millimeters. Its cavity is capable of containing about 57 cc. It consists of two parts, a principal cavity, or sinus venerum, situated posteriorly, and an anterior, smaller portion, the auricula. Sinus venerum, sinus venosus. The sinus venarum is the large quadrangular cavity placed between the two vena cavae. Its walls, which are extremely thin, are connected below with the right ventricle, and medially with the left atrium, but are free in the rest of their extent. Auricula, auricula dextra, right auricular appendix. The auricula is a small, conical, muscular pouch, the margins of which present a dentated edge. It projects from the upper and front part of the sinus forward and toward the left side, overlapping the root of the aorta. The separation of the auricula from the sinus venarum is indicated externally by a groove, the terminal sulcus, which extends from the front of the superior vena cava to the front of the inferior vena cava, and represents the line of union of the sinus venosus of the embryo with the primitive atrium. On the inner wall of the atrium the separation is marked by a vertical, smooth, muscular ridge, the terminal crest. Behind the crest the internal surface of the atrium is smooth, while in front of it the muscular fibers of the wall are raised into parallel ridges resembling the teeth of a comb, and hence named the musculi pectinati. Its interior presents the following parts for examination. Openings: superior vena cava, inferior vena cava, coronary sinus, foramina venarum minimarum, atrioventricular. Valves: valve of the inferior vena cava, valve of the coronary sinus, fossa ovalis, limbus fossae ovalis, intervenous tubercle, musculi pectinati, crista terminalis. The superior vena cava returns the blood from the upper half of the body, and opens into the upper and back part of the atrium, the direction of its orifice being downward and forward. Its opening has no valve. The inferior vena cava, larger than the superior, returns the blood from the lower half of the body, and opens into the lowest part of the atrium near the atrial septum, its orifice being directed upward and backward, and guarded by a rudimentary valve, the valve of the inferior vena cava, eustachian valve. The blood entering the atrium through the superior vena cava is directed downward and forward, that is, toward the atrioventricular orifice, while that entering through the inferior vena cava is directed upward and backward, toward the atrial septum. This is the normal direction of the two currents in fetal life. The coronary sinus opens into the atrium between the orifice of the inferior vena cava and the atrioventricular opening. It returns blood from the substance of the heart, and is protected by a semicircular valve, the valve of the coronary sinus, valve of Thebesius. The foramina venarum minimarum, foramina Thebesii, are the orifices of minute veins, veni cordis minimi, which return blood directly from the muscular substance of the heart. The atrioventricular opening, tricuspid orifice, is the large oval aperture of communication between the atrium and the ventricle. It will be described with the right ventricle. The valve of the inferior vena cava, valvula vena cavae inferioris or Eustachii, Eustachian valve, is situated in front of the orifice of the inferior vena cava. It is semilunar in form, its convex margin being attached to the anterior margin of the orifice. Its concave margin, which is free, ends in two cornua, of which the left is continuous with the anterior edge of the limbus fasciovalis, while the right is lost on the wall of the atrium. The valve is formed by a duplicature of the lining membrane of the atrium, containing a few muscular fibers. In the fetus this valve is of large size, and serves to direct the blood from the inferior vena cava through the foramen ovale into the left atrium. In the adult it occasionally persists, and may assist in preventing the reflux of blood into the inferior vena cava. More commonly it is small, and may present a cribriform or filamentous appearance. Sometimes it is altogether wanting. The valve of the coronary sinus, valvula sinus coronarii, or Thebesii, Thebesian valve, is a semicircular fold of the lining membrane of the atrium, at the orifice of the coronary sinus. It prevents the regurgitation of blood into the sinus during the contraction of the atrium. This valve may be double, or it may be cribriform. The fossa ovalis is an oval depression on the septal wall of the atrium, and corresponds to the situation of the foramen ovale in the fetus. It is situated at the lower part of the septum, above and to the left of the orifice of the inferior vena cava. The limbus fasciovalis, annulus ovalis, is the prominent oval margin of the fossa ovalis. It is most distinct above and at the sides of the fossa. Below it is deficient. A small slit-like valvular opening is occasionally found at the upper margin of the fossa, leading upward beneath the limbus into the left atrium. It is the remains of the fetal aperture between the two atria. The intervenous tubercle, tuberculum intervenosum, tubercle of Lower, is a small projection on the posterior wall of the atrium above the fossa ovalis. It is distinct in the hearts of quadrupeds, but in man is scarcely visible. It was supposed by Lower to direct the blood from the superior vena cava toward the atrioventricular opening. Right ventricle, ventriculus dexter. The right ventricle is triangular in form, and extends from the right atrium to near the apex of the heart. Its antero-superior surface is rounded and convex, and forms the larger part of the sternocostal surface of the heart. Its under surface is flattened, rests upon the diaphragm, and forms a small part of the diaphragmatic surface of the heart. Its posterior wall is formed by the ventricular septum, which bulges into the right ventricle, so that a transverse section of the cavity presents a semilunar outline. Its upper and left angle forms a conical pouch, the conus arteriosus, from which the pulmonary artery arises. A tendinous band, which may be named the tendon of the conus arteriosus, extends upward from the right atrioventricular fibrous ring, and connects the posterior surface of the conus arteriosus to the aorta. The wall of the right ventricle is thinner than that of the left, the proportion between them being as 1 to 3. It is thickest at the base, and gradually becomes thinner toward the apex. The cavity equals in size that of the left ventricle, and is capable of containing about 85 cc. Its interior presents the following parts for examination. Openings: right atrioventricular, pulmonary artery. Valves: tricuspid, pulmonary. Trabeculi carni, chordi tendini. The right atrioventricular orifice is the large oval aperture of communication between the right atrium and ventricle. Situated at the base of the ventricle, it measures about 4 cm in diameter, and is surrounded by a fibrous ring, covered by the lining membrane of the heart. It is considerably larger than the corresponding aperture on the left side, being sufficient to admit the ends of 4 fingers. It is guarded by the tricuspid valve. The opening of the pulmonary artery is circular in form, and situated at the summit of the conus arteriosus, close to the ventricular septum. It is placed above and to the left of the atrioventricular opening, and is guarded by the pulmonary semilunar valves. The tricuspid valve, valvula tricuspidalis, consists of 3 somewhat triangular cusps, or segments. The largest cusp is interposed between the atrioventricular orifice and the conus arteriosus, and is termed the anterior or infundibular cusp. A second, the posterior or marginal cusp, is in relation to the right margin of the ventricle. and a third, the medial or septal cusp, to the ventricular septum. They are formed by duplicatures of the lining membrane of the heart, strengthened by intervening layers of fibrous tissue. Their central parts are thick and strong, their marginal portions thin and translucent, and in the angles between the latter small intermediate segments are sometimes seen. Their bases are attached to a fibrous ring surrounding the atrioventricular orifice, and are also joined to each other so as to form a continuous annular membrane, while their apices project into the ventricular cavity. Their atrial surfaces, directed toward the blood current from the atrium, are smooth; their ventricular surfaces, directed toward the wall of the ventricle, are rough and irregular, and together with the apices and margins of the cusps, give attachment to a number of delicate tendinous cords, the chordae tendineae. The trabeculae carni, columnae carni, are rounded or irregular muscular columns which project from the whole of the inner surface of the ventricle, with the exception of the conus arteriosus. They are of three kinds. Some are attached along their entire length on one side, and merely form prominent ridges. Others are fixed at their extremities, but free in the middle; while a third set, musculi papillares, are continuous by their bases with the wall of the ventricle, while their apices give origin to the chordae tendineae, which pass to be attached to the segments of the tricuspid valve. There are two papillary muscles, anterior and posterior. Of these the anterior is the larger, and its chordae tendineae are connected with the anterior and posterior cusps of the valve. The posterior papillary muscle sometimes consists of 2 or 3 parts. Its chordae tendineae are connected with the posterior and medial cusps. In addition to these, some chordae tendineae spring directly from the ventricular septum or from small papillary eminences on it, and pass to the anterior and medial cusps. A muscular band, well marked in sheep and some other animals, frequently extends from the base of the anterior papillary muscle to the ventricular septum. From its attachments it may assist in preventing over-distension of the ventricle, and so has been named the moderator band. The pulmonary semilunar valves are 3 in number, 2 in front and 1 behind, formed by duplicatures of the lining membrane, strengthened by fibrous tissue. They are attached by their convex margins to the wall of the artery at its junction with the ventricle, their free borders being directed upward into the lumen of the vessel. The free and attached margins of each are strengthened by tendinous fibers, and the former presents, at its middle, a thickened nodule, corpus aurantii. From this nodule tendinous fibers radiate through the segment to its attached margin, but are absent from 2 narrow crescentic portions, the lunulae, placed one on either side of the nodule immediately adjoining the free margin. Between the semilunar valves and the wall of the pulmonary artery are 3 pouches or sinuses, sinuses of Valsalva. Left atrium, atrium sinistrum, left auricle. The left atrium is rather smaller than the right, but its walls are thicker, measuring about 3 millimeters. It consists, like the right, of 2 parts, a principal cavity and an auricula. The principal cavity is cuboidal in form, and concealed in front by the pulmonary artery and aorta. In front and to the right it is separated from the right atrium by the atrial septum. Opening into it on either side are the 2 pulmonary veins. Auricula, auricula sinistra, left auricular appendix. The auricula is somewhat constricted at its junction with the principal cavity. It is longer, narrower, and more curved than that of the right side. Its margins are more deeply indented. It is directed forward and toward the right, and overlaps the root of the pulmonary artery. The interior of the left atrium presents the following parts for examination: openings of the 4 pulmonary veins, left atrioventricular opening, musculi pectinati. The pulmonary veins, 4 in number, open into the upper part of the posterior surface of the left atrium, 2 on either side of its middle line. They are not provided with valves. The 2 left veins frequently end by a common opening. The left atrioventricular opening is the aperture between the left atrium and ventricle, and is rather smaller than the corresponding opening on the right side. The musculi pectinati, fewer and smaller than in the right auricula, are confined to the inner surface of the auricula. On the atrial septum may be seen a lunated impression, bounded below by a crescentic ridge, the concavity of which is turned upward. The depression is just above the fossa ovalis of the right atrium. End of section 6. Section 7 of Gray's Anatomy, Part 3. This is a Librivox recording. All Librivox recordings are in the public domain. For more information or to volunteer, please visit librivox.org. Recorded by Laurie Anne Walden. Anatomy of the Human Body, Part 3, by Henry Gray. The Heart, Part 2. Left ventricle, ventriculus sinister. The left ventricle is longer and more conical in shape than the right, and on transverse section its concavity presents an oval or nearly circular outline. It forms a small part of the sternocostal surface and a considerable part of the diaphragmatic surface of the heart. It also forms the apex of the heart. Its walls are about 3 times as thick as those of the right ventricle. Its interior presents the following parts for examination. Openings: left atrioventricular, aortic. Valves: bicuspid or mitral, aortic. Trabeculae carni, chordae tendinae. The left atrioventricular opening, mitral orifice, is placed below and to the left of the aortic orifice. It is a little smaller than the corresponding aperture of the opposite side, admitting only 2 fingers. It is surrounded by a dense fibrous ring, covered by the lining membrane of the heart, and is guarded by the bicuspid or mitral valve. The aortic opening is a circular aperture, in front and to the right of the atrioventricular, from which it is separated by the anterior cusp of the bicuspid valve. Its orifice is guarded by the aortic semilunar valves. The portion of the ventricle immediately below the aortic orifice is termed the aortic vestibule, and possesses fibrous instead of muscular walls. The bicuspid or mitral valve, valvula bicuspidalis or mitralis, is attached to the circumference of the left atrioventricular orifice in the same way that the tricuspid valve is on the opposite side. It consists of 2 triangular cusps formed by duplicatures of the lining membrane, strengthened by fibrous tissue, and containing a few muscular fibers. The cusps are of unequal size, and are larger, thicker, and stronger than those of the tricuspid valve. The larger cusp is placed in front and to the right between the atrioventricular and aortic orifices, and is known as the anterior or aortic cusp. The smaller or posterior cusp is placed behind and to the left of the opening. 2 smaller cusps are usually found at the angles of junction of the larger. The cusps of the bicuspid valve are furnished with chordae tendinae, which are attached in a manner similar to those on the right side. They are, however, thicker, stronger, and less numerous. The aortic semilunar valves are 3 in number, and surround the orifice of the aorta. 2 are anterior, right and left, and 1 posterior. They are similar in structure and in their mode of attachment to the pulmonary semilunar valves, but are larger, thicker, and stronger. The lunulae are more distinct, and the noduli or corpora arantii thicker and more prominent. Opposite the valves, the aorta presents slight dilatations, the aortic sinuses of Valsalva, which are larger than those at the origin of the pulmonary artery. The trabeculae carneae are of 3 kinds, like those upon the right side, but they are more numerous, and present a dense interlacement, especially at the apex and upon the posterior wall of the ventricle. The musculi papillaris are 2 in number, one being connected to the anterior, the other to the posterior wall. They are of large size, and end in rounded extremities, from which the chordae tendinae arise. The chordae tendinae from each papillary muscle are connected to both cusps of the bicuspid valve. Ventricular septum, septum ventriculorum, interventricular septum. The ventricular septum is directed obliquely backward and to the right, and is curved, with a convexity toward the right ventricle. Its margins correspond with the anterior and posterior longitudinal sulci. The greater portion of it is thick and muscular, and constitutes the muscular ventricular septum; but its upper and posterior part, which separates the aortic vestibule from the lower part of the right atrium and upper part of the right ventricle, is thin and fibrous, and is termed the membranous ventricular septum. An abnormal communication may exist between the ventricles at this part, owing to defective development of the membranous septum. Structure. The heart consists of muscular fibers and of fibrous rings which serve for their attachment. It is covered by the visceral layer of the serous pericardium, epicardium, and lined by the endocardium. Between these two membranes is the muscular wall, or myocardium. The endocardium is a thin, smooth membrane which lines and gives the glistening appearance to the inner surface of the heart. It assists in forming the valves by its reduplications, and is continuous with the lining membrane of the large blood vessels. It consists of connective tissue and elastic fibers, and is attached to the muscular structure by loose elastic tissue, which contains blood vessels and nerves. Its free surface is covered by endothelial cells. The fibrous rings surround the atrioventricular and arterial orifices, and are stronger upon the left than on the right side of the heart. The atrioventricular rings serve for the attachment of the muscular fibers of the atria and ventricles, and for the attachment of the bicuspid and tricuspid valves. The left atrioventricular ring is closely connected, by its right margin, with the aortic arterial ring. Between these and the right atrioventricular ring is a triangular mass of fibrous tissue, the trigonum fibrosum, which represents the os cordis seen in the heart of some of the larger animals, as the ox and elephant. Lastly, there is the tendinous band, already referred to, the posterior surface of the conus arteriosus. The fibrous rings surrounding the arterial orifices serve for the attachment of the great vessels and semilunar valves. Each ring receives, by its ventricular margin, the attachment of some of the muscular fibers of the ventricles. Its opposite margin presents 3 deep semicircular notches, to which the middle coat of the artery is firmly fixed. The attachment of the artery to its fibrous ring is strengthened by the external coat and serous membrane externally, and by the endocardium internally. From the margins of the semicircular notches, the fibrous structure of the ring is continued into the segments of the valves. The middle coat of the artery in this situation is thin, and the vessel is dilated to form the sinuses of the aorta and pulmonary artery. Cardiac muscular tissue. The fibers of the heart differ very remarkably from those of other striped muscles. They are smaller by 1/3, and their transverse striae are by no means so well marked. They show faint longitudinal striation. The fibers are made up of distinct quadrangular cells, joined end to end so as to form a syncytium. Each cell contains a clear oval nucleus situated near its center. The extremities of the cells have a tendency to branch or divide, the subdivisions uniting with offsets from other cells, and thus producing an anastomosis of the fibers. The connective tissue between the bundles of fibers is much less than in ordinary striped muscle, and no sarcolemma has been proved to exist. Purkinje fibers. Between the endocardium and the ordinary cardiac muscle are found, embedded in a small amount of connective tissue, peculiar fibers known as Purkinje fibers. They are found in certain mammals and in birds, and can be best seen in the sheep's heart, where they form a considerable portion of the moderator band, and also appear as gelatinous-looking strands on the inner walls of the atria and ventricles. They also occur in the human heart, associated with the terminal distributions of the bundle of His. The fibers are very much larger in size than the cardiac cells, and differ from them in several ways. In longitudinal section they are quadrilateral in shape, being about twice as long as they are broad. The central portion of each fiber contains one or more nuclei, and is made up of granular protoplasm, with no indication of striations. while the peripheral portion is clear and has distinct transverse striations. The fibers are intimately connected with each other, possess no definite sarcolemma, and do not branch. The muscular structure of the heart consists of bands of fibers which present an exceedingly intricate interlacement. They comprise a. the fibers of the atria, b. the fibers of the ventricles, and c. the atrioventricular bundle of His. The fibers of the atria are arranged in two layers, a superficial, common to both cavities, and a deep, proper to each. The superficial fibers are most distinct on the front of the atria, across the bases of which they run in a transverse direction, forming a thin and incomplete layer. Some of these fibers run into the atrial septum. The deep fibers consist of looped and annular fibers. The looped fibers pass upward over each atrium, being attached by their two extremities to the corresponding atrioventricular ring, in front and behind. The annular fibers surround the auriculae, and form annular bands around the terminations of the veins and around the fossa ovalis. The fibers of the ventricles are arranged in a complex manner, and various accounts have been given of their course and connections. The following description is based on the work of McCallum. They consist of superficial and deep layers, all of which, with the exception of two, are inserted into the papillary muscles of the ventricles. The superficial layers consist of the following: a. Fibers which spring from the tendon of the conus arteriosus, and sweep downward and toward the left, across the anterior longitudinal sulcus, and around the apex of the heart, where they pass upward and inward to terminate in the papillary muscles of the left ventricle. Those arising from the upper half of the tendon of the conus arteriosus pass to the anterior papillary muscle. Those from the lower half to the posterior papillary muscle and the papillary muscles of the septum. b. Fibers which arise from the right atrioventricular ring and run diagonally across the diaphragmatic surface of the right ventricle and around its right border onto its costosternal surface, where they dip beneath the fibers just described, and, crossing the anterior longitudinal sulcus, wind around the apex of the heart and end in the posterior papillary muscle of the left ventricle. c. Fibers which spring from the left atrioventricular ring and, crossing the posterior longitudinal sulcus, pass successively into the right ventricle and end in its papillary muscles. The deep layers are 3 in number. They arise in the papillary muscles of one ventricle, and, curving in an S-shaped manner, turn in at the longitudinal sulcus and end in the papillary muscles of the other ventricle. The layer which is most superficial in the right ventricle lies next the lumen of the left, and vice versa. Those of the first layer almost encircle the right ventricle, and, crossing in the septum to the left, unite with the superficial fibers from the right atrioventricular ring to form the posterior papillary muscle. Those of the second layer have a less extensive course in the wall of the right ventricle, and a correspondingly greater course in the left, where they join with the superficial fibers from the anterior half of the tendon of the conus arteriosus to form the papillary muscles of the septum. Those of the third layer pass almost entirely around the left ventricle and unite with the superficial fibers from the lower half of the tendon of the conus arteriosus to form the anterior papillary muscle. Besides the layers just described, there are 2 bands which do not end in papillary muscles. One springs from the right atrioventricular ring and crosses in the atrioventricular septum. It then encircles the deep layers of the left ventricle and ends in the left atrioventricular ring. The second band is apparently confined to the left ventricle. It is attached to the left atrioventricular ring, and encircles the portion of the ventricle adjacent to the aortic orifice. The atrioventricular bundle of His is the only direct muscular connection known to exist between the atria and the ventricles. Its cells differ from ordinary cardiac muscle cells in being more spindle-shaped. They are, moreover, more loosely arranged, and have a richer vascular supply than the rest of the heart muscle. It arises in connection with 2 small collections of spindle-shaped cells, the sinoatrial and atrioventricular nodes. The sinoatrial node is situated on the anterior border of the opening of the superior vena cava. From it strands of fusiform fibers run under the endocardium of the wall of the atrium to the atrioventricular node. The atrioventricular node lies near the orifice of the coronary sinus in the annular and septal fibers of the right atrium. From it the atrioventricular bundle passes forward in the lower part of the membranous septum and divides into right and left fasciculi. These run down in the right and left ventricles, one on either side of the ventricular septum covered by endocardium. In the lower parts of the ventricles they break up into numerous strands which end in the papillary muscles and in the ventricular muscle generally. The greater portion of the atrioventricular bundle consists of narrow, somewhat fusiform fibers, but its terminal strands are composed of Purkinje fibers. Dr. A. Morrison has shown that in the sheep and pig the atrioventricular bundle is a great avenue for the transmission of nerves from the auricular to the ventricular heart, large and numerous nerve trunks entering the bundle and coursing with it. From these, branches pass off and form plexuses around groups of Purkinje cells, and from these plexuses fine fibrils go to innervate individual cells. Clinical and experimental evidence go to prove that this bundle conveys the impulse to systolic contraction from the atrial septum to the ventricles. Vessels and nerves. The arteries supplying the heart are the right and left coronary from the aorta. The veins end in the right atrium. The lymphatics end in the thoracic and right lymphatic ducts. The nerves are derived from the cardiac plexus, which are formed partly from the vagi and partly from the sympathetic trunks. They are freely distributed both on the surface and in the substance of the heart, the separate nerve filaments being furnished with small ganglia. The cardiac cycle and the actions of the valves. By the contractions of the heart, the blood is pumped through the arteries to all parts of the body. These contractions occur regularly, and at the rate of about 70 per minute. Each wave of contraction, or period of activity, is followed by a period of rest, the two periods constituting what is known as a cardiac cycle. Each cardiac cycle consists of 3 phases, which succeed each other as follows. 1. A short simultaneous contraction of both atria, termed the atrial systole, followed, after a slight pause, by 2. A simultaneous, but more prolonged, contraction of both ventricles, named the ventricular systole, and 3. A period of rest, during which the whole heart is relaxed. The atrial contraction commences around the venous openings, and sweeping over the atria forces their contents through the atrioventricular openings into the ventricles, regurgitation into the veins being prevented by the contraction of their muscular coats. When the ventricles contract, the tricuspid and bicuspid valves are closed and prevent the passage of the blood back into the atria. The musculi papillaris at the same time are shortened, and pulling on the chordae tendineae prevent the inversion of the valves into the atria. As soon as the pressure in the ventricles exceeds that in the pulmonary artery and aorta, the valves guarding the orifices of these vessels are opened, and the blood is driven from the right ventricle into the pulmonary artery, and from the left into the aorta. The moment the systole of the ventricles ceases, the pressure of the blood in the pulmonary artery and aorta closes the pulmonary and aortic semilunar valves to prevent regurgitation of blood into the ventricles, the valves remaining shut until reopened by the next ventricular systole. During the period of rest, the tension of the tricuspid and bicuspid valves is relaxed, and blood is flowing from the veins into the atria, being aspirated by negative intrathoracic pressure, and slightly also from the atria into the ventricles. The average duration of a cardiac cycle is about 0.8 of a second, made up as follows: atrial systole 1/10, atrial diastole 7/10, ventricular systole 3/10, ventricular diastole 5/10, total systole 4/10, complete diastole 4/10. The rhythmical action of the heart is muscular in origin. That is to say, the heart muscle itself possesses the inherent property of contraction, apart from any nervous stimulation. The more embryonic the muscle, the better is it able to initiate and propagate the contraction wave. This explains why the normal systole of the heart starts at the entrance of the veins, for there the muscle is most embryonic in nature. At the atrioventricular junction there is a slight pause in the wave of muscular contraction. To obviate this, so far as possible, a peculiar band of marked embryonic type passes across the junction, and so carries on the contraction wave to the ventricles. This band, composed of special fibers, is the atrioventricular bundle of His. The nerves, although not concerned in originating the contractions of the heart muscle, play an important role in regulating their force and frequency in order to subserve the physiological needs of the organism. End of section 7.
Section 8 of Gray's Anatomy, Part 3. This is a Librivox recording. All Librivox recordings are in the public domain. For more information or to volunteer, please visit Librivox.org. Recording by Leanne Howlett. Anatomy of the Human Body, Part 3, by Henry Gray. Peculiarities in the Vascular System in the Fetus. The chief peculiarities of the fetal heart are the direct communication between the atria through the foramen oval and the large size of the valve of the inferior vena cava. Among other peculiarities the following may be noted. 1. In early fetal life the heart lies immediately below the mandibular arch and is relatively large in size. As development proceeds it is gradually drawn within the thorax, but at first it lies in the middle line. Toward the end of pregnancy it gradually becomes oblique in direction. 2. For a time the atrial portion exceeds the ventricular in size, and the walls of the ventricles are of equal thickness. Toward the end of fetal life the ventricular portion becomes the larger, and the wall of the left ventricle exceeds that of the right in thickness. 3. Its size is large as compared with that of the rest of the body, the proportion at the second month being 1:50, and at birth 1:120, while in the adult the average is about 1:160. The foramen oval, situated at the lower part of the atrial septum, forms a free communication between the atria until the end of fetal life. A septum, septum secundum, grows down from the upper wall of the atrium to the right of the primary septum in which the foramen oval is situated. Shortly after birth it fuses with the primary septum and the foramen oval is obliterated. The valve of the inferior vena cava serves to direct the blood from that vessel through the foramen oval into the left atrium. The peculiarities in the arterial system of the fetus are the communication between the pulmonary artery and the aorta by means of the ductus arteriosus, and the continuation of the hypogastric arteries as the umbilical arteries to the placenta. The ductus arteriosus is a short tube, about 1.25 centimeters in length at birth, and of the diameter of a goose quill. In the early condition it forms the continuation of the pulmonary artery and opens into the aorta just beyond the origin of the left subclavian artery, and so conducts the greater amount of the blood from the right ventricle into the aorta. When the branches of the pulmonary artery have become larger relatively to the ductus arteriosus, the latter is chiefly connected to the left pulmonary artery. The hypogastric arteries run along the sides of the bladder and thence upward on the back of the anterior abdominal wall to the umbilicus. Here they pass out of the abdomen and are continued as the umbilical arteries in the umbilical cord to the placenta. They convey the fetal blood to the placenta. The peculiarities in the venous system of the fetus are the communications established between the placenta and the liver and portal vein through the umbilical vein, and between the umbilical vein and the inferior vena cava through the ductus venosus. Fetal circulation. The fetal blood is returned from the placenta to the fetus by the umbilical vein. This vein enters the abdomen at the umbilicus and passes upward along the free margin of the falciform ligament of the liver to the under surface of that organ, where it gives off 2 or 3 branches, one of large size to the left lobe, and others to the lobus quadratus and lobus caudatus. At the porta hepatis, transverse fissure of the liver, it divides into 2 branches. Of these the larger is joined by the portal vein and enters the right lobe. The smaller is continued upward under the name of the ductus venosus, and joins the inferior vena cava. The blood, therefore, which traverses the umbilical vein, passes to the inferior vena cava in 3 different ways. A considerable quantity circulates through the liver with the portal venous blood, before entering the inferior vena cava by the hepatic veins. Some enters the liver directly, and is carried to the inferior cava by the hepatic veins. The remainder passes directly into the inferior vena cava through the ductus venosus. In the inferior vena cava, the blood carried by the ductus venosus and hepatic veins become mixed with that returning from the lower extremities and abdominal wall. It enters the right atrium, and, guided by the valve of the inferior vena cava, passes through the foramen oval into the left atrium, where it mixes with a small quantity of blood returned from the lungs by the pulmonary veins. From the left atrium it passes into the left ventricle, and from the left ventricle into the aorta, by means of which it is distributed almost entirely to the head and upper extremities, a small quantity being probably carried into the descending aorta. From the head and upper extremities the blood is returned by the superior vena cava to the right atrium, where it mixes with a small portion of the blood from the inferior vena cava. From the right atrium it descends into the right ventricle, and thence passes into the pulmonary artery. The lungs of the fetus being inactive, only a small quantity of the blood of the pulmonary artery is distributed to them by the right and left pulmonary arteries, and returned by the pulmonary veins to the left atrium. The greater part passes through the ductus arteriosus into the aorta, where it mixes with a small quantity of the blood transmitted by the left ventricle into the aorta. Through this vessel it descends, and is in part distributed to the lower extremities and the viscera of the abdomen and pelvis, but the greater amount is conveyed by the umbilical arteries to the placenta. From the preceding account of the circulation of the blood in the fetus the following facts will be evident. 1. The placenta serves the purposes of nutrition and excretion, receiving the impure blood from the fetus, and returning it purified and charged with additional nutritive material. 2. Nearly the whole of the blood of the umbilical vein traverses the liver before entering the inferior vena cava. Hence the large size of the liver, especially at an early period of fetal life. 3. The right atrium is the point of meeting of a double current, the blood in the inferior vena cava being guided by the valve of this vessel into the left atrium, while that in the superior vena cava descends into the right ventricle. At an early period of fetal life it is highly probable that the 2 streams are quite distinct, for the inferior vena cava opens almost directly into the left atrium, and the valve of the inferior vena cava would exclude the current from the right ventricle. At a later period, as the separation between the 2 atria becomes more distinct, it seems probable that some mixture of the 2 streams must take place. 4. The pure blood carried from the placenta to the fetus by the umbilical vein, mixed with the blood from the portal vein and inferior vena cava, passes almost directly to the arch of the aorta, and is distributed by the branches of that vessel to the head and upper extremities. 5. The blood contained in the descending aorta, chiefly derived from that which has already circulated through the head and limbs, together with a small quantity from the left ventricle, is distributed to the abdomen and lower extremities. Changes in the vascular system at birth. At birth, when respiration is established, an increased amount of blood from the pulmonary artery passes through the lungs, and the placental circulation is cut off. The foramen oval is closed by about the 10th day after birth. The valvular fold above mentioned adheres to the margin of the foramen for the greater part of its circumference, but a slit-like opening is left between the two atria above, and this sometimes persists. The ductus arteriosus begins to contract immediately after respiration is established, and is completely closed from the 4th to the 10th day. It ultimately degenerates into an impervious cord, the ligamentum arteriosum, which connects the left pulmonary artery to the arch of the aorta. Of the hypogastric arteries, the parts extending from the sides of the bladder to the umbilicus become obliterated between the 2nd and 5th days after birth, and project as fibrous cords, the lateral umbilical ligaments toward the abdominal cavity carrying on them folds of peritoneum. The umbilical vein and ductus venosus are completely obliterated between the 2nd and 5th days after birth. The former becomes the ligamentum teres, the latter the ligamentum venosum of the liver. End of Section 8. Recording by Leanne Howlett.
Section 9 of Gray's Anatomy Part 3. This is a Librivox recording. All Librivox recordings are in the public domain. For more information or to volunteer, please visit librivox.org. Anatomy of the Human Body, Part 3, by Henry Gray. The Arteries, Introduction. The distribution of the systemic arteries is like a highly ramified tree, the common trunk of which, formed by the aorta, commences at the left ventricle, while the smallest ramifications extend to the peripheral parts of the body and the contained organs. Arteries are found in all parts of the body except in the hairs, nails, epidermis, cartilages, and cornea. The larger trunks usually occupy the most protected situations, running in the limbs along the flexor surface, where they are less exposed to injury. There is considerable variation in the mode of division of the arteries. Occasionally a short trunk subdivides into several branches at the same point, as may be observed in the celiac artery and the thyrocervical trunk. The vessel may give off several branches in succession and still continue as the main trunk, as is seen in the arteries of the limbs, or the division may be dichotomous, as for instance when the aorta divides into the two common iliacs. A branch of an artery is smaller than the trunk from which it arises, but if an artery divides into two branches, the combined sectional area of the two vessels is, in nearly every instance, somewhat greater than that of the trunk, and the combined sectional area of all the arterial branches greatly exceeds that of the aorta, so that the arteries collectively may be regarded as a cone, the apex of which corresponds to the aorta, and the base to the capillary system. The arteries, in their distribution, communicate with one another, forming what are called anastomoses, and these communications are very free between the large as well as between the smaller branches. The anastomosis between trunks of equal size is found where great activity of the circulation is requisite, as in the brain. Here the two vertebral arteries unite to form the basilar, and the two anterior cerebral arteries are connected by a short communicating trunk. It is also found in the abdomen, where the intestinal arteries have very ample anastomoses between their larger branches. In the limbs, the anastomoses are most numerous and of largest size around the joints, the branches of an artery above uniting with branches from the vessels below. These anastomoses are of considerable interest to the surgeon, as it is by their enlargement that a collateral circulation is established after the application of a ligature to an artery. The smaller branches of arteries anastomose more frequently than the larger, and between the smallest twigs these anastomoses become so numerous as to constitute a close network that pervades nearly every tissue of the body. Throughout the body, generally, the larger arterial branches pursue a fairly straight course, but in certain situations they are tortuous. Thus the external maxillary artery in its course over the face and the arteries of the lips are extremely tortuous to accommodate themselves to the movements of the parts. The uterine arteries are also tortuous to accommodate themselves to the increase in size which the uterus undergoes during pregnancy. The pulmonary artery conveys the venous blood from the right ventricle of the heart to the lungs. It is a short, wide vessel, about 5 centimeters in length and 3 centimeters in diameter, arising from the conus arteriosus of the right ventricle. It extends obliquely upward and backward. passing at first in front and then to the left of the ascending aorta, as far as the undersurface of the aortic arch, where it divides, about the level of the fibrocartilage between the fifth and sixth thoracic vertebrae, into right and left branches of nearly equal size. Relations. The whole of this vessel is contained within the pericardium. It is enclosed with the ascending aorta in a single tube of the visceral layer of the serous pericardium, which is continued upward upon them from the base of the heart. The fibrous layer of the pericardium is gradually lost upon the external coats of the two branches of the artery. In front, the pulmonary artery is separated from the anterior end of the second left intercostal space by the pleura and the left lung, in addition to the pericardium. It rests at first upon the ascending aorta, and higher up lies in front of the left atrium on a plane posterior to the ascending aorta. On either side of its origin is the auricula of the corresponding atrium and a coronary artery, the left coronary artery passing, in the first part of its course, behind the vessel. The superficial part of the cardiac plexus lies above its bifurcation, between it and the arch of the aorta. The right branch of the pulmonary artery, ramus dexter A pulmonalis, longer and larger than the left, runs horizontally to the right, behind the ascending aorta and superior vena cava, and in front of the right bronchus, to the root of the right lung, where it divides into two branches. The lower and larger of these goes to the middle and lower lobes of the lung. The upper and smaller is distributed to the upper lobe. The left branch of the pulmonary artery, ramus sinister A pulmonalis, shorter and somewhat smaller than the right, passes horizontally in front of the descending aorta and left bronchus to the root of the left lung, where it divides into two branches, one for each lobe of the lung. Above, it is connected to the concavity of the aortic arch by the ligamentum arteriosum, on the left of which is the left recurrent nerve, and on the right the superficial part of the cardiac plexus. Below, it is joined to the upper left pulmonary vein by the ligament of the left vena cava. The terminal branches of the pulmonary arteries will be described with the anatomy of the lungs. End of section 9.
Section 10 of Gray's Anatomy, Part 3. This is a Librivox recording. All Librivox recordings are public domain. For more information or to volunteer, please visit Librivox.org. Recording by Suzanne Olson. Anatomy of the Human Body, Part 3, by Henry Gray, The Aorta. The aorta is the main trunk of a series of vessels which convey the oxygenated blood to the tissues of the body for their nutrition. It commences at the upper part of the left ventricle, where it is about 3 centimeters in diameter, and after ascending for a short distance, arches backward and to the left side over the root of the left lung. It then descends within the thorax on the left side of the vertebral column, passes into the abdominal cavity through the aortic hiatus in the diaphragm, and ends considerably diminished in size, about 1.75 centimeters in diameter, opposite the lower border of the fourth lumbar vertebra, by dividing into the right and left common iliac arteries. Hence it is described in several portions, viz., the ascending aorta, the arch of the aorta, and the descending aorta, which last is again divided into the thoracic and abdominal aortae. The ascending aorta, aorta ascendens. The ascending aorta is about 5 centimeters in length. It commences at the upper part of the base of the left ventricle on a level with the lower border of the third costal cartilage behind the left half of the sternum. It passes obliquely upward, forward, and to the right in the direction of the heart's axis, as high as the upper border of the second right costal cartilage, describing a slight curve in its course, and being situated about 6 centimeters behind the posterior surface of the sternum. At its origin it presents, opposite the segments of the aortic valve, 3 small dilatations called the aortic sinuses. At the union of the ascending aorta with the aortic arch, the caliber of the vessel is increased, owing to a bulging of its right wall. This dilatation is termed the bulb of the aorta, and on transverse section presents a somewhat oval figure. The ascending aorta is contained within the pericardium and is enclosed in a tube of the serous pericardium common to it and the pulmonary artery. Relations. The ascending aorta is covered at its commencement by the trunk of the pulmonary artery in the right auricula, and higher up is separated from the sternum by the pericardium, the right pleura, the anterior margin of the right lung, and some loose areolar tissue, and the remains of the thymus. Posteriorly, it rests upon the left atrium and right pulmonary artery. On the right side, it is in relation with the superior vena cava and right atrium, the former lying partly behind it on the left side with the pulmonary artery. Branches. The only branches of the ascending aorta are the two coronary arteries which supply the heart. They arise near the commencement of the aorta immediately above the attached margins of the semilunar valves. The coronary arteries. The right coronary artery, A coronaria cordis dextra, arises from the right anterior aortic sinus. It passes at first between the conus arteriosus and the right auricula, and then runs in the right portion of the coronary sulcus, coursing at first from left to right, and then on the diaphragmatic surface of the heart, from right to left, as far as the posterior longitudinal sulcus, down which it continued to the apex of the heart as a posterior descending branch. It gives off a large marginal branch which follows the acute margin of the heart and supplies branches to both surfaces of the right ventricle. It also gives twigs to the right atrium and to the part of the left ventricle which adjoins the posterior longitudinal sulcus. The left coronary artery, A coronaria cordis sinistra, larger than the right, arises from the left anterior aortic sinus and divides into an anterior descending and a circumflex branch. The anterior descending branch passes at first behind the pulmonary artery and then comes forward between that vessel and the left auricula to reach the anterior longitudinal sulcus, along which it descends to the incisura apicus cordis. It gives branches to both ventricles. The circumflex branch follows the left part of the coronary sulcus, running first to the left and then to the right, reaching nearly as far as the posterior longitudinal sulcus. It gives branches to the left atrium and ventricle. There is a free anastomosis between the minute branches of the two coronary arteries in the substance of the heart. Peculiarities. These vessels occasionally arise by a common trunk, or their number may be increased to 3, the additional branch being of small size. More rarely, there are 2 additional branches. The arch of the aorta. Arcus aortae transverse aorta. The arch of the aorta begins at the level of the upper border of the second sternocostal articulation of the right side and runs at first upward, backward, and to the left in front of the trachea. It is then directed backward on the left side of the trachea and finally passes downward on the left side of the body of the fourth thoracic vertebra, at the lower border of which it becomes continuous with the descending aorta. It thus forms 2 curvatures, one with its convexity upward, the other with its convexity forward and to the left. Its upper border is usually about 2.5 centimeters below the superior border to the manubrium sterni. Relations. The arch of the aorta is covered anteriorly by the pleurae and anterior margins of the lungs and by the remains of the thymus. As the vessel runs backward, its left side is in contact with the left lung and pleura. Passing downward on the left side of this part of the arch are 4 nerves. In order from backwards, these are the left phrenic, the lower of the superior cardiac branches of the left vagus, the superior cardiac branch of the left sympathetic, and the trunk of the left vagus. As the last nerve crosses the arch, it gives off its recurrent branch, which hooks around below the vessel and then passes upward on its right side. The highest left intercostal vein runs obliquely upward and forward on the left side of the arch between the phrenic and vagus nerves. On the right are the deep part of the cardiac plexus, the left recurrent nerve, the esophagus, and the thoracic duct. The trachea lies behind and to the right of the vessel. Above are the innominate, left common carotid, and left subclavian arteries, which arise from the convexity of the arch and are crossed close to their origins by the left innominate vein. Below are the bifurcation of the pulmonary artery, the left bronchus, the ligamentum anteriosum, the superficial part of the cardiac plexus, and the left recurrent nerve. As already stated, the ligamentum arteriosum connects the commencement of the left pulmonary artery to the aortic arch. Between the origin of the left subclavian artery and the attachment of the ductus arteriosus, the lumen of the fetal aorta is considerably narrowed, forming what is termed the aortic isthmus, while immediately beyond the ductus arteriosus, the vessel presents a fusiform dilation, which His has named the aortic spindle, the point of junction of the two parts being marked in the concavity of the arch by an indentation or angle. These conditions persist to some extent in the adult, where His found that the average diameter of the spindle exceeded that of the isthmus by 3 millimeters. Distinct from this diffuse and moderate stenosis at the isthmus is the condition known as coarctation of the aorta, or marked stenosis, often amounting to complete obliteration of its lumen, seen in adults and occurring at or near, oftenest a little below, the insertion of the ligamentum arteriosum into the aorta. According to Bonnet, this coarctation is never found in the fetus or at birth, and is due to an abnormal extension of the peculiar tissue of the ductus into the aortic wall, which gives rise to a simultaneous stenosis of both vessels as it contracts after birth. The ductus is usually obliterated in these cases. An extensive collateral circulation is set up by the costocervicals, internal mammaries, and the descending branches of the transverse cervical above the stenosis, and below it by the first 4 aortic intercostals, the pericardiacophrenics, and the superior and inferior epigastrics. Peculiarities. The height to which the aorta rises in the thorax is usually about 2.5 centimeters below the upper border of the sternum, but it may ascend nearly to the top of the bone. Occasionally it's found 4 centimeters, more rarely from 5 to 8 centimeters below this point. Occasionally it is found 4 centimeters, more rarely from 5 to 8 centimeters below this point. Sometimes the aorta arches over the root of the right lung, right aortic arch, instead of over that of the left, and passes down on the right side of the vertebral column, a condition which is found in birds. In such cases, all the thoracic and abdominal viscera are transposed. Less frequently the aorta, after arching over the root of the right lung, is directed to its usual position on the left side of the vertebral column. This peculiarity is not accompanied by transposition of the viscera. The aorta occasionally divides, as in some quadrupeds, into an ascending and a descending trunk, the former of which is directed vertically upward and subdivides into 3 branches to supply the head and upper extremities. Sometimes the aorta subdivides near its origin into 2 branches which soon reunite. In one of these cases, the esophagus and trachea were found to pass through the interval between the 2 branches. This is the normal condition of the vessel in the reptilia. Branches. The branches given off from the arch of the aorta are 3 in number: the innominate, the left common carotid, and the left subclavian. Peculiarities. Position of the branches. The branches, instead of arising from the highest part of the arch, may spring from the commencement of the arch or upper part of the ascending aorta, or the distance between them at their origins may be increased or diminished, the most frequent change in this respect being the approximation of the left carotid toward the innominate artery. The number of the primary branches may be reduced to one, or more commonly two, the left carotid arising from the innominate artery, or, more rarely, the carotid and subclavian arteries of the left side arising from a left innominate artery. But the number may be increased to four from the right carotid and subclavian arteries arising directly from the aorta, the innominate being absent. In most of these latter cases, the right subclavian has been found to arise from the left end of the arch. In other cases, it is the second or third branch given off instead of the first. Another common form in which there are four primary branches is that in which the left vertebral artery arises from the arch of the aorta between the left carotid and subclavian arteries. Lastly, the number of trunks from the arch may be increased to 5 or 6. In these instances, the external and internal carotids arise separately from the arch, the common carotid being absent on one or both sides. In some cases, 6 branches have been found, and this condition is associated with the origin of both vertebral arteries from the arch. Number usual, arrangement different. When the aorta arches over to the right side, the 3 branches have an arrangement the reverse of what is usual. The innominate artery is the left one, and the right carotid and subclavian arise separately. In other cases, where the aorta takes its usual course, the 2 carotids may be joined in a common trunk, and the subclavians arise separately from the arch, the right subclavian generally arising from the left end of the arch. In some instances, other arteries spring from the arch of the aorta. Of these, the most common are the bronchial, one or both, and the thyroidea, ema, but the internal mammary and the inferior thyroid have been seen to arise from this vessel. The innominate artery, A. Anonyma brachiocephalic artery. The innominate artery is the largest branch of the arch of the aorta and is from 4 to 5 centimeters in length. It arises on a level with the upper border of the second right costal cartilage from the commencement of the arch of the aorta on a plane anterior to the origin of the left carotid. It ascends obliquely upward, backward, and to the right to the level of the upper border of the right sternoclavicular articulation, where it divides into the right common carotid and right subclavian arteries. Relations. Anteriorly, it is separated from the manubrium sterni by the sternohyoides and sternothyroideus, the remains of the thymus, the left innominate and right inferior thyroid veins which cross its root, and sometimes the superior cardiac branches of the right vagus. Posterior to it is the trachea, which it crosses obliquely. On the right side are the right innominate vein, the superior vena cava, the right phrenic nerve, and the pleura, and on the left side the remains of the thymus, the origin of the left common carotid artery, the inferior thyroid veins, and the trachea. Branches. The innominate artery usually gives off no branches, but occasionally a small branch, the thyroidea ema, arises from it. Sometimes it gives off a thymic or bronchial branch. The thyroidea ema, A. thyroidea ema, ascends in front of the trachea to the lower part of the thyroid gland which it supplies. It varies greatly in size and appears to compensate for deficiency or absence of one of the other thyroid vessels. It occasionally arises from the aorta, the right common carotid, the subclavian, or the internal mammary. Point of division. The innominate artery sometimes divides the level of the sternoclavicular joint, less frequently below it. Position. When the aortic arch is on the right side, the innominate is directed to the left side of the neck. Collateral circulation. Alan Burns demonstrated on the dead subject the possibility of the establishment of the collateral circulation after ligature of the innominate artery by tying and dividing that artery. He then found that even course injection impelled into the aorta passed freely by the anastomosing branches into the arteries of the right arm, filling them and all the vessels of the head completely. The branches by which this circulation would be carried on are very numerous. Thus, all the communications across the middle line between the branches of the carotid arteries of the opposite sides would be available for the supply of blood to the right side of the head and neck, while the anastomosis between the costocervical of the subclavian and the first aortic intercostal, see infra on the collateral circulation after obliteration of the thoracic aorta, would bring the blood by a free and direct course into the right subclavian. The numerous connections also between the intercostal arteries and the branches of the axillary and internal mammary arteries would doubtless assist in the supply of blood to the right arm, while the inferior epigastric from the external iliac would, by means of its anastomosis with the internal mammary, compensate for any deficiency in the vascularity of the wall of the chest.
End of Section 10, Part 3, recorded by Suzanne Olson.
Section 11 of Gray's Anatomy, Part 3. This is a Librivox recording. All Librivox recordings are in the public domain. For more information or to volunteer, please visit Librivox.org. Anatomy of the Human Body, Part 3, by Henry Gray. The common carotid artery. The principal arteries of supply to the head and neck are the two common carotids. They ascend in the neck, and each divides into two branches, viz. 1. The external carotid, supplying the exterior of the head, the face, and the greater part of the neck. 2. The internal carotid, supplying to a great extent the parts within the cranial and orbital cavities. The common carotid artery. A. Carotis communis. The common carotid arteries differ in length and in their mode of origin. The right begins at the bifurcation of the innominate artery behind the sternoclavicular joint and is confined to the neck. The left springs from the highest part of the arch of the aorta to the left of and on a plane posterior to the innominate artery, and therefore consists of a thoracic and a cervical portion. The thoracic portion of the left common carotid artery ascends from the arch of the aorta through the superior mediastinum to the level of the left sternoclavicular joint, where it is continuous with the cervical portion. Relations. In front, it is separated from the manubrium sterni by the sternohyoideus and sternothyroideus, the anterior portions of the left pleura and lung, the left innominate vein, and the remains of the thymus. Behind, it lies on the trachea, esophagus, left recurrent nerve, and thoracic duct. To its right side below is the innominate artery, and above, the trachea, the inferior thyroid veins, and the remains of the thymus. To its left side are the left vagus and phrenic nerves, left pleura, and lung. The left subclavian artery is posterior and slightly lateral to it. The cervical portions of the common carotids resemble each other so closely that one description will apply to both. Each vessel passes obliquely upward from behind the sternoclavicular articulation to the level of the upper border of the thyroid cartilage, where it divides into the external and internal carotid arteries. At the lower part of the neck, the two common carotid arteries are separated from each other by a very narrow interval which contains the trachea, but at the upper part, the thyroid gland, the larynx, and pharynx project forward between the two vessels. The common carotid artery is contained in a sheath, which is derived from the deep cervical fascia and encloses also the internal jugular vein and vagus nerve. The vein lying lateral to the artery, and the nerve between the artery and vein, on a plane posterior to both. On opening the sheath, each of these three structures is seen to have a separate fibrous investment. Relations. At the lower part of the neck, the common carotid artery is very deeply seated, being covered by the integument, superficial fascia, platysma, and deep cervical fascia, the sternocleidomastoideus, sternohyoideus, sternothyroideus, and omohyoideus. In the upper part of its course it is more superficial, being covered merely by the integument, the superficial fascia, platysma, deep cervical fascia, and medial margin of the sternocleidomastoideus. When the latter muscle is drawn backward, the artery is seen to be contained in a triangular space, the carotid triangle, bounded behind by the sternocleidomastoideus, above by the stylohyoideus and posterior belly of the digastricus, and below by the superior belly of the omohyoideus. This part of the artery is crossed obliquely from its medial to its lateral side by the sternocleidomastoid branch of the superior thyroid artery. It is also crossed by the superior and middle thyroid veins, which end in the internal jugular. Descending in front of its sheath is the descending branch of the hypoglossal nerve, this filament being joined by one or two branches from the cervical nerves, which cross the vessel obliquely. Sometimes the descending branch of the hypoglossal nerve is contained within the sheath. The superior thyroid vein crosses the artery near its termination, and the middle thyroid vein a little below the level of the cricoid cartilage. The anterior jugular vein crosses the artery just above the clavicle, but is separated from it by the sternohyoideus and sternothyroideus. Behind, the artery is separated from the transverse processes of the cervical vertebrae by the longus colli and longus capitis, the sympathetic trunk being interposed between it and the muscles. The inferior thyroid artery crosses behind the lower part of the vessel. Medially, it is in relation with the esophagus, trachea, and thyroid gland, which overlaps it, the inferior thyroid artery and recurrent nerve being interposed, higher up, with the larynx and pharynx. Lateral to the artery are the internal jugular vein and vagus nerve. At the lower part of the neck, the right recurrent nerve crosses obliquely behind the artery. The right internal jugular vein diverges from the artery, but the left approaches and often overlaps the lower part of the artery. Behind the angle of bifurcation of the common carotid artery is a reddish-brown oval body, known as the glomus caroticum, carotid body. It is similar in structure to the glomus coccygum, coccygeal body, which is situated on the middle sacral artery. Peculiarities as to origin. The right common carotid may arise above the level of the upper border of the sternoclavicular articulation. This variation occurs in about 12% of cases. In other cases, the artery may arise as a separate branch from the arch of the aorta or in conjunction with the left carotid. The left common carotid varies in its origin more than the right. In the majority of abnormal cases, it arises with the innominate artery. If that artery is absent, the two carotids arise usually by a single trunk. It is rarely joined with the left subclavian, except in cases of transposition of the aortic arch. Peculiarities as to point of division. In the majority of abnormal cases, this occurs higher than usual, the artery dividing opposite or even above the hyoid bone. More rarely it occurs below, opposite the middle of the larynx or the lower border of the cricoid cartilage. One case is related by Morgagni, where the artery was only 4 centimeters in length and divided at the root of the neck. Very rarely the common carotid ascends in the neck without any subdivision, either the external or the internal carotid being wanting, and in a few cases the common carotid has been found to be absent, the external and internal carotids arising directly from the arch of the aorta. This peculiarity existed on both sides in some instances, on one side in others. Occasional branches. The common carotid usually gives off no branch previous to its bifurcation, but it occasionally gives origin to the superior thyroid or its laryngeal branch, the ascending pharyngeal, the inferior thyroid, or, more rarely, the vertebral artery. Collateral circulation. After ligature of the common carotid, the collateral circulation can be perfectly established by the free communication which exists between the carotid arteries of opposite sides, both without and within the cranium, and by enlargement of the branches of the subclavian artery on the side corresponding to that on which the vessel has been tied. The chief communications outside the skull take place between the superior and inferior thyroid arteries and the profunda cervicis and ramus descendens of the occipital. The vertebral takes the place of the internal carotid within the cranium. End of section 11. Recording by Selena Arter. Section 12 of Gray's Anatomy, Part 3. This is a Librivox recording. All Librivox recordings are in the public domain. For more information or to volunteer, please visit Librivox.org. Anatomy of the Human Body, Part 3, by Henry Gray. Section 12, Part 1. The External Carotid Artery. A. Carotis externa. The external carotid artery begins opposite the upper border of the thyroid cartilage, and, taking a slightly curved course, passes upward and forward, and then inclines backward to the space behind the neck of the mandible, where it divides into the superficial temporal and internal maxillary arteries. It rapidly diminishes in size in its course up the neck, owing to the number and large size of the branches given off from it. In the child, it is somewhat smaller than the internal carotid, but in the adult the two vessels are of nearly equal size. At its origin, this artery is more superficial, and placed nearer the middle line than the internal carotid, and is contained within the carotid triangle. Relations. The external carotid artery is covered by the skin, superficial fascia, platysma, deep fascia, and anterior margin of the sternocleidomastoideus. It is crossed by the hypoglossal nerve, by the lingual, rhinine, common facial, and superior thyroid veins, and by the digastricus and stylohyoideus. Higher up it passes deeply into the substance of the parotid gland, where it lies deep to the facial nerve and the junction of the temporal and internal maxillary veins. Medial to it are the hyoid bone, the wall of the pharynx, the superior laryngeal nerve, and a portion of the parotid gland. Lateral to it, in the lower part of its course, is the internal carotid artery. Posterior to it, near its origin, is the superior laryngeal nerve, and higher up it is separated from the internal carotid by the styloglossus and stylopharyngeus, the glossopharyngeal nerve, the pharyngeal branch of the vagus, and part of the parotid gland. Branches. The branches of the external carotid artery may be divided into 4 sets. 1. Anterior. Superior thyroid, lingual, external maxillary. 2. Posterior. Occipital, posterior auricular. 3. Ascending. Ascending pharyngeal. 4. Terminal. Superficial temporal, internal maxillary. 1. The superior thyroid artery, A. thyroidea superior, arises from the external carotid artery just below the level of the greater cornu of the hyoid bone, and ends in the thyroid gland. Relations. From its origin under the anterior border of the sternocleidomastoideus, it runs upward and forward for a short distance in the carotid triangle, where it is covered by the skin, platysma, and fascia. It then arches downward beneath the omohyoideus, sternohyoideus, and sternothyroideus. To its medial side are the constrictor pharyngeus inferior and the external branch of the superior laryngeal nerve. Branches. It distributes twigs to the adjacent muscles and numerous branches to the thyroid gland, anastomosing with its fellow of the opposite side, and with the inferior thyroid arteries. The branches to the gland are generally two in number. One, the larger, supplies principally the anterior surface on the isthmus of the gland. It anastomoses with the corresponding artery of the opposite side. A second branch descends on the posterior surface of the gland and anastomoses with the inferior thyroid artery. Besides the arteries distributed to the muscles and to the thyroid gland, the branches of the superior thyroid are hyoid, sternocleidomastoid, superior laryngeal, cricothyroid. The hyoid branch, ramus hyoideus infrahyoid branch, is small and runs along the lower border of the hyoid bone, beneath the thyrohyoideus, and anastomoses with the vessel of the opposite side. The sternocleidomastoid branch, ramus sternocleidomastoideus sternomastoid branch, runs downward and lateralward across the sheath of the common carotid artery, and supplies the sternocleidomastoideus and neighboring muscles and integument. It frequently arises as a separate branch from the external carotid. The superior laryngeal artery, A. laryngea superior, larger than either of the preceding, accompanies the internal laryngeal branch of the superior laryngeal nerve beneath the thyrohyoideus. It pierces the hyothyroid membrane and supplies the muscles, mucous membrane, and glands of the larynx, anastomosing with the branch from the opposite side. The cricothyroid branch, ramus cricothyroideus, is small and runs transversely across the cricothyroid membrane, communicating with the artery of the opposite side. 2. The lingual artery, A. lingualis, arises from the external carotid between the superior thyroid and external maxillary. It first runs obliquely upward and medialward to the greater cornu of the hyoid bone. It then curves downward and forward, forming a loop which is crossed by the hypoglossal nerve, and passing beneath the digastricus and the stylohyoideus, it runs horizontally forward, beneath the hyoglossus, and finally, ascending almost perpendicularly to the tongue, turns forward on its lower surface as far as the tip, under the name of the profunda linguae. Relations. Its first or oblique portion is superficial and is contained within the carotid triangle. It rests upon the constrictor pharyngeus medius and is covered by the platysma and the fascia of the neck. Its second or curved portion also lies upon the constrictor pharyngeus medius, being covered at first by the tendon of the digastricus and by the stylohyoideus, and afterward by the hyoglossus. Its third or horizontal portion lies between the hyoglossus and genioglossus. The fourth or terminal part, under the name of the profunda linguae, ranine artery, runs along the under surface of the tongue to its tip. Here it is superficial, being covered only by the mucous membrane. Above it is the longitudinalis inferior, and on the medial side the genioglossus. The hypoglossal nerve crosses the first part of the lingual artery, but is separated from the second part by the hyoglossus. Branches. The branches of the lingual artery are hyoid, dorsalis linguae, sublingual, profunda linguae. The hyoid branch, ramus hyoideus, suprahyoid branch, runs along the upper border of the hyoid bone, supplying the muscles attached to it and anastomosing with its fellow of the opposite side. The arteriae dorsalis linguae, rami dorsalis linguae, consist usually of 2 or 3 small branches which arise beneath the hyoglossus. They ascend to the back of the dorsum of the tongue, and supply the mucous membrane in this situation, the glossopalatine arch, the tonsil, soft palate, and epiglottis, anastomosing with the vessels of the opposite side. The sublingual artery, A. sublingualis, arises at the anterior margin of the hyoglossus, and runs forward between the genioglossus and mylohyoideus to the sublingual gland. It supplies the gland and gives branches to the mylohyoideus and neighboring muscles, and to the mucous membrane of the mouth and gums. One branch runs behind the alveolar process of the mandible in the substance of the gum to anastomose with a similar artery from the other side. Another pierces the mylohyoideus and anastomoses with the submental branch of the external maxillary artery. The arteria profunda linguae, ranine artery, deep lingual artery, is the terminal portion of the lingual artery. It pursues a tortuous course and runs along the under surface of the tongue, below the longitudinalis inferior, and above the mucous membrane. It lies on the lateral side of the genioglossus, accompanied by the lingual nerve. At the tip of the tongue, it is said to anastomose with the artery of the opposite side, but this is denied by Hürtel. In the mouth, these vessels are placed one on either side of the frenulum linguae. 3. The external maxillary artery, A. Maxillaris externa, facial artery, arises in the carotid triangle a little above the lingual artery, and, sheltered by the ramus of the mandible, passes obliquely up beneath the digastricus and stylohyoideus, over which it arches to enter a groove on the posterior surface of the submaxillary gland. It then curves upward over the body of the mandible at the antero-inferior angle of the masseter, passes forward and upward across the cheek to the angle of the mouth, then ascends along the side of the nose, and ends at the medial commissure of the eye, under the name of the angular artery. This vessel, both in the neck and on the face, is remarkably tortuous, in the former situation, to accommodate itself to the movements of the pharynx in deglutition, and in the latter, to the movements of the mandible, lips, and cheeks. Relations. In the neck, its origin is superficial, being covered by the integument, platysma, and fascia. It then passes beneath the digastricus and stylohyoideus muscles and part of the submaxillary gland, and frequently beneath the hypoglossal nerve. It lies upon the constrictores pharyngeus medius and superior, the latter of which separates it, at the summit of its arch, from the lower and back part of the tonsil. On the face, where it passes over the body of the mandible, it is comparatively superficial, lying immediately beneath the platysma. In its course over the face, it is covered by the integument, the fat of the cheek, and, near the angle of the mouth, by the platysma, rhizorius, and zygomaticus. It rests on the buccinator and caninus, and passes either over or under the infraorbital head of the quadratus labii superioris. The anterior facial vein lies lateral to the artery, and takes a more direct course across the face, where it is separated from the artery by a considerable interval. In the neck it lies superficial to the artery. The branches of the facial nerve cross the artery from behind forward. Branches. The branches of the artery may be divided into two sets, those given off in the neck, cervical, and those on the face, facial. Cervical branches. Ascending palatine, tonsillar, glandular, submental, muscular. Facial branches. Inferior labial, superior labial, lateral nasal, angular, muscular. The ascending palatine artery, A. palatina ascendens, arises close to the origin of the external maxillary artery, and passes up between the styloglossus and stylopharyngeus to the side of the pharynx, along which it is continued between the constrictor pharyngeus superior and the pterygoideus internus to near the base of the skull. It divides near the levator villi palatini into two branches. One follows the course of this muscle, and, winding over the upper border of the constrictor pharyngeus superior, supplies the soft palate and the palatine glands, anastomosing with its fellow of the opposite side and with the descending palatine branch of the internal maxillary artery. The other pierces the constrictor pharyngeus superior and supplies the palatine tonsil and auditory tube, anastomosing with the tonsillar and ascending pharyngeal arteries. The tonsillar branch, ramus tonsillaris, ascends between the pterogordius internus and styloglossus, and then along the side of the pharynx, perforating the constrictor pharyngeus superior to ramify in the substance of the palatine tonsil and root of the tongue. The glandular branches, rami glandulari submaxillary branches, consist of 3 or 4 large vessels which supply the submaxillary gland, some being prolonged to the neighboring muscles, lymph glands, and integument. The submental artery, A. submentalis, the largest of the cervical branches, is given off from the facial artery just as that vessel quits the submaxillary gland. It runs forward upon the mylohyoideus, just below the body of the mandible, and beneath the digastricus. It supplies the surrounding muscles, and anastomoses with the sublingual artery and with the mylohyoid branch of the inferior alveolar. At the symphysis mentae, it turns upward over the border of the mandible and divides into a superficial and a deep branch. The superficial branch passes between the integument and quadratus labii inferioris, and anastomoses with the inferior labial artery. The deep branch runs between the muscle and the bone, supplies the lip, and anastomoses with the inferior labial and mental arteries. The inferior labial artery, A. labialis inferior, inferior coronary artery, arises near the angle of the mouth. It passes upward and forward beneath the triangularis and, penetrating the orbicularis oris, runs in a tortuous course along the edge of the lower lip between this muscle and the mucous membrane. It supplies the labial glands, the mucous membrane, and the muscles of the lower lip, and anastomoses with the artery of the opposite side, and with the mental branch of the inferior alveolar artery. The superior labial artery, A. labialis superior, superior coronary artery, is larger and more tortuous than the inferior. It follows a similar course along the edge of the upper lip, lying between the mucous membrane and the orbicularis oris, and anastomoses with the artery of the opposite side. It supplies the upper lip and gives off in its course 2 or 3 vessels which ascend to the nose. A septal branch ramifies on the nasal septum as far as the point of the nose, and an alar branch supplies the ala of the nose. The lateral nasal branch is derived from the external maxillary, as that vessel ascends along the side of the nose. It supplies the ala and dorsum of the nose, anastomosing with its fellow, with the septal and alar branches, with the dorsal nasal branch of the ophthalmic, and with the infraorbital branch of the internal maxillary. The angular artery, A. angularis, is the terminal part of the external maxillary. It ascends to the medial angle of the orbit, embedded in the fibers of the angular head of the quadratus labii superioris, and accompanied by the angular vein. On the cheek it distributes branches which anastomose with the infraorbital, after supplying the lacrimal sac and orbicularis oculi. It ends by anastomosing with the dorsal nasal branch of the ophthalmic artery. The muscular branches in the neck are distributed to the pterogordius internus and stylohyoideus, and on the face to the masseter and buccinator. The anastomoses of the external maxillary artery are very numerous, not only with the vessel of the opposite side, but, in the neck, with the sublingual branch of the lingual, with the ascending pharyngeal, and by its ascending palatine and tonsillar branches with the palatine branch of the internal maxillary. On the face, with the mental branch of the inferior alveolar as it emerges from the mental foramen, with the transverse facial branch of the superficial temporal, with the infraorbital branch of the internal maxillary, and with the dorsal nasal branch of the ophthalmic. Peculiarities. The external maxillary artery not infrequently arises in common with the lingual. It varies in its size and in the extent to which it supplies the face. It occasionally ends at the submental, and not infrequently extends only as high as the angle of the mouth or nose. The deficiency is then compensated for by enlargement of one of the neighboring arteries. 4. The occipital artery, A. occipitalis, arises from the posterior part of the external carotid, opposite the external maxillary, near the lower margin of the posterior belly of the digastricus, and ends in the posterior part of the scalp. Course and relations. At its origin, it is covered by the posterior belly of the digastricus and the stylohyoideus, and the hypoglossal nerve winds around it from behind forward. Higher up, it crosses the internal carotid artery, the internal jugular vein, and the vagus and accessory nerves. It next ascends to the interval between the transverse process of the atlas and the mastoid process of the temporal bone, and passes horizontally backward, grooving the surface of the latter bone, being covered by the sternocleidomastoideus, splenius capitis, longissimus capitis, and digastricus, and resting upon the rectus capitis lateralis, the obliquus superior, and semispinalis capitis. It then changes its course and runs vertically upward, pierces the fascia connecting the cranial attachment of the trapezius with the sternocleidomastoideus, and ascends in a tortuous course in the superficial fascia of the scalp, where it divides into numerous branches, which reach as high as the vertex of the skull, and anastomose with the posterior auricular and superficial temporal arteries. Its terminal portion is accompanied by the greater occipital nerve. Branches. The branches of the occipital artery are muscular, sternocleidomastoid, auricular, meningeal, descending. The muscular branches, rami muscularis, supply the digastricus, stylohyoideus, splenius, and longissimus capitis. The sternocleidomastoid artery, A. sternocleidomastoidea, sternomastoid artery, generally arises from the occipital close to its commencement, but sometimes springs directly from the external carotid. It passes downward and backward over the hypoglossal nerve, and enters the substance of the muscle, in company with the accessory nerve. The auricular branch, ramus auricularis, supplies the back of the concha, and frequently gives off a branch, which enters the skull through the mastoid foramen and supplies the dura mater, the diploe, and the mastoid cells. This latter branch sometimes arises from the occipital artery, and is then known as the mastoid branch. The meningeal branch, ramus meningeus dural branch, ascends from the internal jugular vein and enters the skull through the jugular foramen and condyloid canal, to supply the dura mater in the posterior fossa. The descending branch, ramus descendens, arteria princeps cervicis. The largest branch of the occipital descends on the back of the neck and divides into a superficial and deep portion. The superficial portion runs beneath the splenius, giving off branches which pierce that muscle to supply the trapezius, and anastomose with the ascending branch of the transverse cervical. The deep portion runs down between the semispinalis capitis and coli, and anastomoses with the vertebral and with the A profunda cervicalis, a branch of the costocervical trunk. The anastomosis between these vessels assists in establishing the collateral circulation after ligature of the common carotid or subclavian artery. The terminal branches of the occipital artery are distributed to the back of the head. They are very tortuous, and lie between the integument and the occipitalis, anastomosing with the artery of the opposite side and with the posterior auricular and temporal arteries, and supplying the occipitalis, the integument, and pericranium. One of the terminal branches may give off a meningeal twig, which passes through the parietal foramen. End of Section 12. Recording by Selena Arter. Section 13 of Gray's Anatomy, Part 3. This is a Librivox recording. All Librivox recordings are in the public domain. For more information or to volunteer, please visit Librivox.org. Anatomy of the Human Body, Part 3, by Henry Gray. The external carotid artery. Part 2. 5. The posterior auricular artery, A. auricularis posterior, is small and arises from the external carotid, above the digastricus and stylohyoideus, opposite the apex of the styloid process. It ascends under cover of the parotid gland on the styloid process of the temporal bone to the groove between the cartilage of the ear and the mastoid process, immediately above which it divides into its auricular and occipital branches. Branches. Besides several small branches to the digastricus, stylohyoideus, and sternocleidomastoideus, and to the parotid gland, this vessel gives off 3 branches: stylomastoid, auricular, occipital. The stylomastoid artery, A. stylomastoidea, enters the styloid mastoid foramen and supplies the tympanic cavity, the tympanic antrum and mastoid cells, and the semicircular canals. In the young subject a branch from this vessel forms, with the anterior tympanic artery from the internal maxillary, a vascular circle which surrounds the tympanic membrane, and from which delicate vessels ramify on that membrane. It anastomoses with the superficial petrosal branch of the middle meningeal artery by a twig which enters the hiatus canalis fascialis. The auricular branch, ramus auricularis, ascends behind the ear, beneath the auricularis posterior, and is distributed to the back of the auricula, upon which it ramifies minutely, some branches curving around the margin of the cartilage, others perforating it, to supply the anterior surface. It anastomoses with the parietal and anterior auricular branches of the superficial temporal. The occipital branch, ramus occipitalis, passes backward over the sternocleidomastoideus to the scalp above and behind the ear. It supplies the occipitalis and the scalp in this situation and anastomoses with the occipital artery. 6. The ascending pharyngeal artery, A. pharyngea ascendens, the smallest branch of the external carotid, is a long, slender vessel, deeply seated in the neck, beneath the other branches of the external carotid and under the stylopharyngeus. It arises from the back part of the external carotid, near the commencement of that vessel, and ascends vertically between the internal carotid and the side of the pharynx, to the under surface of the base of the skull, lying on the longus capitis. Branches. Its branches are pharyngeal, palatine, prevertebral, inferior tympanic, posterior meningeal. The pharyngeal branches, rami pharyngeae, are 3 or 4 in number. 2 of these descend to supply the constrictores pharyngeus medius and inferior, and the stylopharyngeus, ramifying in their substance and in the mucous membrane lining them. The palatine branch varies in size, and may take the place of the ascending palatine branch of the facial artery, when that vessel is small. It passes inward upon the constrictor pharyngeus superior, sends ramifications to the soft palate and tonsil, and supplies a branch to the auditory tube. The prevertebral branches are numerous small vessels, which supply the longi capitis and coli, the sympathetic trunk, the hypoglossal and vagus nerves, and the lymph glands. They anastomose with the ascending cervical artery. The inferior tympanic artery, A. tympanica inferior, is a small branch which passes through a minute foramen in the petrous portion of the temporal bone, in company with the tympanic branch of the glossopharyngeal nerve, to supply the medial wall of the tympanic cavity and anastomose with the other tympanic arteries. The meningeal branches are several small vessels, which supply the dura mater. 1. The posterior meningeal enters the cranium through the jugular foramen. A second passes through the foramen lacerum, and occasionally a third through the canal for the hypoglossal nerve. The superficial temporal artery, A. temporalis superficialis, the smaller of the 2 terminal branches of the external carotid, appears, from its direction, to be the continuation of that vessel. It begins in the substance of the parotid gland, behind the neck of the mandible, and crosses over the posterior root of the zygomatic process of the temporal bone. About 5 centimeters above this process, it divides into 2 branches, a frontal and a parietal. Relations. As it crosses the zygomatic process, it is covered by the auricularis anterior muscle and by a dense fascia. It is crossed by the temporal and zygomatic branches of the facial nerve and 1 or 2 veins, and is accompanied by the auriculotemporal nerve, which lies immediately behind it. Branches. Besides some twigs to the parotid gland, to the temporomandibular joint, and to the masseter muscle, its branches are transverse facial, middle temporal, anterior auricular, frontal, parietal. The transverse facial artery, A. transversa fascia, is given off from the superficial temporal before that vessel quits the parotid gland. Running forward through the substance of the gland, it passes transversely across the side of the face, between the parotid duct and the lower border of the zygomatic arch, and divides into numerous branches which supply the parotid gland and duct, the masseter and the integument, and anastomose with the external maxillary, masseteric, buccinator, and infraorbital arteries. This vessel rests on the masseter, and is accompanied by 1 or 2 branches of the facial nerve. The middle temporal artery, A. temporalis media, arises immediately above the zygomatic arch, and, perforating the temporal fascia, gives branches to the temporalis, anastomosing with the deep temporal branches of the internal maxillary. It occasionally gives off a zygomatico-orbital branch, which runs along the upper border of the zygomatic arch, between the 2 layers of the temporal fascia, to the lateral angle of the orbit. This branch, which may arise directly from the superficial temporal artery, supplies the orbicularis oculi, and anastomoses with the lacrimal and palpebral branches of the ophthalmic artery. The anterior auricular branches, rami auricularis anteriores, are distributed to the anterior portion of the auricula, the lobule, and part of the external meatus, anastomosing with the posterior auricular. The frontal branch, ramus frontalis anterior temporal, runs tortuously upward and forward to the forehead, supplying the muscles, integument, and pericranium in this region, and anastomosing with the supraorbital and frontal arteries. The parietal branch, ramus parietalis posterior temporal, larger than the frontal, curves upward and backward on the side of the head, lying superficial to the temporal fascia, and anastomosing with its fellow of the opposite side, and with the posterior auricular and occipital arteries. 8. The internal maxillary artery, A. maxillaris interna, the larger of the 2 terminal branches of the external carotid, arises behind the neck of the mandible, and is at first embedded in the substance of the parotid gland. It passes forward between the ramus of the mandible and the spheno-mandibular ligament, and then runs, either superficial or deep, to the pterygoideus externus, to the pterygopalatine fossa. It supplies the deep structures of the face, and may be divided into mandibular, pterygoid, and pterygopalatine portions. The first, or mandibular portion, passes horizontally forward, between the ramus of the mandible and the spheno-mandibular ligament, where it lies parallel to and a little below the auriculotemporal nerve. It crosses the inferior alveolar nerve and runs along the lower border of the pterygoideus externus. The second, or pterygoid portion, runs obliquely forward and upward under cover of the ramus of the mandible and insertion of the temporalis on the superficial, very frequently on the deep, surface of the pterygoideus externus. It then passes between the 2 heads of origin of this muscle and enters the fossa. The third, or pterygopalatine portion, lies in the pterygopalatine fossa in relation with the spheno-palatine ganglion. The branches of this vessel may be divided into 3 groups, corresponding with its 3 divisions. Branches of the first or mandibular portions: anterior tympanic, deep auricular, middle meningeal, accessory meningeal, inferior alveolar. The anterior tympanic artery, A. tympanica anterior, tympanic artery, passes upward behind the temporomandibular articulation, enters the tympanic cavity through the petrotympanic fissure, and ramifies upon the tympanic membrane, forming a vascular circle around the membrane with the stylomastoid branch of the posterior auricular, and anastomosing with the artery of the pterygoid canal and with the carodico-tympanic branch from the internal carotid. The deep auricular artery, A. auricularis profunda, often arises in common with the preceding. It ascends in the substance of the parotid gland, behind the temporomandibular articulation, pierces the cartilaginous or bony wall of the external acoustic meatus, and supplies its cuticular lining and the outer surface of the tympanic membrane. It gives a branch to the temporomandibular joint. The middle meningeal artery, A. meningia media, medidural artery, is the largest of the arteries which supply the dura mater. It ascends between the sphenomandibular ligament and the pterygoideus externus, and between the 2 roots of the auriculotemporal nerve to the foramen spinosum of the sphenoid bone, through which it enters the cranium. It then runs forward in a groove on the great wing of the sphenoid bone, and divides into 2 branches, anterior and posterior. The anterior branch, the larger, crosses the great wing of the sphenoid, reaches the groove or canal in the sphenoidal angle of the parietal bone, and then divides into branches which spread out between the dura mater and internal surface of the cranium, some passing upward as far as the vertex, and others backward to the occipital region. The posterior branch curves backward on the squama of the temporal bone, and, reaching the parietal some distance in front of its mastoid angle, divides into branches which supply the posterior part of the dura mater and cranium. The branches of the middle meningeal artery are distributed partly to the dura mater, but chiefly to the bones. They anastomose with the arteries of the opposite side, and with the anterior and posterior meningeal. The middle meningeal on entering the cranium gives off the following branches. 1. Numerous small vessels supply the semilunar ganglion and the dura mater in this situation. 2. A superficial petrosal branch enters the hiatus of the facial canal, supplies the facial nerve, and anastomoses with the stylomastoid branch of the posterior auricular artery. 3. A superior tympanic artery runs in the canal for the tensor tympani, and supplies this muscle and the lining membrane of the canal. 4. Orbital branches pass through the superior orbital fissure or through separate canals in the great wing of the sphenoid, to anastomose with the lacrimal or other branches of the ophthalmic artery. 5. Temporal branches pass through foramina in the great wing of the sphenoid, and anastomose in the temporal fossa with the deep temporal arteries. The accessory meningeal branch, ramus, meningeus accessorius, small meningeal, or parvidoral branch, is sometimes derived from the preceding. It enters the skull through the foramen ovale, and supplies the semilunar ganglion and dura mater. The inferior alveolar artery, A. alveolaris inferior, inferior dental artery, descends with the inferior alveolar nerve to the mandibular foramen on the medial surface of the ramus of the mandible. It runs along the mandibular canal in the substance of the bone, accompanied by the nerve, and opposite the first premolar tooth divides into 2 branches, incisor and mental. The incisor branch is continued forward beneath the incisor teeth as far as the middle line, where it anastomoses with the artery of the opposite side. The mental branch escapes with the nerve at the mental foramen, supplies the chin, and anastomoses with the submental and inferior labial arteries. Near its origin, the inferior alveolar artery gives off a lingual branch, which descends with the lingual nerve and supplies the mucous membrane of the mouth. As the inferior alveolar artery enters the foramen, it gives off a mylohyoid branch, which runs in the mylohyoid groove, and ramifies on the undersurface of the mylohyoideus. The inferior alveolar artery and its incisor branch during their course through the substance of the bone give off a few twigs, which are lost in the cancellous tissue, and a series of branches which correspond in number to the roots of the teeth. These enter the minute apertures at the extremities of the roots, and supply the pulp of the teeth. Branches of the second or pterygoid portion: deep temporal, pterygoid, mesenteric, buccinator. The deep temporal branches, 2 in number, anterior and posterior, ascend between the temporalis and the pericranium. They supply the muscle and anastomose with the middle temporal artery. The anterior communicates with the lacrimal artery by means of small branches which perforate the zygomatic bone and great wing of the sphenoid. The pterygoid branches, rami pterygoidae, irregular in their number and origin, supply the pterygoidae. The mesenteric artery, A. mesenterica, is small and passes lateralward through the mandibular notch to the deep surface of the masseter. It supplies the muscle and anastomoses with the mesenteric branches of the external maxillary and with the transverse facial artery. The buccinator artery, A. buccinatoria, buccal artery, is small and runs obliquely forward between the pterygoideus internus and the insertion of the temporalis to the outer surface of the buccinator, to which it is distributed, anastomosing with branches of the external maxillary and with the infraorbital. Branches of the third or pterygopalatine portion: posterior superior alveolar, infraorbital, descending palatine, artery of the pterygoid canal, pharyngeal, sphenopalatine. The posterior superior alveolar artery, A. alveolaris superior posterior alveolar or posterior dental artery, is given off from the internal maxillary, frequently in conjunction with the infraorbital just as the trunk of the vessel is passing into the pterygopalatine fossa. Descending upon the tuberosity of the maxilla, it divides into numerous branches, some of which enter the alveolar canals to supply the molar and premolar teeth and the lining of the maxillary sinus, while others are continued forward on the alveolar process to supply the gums. The infraorbital artery, A. infraorbitalis, appears from its direction to be the continuation of the trunk of the internal maxillary, but often arises in conjunction with the posterior superior alveolar. It runs along the infraorbital groove and canal with the infraorbital nerve, and emerges on the face through the infraorbital foramen, beneath the infraorbital head of the quadratus labii superioris. While in the canal, it gives off A. orbital branches which assist in supplying the rectus inferior and obliquus inferior and the lacrimal sac, and B. anterior superior alveolar branches which descend through the anterior alveolar canals to supply the upper incisor and canine teeth and the mucous membrane of the maxillary sinus. On the face, some branches pass upward to the medial angle of the orbit and the lacrimal sac, anastomosing with the angular branch of the external maxillary artery. Others run toward the nose, anastomosing with the dorsal nasal branch of the ophthalmic, and others descend between the quadratus labii superioris and the caninus, and anastomose with the external maxillary, transverse facial, and buccinator arteries. The 4 remaining branches arise from that portion of the internal maxillary which is contained in the pterygopalatine fossa. The descending palatine artery, A. palatina descendens, descends through the pterygopalatine canal with the anterior palatine branch of the sphenopalatine ganglion, and, emerging from the greater palatine foramen, runs forward in a groove on the medial side of the alveolar border of the hard palate to the incisive canal. The terminal branch of the artery passes upward through this canal to anastomose with the sphenopalatine artery. Branches are distributed to the gums, the palatine glands, and the mucous membrane of the roof of the mouth, while in the pterygopalatine canal it gives off twigs which descend in the lesser palatine canals to supply the soft palate and palatine tonsil, anastomosing with the ascending palatine artery. The artery of the pterygoid canal, A. canalis pterygoidea, Vidian artery, passes backward along the pterygoid canal with the corresponding nerve. It is distributed to the upper part of the pharynx and to the auditory tube, sending into the tympanic cavity a small branch which anastomoses with the other tympanic arteries. The pharyngeal branch is very small. It runs backward through the pharyngeal canal with the pharyngeal nerve, and is distributed to the upper part of the pharynx and to the auditory tube. The sphenopalatine artery, A. sphenopalatina nasopalatine artery, passes through the sphenopalatine foramen into the cavity of the nose, at the back part of the superior meatus. Here it gives off its posterior lateral nasal branches which spread forward over the conchae and meatuses, anastomose with the ethmoidal arteries and the nasal branches of the descending palatine, and assist in supplying the frontal, maxillary, ethmoidal, and sphenoidal sinuses. Crossing the undersurface of the sphenoid, the sphenopalatine artery ends on the nasal septum as the posterior septal branches. These anastomose with the ethmoidal arteries and the septal branch of the superior labial. One branch descends in a groove on the vomer to the incisive canal and anastomoses with the descending palatine artery. End of section 13. Recording by Selena Arter.
Section 14 of Gray's Anatomy, Part 3. This is a Librivox recording. All Librivox recordings are in the public domain. For more information or to volunteer, please visit librivox.org. Read by Carolyn Power. The side of the neck presents a somewhat quadrilateral outline, limited above by the lower border of the body of the mandible and an imaginary line extending from the angle of the mandible to the mastoid process, below by the upper border of the clavicle, in front by the middle line of the neck, behind by the anterior margin of the trapezius. This space is subdivided into 2 large triangles by the sternocleidomastoideus, which passes obliquely across the neck from the sternum and clavicle below to the mastoid process and occipital bone above. The triangular space in front of this muscle is called the anterior triangle, and that behind it the posterior triangle. Anterior triangle. The anterior triangle is bounded in front by the middle line of the neck, behind by the anterior margin of the sternocleidomastoideus. Its base, directed upward, is formed by the lower border of the body of the mandible, and a line extending from the angle of the mandible to the mastoid process. Its apex is below, at the sternum. This space is subdivided into 4 smaller triangles by the digastricus above and the superior belly of the omohyoideus below. These smaller triangles are named the inferior carotid, the superior carotid, the submaxillary, and the suprahyoid. The inferior carotid or muscular triangle is bounded in front by the median line of the neck from the hyoid bone to the sternum, behind by the anterior margin of the sternocleidomastoideus, above by the superior belly of the omohyoideus. It is covered by the integument superficial fascia, platysma, and deep fascia, ramifying in which are some of the branches of the supraclavicular nerves. Beneath these superficial structures are the sternohyoideus and the sternothyroideus, which, together with the anterior margin of the sternocleidomastoideus, conceal the lower part of the common carotid artery. Footnote: Therefore, the common carotid artery and the internal jugular vein are not, strictly speaking, contained in this triangle, since they are covered by the sternocleidomastoideus, that is to say, they lie under that muscle which forms the posterior border of the triangle. But as they lie very close to the structures which are really contained in the triangle, and whose position it is essential to remember in operating on this part of the artery, it is expedient to study the relations of all these parts together. End footnote. This vessel is enclosed within its sheath, together with the internal jugular vein and vagus nerve. The vein lies lateral to the artery on the right side of the neck, but overlaps it below on the left side. The nerve lies between the artery and vein, on a plane posterior to both. In front of the sheath are a few descending filaments from the ansa hypoglossae. Behind the sheath are the inferior thyroid artery, the recurrent nerve, and the sympathetic trunk, and on its medial side, the esophagus, the trachea, the thyroid gland, and the lower part of the larynx. By cutting into the upper part of this space and slightly displacing the sternocleidomastoideus, the common carotid artery may be tied below the omohyoideus. The superior carotid or carotid triangle is bounded behind by the sternocleidomastoideus, below by the superior belly of the omohyoideus, and above by the stylohyoideus and the posterior belly of the digastricus. It is covered by the integument superficial fascia, platysma, and deep fascia, ramifying in which are branches of the facial and cutaneous cervical nerves. Its floor is formed by parts of the thyrohyoideus, hyoglossus, and the constrictors pharyngeus medius and inferior. This space, when dissected, is seen to contain the upper part of the common carotid artery, which bifurcates opposite the upper border of the thyroid cartilage into the external and internal carotid. These vessels are somewhat concealed from view by the anterior margin of the sternocleidomastoideus, which overlaps them. The external and internal carotids lie side by side, the external being the more anterior of the two. The following branches of the external carotid are also met with in this space: the superior thyroid running forward and downward, the lingual directly forward, the external maxillary forward and upward, the occipital backward, and the ascending pharyngeal directly upward on the medial side of the internal carotid. The veins met with are the internal jugular, which lies on the lateral side of the common and internal carotid arteries, and veins corresponding to the above-mentioned branches of the external carotid, viz., the superior thyroid, the lingual, common facial, ascending pharyngeal, and sometimes the occipital, all of which end in the internal jugular. The nerves in this space are the following. In front of the sheath of the common carotid is the ramus descendens hypoglossae. The hypoglossal nerve crosses both the internal and external carotids above, curving around the origin of the occipital artery. Within the sheath, between the artery and vein, and behind both, is the vagus nerve. Behind the sheath, the sympathetic trunk. On the lateral side of the vessels, the accessory nerve runs for a short distance before it pierces the sternocleidomastoideus, and on the medial side of the external carotid, just below the hyoid bone, may be seen the internal branch of the superior laryngeal nerve, and still more inferiorly, the external branch of the same nerve. The upper portion of the larynx and lower portion of the pharynx are also found in the front part of this space. The submaxillary or digastric triangle corresponds to the region of the neck immediately beneath the body of the mandible. It is bounded above by the lower border of the body of the mandible and a line drawn from its angle to the mastoid process, below by the posterior belly of the digastricus and the stylohyoideus, in front by the anterior belly of the digastricus. It is covered by the integument, superficial fascia, platysma, and deep fascia, ramifying in which are branches of the facial nerve and ascending filaments of the cutaneous cervical nerve. Its floor is formed by the mylohyoideus, hyoglossus, and constrictor pharyngis superior. It is divided into an anterior and a posterior part by the stylomandibular ligament. The anterior part contains the submaxillary gland, superficial to which is the anterior facial vein, while embedded in the gland is the external maxillary artery and its glandular branches. Beneath the gland, on the surface of the mylohyoideus, are the submental artery and the mylohyoid artery and nerve. The posterior part of this triangle contains the external carotid artery ascending deeply in the substance of the parotid gland. This vessel lies here in front of and superficial to the internal carotid, being crossed by the facial nerve, and gives off in its course the posterior auricular, superficial temporal, and internal maxillary branches. More deeply are the internal carotid, the internal jugular vein, and the vagus nerve, separated from the external carotid by the styloglossus and the stylopharyngeus, and the glossopharyngeal nerve. Footnote: the remark made about the inferior carotid triangle applies also to this one. The structures enumerated as contained in its posterior part lie, strictly speaking, beneath the muscles which form the posterior boundary of the triangle, but as it is very important to bear in mind their close relation to the parotid gland, all these parts are spoken of together. End footnote. The suprahyoid triangle is limited behind by the anterior belly of the digastricus, in front by the middle line of the neck between the mandible and the hyoid bone, below by the body of the hyoid bone. Its floor is formed by the mylohyoideus. It contains 1 or 2 lymph glands and some small veins. The latter unite to form the anterior jugular vein. Posterior triangle. The posterior triangle is bounded in front by the sternocleidomastoideus, behind by the anterior margin of the trapezius. Its base is formed by the middle third of the clavicle, its apex by the occipital bone. The space is crossed about 2.5 centimeters above the clavicle by the inferior belly of the omohyoideus, which divides it into 2 triangles, an upper or occipital and a lower or subclavian. The occipital triangle, the larger division of the posterior triangle, is bounded in front by the sternocleidomastoideus, behind by the trapezius, below by the omohyoideus. Its floor is formed from the above downward by the splenius capitis, levator scapulae, and the scaleni medius and posterior. It is covered by the skin, the superficial and deep fasciae, and by the platysma below. The accessory nerve is directed obliquely across the space from the sternocleidomastoideus, which it pierces, to the under surface of the trapezius. Below, the supraclavicular nerves and the transverse cervical vessels and the upper part of the brachial plexus cross this space. A chain of lymph glands is also found running along the posterior border of the sternocleidomastoideus from the mastoid process to the root of the neck. The subclavian triangle, the smaller division of the posterior triangle, is bounded above by the inferior belly of the omohyoideus, below by the clavicle. Its base is formed by the posterior border of the sternocleidomastoideus. Its floor is formed by the first rib with the first digitation of the serratus anterior. The size of the subclavian triangle varies with the extent of attachment of the clavicular portions of the sternocleidomastoideus and trapezius, and also with the height at which the omohyoideus crosses the neck. Its height also varies according to the position of the arm, being diminished by raising the limb on account of the ascent of the clavicle, and increased by drawing the arm downward when that bone is depressed. This space is covered by the integument, the superficial and deep fasciae, and the platysma, and crossed by the supraclavicular nerves. Just above the level of the clavicle, the third portion of the subclavian artery curves lateralward and downward from the lateral margin of the scalenus anterior across the first rib to the axilla, and this is the situation most commonly chosen for ligaturing the vessel. Sometimes this vessel rises as high as 4 centimeters above the clavicle. Occasionally, it passes in front of the scalenus anterior or pierces the fibers of that muscle. The subclavian vein lies behind the clavicle and is not usually seen in this space, but in some cases it rises as high as the artery, and has even been seen to pass with that vessel behind the scalenus anterior. The brachial plexus of nerves lies above the artery and in close contact with it. Passing transversely behind the clavicle are the transverse scapular vessels, and traversing its upper angle in the same direction, the transverse cervical artery and vein. The external jugular vein runs vertically downward behind the posterior border of the sternocleidomastoideus to terminate in the subclavian vein. It receives the transverse cervical and transverse scapular veins, which form a plexus in front of the artery, and occasionally a small vein which crosses the clavicle from the cephalic. The small nerve to the subclavius also crosses this triangle about its middle, and some lymph glands are usually found in the space. End of section 14. Read by Carolyn Power.
Section 15 of Gray's Anatomy, Part 3. This is a Librivox recording. All Librivox recordings are in the public domain. For more information or to volunteer, please visit LibriVox.org. Recording by Ellie. Anatomy of the Human Body, Part 3 by Henry Gray. The internal carotid artery, arteria carotis interna. The internal carotid artery supplies the anterior part of the brain, the eye and its appendages, and sends branches to the forehead and nose. Its size in the adult is equal to that of the external carotid, though in the child it is larger than that vessel. It is remarkable for the number of curvatures that it presents in different parts of its course. It occasionally has 1 or 2 flexures near the base of the skull, while in its passage through the carotid canal and along the side of the body of the sphenoid bone, it describes a double curvature and resembles the italic letter S. Course and relations. In considering the course and relations of this vessel, it may be divided into 4 portions: cervical, petrous, cavernous, and cerebral. Cervical portion. This portion of the internal carotid begins at the bifurcation of the common carotid, opposite the upper border of the thyroid cartilage, and runs perpendicularly upward, in front of the transverse processes of the upper 3 vertebrae, to the carotid canal in the petrous portion of the temporal bone. It is comparatively superficial at its commencement, where it is contained in the carotid triangle, and lies behind and lateral to the external carotid, overlapped by the sternocleidomastoideus and covered by the deep fascia, platysma, and integument. It then passes beneath the parotid gland, being crossed by the hypoglossal nerve and the digastricus and stylohyoideus, and the occipital and posterior auricular arteries. Higher up, it is separated from the external carotid by the styloglossus and stylopharyngeus, the tip of the styloid process and the stylohyoid ligament, the glossopharyngeal nerve, the pharyngeal branch of the vagus. It is in relation, behind, with the longus capitis, the superior cervical ganglion of the sympathetic trunk, and the superior laryngeal nerve. Laterally with the internal jugular vein and vagus nerve, the nerve lying on a plane posterior to the artery. Medially with the pharynx, superior laryngeal nerve, and ascending pharyngeal artery. At the base of the skull, the glossopharyngeal, vagus, accessory, and hypoglossal nerves lie between the artery and the internal jugular vein. Petrous portion. When the internal carotid artery enters the canal in the petrous portion of the temporal bone, it first ascends a short distance, then curves forward and medialward, and again ascends as it leaves the canal to enter the cavity of the skull between the lingula and the petrosal process of the sphenoid. The artery lies at first in front of the cochlear and tympanic cavity. From the latter cavity it is separated by a thin bony lamella, which is cribriform in the young subject, and often partially absorbed in old age. Further forward it is separated from the semilunar ganglion by a thin plate of bone, which forms the floor of the fossa for the ganglion and the roof of the horizontal portion of the canal. Frequently this bony plate is more or less deficient, and then the ganglion is separated from the artery by fibrous membrane. The artery is separated from the bony wall of the carotid canal by a prolongation of dura mater, and is surrounded by a number of small veins and by filaments of the carotid plexus, derived from the ascending branch of the superior cervical ganglion of the sympathetic trunk. Cavernous portion. In this part of its course the artery is situated between the layers of the dura mater, forming the cavernous sinus, but covered by the lining membrane of the sinus. It at first ascends toward the posterior clinoid process, then passes forward by the side of the body of the sphenoid bone, and again curves upward on the medial side to the anterior clinoid process, and perforates the dura mater, forming the roof of the sinus. This portion of the artery is surrounded by filaments of the sympathetic nerve, and on its lateral side is the abducent nerve. Cerebral portion. Having perforated the dura mater on the medial side of the anterior clinoid process, the internal carotid passes between the optic and oculomotor nerves to the anterior perforated substance at the medial extremity of the lateral cerebral fissure, where it gives off its terminal or cerebral branches. Peculiarities. The length of the internal carotid varies according to the length of the neck, and also according to the point of bifurcation from the common carotid. It arises sometimes from the arch of the aorta. In such rare instances, this vessel has been found to be placed nearer the middle line of the neck than the external carotid, as far upward as the larynx, when the latter vessel crossed the internal carotid. The course of the artery, instead of being straight, may be very tortuous. A few instances are recorded in which this vessel was altogether absent. In one of these, the common carotid passed up the neck and gave off the usual branches of the external carotid. The cranial portion of the internal carotid was replaced by two branches of the internal maxillary, which entered the skull through the foramen rotundum and foramen ovale, and joined to form a single vessel. Branches. The cervical portion of the internal carotid gives off no branches. Those from the other portions are: from the petrous portion, caretico-tympanic, artery of the pterygoid canal; from the cavernous portion, cavernous, hypophyseal, semilunar, anterior meningeal, ophthalmic; from the cerebral portion, anterior cerebral, middle cerebral, posterior communicating, choroidal. 1. The caretico-tympanic branch. Ramus caretico-tympanicus. Tympanic branch is small. It enters the tympanic cavity through a minute foramen in the carotid canal, and anastomosis is the anterior tympanic branch of the internal maxillary and with the stylomastoid artery. 2. The artery of the pterygoid canal. Arteria canalis pterygoideae, Vidian artery. is a small, inconstant branch which passes through the pterygoid canal and anastomoses with the branch of the internal maxillary artery. 3. The cavernous branches are numerous small vessels which supply the hypophysis, the semilunar ganglion, and the walls of the cavernous and inferior petrosal sinuses, some of them anastomosed with branches of the middle meningeal. 4. The hypophyseal branches are one or two minute vessels supplying the hypophysis. 5. The semilunar branches are small vessels to the semilunar ganglion. 6. The anterior meningeal branch, Arteria meningia anterior, is a small branch which passes over the small wing of the sphenoid to supply the dura mater of the anterior cranial fossa. It anastomoses with the meningeal branch from the posterior ethmoidal artery. 7. The ophthalmic artery, Arteria ophthalmica, arises from the internal carotid just as that vessel is emerging from the cavernous sinus on the medial side of the anterior clinoid process, and enters the orbital cavity through the optic foramen, below and lateral to the optic nerve. It then passes over the nerve to reach the medial wall of the orbit and thence horizontally forward, beneath the lower border of the obliquus superior, and divides it into two terminal branches, the frontal and the dorsal nasal. As the artery crosses the optic nerve, it is accompanied by the nasociliary nerve, and is separated from the frontal nerve by the rectus superior and the levator palpebrae superioris. Branches. The branches of the ophthalmic artery may be divided into an orbital group distributed to the orbit and the surrounding parts, and an ocular group to the muscles and bulb of the eye. Orbital group: lacrimal, supraorbital, posterior ethmoidal, anterior ethmoidal, medial palpebral, frontal, dorsal nasal. Ocular group: central artery of the retina, short posterior ciliary, long posterior ciliary, anterior ciliary, muscular. The lacrimal artery, arteria lacrimalis, arises from the optic foramen, and is one of the largest branches derived from the ophthalmic. Not infrequently, it is given off before the artery enters the orbit. It accompanies the lacrimal nerve along the upper border of the rectus lateralis, and supplies the lacrimal gland. Its terminal branches, escaping from the gland, are distributed to the eyelids and conjunctiva. Of those supplying the eyelids, two are of considerable size and are named the lateral palpebral arteries. They run medialward in the upper and lower lids respectively, and anastomose with the medial palpebral arteries, forming an arterial circle in its situation. The lacrimal artery give off one or two zygomatic branches, one of which passes through the zygomatico-temporal foramen to reach the temporal fossa and anastomoses with the deep temporal arteries. Another appears on the cheek through the zygomatico-facial foramen, and anastomoses with the transverse facial. A recurrent branch passes backward through the lateral part of the superior orbital fissure to the dura mater, and anastomoses with the branch of the middle meningeal artery. The lacrimal artery is sometimes derived from one of the anterior branches of the middle meningeal artery. And the supraorbital artery, arteria supraorbitalis, springs from the ophthalmic, as that vessel is crossing over the optic nerve. It passes upward to the medial borders of the rectus superior and levator palpebrae, and meeting the supraorbital nerve accompanies it between the periostrum and levator palpebrae to the supraorbital foramen. Passing through this, it divides into a superficial and a deep branch, which supply the integument, the muscles, and the pericranium of the forehead, anastomosing with the frontal branch of the superficial temporal, and the artery of the opposite side. This artery in the orbit supplies the rectus superior and the levator palpebrae, and sends a branch across the pulley to the obliquus superior to supply the parts of the medial palpebral commissure. At the supraorbital foramen, it frequently transmits a branch to the diploe. The ethmoidal arteries are two in number, posterior and anterior. The posterior ethmoidal artery, the smaller, passes through the posterior ethmoidal canal, supplies the posterior ethmoidal cells, and, entering the cranium, gives off a meningeal branch to the dura mater and nasal branches which descend into the nasal cavity through the apertures in the cribriform plate, anastomosing with branches of the sphenopalatine. The anterior ethmoidal artery accompanies the nasociliary nerve through the anterior ethmoidal canal, supplies the anterior and middle ethmoidal cells and frontal sinus, and, entering the cranium, gives off a meningeal branch to the dura mater, and nasal branches. These latter descend into the nasal cavity through the slit by the side of the crista galli, and running along the groove on the inner surface of the nasal bone, supply branches to the lateral wall and septum of the nose, and the terminal branch which appears on the dorsum of the nose between the nasal bone and the lateral cartilage. The medial palpebral arteries, arteria palpebralis medialis, internal palpebral arteries, 2 in number, superior and inferior, arise from the ophthalmic, opposite the pulli of the obliquus superior. They leave the orbit to encircle the eyelids near their free margins, forming a superior and an inferior arch, which lie between the orbicularis oculi and the tarsi. The superior palpebral anastomosis at the lateral angle of the orbit is the zygomatic-orbital branch of the temporal artery, and with the upper two of the 2 lateral palpebral branches from the lacrimal artery. The inferior palpebral anastomosis at the lateral angle of the orbit, with the lower of the 2 lateral palpebral branches from the lacrimal, and with the transverse facial artery, and at the medial part of the lid with a branch from the angular artery. From this last anastomosis a branch passes to the nasolacrimal duct, ramifying in its mucous membrane as far as the inferior meatus of the nasal cavity. The frontal artery, arteria frontalis, is one of the terminal branches of the ophthalmic, leaves the orbit at its medial angle with the supratrochlear nerve, and ascending to the forehead supplies the integument, muscles, and pericranium, anastomosing with the supraorbital artery and with the artery of the opposite side. The dorsal nasal artery, arteria dorsalis nasi, nasal artery. The other terminal branch of the ophthalmic emerges from the orbit above the medial palpebral ligament, and, after giving a twig to the upper part of the lacrimal sac, divides into 2 branches, one of which crosses the root of the nose and anastomoses with the angular artery. The other runs along the dorsum of the nose, supplies its outer surface and anastomoses with the artery of the opposite side, and with the lateral nasal branch of the external artery. The central artery of the retina, arteria centralis retinae, is the first and one of the smallest branches of the ophthalmic artery. It runs for a short distance within the dural sheath of the optic nerve, but about 2.25 cm behind the eyeball it pierces the nerve obliquely, and runs forward in the center of its substance to the retina. Its mode of distribution will be described with the anatomy of the eye. The ciliary arteries, arteria ciliaris, are divisible into 3 groups: the long and short, posterior, and the anterior. The short posterior ciliary arteries, from 6 to 12 in number, arise from the ophthalmic or its branches. They pass forward around the optic nerve to the posterior part of the eyeball, pierce the sclera around the entrance of the nerve, and supply the choroid and ciliary processes. The long posterior ciliary arteries, 2 in number, pierce the posterior part of the sclera at some distance from the optic nerve, and run forward, along either side of the eyeball, between the sclera and the choroid, to the ciliary muscle, where they divide into 2 branches. These form an anterior circle, the circulus arteriosus major, around the circumference of the iris, from which numerous converging branches run in the substance of the iris to its pupillary margin, where they form a second arterial circle, the circulus arteriosus minor. The anterior ciliary arteries are derived from the muscular branches. They run to the front of the eyeball in company with the tendons of the recti, form a vascular zone beneath the conjunctiva, and then pierce the sclera a short distance from the cornea, and end in the circulus arteriosus major. The muscular branches, remi muscularis, 2 in number, superior and inferior, frequently spring from a common trunk. The superior, often wanting, supplies the levator palpebrae superioris, rectus superior, and obliquus superior. The inferior, more constantly present, passes forward between the optic nerve and the rectus inferior, and is distributed to the recti lateralis, medialis, and inferior, and the obliquus inferior. This vessel gives off most of the anterior ciliary arteries. Additional muscular branches are given off from the lacrimal and the supraorbital arteries, or form the trunk of the ophthalmic. 8. Anterior cerebral artery, arteria cerebri anterior, arises from the internal carotid at the medial extremity of the lateral cerebral fissure. It passes forward and medialward across the anterior perforated substance above the optic nerve to the commencement of the longitudinal fissure. Here it comes into close relationship with the opposite artery to which it is connected by a short trunk, the anterior communicating artery. From this point the two vessels run side by side in the longitudinal fissure, curve around the geno of the corpus callosum, and turning backward, continue along the upper surface of the corpus callosum to its posterior part, where they end by anastomosing with the posterior cerebral arteries. Branches. In its course the anterior cerebral artery gives off the following branches: anterior medial ganglionic, anterior, posterior, inferior, and middle. The anterior medial ganglionic branches form a group of small arteries which arise at the commencement of the anterior cerebral artery. They pierce the anterior perforated substance in lamina terminalis, and supply the rostrum of the corpus callosum, the septum pellucidum, and the head of the caudate nucleus. The inferior branches, 2 or 3 in number, are distributed to the orbital surface of the frontal lobe, where they supply the olfactory lobe, gyrus rectus, and internal orbital gyrus. The anterior branches supply a part of the superior frontal gyrus, and send twigs over the edge of the hemisphere to the superior and middle frontal gyri, and upper part of the anterior central gyrus. The middle branches supply the corpus callosum, the cingulate gyrus, the medial surface of the superior frontal gyrus, and the upper part of the anterior central gyrus. The posterior branches supply the precuneus and adjacent lateral surface of the hemisphere. The anterior communicating artery, arteria communicans anterior, connects the 2 anterior cerebral arteries across the commencement of the longitudinal fissure. Sometimes this vessel is wanting, the 2 arteries joining together to form a single trunk which afterward divides, or it may be wholly or partially divided into 2. Its length averages about 4 millimeters, but varies greatly. It gives off some of the intermedial ganglionic vessels, but these are principally derived from the anterior cerebral. 9. The middle cerebral artery, arteria cerebri media, the largest branch of the internal carotid, runs at first lateralward in the lateral cerebral or sylvian fissure, and then backward and upward to the surface of the insula, where it divides into a number of branches which are distributed to the lateral surface of the cerebral hemisphere. Branches. The branches of this vessel are anterolateral ganglionic, inferior lateral frontal, ascending parietal, parietotemporal, and temporal. The anterolateral ganglionic branches, a group of small arteries which arise at the commencement of the middle cerebral artery, are arranged in 2 sets. 1. The internal striate passes upward through the inner segments of the lentiform nucleus, and supplies it, the caudate nucleus, and the internal capsule. The other, the external striate, ascends through the outer segment of the lentiform nucleus, and supplies the caudate nucleus and thalamus. One artery of this group is of larger size than the rest, and is of special importance, as being the artery in the brain most frequently ruptured. It has been termed by Charcot the artery of cerebral hemorrhage. It ascends between the lentiform nucleus and the external capsule, and ends in the caudate nucleus. The inferior lateral frontal supplies the inferior frontal gyrus, Broca's convolution, and the lateral part of the orbital surface of the frontal lobe. The ascending frontal supplies the anterior central gyrus. The ascending parietal is distributed to the posterior central gyrus and the lower part of the superior parietal lobule. The parietotemporal supplies the supramarginal and angular gyri. The parietotemporal supplies the supramarginal and angular gyri, and posterior parts of the superior and middle temporal gyri. The temporal branches, 2 or 3 in number, are distributed to the lateral surface of the temporal lobe. 10. The posterior communicating artery, arteria communicans posterior, runs backward from the internal carotid and anastomoses with the posterior cerebral, a branch of the basilar. It varies in size, being sometimes small and occasionally so large that the posterior cerebral may be considered as arising from the internal carotid rather than from the basilar. It is frequently larger on one side than on the other. From its posterior half are given off a number of small branches, the posteromedial ganglionic branches, which, with similar vessels from the posterior cerebral, pierce the posterior perforated substance and supply the medial surface of the thalami and the walls of the third ventricle. 11. The anterior choroidal, anteriochoroidea, choroid artery, is a small but constant branch which arises from the internal carotid near the posterior communicating artery. Passing backward and lateralward between the temporal lobe and the cerebral peduncle, it enters the inferior horn of the lateral ventricle through the choroidal fissure and ends in the choroid plexus. It is distributed to the hippocampus, fimbria, talar choroidea of the third ventricle, and choroid plexus. End of section 15. Recording by Ellie.
Section 16 of Gray's Anatomy, Part 3. This is a Librivox recording. All Librivox recordings are in the public domain. For more information or to volunteer, please visit Librivox.org. Anatomy of the Human Body, Part 3, by Henry Gray. The Arteries of the Brain. Since the mode of distribution of the vessels of the brain has an important bearing upon the considerable number of pathological lesions which may occur in this part of the nervous system, it is important to consider a little in more detail the manner in which the vessels are distributed. The cerebral arteries are derived from the internal carotid and vertebral, which at the base of the brain form a remarkable anastomosis known as the arterial circle of Willis. It is formed in front by the anterior cerebral arteries, branches of the internal carotid, which are connected together by the anterior communicating, behind by the two posterior cerebral arteries, branches of the basilar, which are connected on either side with the internal carotid by the posterior communicating. The parts of the brain included within this arterial circle are the lamina terminalis, the optic chiasma, the infundibulum, the tuber cinereum, the corpora mammillaria, and the posterior perforated substance. The three trunks which together supply each cerebral hemisphere arise from the arterial circle of Willis. From its anterior part proceed the two anterior cerebrals, from its anterolateral parts the middle cerebrals, and from its posterior part the posterior cerebrals. Each of these principal arteries gives origin to two different systems of secondary vessels. One of these is named the ganglionic system, and the vessels belonging to it supply the thalami and corpora striata. The other is the cortical system, and its vessels ramify in the pia mater and supply the cortex and subadjacent brain substance. These two systems do not communicate at any point of their peripheral distribution, but are entirely independent of each other, and there is between the parts supplied by the two systems a borderland of diminished nutritive activity, where, it is said, softening is especially liable to occur in the brains of old people. The ganglionic system. All the vessels of this system are given off from the arterial circle of Willis, or from the vessels close to it. They form 6 principal groups. 1. The anteromedial group, derived from the anterior cerebrals and anterior communicating. 2. The posteromedial group, from the posterior cerebrals and posterior communicating. 3 and 4, the right and left anterolateral groups, from the middle cerebrals, and 5 and 6, the right and left posterolateral groups, from the posterior cerebrals, after they have wound around the cerebral peduncles. The vessels of this system are larger than those of the cortical system, and are what Cohnheim designated terminal arteries, that is to say, vessels from which their origin to their termination neither supply nor receive any anastomotic branch, so that, through any one of the vessels, only a limited area of the thalamus or corpus striatum can be injected, and the injection cannot be driven beyond the area of the part supplied by the particular vessel which is the subject of the experiment. The cortical arterial system. The vessels forming this system are the terminal branches of the anterior, middle, and posterior cerebral arteries. They divide and ramify in the substance of the pia mater, and give off branches which penetrate the brain cortex perpendicularly. These branches are divisible into 2 classes, long and short. The long or medullary arteries pass through the gray substance and penetrate the subadjacent white substance to the depth of 3 or 4 centimeters, without intercommunicating otherwise than by very fine capillaries, and thus constitute so many independent small systems. The short vessels are confined to the cortex, where they form with the long vessels a compact network in the middle zone of the gray substance, the outer and inner zones being sparingly supplied with blood. The vessels of the cortical arterial system are not so strictly terminal as those of the ganglionic system, but they approach this type very closely, so that injection of one area from the vessel of another area, though possible, is frequently very difficult, and is only effected through vessels of small caliber. As a result of this, obstruction of one of the main branches or its divisions may have the effect of producing softening in a limited area of the cortex. End of section 16.
Section number 17 of Gray's Anatomy, Part 3. This is a LibriVox recording. All LibriVox recordings are in the public domain. For more information or to volunteer, please visit LibriVox.org. Recording by Morgan Scorpion. Anatomy of the Human Body, Part 3, by Henry Gray. Arteries of the upper extremity, the subclavian artery, Part 1. The artery which supplies the upper extremity continues as a single trunk from its commencement down to the elbow, but different portions of it have received different names, according to the regions through which they pass. The part of the vessel which extends from its origin to the outer border of the first rib is termed the subclavian. Beyond this point to the lower border of the axilla, it is named the axillary, and from the lower margin of the axillary space to the bend of the elbow it is termed brachial. Here the trunk ends by dividing into two branches, the radial and ulnar. The subclavian artery, arteria subclavia. On the right side, the subclavian artery arises from the innominate artery behind the right sternoclavicular articulation. On the left side it springs from the arch of the aorta. The two vessels, therefore, in the first part of their course, differ in length, direction, and relation with neighbouring structures. In order to facilitate the description, each subclavian artery is divided into three parts. The first portion extends from the origin of the vessel to the medial border of the scalenus anterior, the second lies behind this muscle, and the third extends from the lateral margin of the muscle to the outer border of the first rib, where it becomes the axillary artery. The first portions of the two vessels require separate descriptions. The second and third parts of the two arteries are practically alike. First part of the right subclavian artery. The first part of the right subclavian artery arises from the innominate artery behind the upper part of the right sternoclavicular articulation and passes upward and lateralward to the medial margin of the scalenus anterior. It extends a little above the clavicle, the extent to which it does so varying in different cases. Relations. It is covered, in front, by the integument, superficial fascia, platysma, deep fascia, the clavicular origin of the sternocleidomastoideus, the sternohyoideus, and sternothyreoideus, and another layer of the deep fascia. It is crossed by the internal jugular and vertebral veins, by the vagus nerve and the cardiac branches of the vagus and sympathetic, and by the subclavian loop of the sympathetic trunk which forms a ring around the vessel. The anterior jugular vein is directed lateralward in front of the artery, but is separated from it by the sternohyoideus and sternothyreoideus. Below and behind the artery is the pleura, which separates it from the apex of the lung. Behind is the sympathetic trunk, the longus colli, and the first thoracic vertebra. The right recurrent nerve winds around the lower and back part of the vessel. First part of the left subclavian artery. The first part of the left subclavian artery arises from the arch of the aorta, behind the left common carotid and at the level of the fourth thoracic vertebra. It ascends in the superior mediastinal cavity to the root of the neck and then arches lateralward to the medial border of the scalenus anterior. Relations. It is in relation, in front, with the vagus, cardiac, and phrenic nerves, which lie parallel with it, the left common carotid artery, left internal jugular and vertebral veins, and the commencement of the left innominate vein, and is covered by the sternothyreoideus, sternohyoideus, and sternocleidomastoideus. Behind, it is in relation with the esophagus, thoracic duct, left recurrent nerve, inferior cervical ganglion of the sympathetic trunk, and longus colli. Higher up, however, the esophagus and thoracic duct lie to its right side, the latter ultimately arching over the vessel to join the angle of union between the subclavian and internal jugular veins. Medial to it are the esophagus, trachea, thoracic duct, and left recurrent nerve. Lateral to it, the left pleura and lung. second and third parts of the subclavian artery. The second portion of the subclavian artery lies behind the scalenus anterior. It is very short, and forms the highest part of the arch described by the vessel. Relations. It is covered in front by the skin, superficial fascia, platysma, deep cervical fascia, sternocleidomastoideus, and scalenus anterior. On the right side of the neck the phrenic nerve is separated from the second part of the artery by the scalenus anterior, while on the left side it crosses the first part of the artery close to the medial edge of the muscle. Behind the vessel are the pleura and the scalenus medius, above the brachial plexus of nerves, below the pleura. The subclavian vein lies below and in front of the artery, separated from it by the scalenus anterior. The third portion of the subclavian artery runs downward and lateralward from the lateral margin of the scalenus anterior to the outer border of the first rib, where it becomes the axillary artery. This is the most superficial portion of the vessel, and is contained in the subclavian triangle. Relations. It is covered in front by the skin, the superficial fascia, the platysma, the supraclavicular nerves, and the deep cervical fascia. The external jugular vein crosses its medial part and receives the transverse scapular, transverse cervical, and anterior jugular veins, which frequently form a plexus in front of the artery. Behind the veins, the nerve to the subclavius descends in front of the artery. The terminal part of the artery lies behind the clavicle and the subclavius, and is crossed by the transverse scapular vessels. The subclavian vein is in front of and at a slightly lower level than the artery. Behind, it lies on the lowest trunk of the brachial plexus, which intervenes between it and the scalenus medius. Above and to its lateral side are the upper trunks of the brachial plexus and the omohyoideus. Below, it rests on the upper surface of the first rib. Peculiarities. The subclavian arteries vary in their origin, their course, and the height to which they rise in the neck. The origin of the right subclavian from the innominate takes place, in some cases, above the sternoclavicular articulation, and occasionally, but less frequently, below that joint. The artery may arise as a separate trunk from the arch of the aorta, and in such cases it may be either the first, second, third, or even the last branch derived from that vessel. In the majority, however, it is the first or last, rarely the second or third. When it is the first branch, it occupies the ordinary position of the innominate artery; when the second or third, it gains its unusual position by passing behind the right carotid; and when the last branch, it arises from the left extremity of the arch, and passes obliquely toward the right side, usually behind the trachea, esophagus, and right carotid, sometimes between the esophagus and trachea, to the upper border of the first rib, whence it follows its ordinary course. In very rare instances, this vessel arises from the thoracic aorta, as low down as the fourth thoracic vertebra. Occasionally it perforates the scalenus anterior; more rarely it passes in front of that muscle. Sometimes the subclavian vein passes with the artery behind the scalenus anterior. The artery may ascend as high as 4 cm above the clavicle, or any intermediate point between this and the upper border of the bone, the right subclavian usually ascending higher than the left. The left subclavian is occasionally joined at its origin with the left carotid. The left subclavian artery is more deeply placed than the right in the first part of its course, and, as a rule, does not reach quite as high a level in the neck. The posterior border of the sternocleidomastoideus corresponds pretty closely to the lateral border of the scalenus anterior, so that the third portion of the artery, the part most accessible for operation, lies immediately lateral to the posterior border of the sternocleidomastoideus. Collateral circulation. After ligature of the third part of the subclavian artery, the collateral circulation is established mainly by 3 sets of vessels, thus described in a dissection. 1. A posterior set, consisting of the transverse scapula and the descending ramus of the transverse cervical branches of the subclavian, anastomosing with the subscapula from the axillary. 2. A medial set, produced by the connection of the internal mammary on the one hand with the highest intercostal and lateral thoracic arteries, and the branches from the subscapula on the other. 3. A middle or axillary set, consisting of a number of small vessels derived from branches of the subclavian, above, and passing through the axilla, terminating either in the main trunk, or some of the branches of the axillary below. This last set presented most conspicuously the peculiar character of newly formed, or rather dilated arteries, being excessively tortuous and forming a complete plexus. The chief agent in the restoration of the axillary artery below the tumour was the subscapular artery, which communicated most freely with the internal mammary, transverse scapular and descending ramus of the transverse cervical branches of the subclavian, from all of which it received so great an influx of blood as to dilate it to 3 times its natural size. Footnote: Guy's Hospital Reports, Volume 1, 1836, Case of axillary aneurysm, in which Aston Key had tied the subclavian artery on the lateral edge of the scalenus anterior 12 years previously. End of footnote. When a ligature is applied to the first part of the subclavian artery, the collateral circulation is carried on by 1. The anastomosis between the superior and inferior thyroids, 2. The anastomosis of the 2 vertebrals, 3. The anastomosis of the internal mammary with the inferior epigastric and the aortic intercostals, 4. The costocervical anastomosing with the aortic intercostals, 5. The profunda cervicis anastomosing with the descending branch of the occipital, 6. The scapular branches of the thyrocervical trunk anastomosing with the branches of the axillary, and 7. The thoracic branches of the axillary anastomosing with the aortic intercostals. Branches. The branches of the subclavian artery are vertebral, thyrocervical, internal mammary, costocervical. On the left side all 4 branches generally arise from the first portion of the vessel, but on the right side the costocervical trunk usually springs from the second portion of the vessel. On both sides of the neck the first 3 branches arise close together at the medial border of the scalenus anterior in the majority of cases. A free interval of from 1.25 to 2.5 cm exists between the commencement of the artery and the origin of the nearest branch. 1. The vertebral artery, arteria vertebralis, is the first branch of the subclavian and arises from the upper and back part of the first portion of the vessel. It is surrounded by a plexus of nerve fibres derived from the inferior cervical ganglion of the sympathetic trunk, and ascends through the foramina in the transverse processes of the upper 6 cervical vertebrae. Footnote: The vertebral artery sometimes enters the foramen in the transverse process of the 5th vertebra, and has been seen entering that of the 7th vertebra. End of footnote. It then winds behind the superior articular process of the atlas and, entering the skull through the foramen magnum, unites, at the lower border of the pons, with the vessel of the opposite side to form the basilar artery. Relations. The vertebral artery may be divided into 4 parts. The first part runs upward and backward between the longus colli and the scalenus anterior. In front of it are the internal jugular and vertebral veins, and it is crossed by the inferior thyroid artery. The left vertebral is crossed by the thoracic duct also. Behind it are the transverse process of the 7th cervical vertebra, the sympathetic trunk, and its inferior cervical ganglion. The second part runs upward through the foramina in the transverse processes of the upper 6 cervical vertebrae, and is surrounded by branches from the inferior cervical sympathetic ganglion and by a plexus of veins which unite to form the vertebral vein at the lower part of the neck. It is situated in front of the trunks of the cervical nerves, and pursues an almost vertical course as far as the transverse process of the atlas, above which it runs upward and lateralward to the foramen in the transverse process of the atlas. The third part issues from the latter foramen on the medial side of the rectus capitis lateralis, and curves backward behind the superior articular process of the atlas, the anterior ramus of the first cervical nerve being on its medial side. It then lies in the groove on the upper surface of the posterior arch of the atlas, and enters the vertebral canal by passing between the posterior atlanto-occipital membrane. This part of the artery is covered by the semispinalis capitis, and is contained in the suboccipital triangle, a triangular space bounded by the rectus capitis posterior major, the obliquus superior, and the obliquus inferior. The first cervical or suboccipital nerve lies between the artery and the posterior arch of the atlas. The fourth part pierces the dura mater and inclines medialward to the front of the medulla oblongata. It is placed between the hypoglossal nerve and the anterior root of the first cervical nerve, and beneath the first digitation of the ligamentum denticulatum. At the lower border of the pons, it unites with the vessel of the opposite side to form the basilar artery. Branches. The branches of the vertebral artery may be divided into two sets, those given off at the neck and those within the cranium. Cervical branch, spinal, cranial branches, meningeal. Cervical branch, muscular, cranial branches, posterior spinal, anterior spinal, posterior inferior cerebellar, medullary. Spinal branches, rami spinalis, enter the vertebral canal through the intervertebral foramina, and each divides into two branches. Of these, one passes along the roots of the nerves to supply the medulla spinalis and its membranes, anastomosing with the other arteries of the medulla spinalis. The other divides into an ascending and a descending branch, which unite with similar branches from the arteries above and below, so that two lateral anastomotic chains are formed on the posterior surfaces of the bodies of the vertebrae, near the attachment of the pedicles. From these anastomotic chains branches are supplied to the periosteum and the bodies of the vertebrae, and others form communications with similar branches from the opposite side. From these communications small twigs arise which join similar branches above and below, to form a central anastomotic chain on the posterior surface of the bodies of the vertebrae. Muscular branches are given off to the deep muscles of the neck, where the vertebral artery curves around the articular process of the atlas. They anastomose with the occipital and with the ascending and deep cervical arteries. The meningeal branch, ramus meningeus, posterior meningeal branch, springs from the vertebral, opposite the foramen magnum, ramifies between the bone and dura mater in the cerebellar fossa, and supplies the falx cerebelli. It is frequently represented by one or two small branches. The posterior spinal artery, A spinalis posterior, dorsal spinal artery, arises from the vertebral, at the side of the medulla oblongata. Passing backward, it descends on this structure, lying in front of the posterior roots of the spinal nerves, and is reinforced by a succession of small branches, which enter the vertebral canal through the intervertebral foramina. By means of these it is continued to the lower part of the medulla spinalis, and to the cauda equina. Branches from the posterior spinal arteries form a free anastomosis around the posterior roots of the spinal nerves, and communicate, by means of very tortuous transverse branches, with the vessels of the opposite side. Close to its origin, each gives off an ascending branch, which ends at the side of the fourth ventricle. The anterior spinal artery. Arteria spinalis anterior, ventral spinal artery, is a small branch which arises near the termination of the vertebral, and, descending in front of the medulla oblongata, unites with its fellow of the opposite side at the level of the foramen magnum. One of these vessels is usually larger than the other, but occasionally they are about equal in size. The single trunk thus formed descends on the front of the medulla spinalis, and is reinforced by a succession of small branches which enter the vertebral canal through the intervertebral foramina. These branches are derived from the vertebral and the ascending cervical of the inferior thyroid at the neck, from the intercostalis in the thorax, and from the lumbar, iliolumbar, and lateral sacral arteries in the abdomen and pelvis. They unite by means of ascending and descending branches to form a single anterior median artery, which extends as far as the lower part of the medulla spinalis, and is continued as a slender twig on the filum terminale. This vessel is placed in the pia mater along the anterior median fissure. It supplies that membrane and the substance of the medulla spinalis, and sends off branches at its lower part to be distributed to the cauda equina. The posterior inferior cerebellar artery, Arteria cerebelli inferior posterior, the largest branch of the vertebral, winds backward around the upper part of the medulla oblongata, passing between the origins of the vagus and accessory nerves, over the inferior peduncle to the undersurface of the cerebellum, where it divides into two branches. The medial branch is continued backward to the notch between the two hemispheres of the cerebellum, while the lateral supplies the undersurface of the cerebellum, as far as its lateral border, where it anastomoses with the anterior inferior cerebellar and the superior cerebellar branches of the basilar artery. Branches from this artery supply the choroid plexus of the fourth ventricle. The medullary arteries, bulbar arteries, are several minute vessels which spring from the vertebral and its branches, and are distributed to the medulla oblongata. End of section number 17. Section 18 of Gray's Anatomy, Part 3. This is a Librivox recording. All Librivox recordings are in the public domain. For more information or to volunteer, please visit LibriVox.org. Recording by Morgan Scorpion. Anatomy of the Human Body, Part 3, by Henry Gray. The subclavian artery, Part 2. The basilar artery, Arteria basilaris, so named from its position at the base of the skull, is a single trunk formed by the junction of the two vertebral arteries. It extends from the lower to the upper border of the pons, lying in its median groove, under cover of the arachnoid. It ends by dividing into the two posterior cerebral arteries. Its branches on either side are the following: pontine, anterior inferior cerebellar, internal auditory, superior cerebellar, posterior cerebral. The pontine branches, rami ad pontem, transverse branches, are a number of small vessels which come off at right angles from either side of the basilar artery and supply the pons and adjacent parts of the brain. The internal auditory artery, arterius auditiva interna, auditory artery, a long slender branch, arises from near the middle of the artery. It accompanies the acoustic nerve through the internal acoustic meatus, and is distributed to the internal ear. The anterior inferior cerebellar artery, arterius cerebelli inferior anterior, passes backward to be distributed to the anterior part of the undersurface of the cerebellum, anastomosing with the posterior inferior cerebellar branch of the vertebral. The superior cerebellar artery, arterius cerebelli superior, arises near the termination of the basilar. It passes lateralward immediately below the oculomotor nerve, which separates it from the posterior cerebral artery, winds around the cerebral peduncle close to the trochlear nerve, and, arriving at the upper surface of the cerebellum, divides into branches which ramify in the pia mater and anastomose with those of the inferior cerebellar arteries. Several branches are given to the pineal body, the anterior medullary velum and the tela choroidea of the third ventricle. The posterior cerebral artery, arteria cerebri posterior, is larger than the preceding, from which it is separated near its origin by the oculomotor nerve. Passing lateralward, parallel to the superior cerebellar artery, and receiving the posterior communicating from the internal carotid, it winds around the cerebral peduncle and reaches the tentorial surface of the occipital lobe of the cerebrum, where it breaks up into branches for the supply of the temporal and occipital lobes. The branches of the posterior cerebral artery are divided into two sets: ganglionic and cortical. Ganglionic, posterior medial, posterior choroidal, posterolateral. Cortical, anterior temporal, posterior temporal, calcarine, parieto-occipital. Ganglionic. The posteromedial ganglionic branches are a group of small arteries which arise at the commencement of the posterior cerebral artery. These, with similar branches from the posterior communicating, pierce the posterior perforated substance, and supply the medial surfaces of the thalami and the walls of the third ventricle. Posterior choroidal branches run forward beneath the splenium of the corpus callosum, and supply the tela choroidea of the third ventricle and the choroid plexus. The posterolateral ganglionic branches are small arteries which arise from the posterior cerebral artery after it has turned around the cerebral peduncle. They supply a considerable portion of the thalamus. Cortical. The cortical branches are the anterior temporal, distributed to the uncus and the anterior part of the fusiform gyrus, the posterior temporal, to the fusiform and the inferior temporal gyri, the calcarine, to the cuneus and gyrus lingualis and the back part of the convex surface of the occipital lobe, and the parieto-occipital, to the cuneus and the precuneus. 2. The thyrocervical trunk, truncus thyrocervicalis, thyroid axis, is a short, thick trunk which arises from the front of the first portion of the subclavian artery, close to the medial border of the scalenus anterior, and divides almost immediately into three branches: the inferior thyroid, transverse scapular and transverse cervical. The inferior thyroid artery, arteria thyroidea inferior, passes upward in front of the vertebral artery and longus colli, then turns medialward behind the carotid sheath and its contents, and also behind the sympathetic trunk, the middle cervical ganglion resting upon the vessel. Reaching the lower border of the thyroid gland, it divides into two branches, which supply the postero-inferior parts of the gland, and anastomose with the superior thyroid and with the corresponding artery of the opposite side. The recurrent nerve passes upward generally behind, but occasionally in front of the artery. The branches of the inferior thyroid are inferior laryngeal, tracheal, esophageal, ascending cervical, muscular. The inferior laryngeal artery, arteria laryngea inferior, ascends upon the trachea to the back part of the larynx under cover of the constrictor pharyngis inferior, in company with the recurrent nerve, and supplies the muscles and mucous membrane of this part, anastomosing with the branch from the opposite side and with the superior laryngeal branch of the superior thyroid artery. The tracheal branches, rami trachealis, are distributed upon the trachea, and anastomose below with the bronchial arteries. The esophageal branches, rami esophagae, supply the esophagus and anastomose with the esophageal branches of the aorta. The ascending cervical artery, arteria cervicalis ascendens, is a small branch which arises from the inferior thyroid as that vessel is passing behind the carotid sheath. It runs up on the anterior tubercles of the transverse processes of the cervical vertebrae in the interval between the scalenus anterior and longus capitis. To the muscles of the neck it gives twigs which anastomose with branches of the vertebral, and it sends one or two spinal branches into the vertebral canal through the intervertebral foramina to be distributed to the medulla spinalis and its membranes, and to the bodies of the vertebrae, in the same manner as the spinal branches from the vertebral. It anastomoses with the ascending pharyngeal and occipital arteries. The muscular branches supply the depressors of the hyoid bone and the longus colli, scalenus anterior and constrictor pharyngis inferior. The transverse scapular artery, arteria transversa scapulae suprascapular artery, passes at first downward and lateralward across the scalenus anterior and phrenic nerve, being covered by the sternocleidomastoideus. It then crosses the subclavian artery and the brachial plexus, and runs behind and parallel with the clavicle and the subclavius, and beneath the inferior belly of the omohyoideus, to the superior border of the scapula. It passes over the superior transverse ligament of the scapula, which separates it from the suprascapular nerve, and enters the supraspinatus fossa. In this situation it lies close to the bone, and ramifies between it and the supraspinatus, to which it supplies branches. It then descends behind the neck of the scapula, through the great scapular notch and under cover of the inferior transverse ligament, to reach the infraspinatus fossa, where it anastomoses with the scapular circumflex and the descending branch of the transverse cervical. Besides distributing branches to the sternocleidomastoideus, subclavius, and neighbouring muscles, it gives off a suprasternal branch, which crosses over the sternal end of the clavicle to the skin of the upper part of the chest, and an acromial branch, which pierces the trapezius and supplies the skin over the acromion, anastomosing with the thoracoacromial artery. As the artery passes over the superior transverse ligament of the scapula, it sends a branch into the subscapular fossa, where it ramifies beneath the subscapularis, and anastomoses with the subscapular artery, and with the descending branch of the transverse cervical. It also sends articular branches to the acromioclavicular and shoulder joints, and a nutrient artery to the clavicle. The transverse cervical artery, arteria transversa colli, transversalis colli artery, lies at a higher level than the transverse scapula. It passes transversely above the inferior belly of the omohyoideus to the anterior margin of the trapezius, beneath which it divides into an ascending and a descending branch. It crosses in front of the phrenic nerve and the scalene, and in front of or between the divisions of the brachial plexus, and is covered by the platysma and sternocleidomastoideus, and crossed by the omohyoideus and trapezius. The ascending branch, ramus ascendens, superficial cervical artery, ascends beneath the anterior margin of the trapezius, distributing branches to it, and to the neighbouring muscles and lymph glands in the neck, and anastomosing with the superficial branch of the descending ramus of the occipital artery. The descending branch, ramus descendens, posterior scapular artery, passes beneath the levator scapulae to the medial angle of the scapula, and then descends under the rhomboideae along the vertebral border of that bone as far as the inferior angle. It supplies the rhomboideae, latissimus dorsi and trapezius, and anastomosis with the transverse scapular and subscapular arteries, and with the posterior branches of some of the intercostal arteries. Peculiarities. The ascending branch of the transverse cervical frequently arises directly from the thyrocervical trunk, and the descending branch from the third, more rarely from the second, part of the subclavian. The internal mammary artery, arteria mammaria interna, arises from the under surface of the first portion of the subclavian, opposite the thyrocervical trunk. It descends behind the cartilages of the upper 6 ribs at a distance of about 1.25 cm from the margin of the sternum, and at the level of the 6th intercostal space divides into the musculophrenic and superior epigastric arteries. Relations. It is directed at first downward, forward, and medialward behind the sternal end of the clavicle, the subclavian and internal jugular veins, and the first costal cartilage, and passes forward close to the lateral side of the innominate vein. As it enters the thorax, the phrenic nerve crosses from its lateral to its medial side. Below the first costal cartilage it descends almost vertically to its point of bifurcation. It is covered in front by the cartilages of the upper 6 ribs and the intervening intercostales interni and anterior intercostal membranes, and is crossed by the terminal portions of the upper 6 intercostal nerves. It rests on the pleura as far as the third costal cartilage, below this level upon the transversus thoracis. It is accompanied by a pair of veins. These unite above to form a single vessel, which runs medial to the artery and ends in the corresponding innominate vein. Branches. The branches of the internal mammary are pericardiacophrenic, anterior mediastinal, pericardial, sternal, intercostal, perforating, musculophrenic, superior epigastric. The pericardiacophrenic artery, arteria pericardiacophrenica, arteria comes nervi phrenici, is a long slender branch, which accompanies the phrenic nerve, between the pleura and pericardium, to the diaphragm, to which it is distributed. It anastomoses with the musculophrenic and inferior phrenic arteries. The anterior mediastinal arteries, AA mediastinalis anteriores, mediastinal arteries, are small vessels, distributed to the areolar tissue and lymph glands in the anterior mediastinal cavity and to the remains of the thymus. The pericardial branches supply the upper part of the anterior surface of the pericardium. The lower part receives branches from the musculophrenic artery. The sternal branches, rami sternalis, are distributed to the transversus thoracis and to the posterior surface of the sternum. The anterior mediastinal, pericardial, and sternal branches, together with some twigs from the pericardiacophrenic, anastomose with branches from the intercostal and bronchial arteries, and form a subpleural mediastinal plexus. The intercostal branches, rami intercostalis, anterior intercostal arteries, supply the upper 5 or 6 intercostal spaces. 2 in number in each space, these small vessels pass lateralward, one lying near the lower margin of the rib above, and the other near the upper margin of the rib below, and anastomose with the intercostal arteries from the aorta. They are at first situated between the pleura and the intercostales interni, and then between the intercostales interni and externi. They supply the intercostales, and by branches which perforate the intercostales externi, the pectorales and the mamma. The perforating branches, rami perforantes, correspond to the 5 or 6 intercostal spaces. They pass forward through the intercostal spaces and, curving lateralward, supply the pectoralis major and the integument. Those which correspond to the 2nd, 3rd, and 4th spaces give branches to the mamma, and during lactation are of large size. The musculophrenic artery, arteria musculophrenica, is directed obliquely downward and lateralward, behind the cartilages of the false ribs. It perforates the diaphragm at the 8th or 9th costal cartilage, and ends, considerably reduced in size, opposite the last intercostal space. It gives off intercostal branches to the 7th, 8th, and 9th intercostal spaces. These diminish in size as the spaces decrease in length, and are distributed in a manner precisely similar to the intercostals from the internal mammary. The musculophrenic also gives branches to the lower part of the pericardium, and others which run backward to the diaphragm and downward to the abdominal muscles. The superior epigastric artery, arteria epigastrica superior, continues in the original direction of the internal mammary. It descends to the interval between the costal and sternal attachments of the diaphragm, and enters the sheath of the rectus abdominis, at first lying behind the muscle, and then perforating and supplying it, and anastomosing with the inferior epigastric artery from the external iliac. Branches perforate the anterior wall of the sheath of the rectus, and supply the muscles of the abdomen and the integument, and a small branch passes in front of the xiphoid process and anastomoses with the artery of the opposite side. It also gives some twigs to the diaphragm, while from the artery of the right side small branches extend into the falciform ligament of the liver, and anastomose with the hepatic artery. The costicocervical trunk, truncus costicocervicalis, superior intercostal artery, arises from the upper and back part of the subclavian artery, behind the scalenus anterior on the right side, and medial to that muscle on the left side. Passing backward, it gives off the profunda cervicalis, and, continuing as the highest intercostal artery, descends behind the pleura in front of the necks of the first and second ribs, and anastomoses with the first aortic intercostal. As it crosses the neck of the first rib, it lies medial to the anterior division of the first thoracic nerve, and lateral to the first thoracic ganglion of the sympathetic trunk. In the first intercostal space, it gives off a branch which is distributed in a manner similar to the distribution of the aortic intercostals. The branch for the second intercostal space usually joins with one from the highest aortic intercostal artery. This branch is not constant, but is more commonly found on the right side. When absent, its place is supplied by an intercostal branch from the aorta. Each intercostal gives off a posterior branch which goes to the posterior vertebral muscles, and sends a small spinal branch through the corresponding intervertebral foramen to the medulla spinalis and its membranes. The profunda cervicalis, arteria cervicalis profunda, deep cervical branch, arises in most cases from the costocervical trunk, and is analogous to the posterior branch of an aortic intercostal artery. Occasionally it is a separate branch from the subclavian artery. Passing backward, above the 8th cervical nerve and between the transverse process of the 7th cervical vertebra and the neck of the first rib, it runs up the back of the neck, between the semispinalis capitis and colli, as high as the axis vertebra, supplying these and adjacent muscles, and anastomosing with the deep division of the descending branch of the occipital and with branches of the vertebral. It gives off a spinal twig which enters the canal through the intervertebral foramen between the 7th cervical and 1st thoracic vertebra. End of section 18.
Section 19 of Gray's Anatomy Part 3. This is a Librivox recording. All Librivox recordings are in the public domain. For more information or to volunteer, please visit LibriVox.org. Recording by Ellie. Anatomy of the Human Body Part 3 by Henry Gray. The axilla, the axillary artery. 4b. The axilla. The axilla is a pyramidal space situated between the upper lateral part of the chest and the medial side of the arm. Boundaries. The apex, which is directed upward toward the root of the neck, corresponds to the interval between the outer border of the first rib, the superior border of the scapula, and the posterior surface of the clavicle, and through it the axillary vessels and nerves pass. The base, directed downward, is broad at the chest but narrow and pointed at the arm. It is formed by the integument and the thick layer of the fascia, the axillary fascia, extending between the lower border of the pectoralis major in front and the lower border of the latissimus dorsi behind. The anterior wall is formed by the pectoralis major and minor, the former covering the whole of this wall and the latter only its central part. The space between the upper border of the pectoralis minor and the clavicle is occupied by the coracoclavicular fascia. The posterior wall, which extends somewhat lower than the anterior, is formed by the subscapularis above, the teres major and the latissimus dorsi below. On the medial side are the first 4 ribs, with their corresponding intercostals, and part of the serratus anterior. On the lateral side, where the anterior and posterior walls converge, the space is narrow, and bounded by the humerus, the coracobrachialis, and the biceps brachii. Contents. It contains the axillary vessels and the brachial plexus of nerves, with their branches, some branches of the intercostal nerves, together with a quantity of fat and loose areolar tissue, the axillary artery and vein, with the brachial plexus of nerves, extend obliquely along the lateral boundary of the axilla, from its apex to its base, and are placed much nearer to the anterior than the posterior wall, the vein lying to the thoracic side of the artery and partially concealing it. At the forepart of the axilla, in contact with the pectoralis, are the thoracic branches of the axillary artery, and along the lower margin of the pectoralis minor, the lateral thoracic artery extends to the side of the chest. At the back part, in contact with the lower margin of the subscapularis, are the subscapular vessels and nerves. Winding around the lateral border of this muscle are the scapular circumflex vessels, and close to the neck of the humerus, the posterior humeral circumflex vessels, and the axillary nerve curved backward to the shoulder. Along the medial or thoracic side no vessel of importance exists, the upper part of the space being crossed merely by a few small branches from the highest thoracic artery. There are some important nerves, however, in this situation, viz. The long thoracic nerve, descending on the surface of the serratus anterior, to which it is distributed, and the intercostobrachial nerve, perforating the upper and anterior part of this wall, and passing across the axilla to the medial side of the arm. The position and arrangement of the lymph glands are described on pages 699 and 700. 1. The axillary artery, the continuation of the subclavian, commences at the outer border of the first rib and ends at the lower border of the tendon of the teres major, where it takes the name of brachial. Its direction varies with the position of the limb. Thus the vessel is nearly straight when the arm is directed at right angles with the trunk, concave upward when the arm is elevated above this, and convex upward and lateralward when the arm lies by the side. At its origin the artery is very deeply situated, but near its termination is superficial, being covered only by the skin and fascia. To facilitate the description of the vessel, it is divided into three portions. The first part lies above, the second behind, and the third below the pectoralis minor. Relations. The first portion of the axillary artery is covered anteriorly by the clavicular portion of the pectoralis major and the coracoclavicular fascia, and is crossed by the lateral anterior thoracic nerve and the thoracicoacromial and cephalic veins. Posterior to it are the first intercostal space, the corresponding intercostalis externus, the first and second digitations of the serratus anterior, and the long thoracic and medial anterior thoracic nerves, and the medial cord of the brachial plexus. On its lateral side is the brachial plexus, from which it is separated by a little areolar tissue. On its medial or thoracic side is the axillary vein, which overlaps the artery. It is enclosed together with the axillary vein and the brachial plexus in a fibrous sheath, the axillary sheath, continuous above with the deep cervical fascia. The second portion of the axillary artery is covered anteriorly by the pectoralis major and minor. Posterior to it are the posterior cord of the brachial plexus and some areolar tissue, which intervenes between it and the subscapularis. On the medial side is the axillary vein, separated from the artery by the medial cord of the brachial plexus and the medial anterior thoracic nerve. On the lateral side is the lateral cord of the brachial plexus. Brachial plexus thus surrounds the artery on three sides and separates it from direct contact with the vein and adjacent muscles. The third portion of the axillary artery extends from the lower border of the pectoralis minor to the lower border of the tendon of the teres major. In front it is covered by the lower part of the pectoralis major above, but only by the integument and fascia below. Behind it is in relation with the lower part of the subscapularis and the tendons of the latissimus dorsi and the teres major. On its lateral side is the coracobrachialis, and on its medial or thoracic side the axillary vein. The nerves of the brachial plexus bear the following relations to this part of the artery. On the lateral side are the lateral head and the trunk of the median. On the musculocutaneous, for a short distance, on the medial side the ulna between the vein and the artery, and the medial brachial cutaneous on the medial side of the vein. In front are the medial head of the median and the medial antebrachial cutaneous, and behind the radial and axillary, the latter only as far as the lower border of the subscapularis. Collateral circulation of the ligature of the axillary artery. If the artery be tied above the origin of the thoracoacromial, the collateral circulation will be carried on by the same branches as after the ligature on the third part of the subclavian. if at the lower point, between the thoracoacromial and the subscapular, the lateral vessel, by its free anastomosis with the transverse scapula and the transverse cervical branches of the subclavian, will become the chief agent in carrying on the circulation. The lateral thoracic, if it be below the ligature, will materially contribute by its anastomosis with the intercostal and internal mammary arteries. If the point included in the ligature is below the origin of the subscapular artery, it will most probably also be below the origins of the two humeral circumflex arteries. The chief agents in restoring the circulations will then be the subscapular and the two humeral circumflex arteries, anastomosing with the arteria profunda brachii. Branches of the maxillary are: from first part, highest thoracic; from second part, thoracoacromial, lateral thoracic; from third part, subscapular, posterior humeral circumflex, anterior humeral circumflex. 1. The highest thoracic artery, arteria thoracalis suprema, superior thoracic artery, is a small vessel which may arise from the thoracoacromial, running forward and medialward along the upper border of the pectoralis minor. It passes between it and the pectoralis major to the side of the chest. It supplies branches to these muscles and to the parties of the thorax and anastomosis with the internal mammary and intercostal arteries. 2. The thoracoacromial artery, arteria thoracoacromialis, acromiothoracic artery, thoracic axis, is a short trunk which arises from the forepart of the axillary artery, its origin being generally overlapped by the upper edge of the pectoralis minor. Projecting forward to the upper border of this muscle, it pierces the coracoclavicular fascia and divides it into 4 branches: pectoral, acromial, clavicular, and deltoid. The pectoral branch descends between the two pectorales and is distributed to them and to the mamma, anastomosing with the intercostal branches of the internal mammary and with the lateral thoracic. The acromial branch runs lateralward over the coracoid process and under the deltoideus, to which it gives branches. It then pierces the muscle and ends in the acromion in an arterial network formed by branches from the transverse scapular, thoracoacromial, and posterior humeral circumflex arteries. The clavicular branch runs upward and medialward to the sternoclavicular joint, supplying this articulation and the subclavius. The deltoid humeral branch, often arising with the acromial, crosses over the pectoralis minor and passes in the same groove as the cephalic vein, between the pectoralis major and deltoideus, and gives branches to both muscles. 3. The lateral thoracic artery, arteria thoracalis lateralis, long thoracic artery, external mammary artery, follows the lower border of the pectoralis minor to the side of the chest, supplying the serratus anterior and the pectoralis, and sending branches across the axilla to the axillary glands and the subscapularis. It anastomoses with the internal mammary, subscapular, and intercostal arteries, and with the pectoral branch of the thoracoacromial. In the female it supplies the external mammary branch, which turns around the free edge of the pectoralis major and supplies the mamma. 4. The subscapular artery, arteria subscapularis. The largest branch of the axillary artery arises at the lower border of the subscapularis, which it follows to the inferior angle of the scapula, where it anastomoses with the lateral thoracic and intercostal arteries, and with the descending branch of the transverse cervical, and ends in the neighboring muscles. About 4 centimeters from its origin it gives off a branch, the scapular circumflex artery. The scapular circumflex artery, arteria circumflexa scapulae, dorsalis scapulae artery, is generally larger than the continuation of the subscapular. It curves around the axillary border of the scapula, transversing the space between the subscapularis above, teres major below, and the long head of the triceps laterally. It enters the infraspinatus fossa, under cover of the teres minor, and anastomoses with the transverse scapular artery and the descending branch of the transverse cervical. In its course it gives off 2 branches. 1. Infrascapular, enters the subscapular fossa beneath the subscapularis, which it supplies, anastomosing with the transverse scapular artery and the descending branch of the transverse cervical. The other is continued along the axillary border of the scapula, between the teres major and minor, and at the dorsal surface of the inferior angle anastomosis with the descending branch of the transverse cervical. In addition to these, small branches are distributed to the back part of the deltoideus. and the long head of the triceps brachii, anastomosing with the ascending branch of the arteria profunda brachii. 5. The posterior humeral circumflex artery, arteria circumflexa humeri posterior, posterior circumflex artery, arises from the axillary artery at the lower border of the subscapularis, and runs backward with the axillary nerve through the quadrangular space bounded by the subscapularis and teres minor above, the teres major below, the long head or the triceps brachii medially, and the surgical neck of the humerus laterally. It winds around the neck of the humerus and is distributed to the deltoideus and shoulder joint, anastomosing with the anterior humeral circumflex and profunda brachii. 6. The anterior humeral circumflex artery, arteria circumflexa humeri anterior, anterior circumflex artery, considerably smaller than the posterior, arises nearly opposite it, from the lateral side of the axillary artery. It runs horizontally beneath the coracobrachialis and short head of the biceps brachii in front of the neck of the humerus. On reaching the intertubercular sulcus, it gives off a branch which ascends the sulcus to supply the head of the humerus and the shoulder joint. The trunk of the vessel is then continued onward beneath the long head of the biceps brachii and the deltoideus, and anastomosis with the posterior humeral circumflex artery. Peculiarities. The branches of the axillary artery vary considerably in different subjects. Occasionally the subscapular, humeral circumflex and profunda arteries arise from a common trunk, and when this occurs the branches of the brachial plexus surround this trunk instead of the main vessel. Sometimes the axillary artery divides into the radial and ulnar arteries, and occasionally it gives origin to the volar interosseous artery of the forearm. End of section 19. Recording by Ellie, August 2009.
Section 20 of Gray's Anatomy, Part 3. This is a Librivox recording. All Librivox recordings are in the public domain. For more information or to volunteer, please visit Librivox.org. Recording by Leanne Howlett. Anatomy of the Human Body, Part 3, by Henry Gray. The Brachial Artery. 4b. 2. The brachial artery. A. Brachialis. The brachial artery commences at the lower margin of the tendon of the teres major, and, passing down the arm, ends about 1 centimeter below the bend of the elbow, where it divides into the radial and ulnar arteries. At first the brachial artery lies medial to the humerus, but as it runs down the arm it gradually gets in front of the bone, and at the bend of the elbow it lies midway between its two epicondyles. Relations. The artery is superficial throughout its entire extent, being covered in front by the integument and the superficial and deep fascia. The laceratus fibrosus, bicipital fascia, lies in front of it opposite the elbow, and separates it from the vena mediana cubiti. The median nerve crosses from its lateral to its medial side opposite the insertion of the coracobrachialis. Behind, it is separated from the long head of the triceps brachii by the radial nerve and A profunda brachii. It then lies upon the medial head of the triceps brachii, next upon the insertion of the coracobrachialis, and lastly on the brachialis. Laterally, it is in relation above with the median nerve and the coracobrachialis, below with the biceps brachii, the two muscles overlapping the artery to a considerable extent. Medially, its upper half is in relation with the medial antebrachial cutaneous and ulnar nerves, its lower half with the median nerve. The basilic vein lies on its medial side, but is separated from it in the lower part of the arm by the deep fascia. The artery is accompanied by two venae comitants, which lie in close contact with it, and are connected together at intervals by short transverse branches. The antecubital fossa. At the bend of the elbow the brachial artery sinks deeply into a triangular interval, the antecubital fossa. The base of the triangle is directed upward, and is represented by a line connecting the two epicondyles of the humerus. The sides are formed by the medial edge of the brachioradialis and the lateral margin of the pronator teres. The floor is formed by the brachialis and supinator. This space contains the brachial artery, with its accompanying veins, the radial and ulnar arteries, the median and radial nerves, and the tendon of the biceps brachii. The brachial artery occupies the middle of the space and divides opposite the neck of the radius into the radial and ulnar arteries. It is covered in front by the integument, the superficial fascia, and the vena mediana cubiti, the last being separated from the artery by the lacertus fibrosus. Behind it is the brachialis, which separates it from the elbow joint. The median nerve lies close to the medial side of the artery above, but is separated from it below by the ulnar head of the pronator teres. The tendon of the biceps brachii lies to the lateral side of the artery. The radial nerve is situated upon the supinator and concealed by the brachioradialis. Peculiarities of the brachial artery as regards its course. The brachial artery, accompanied by the median nerve, may leave the medial border of the biceps brachii and descend toward the medial epicondyle of the humerus. In such cases it usually passes behind the supracondylar process of the humerus, from which a fibrous arch is in most cases thrown over the artery. It then runs beneath or through the substance of the pronator teres to the bend of the elbow. This variation bears considerable analogy with the normal condition of the artery in some of the carnivora. It has been referred to in the description of the humerus. As regards its division, occasionally the artery is divided for a short distance at its upper part into two trunks, which are united below. Frequently the artery divides at a higher level than usual, and the vessels concerned in this high division are three, viz., radial, ulnar, and interosseous. Most frequently the radial is given off high up, the other limb of the bifurcation consisting of the ulnar and interosseous. In some instances the ulnar rises above the ordinary level, and the radial and interosseous form the other limb of the division. Occasionally the interosseous arises high up. Sometimes long, slender vessels, vasa aberrantia, connect the brachial or the axillary artery with one of the arteries of the forearm or branches from them. These vessels usually join the radial. Varieties in muscular relations. The brachial artery is occasionally concealed in some part of its course by muscular or tendinous slips derived from the coracobrachialis, biceps brachii, brachialis, or pronator teres. Collateral circulation. After the application of a ligature to the brachial artery and the upper third of the arm, the circulation is carried on by branches from the humeral circumflex and subscapular arteries, anastomizing with ascending branches from the profunda brachii. If the artery be tied below the origin of the profunda brachii and superior ulnar collateral, the circulation is maintained by the branches of these two arteries anastomosing with the inferior ulnar collateral, the radial and ulnar recurrents, and the dorsal interosseous. Branches. The branches of the brachial artery are profunda brachii, superior ulnar collateral, nutrient, inferior ulnar collateral, muscular. 1. The arteria profunda brachii, superior profunda artery, is a large vessel which arises from the medial and back part of the brachial, just below the lower border of the teres major. It follows closely the radial nerve, running at first backward between the medial and lateral heads of the triceps brachii, then along the groove for the radial nerve, where it is covered by the lateral head of the triceps brachii to the lateral side of the arm. There it pierces the lateral intermuscular septum, and, descending between the brachioradialis and the brachialis to the front of the lateral epicondyle of the humerus, ends by anastomosing with the radial recurrent artery. It gives branches to the deltoideus and to the muscles between which it lies. It supplies an occasional nutrient artery which enters the humerus behind the deltoid tuberosity. A branch ascends between the long and lateral heads of the triceps brachii to anastomose with the posterior humeral circumflex artery. A middle collateral branch descends in the middle head of the triceps brachii and assists in forming the anastomosis above the olecranon, and lastly a radial collateral branch runs down behind the lateral intermuscular septum to the back of the lateral epicondyle of the humerus, where it anastomoses with the interosseous recurrent and the inferior ulnar collateral arteries. 2. The nutrient artery, A. nutritia humeri, of the body of the humerus, arises about the middle of the arm and enters the nutrient canal near the insertion of the coracobrachialis. 3. The superior ulnar collateral artery, A. collateralis ulnaris superior, inferior profunda artery, of small size, arises from the brachial a little below the middle of the arm. It frequently springs from the upper part of the A. profunda brachii. It pierces the medial intermuscular septum and descends on the surface of the medial head of the triceps brachii to the space between the medial epicondyle and olecranon, accompanied by the ulnar nerve, and ends under the flexor carpi ulnaris by anastomosing with the posterior ulnar recurrent and inferior ulnar collateral. It sometimes sends a branch in front of the medial epicondyle to anastomose with the inferior ulnar recurrent. 4. The inferior ulnar collateral artery, A. collateralis ulnaris inferior, anastomotica magna artery, arises about 5 cm above the elbow. It passes medialward upon the brachialis, and piercing the medial intermuscular septum, winds around the back of the humerus between the triceps brachii and the bone, forming, by its junction with the profunda brachii, an arch above the olecranon fossa. As the vessel lies on the brachialis, it gives off branches which ascend to join the superior ulnar collateral. Others descend in front of the medial epicondyle to anastomose with the anterior ulnar recurrent. Behind the medial epicondyle, a branch anastomoses with the superior ulnar collateral and posterior ulnar recurrent arteries. 5. The muscular branches, rami muscularis, 3 or 4 in number, are distributed to the coracobrachialis, biceps brachii, and brachialis. The anastomosis around the elbow joint. The vessels engaged in this anastomosis may be conveniently divided into those situated in front of and those behind the medial and lateral epicondyles of the humerus. The branches anastomosing in front of the medial epicondyle are the anterior branch of the inferior ulnar collateral, the anterior ulnar recurrent, and the anterior branch of the superior ulnar collateral. Those behind the medial epicondyle are the inferior ulnar collateral, the posterior ulnar recurrent, and the posterior branch of the superior ulnar collateral. The branches anastomosing in front of the lateral epicondyle are the radial recurrent and the terminal part of the profunda brachii. Those behind the lateral epicondyle, perhaps more properly described as being situated between the lateral epicondyle and the olecranon, are the inferior ulnar collateral, the interosseous recurrent, and the radial collateral branch of the profunda brachii. There is also an arch of anastomosis above the olecranon formed by the interosseous recurrent joining with the inferior ulnar collateral and posterior ulnar recurrent. End of section 20. Recording by Leanne Howlett. Section 21 of Gray's Anatomy, Part 3. This is a Librivox recording. All Librivox recordings are in the public domain. For more information or to volunteer, please visit Librivox.org. Recording by Leanne Howlett. Anatomy of the Human Body, Part 3, by Henry Gray. The Radial Artery. 4b. 3. The radial artery. A. Radialis. The radial artery appears, from its direction, to be the continuation of the brachial, but it is smaller in caliber than the ulnar. It commences at the bifurcation of the brachial, just below the bend of the elbow, and passes along the radial side of the forearm to the wrist. It then winds backward, around the lateral side of the carpus, beneath the tendons of the abductor pollicis longus and extensors pollicis longus and brevis, to the upper end of the space between the metacarpal bones of the thumb and index finger. Finally it passes forward between the two heads of the first interosseous dorsalis into the palm of the hand, where it crosses the metacarpal bones, and at the ulnar side of the hand unites with the deep volar branch of the ulnar artery to form the deep volar arch. The radial artery therefore consists of three portions, one in the forearm, a second at the back of the wrist, and a third in the hand. Relations. A. In the forearm, the artery extends from the neck of the radius to the forepart of the styloid process, being placed to the medial side of the body of the bone above and in front of it below. Its upper part is overlapped by the fleshy belly of the brachioradialis. The rest of the artery is superficial, being covered by the integument and the superficial and deep fasciae. In its course downward it lies upon the tendon of the biceps brachii, the supinator, the pronator teres, the radial origin of the flexor digitorum sublimis, the flexor pollicis longus, the pronator quadratus, and the lower end of the radius. In the upper third of its course it lies between the brachioradialis and the pronator teres. In the lower two-thirds, between the tendons of the brachioradialis and flexor carpi radialis. The superficial branch of the radial nerve is close to the lateral side of the artery and the middle third of its course, and some filaments of the lateral antibrachial cutaneous nerve run along the lower part of the artery as it winds around the wrist. The vessel is accompanied by a pair of venae comitants throughout its whole course. B. At the wrist, the artery reaches the back of the carpus by passing between the radial collateral ligament of the wrist and the tendons of the abductor pollicis longus and extensor pollicis brevis. It then descends on the navicular and greater multangular bones, and before disappearing between the heads of the first interosseous dorsalis, is crossed by the tendon of the extensor pollicis longus. In the interval between the two extensors pollicis, it is crossed by the digital rami of the superficial branch of the radial nerve, which go to the thumb and index finger. C. In the hand it passes from the upper end of the first interosseous space, between the heads of the first interosseous dorsalis, transversely across the palm between the adductor pollicis obliquus and adductor pollicis transversus, but sometimes piercing the latter muscle to the base of the metacarpal bone of the little finger, where it anastomoses with the deep volar branch from the ulnar artery, completing the deep volar arch. Peculiarities. The origin of the radial artery is, in nearly 1 case in 8, higher than usual. More often it arises from the axillary or upper part of the brachial than from the lower part of the latter vessel. In the forearm it deviates less frequently from its normal position than the ulnar. It has been found lying on the deep fascia instead of beneath it. It has also been observed on the surface of the brachioradialis instead of under its medial border, and in turning around the wrist it has been seen lying on instead of beneath the extensor tendons of the thumb. Branches. The branches of the radial artery may be divided into 3 groups, corresponding with the 3 regions in which the vessel is situated. In the forearm: radial recurrent, muscular, volar carpal, superficial volar. At the wrist: dorsal carpal, first dorsal metacarpal. In the hand: princeps pollicis, volaris indicis radialis, volar metacarpal, perforating, recurrent. The radial recurrent artery, A. recurrens radialis, arises immediately below the elbow. It ascends between the branches of the radial nerve, lying on the supinator and then between the brachioradialis and brachialis, supplying these muscles and the elbow joint, and anastomosing with the terminal part of the profunda brachii. The muscular branches, rami muscularis, are distributed to the muscles on the radial side of the forearm. The volar carpal branch, ramus carpius volaris, anterior radial carpal artery, is a small vessel which arises near the lower border of the pronator quadratus, and, running across the front of the carpus, anastomoses with the volar carpal branch of the ulnar artery. This anastomosis is joined by a branch from the volar interosseous above, and by recurrent branches from the deep volar arch below, thus forming a volar carpal network, which supplies the articulations of the wrist and carpus. The superficial volar branch, ramus volaris superficialis, superficialis voli artery, arises from the radial artery, just where this vessel is about to wind around the lateral side of the wrist. Running forward, it passes through, occasionally over, the muscles of the ball of the thumb, which it supplies, and sometimes anastomoses with the terminal portion of the ulnar artery, completing the superficial volar arch. This vessel varies considerably in size. Usually it is very small, and ends in the muscles of the thumb. Sometimes it is as large as the continuation of the radial. The dorsal carpal branch, ramus carpius dorsalis, posterior radial carpal artery, is a small vessel which arises beneath the extensor tendons of the thumb. Crossing the carpus transversely toward the medial border of the hand, it anastomoses with the dorsal carpal branch of the ulnar and with the volar and dorsal interosseous arteries to form a dorsal carpal network. From this network are given off three slender dorsal metacarpal arteries, which run downward on the second, third, and fourth interossei dorsalis and bifurcate into the dorsal digital branches for the supply of the adjacent sides of the middle, ring, and little fingers respectively, communicating with the proper volar digital branches of the superficial volar arch. Near their origins they anastomose with the deep volar arch by the superior perforating arteries, and near their points of bifurcation with the common volar digital vessels of the superficial volar arch by the inferior perforating arteries. The first dorsal metacarpal arises just before the radial artery passes between the two heads of the first interosseous dorsalis and divides almost immediately into two branches, which supply the adjacent sides of the thumb and index finger. The radial side of the thumb receives a branch directly from the radial artery. The arterior princeps pollicis arises from the radial just as it turns medialward to the deep part of the hand. It descends between the first interosseous dorsalis and adductor pollicis obliquus, along the ulnar side of the metacarpal bone of the thumb to the base of the first phalanx, where it lies beneath the tendon of the flexor pollicis longus, and divides into two branches. These make their appearance between the medial and lateral insertions of the adductor pollicis obliquus, and run along the sides of the thumb, forming on the volar surface of the last phalanx an arch, from which branches are distributed to the integument and subcutaneous tissue of the thumb. The arterior volaris indicis radialis indicis artery, arises close to the preceding, descends between the first interosseous dorsalis and adductor pollicis transversus, and runs along the radial side of the index finger to its extremity, where it anastomoses with the proper digital artery, supplying the ulnar side of the finger. At the lower border of the adductor pollicis transversus, this vessel anastomoses with the princeps pollicis and gives a communicating branch to the superficial volar arch. The A. princeps pollicis and A. volaris indicis radialis may spring from a common trunk termed the first volar metacarpal artery. The deep volar arch, arcus volaris profundus, deep palmar arch, is formed by the anastomosis of the terminal part of the radial artery with the deep volar branch of the ulnar. It lies upon the carpal extremities of the metacarpal bones and on the interossei, being covered by the adductor pollicis obliquus, the flexor tendons of the fingers, and the lumbricalis. Alongside of it, but running in the opposite direction, that is to say, toward the radial side of the hand, is the deep branch of the ulnar nerve. The volar metacarpal arteries, A.A. metacarpi, volaris, palmar interosseous arteries, 3 or 4 in number, arise from the convexity of the deep volar arch. They run distally upon the interossei and anastomose at the clefts of the fingers, with the common digital branches of the superficial volar arch. The perforating branches, rami perforantis, 3 in number, pass backward from the deep volar arch through the 2nd, 3rd, and 4th interosseous spaces and between the heads of the corresponding interossei dorsalis to anastomose with the dorsal metacarpal arteries. The recurrent branches arise from the concavity of the deep volar arch. They ascend in front of the wrist, supply the intercarpal articulations, and end in the volar carpal network. End of section 21. Recording by Leanne Howlett. Section 22 of Gray's Anatomy, Part 3. This is a Librivox recording. All Librivox recordings are in the public domain. For more information or to volunteer, please visit Librivox.org. Recording by Leanne Howlett. Anatomy of the Human Body, Part 3 by Henry Gray. The Ulnar Artery. 4b. 4. The ulnar artery. A. ulnaris. The ulnar artery, the larger of the 2 terminal branches of the brachial, begins a little below the bend of the elbow, and, passing obliquely downward, reaches the ulnar side of the forearm at a point about midway between the elbow and the wrist. It then runs along the ulnar border to the wrist, crosses the transverse carpal ligament on the radial side of the pisiform bone, and immediately beyond this bone divides into 2 branches, which enter into the formation of the superficial and deep volar arches. Relations. A. In the forearm. In its upper half it is deeply seated, being covered by the pronator teres, flexor carpi radialis, palmaris longus, and flexor digitorum sublimis. It lies upon the brachialis and flexor digitorum profundus. The median nerve is in relation with the medial side of the artery for about 2.5 centimeters, and then crosses the vessel, being separated from it by the ulnar head of the pronator teres. In the lower half of the forearm it lies upon the flexor digitorum profundus, being covered by the integument and the superficial and deep fasciae, and placed between the flexor carpi ulnaris and flexor digitorum sublimis. It is accompanied by 2 venae comitantes, and is overlapped in its middle third by the flexor carpi ulnaris. The ulnar nerve lies on the medial side of the lower 2/3 of the artery, and the palmar cutaneous branch of the nerve descends on the lower part of the vessel to the palm of the hand. B. At the wrist the ulnar artery is covered by the integument and the volar carpal ligament, and lies upon the transverse carpal ligament. On its medial side is the pisiform bone, and somewhat behind the artery the ulnar nerve. Peculiarities. The ulnar artery varies in its origin in the proportion of about 1 in 13 cases. It may arise about 5 to 7 centimeters below the elbow, but more frequently higher, the brachial being more often the source of origin than the axillary. Variations in the position of this vessel are more common than in the radial. When its origin is normal, the course of the vessel is rarely changed. When it arises high up, it is almost invariably superficial to the flexor muscles in the forearm, lying commonly beneath the fascia, more rarely between the fascia and integument. In a few cases its position was subcutaneous in the upper part of the forearm, and subaponeurotic in the lower part. Branches. The branches of the ulnar artery may be arranged in the following groups. In the forearm, anterior recurrent, posterior recurrent, common interosseous. At the wrist, volar carpal, dorsal carpal, deep volar. In the hand, muscular, superficial volar arch. The anterior ulnar recurrent artery, A recurrens ulnaris anterior, arises immediately below the elbow joint, runs upward between the brachialis and pronator teres, supplies twigs to those muscles, and in front of the medial epicondyle, anastomoses with the superior and inferior ulnar collateral arteries. The posterior ulnar recurrent artery, A recurrens ulnaris posterior, is much larger, and arises somewhat lower than the preceding. It passes backward and medialward on the flexor digitorum profundus, behind the flexor digitorum sublimis, and ascends behind the medial epicondyle of the humerus. In the interval between this process and the olecranon, it lies beneath the flexor carpi ulnaris, and ascending between the heads of that muscle, in relation with the ulnar nerve, it supplies the neighboring muscles and the elbow joint, and anastomoses with the superior and inferior ulnar collateral and the interosseous recurrent arteries. The common interosseous artery, A interossea communis, about 1 centimeter in length, arises immediately below the tuberosity of the radius, and, passing backward to the upper border of the interosseous membrane, divides into two branches, the volar and dorsal interosseous arteries. The volar interosseous artery, A interossea volaris, anterior interosseous artery, passes down the forearm on the volar surface of the interosseous membrane. It is accompanied by the volar interosseous branch of the median nerve, and overlapped by the contiguous margins of the flexor digitorum profundus and flexor pollicis longus, giving off in this situation muscular branches, and the nutrient arteries of the radius and ulna. At the upper border of the pronator quadratus, it pierces the interosseous membrane and reaches the back of the forearm, where it anastomoses with the dorsal interosseous artery. It then descends, in company with the terminal portion of the dorsal interosseous nerve, to the back of the wrist to join the dorsal carpal network. The volar interosseous artery gives off a slender branch, the arteria mediana, which accompanies the median nerve, and gives offsets to its substance. This artery is sometimes much enlarged, and runs with the nerve into the palm of the hand. Before it pierces the interosseous membrane, the volar interosseous sends a branch downward behind the pronator quadratus to join the volar carpal network. The dorsal interosseous artery, A interossea dorsalis, posterior interosseous artery, passes backward between the oblique cord and the upper border of the interosseous membrane. It appears between the contiguous borders of the supinator and the abductor pollicis longus, and runs down the back of the forearm between the superficial and deep layers of muscles, to both of which it distributes branches. Where it lies upon the abductor pollicis longus and the extensor pollicis brevis, it is accompanied by the dorsal interosseous nerve. At the lower part of the forearm, an anastomoses with the termination of the volar interosseous artery, and with the dorsal carpal network. It gives off near its origin the interosseous recurrent artery, which ascends to the interval between the lateral epicondyle and olecranon, on or through the fibers of the supinator, but beneath the anconeus, and anastomoses with the radial collateral branch of the profunda brachii, the posterior ulnar recurrent, and the inferior ulnar collateral. The muscular branches, rami muscularis, are distributed to the muscles along the ulnar side of the forearm. The volar carpal branch, ramus carpius volaris, anterior ulnar carpal artery, is a small vessel which crosses the front of the carpus beneath the tendons of the flexor digitorum profundus and anastomoses with the corresponding branch of the radial artery. The dorsal carpal branch, ramus carpius dorsalis, posterior ulnar carpal artery, arises immediately above the pisiform bone and winds backward beneath the tendon of the flexor carpi ulnaris. It passes across the dorsal surface of the carpus beneath the extensor tendons to anastomose with a corresponding branch of the radial artery. Immediately after its origin, it gives off a small branch, which runs along the ulnar side of the fifth metacarpal bone and supplies the ulnar side of the dorsal surface of the little finger. The deep volar branch, ramus volaris profundus, profunda branch, passes between the abductor digiti quinti and flexor digiti quinti brevis, and through the origin of the opponens digiti quinti, it anastomoses with the radial artery and completes the deep volar arch. The superficial volar arch, arcus volaris superficialis, superficial palmar arch, is formed by the ulnar artery and is usually completed by a branch from the A volaris indicis radialis, but sometimes by the superficial volar or by a branch from the A princeps pollicis of the radial artery. The arch passes across the palm, describing a curve with its convexity downward. Relations. The superficial volar arch is covered by the skin, the palmaris brevis and the palmar aponeurosis. It lies upon the transverse carpal ligament, the flexor digiti quinti brevis and opponens digiti quinti, the tendons of the flexor digitorum sublimis, the lumbricals, and the divisions of the median and ulnar nerves. Three common volar digital arteries, AA digitalis volaris communis, palmar digital arteries, arise from the convexity of the arch and proceed downward on the second, third, and fourth lumbricals. Each receives the corresponding volar metacarpal artery and then divides into a pair of proper volar digital arteries, AA digitalis volaris proprii, collateral digital arteries, which run along the contiguous sides of the index, middle, ring, and little fingers behind the corresponding digital nerves. They anastomose freely in the subcutaneous tissue of the fingertips and by smaller branches near the interphalangeal joints. Each gives off a couple of dorsal branches which anastomose with the dorsal digital arteries and supply the soft parts on the back of the second and third phalanges, including the matrix of the fingernail. The proper volar digital artery for medial side of the little finger springs from the ulnar artery under cover of the palmaris brevis. End of section 22. Recording by Leanne Howlett.
Section 23 of Gray's Anatomy, Part 3. This is a Librivox recording. All Librivox recordings are in the public domain. For more information or to volunteer, please visit librivox.org. Recording by Marianne Coleman Hipkins. Anatomy of the Human Body, Part 3 by Henry Gray. Arteries of the trunk, descending aorta, thoracic aorta. The descending aorta is divided into two portions, the thoracic and abdominal, in correspondence with the two great cavities of the trunk in which it is situated. 1. The thoracic aorta. Aorta thoracalis. The thoracic aorta is contained in the posterior mediastinal cavity. It begins at the lower border of the fourth thoracic vertebra, where it is continuous with the aortic arch, and ends in front of the lower border of the 12th, at the aortic hiatus in the diaphragm. At its commencement, it is situated on the left of the vertebral column. It approaches the median line as it descends, and, at its termination, lies directly in front of the column. The vessel describes a curve which is concave forward, and as the branches given off from it are small, its diminution in size is inconsiderable. Relations. It is in relation anteriorly from above downward, with the root of the left lung, the pericardium, the esophagus, and the diaphragm posteriorly, with the vertebral column and the hemiazygous veins on the right side, with the azygous vein and thoracic duct on the left side, with the left pleura and lung. The esophagus, with its accompanying plexus of nerves, lies on the right side of the aorta, above, but at the lower part of the thorax. It is placed in front of the aorta, and, close to the diaphragm, is situated on its left side. Peculiarities. The aorta is occasionally found to be obliterated at the junction of the arch with the thoracic aorta, just below the ductus arteriosus. Whether this is the result of disease or of congenital malformation is immaterial to our present purpose. It affords an interesting opportunity of observing the resources of the collateral circulation. The course of the anastomosing vehicles by which the blood is brought from the upper to the lower part of the artery will be found well described in an account of 2 cases in the Pathological Transactions, volumes 8 and 10. In the former, Sidney Jones thus sums up the detailed description of the anastomosing vessels. The principal communications by which the circulation was carried on were 1. The internal mammary anastomosing with the intercostal arteries, with the inferior phrenic of the abdominal aorta by means of the musculophrenic and periocardiacophrenic, and largely with the inferior epigastric. 2. The costo-cervical trunk anastomosing anteriorly by means of a large branch, with the first aortic intercostal, and posteriorly with the posterior branch of the same artery. 3. The inferior thyroid by means of a branch about the size of an ordinary radial, forming a communication with the first aortic intercostal. 4. The transverse cervical by means of a very large communications with the posterior branches of the intercostals. 5. The branches of the subclavian and axillary going to the side of the chest were large, and anastomosed freely with the lateral branches of the intercostals. In the second case, Wood describes the anastomoses in a somewhat similar manner, adding the remark that the blood which was brought into the aorta through the anastomosis of the intercostal arteries appeared to be expanded principally in supplying the abdomen and pelvis, while the supply to the lower extremities had passed through the internal mammary and epigastrics. In a few cases, an apparently double descending thoracic aorta has been found, the 2 vessels lying side by side, and eventually fusing to form a single tube in the lower part of the thorax, or in the abdomen. One of them is the aorta. The other represents a dissecting aortic aneurysm, which has become canalized, opening above and below into the true aorta, and at first sight presenting the appearances of a proper blood vessel. Branches of the thoracic aorta. Visceral. Pericardial, bronchial, esophageal, mediastinal. Parietal, intercostal, subcostal, superior phrenic. The pericardial branches, rami pericardiaceae, consist of a few small vessels which are distributed to the posterior surface of the pericardium. The bronchial arteries, AA bronchialis, vary in number, size, and origin. There is, as a rule, only one right bronchial artery, which arises from the first aortic intercostal, or from the upper left bronchial artery. The left bronchial arteries are usually 2 in number, and arise from the thoracic aorta. The upper left bronchiole arises opposite the fifth thoracic vertebra, the lower just below the level of the left bronchus. Each vessel runs on the back part of its bronchus, dividing and subdividing along the bronchial tubes, supplying them the areolar tissue of the lungs, the bronchial lymph glands, and the esophagus. The esophageal arteries, AA esophageae, 4 or 5 in number, arise from the front of the aorta and pass obliquely downward to the esophagus, forming a chain of anastomoses along that tube, anastomosing with the esophageal branches of the inferior thyroid arteries above and with ascending branches from the left inferior phrenic and left gastric arteries below. The mediastinal branches, rami mediastinalis, are numerous small vessels which supply the lymph glands and loose areolar tissue in the posterior mediastinum. Intercostal arteries, AA intercostalis. There are usually 9 pairs of aortic intercostal arteries. They arise from the back of the aorta and are redistributed to the lower 9 intercostal spaces, the first 2 spaces being supplied by the highest intercostal artery, a branch of the costo-cervical trunk of the subclavian. The right aortic intercostals are longer than the left, on account of the position of the aorta on the left side of the vertebral column. They pass across the bodies of the vertebrae behind the esophagus, thoracic duct, and vena ascygos, and are covered by the right lung and pleura. The left aortic intercostals run backward on the sides of the vertebrae and are covered by the left lung and pleura. The upper 2 vessels are crossed by the highest left intercostal vein, the lower vessels by the hemizygos veins. The further course of the intercostal arteries is practically the same on both sides. Opposite the heads of the ribs, the sympathetic trunk passes downward in front of them, and the splenic nerves also descend in front by the lower arteries. Each artery then divides into an anterior and a posterior ramus. The anterior ramus crosses the corresponding intercostal space obliquely toward the angle of the upper rib, and thence is continued forward in the costal groove. It is placed at first between the pleura and the posterior intercostal membrane. Then it pierces this membrane and lies between it and the intercostalis externus as far as the angle of the rib. From this onward it runs between the intercostalis externus and internus, and anastomoses in front with the intercostal branch of the internal mammary or musculophrenic. Each artery is accompanied by a vein and a nerve, the former being above and the latter below the artery, except in the upper spaces where the nerve is at first above the artery. The first aortic intercostal artery, anastomoses, with the intercostal branch of the costo-cervical trunk, and may form the chief supply of the second intercostal space. The lower 2 intercostal arteries are continued anteriorly from the intercostal spaces into the abdominal wall, and anastomose with the subcostal, superior epigastric, and lumbar arteries. Branches. The anterior rami give off the following branches: collateral intercostal, muscular, lateral cutaneous, mammary. The intercostal branch comes off from the intercostal artery near the angle of the rib, and descends to the upper border of the rib below, along which it courses to anastomose with the intercostal branch of the internal mammary. Muscular branches are given to the intercostalis and pectoralis, and to the serratus anterior. They anastomize with the highest and lateral thoracic branches of the axillary artery. The lateral cutaneous branches accompany the lateral cutaneous branches of the thoracic nerves. Mammary branches are given off by the vessels in the 3rd, 4th, and 5th spaces. They supply the mamma and increase considerably in size during the period of lactation. The posterior ramus runs backward through a space which is bounded above and below by the necks of the ribs, medially by the body of a vertebra, and laterally by an anterior costotransverse ligament. It gives off a spinal branch which enters the vertebral canal through the intervertebral foramen and is distributed to the medulla spinalis and its membranes and the vertebrae. It then courses over the transverse process with the posterior division of the thoracic nerve, supplies branches to the muscles of the back and cutaneous branches, which accompany the corresponding cutaneous branches of the posterior division of the nerve. The subcostal arteries, so named because they lie below the last ribs, constitute the lowest pair of branches derived from the thoracic aorta and are in series with the intercostal arteries. Each passes along the lower border of the 12th rib behind the kidney and in front of the quadratus lumborum muscle, and is accompanied by the 12th thoracic nerve. It then pierces the posterior aponeurosis of the transversus abdominis, and passing forward between this muscle and the obliquus internus anastomoses with the superior epigastric, lower intercostal, and lumbar arteries. Each subcostal artery gives off a posterior branch which has a similar distribution to the posterior ramus of an intercostal artery. The superior phrenic branches are small and arise from the lower part of the thoracic aorta. They are distributed to the posterior part of the upper surface of the diaphragm and anastomose with the musculophrenic and the periocardiacophrenic arteries. A small aberrant artery is sometimes found arising from the right side of the thoracic aorta, near the origin of the right bronchiole. It passes upward into the right behind the trachea and the esophagus, and may anastomose with the highest right intercostal artery. It represents the remains of the right dorsal aorta, and in a small proportion of cases is enlarged to form the first part of the right subclavian artery. End of section 23. Recording by Mary Ann Coleman Hipkins. Section 24 of Gray's Anatomy, Part 3. This is a Librivox recording. All Librivox recordings are in the public domain. For more information or to volunteer, please visit librivox.org. Recording by Mary Ann Coleman Hipkins. Anatomy of the Human Body, Part 3, by Henry Gray. The Abdominal Aorta, Part 1. 5a-2. The Abdominal Aorta. Aorta abdominalis. The abdominal aorta begins at the aortic hiatus of the diaphragm, in front of the lower border of the body of the last thoracic vertebra, and, descending in front of the vertebral column, ends on the body of the fourth lumbar vertebra, commonly a little to the left of the middle line. Footnote 103. Lord Lister, having accurately examined 30 bodies in order to ascertain the exact point of termination of this vessel, found it either absolutely or almost absolutely mesial in 15, while in 13 it deviated more or less to the left, and in 2 was slightly to the right. System of Surgery, edited by T. Holmes, 2nd edition, verse 652. End of footnote. By dividing into the 2 common iliac arteries, it diminishes rapidly in size in consequence of the many large branches which it gives off as it lies upon the bodies of the vertebrae. The curve which it describes is convex forward, the summit of the convexity corresponding to the third lumbar vertebra. Relations. The abdominal aorta is covered anteriorly by the lesser omentum and stomach, behind which are the branches of the celiac artery and the celiac plexus. Below these by the lyinal vein, the pancreas, the the left renal vein, the inferior part of the duodenum, the mesentery and aortic plexus. Posteriorly, it is separated from the lumbar vertebrae and intervertebral fibrocartilages by the anterior longitudinal ligament and left lumbar veins. On the right side, it is in relation above with the azygos vein, cisterna chyli, thoracic duct, and the right crus of the diaphragm, the last separating it from the upper part of the inferior vena cava and from the right celiac ganglion. The inferior vena cava is in contact with the aorta below. On the left side are the left crus of the diaphragm, the left celiac ganglion, the ascending part of the duodenum, and some coils of the small intestine. Collateral circulation. The collateral circulation would be carried on by the anastomoses between the internal mammary and the inferior epigastric, by the free communication between the superior and inferior mesenterics, if the ligature were placed between these vessels, or by the anastomosis between the inferior mesenteric and the internal pudendal, when, as is more common, the point of ligature is below the origin of the inferior mesenteric, and possibly by the anastomoses of the lumbar arteries with the branches of the hypogastric. Branches. The branches of the abdominal aorta may be divided into three sets: visceral, parietal, and terminal. Visceral branches: celiac, superior mesenteric, inferior mesenteric, middle suprarenals, renals, internal spermatics, ovarian in the female. Parietal branches: inferior phrenics, lumbars, middle sacral. Terminal branches: common iliacs. Of the visceral branches, the celiac artery and the superior and inferior mesenteric arteries are unpaired, while the suprarenals, renals, internal spermatics, and ovarian are paired. Of the parietal branches, the inferior phrenics and lumbars are paired, the middle sacral is unpaired, the terminal branches are paired. The celiac artery, A. ciliaca, celiac axis, is a short, thick trunk, about 1.25 centimeters in length, which arises from the front of the aorta, just below the aortic hiatus of the diaphragm, and passing nearly horizontally forward, divides into three large branches: the left gastric, the hepatic, and the splenic. It occasionally gives off one of the inferior phrenic arteries. Relations. The celiac artery is covered by the lesser omentum. On the right side, it is in relation with the right celiac ganglion and the caudate process of the liver. On the left side, with the left celiac ganglion and the cardiac end of the stomach. Below, it is in relation to the upper border of the pancreas and the lyinal vein. 1. The left gastric artery, A. gastrica sinistra, gastric or coronary artery. The smallest of the three branches of the celiac artery passes upward and to the left, posterior to the omental bursa, to the cardiac orifice of the stomach. Here it distributes branches to the esophagus, which anastomose with the aortic esophageal arteries. Others supply the cardiac part of the stomach, anastomosing with branches of the lyinal artery. It then runs from left to right along the lesser curvature of the stomach to the pylorus, between the layers of the lesser omentum. It gives branches to both surfaces of the stomach and anastomoses with the right gastric artery. 2. The hepatic artery, A. hepatica. In the adult is intermediate in size between the left gastric and lyinal. In the fetus, it is the largest of the three branches of the celiac artery. It is first directed forward and to the right, to the upper margin of the superior part of the duodenum, forming the lower boundary of the epiploic foramen of Winslow. It then crosses the portal vein anteriorly and ascends between the layers of the lesser omentum and in front of the epiploic foramen to the portal hepatis, where it divides into two branches, right and left, which supply the corresponding lobes of the liver, accompanying the ramifications of the portal vein and hepatic ducts. The hepatic artery, in its course along the right border of the lesser omentum, is in relation with the common bile duct and portal vein, the duct lying to the right of the artery and the vein behind. Its branches are right gastric epiploic, gastroduodenal, superior pancreatico-duodenal, cystic. The right gastric artery, A gastrica dextra, pyloric artery, arises from the hepatic above the pylorus, descends to the pyloric end of the stomach, and passes from right to left along its lesser curvature, supplying it with branches and anastomosing with the left gastric artery. The gastroduodenal artery, A gastroduodenalis, is a short but large branch which descends near the pylorus, between the superior part of the duodenum and the neck of the pancreas, and divides at the lower border of the duodenum into two branches, the right gastric epiploic and the superior pancreatico-duodenal. Previous to its division, it gives off 2 or 3 small branches to the pyloric end of the stomach and to the pancreas. The right gastric epiploic artery, A gastroepiploica dextra, runs from right to left along the greater curvature of the stomach, between the layers of the greater omentum, anastomosing with the left gastroepiploic branch of the lyinal artery. Except at the pylorus, where it is in contact with the stomach, it lies about a finger's breadth from the greater curvature. This vessel gives off numerous branches, some of which ascend to supply both surfaces of the stomach, while others descend to supply the greater omentum and anastomose with branches of the middle colic. The superior pancreatico-duodenal artery, A pancreatico-duodenalis superior, descends between the contiguous margins of the duodenum and pancreas. It supplies both these organs and anastomoses with the inferior pancreatico-duodenal branch of the superior mesenteric artery and with the pancreatic branches of the lyinal artery. The cystic artery, A cystica, usually a branch of the right hepatic, passes downward and forward along the neck of the gallbladder and divides into two branches, one of which ramifies on the free surface, the other on the attached surface of the gallbladder. The lyinal or splenic artery, A lyinalis, the largest branch of the celiac artery, is remarkable for the tortuosity of its course. It passes horizontally to the left side, behind the stomach and the omental bursa of the peritoneum, and along the upper border of the pancreas, accompanied by the lyinal vein, which lies below it. It crosses in front of the upper part of the left kidney, and on arriving near the spleen, divides into branches, some of which enter the hilus of that organ between the two layers of the phrenicolyinal ligament, to be distributed to the tissues of the spleen. Some are given to the pancreas, while others pass to the greater curvature of the stomach between the layers of the gastrolyinal ligament. Its branches are pancreatic, short gastric, left gastroepiploic. The pancreatic branches, rami pancreatici, are numerous small vessels derived from the lyinal as it runs behind the upper border of the pancreas, supplying its body and tail. One of these, larger than the rest, is sometimes given off near the tail of the pancreas. It runs from left to the right, near the posterior surface of the gland, following the course of the pancreatic duct, and is called the arteria pancreatica magna. These vessels anastomose with the pancreatic branches of the pancreatico-duodenal and superior mesenteric arteries. The short gastric arteries, AA gastricae brevis, vasa brevia, consist of from 5 to 7 small branches, which arise from the end of the lional artery and from its terminal divisions. They pass from left to right between the layers of the gastrolional ligament and are distributed to the greater curvature of the stomach, anastomosing with the branches of the left gastric and left gastropiploic arteries. The left gastropiploic artery, A. gastroepiploica sinistra, the largest branch of the lional, runs from left to right, about a finger's breadth or more from the greater curvature of the stomach, between the layers of the greater omentum, and anastomoses with the right gastroepiploic. In its course it distributes several ascending branches to both surfaces of the stomach. Others descend to supply the greater omentum and anastomose with branches of the middle colic. The superior mesenteric artery, A. mesenterica superior, is a large vessel which supplies the whole length of the small intestine except the superior part of the duodenum. It also supplies the caecum and the ascending part of the colon, and about 1/2 of the transverse part of the colon. It arises from the front of the aorta, about 1.25 centimeters below the celiac artery, and is crossed at its origin by the lional vein and the neck of the pancreas. It passes downward and forward, anterior to the processus inclinatus of the head of the pancreas and inferior part of the duodenum, and descends between the layers of the mesentery to the right iliac fossa, where, considerably diminished in size, it anastomoses with one of its own branches, viz. the ileocolic. In its course it crosses in front of the inferior vena cava, the right ureter and psoas major, and forms an arch, the convexity of which is directed forward and downward to the left side, the concavity backward and upward to the right. It is accompanied by the superior mesenteric vein, which lies to its right side, and it is surrounded by the superior mesenteric plexus of nerves. Branches. Its branches are inferior pancreatico-duodenal, intestinal, ileocolic, right colic, middle colic. The inferior pancreatico-duodenal artery, A. pancreatico-duodenalis inferior, is given off from the superior mesenteric or from its first intestinal branch, opposite the upper border of the inferior part of the duodenum. It courses to the right between the head of the pancreas and duodenum, and then ascends to anastomose with the superior pancreatico-duodenal artery. It distributes branches to the head of the pancreas and to the descending and inferior parts of the duodenum. The intestinal arteries, AA intestinalis, vasa intestini tenis, arise from the convex side of the superior mesenteric artery. They are usually from 12 to 15 in number, and are distributed to the jejunum and ileum. They run nearly parallel with one another between the layers of the mesentery, each vessel dividing into 2 branches, which unite with adjacent branches, forming a series of arches, the convexities of which are directed toward the intestine. From this first set of arches, branches arise, which unite with similar branches from above and below, and thus a second series of arches is formed. From the lower branches of the artery, a third, a fourth, or even a fifth series of arches may be formed, diminishing in size the nearer they approach the intestine. In the short upper part of the mesentery, only one set of arches exists, but as the depth of the mesentery increases, second, third, fourth, or even fifth groups are developed. From the terminal arches, numerous small, slight vessels arise, which encircle the intestine, upon which they are distributed, ramifying between its coats. From the intestinal arteries, small branches are given off to the lymph glands and other structures between the layers of the mesentery. The ileocolic artery, A. ileocolica, is the lowest branch arising from the concavity of the superior mesenteric artery. It passes downward and to the right behind the peritoneum toward the right iliac fossa, where it divides into a superior and an inferior branch. The inferior anastomoses with the end of the superior mesenteric artery, the superior with the right colic artery. The inferior branch of the ileocolic runs toward the upper border of the ileocolic junction and supplies the following branches: A. Colic, which pass upward on the ascending colon. B. Anterior and posterior cecal, which are distributed to the front and back of the cecum. C. An appendicular artery, which descends behind the termination of the ileum and enters the mesenteriole of the veriform process. It runs near the free margin of this mesenteriole and ends in branches which supply the veriform process. And D. Ileal, which run upward and to the left on the lower part of the ileum and anastomose with the termination of the superior mesenteric. The right colic artery, A. colica dextra, arises from the middle of the concavity of the superior mesenteric artery, or from a stem common to it and the ileocolic. It passes to the right behind the peritoneum and in front of the right internal spermatic or ovarian vessels, the right ureter and the psoas major, toward the middle of the ascending colon. Sometimes the vessel lies at a higher level and crosses the descending part of the duodenum and the lower end of the right kidney. At the colon it divides into a descending branch which anastomoses with the ileocolic, and an ascending branch which anastomoses with the middle colic. These branches form arches, from the convexity of which the vessels are distributed to the ascending colon. The middle colic artery, A. colica media, arises from the superior mesenteric just below the pancreas, and passing downward and forward between the layers of the transverse mesocolon, divides into two branches, right and left. The former anastomoses with the right colic, the latter with the left colic, a branch of the inferior mesenteric. The arches thus formed are placed about 2 fingers' breadth from the transverse colon, to which they distribute branches. End of section 24. Recording by Mary Ann Coleman Hipkins. Section 25 of Gray's Anatomy, Part 3. This is a Librivox recording. All Librivox recordings are in the public domain. For more information or to volunteer, please visit librivox.org. Recording by Mary Ann Coleman Hipkins. Anatomy of the Human Body, Part 3, by Henry Gray. The Abdominal Aorta, Part 1. 5A2. The Abdominal Aorta, Aorta Abdominalis. The Inferior Mesenteric Artery, A. mesenterica inferior, supplies the left half of the transverse part of the colon, the whole of the descending and iliac parts of the colon, the sigmoid colon, and the greater part of the rectum. It is smaller than the superior mesenteric and arises from the aorta about 3 or 4 centimeters above its division into the common iliacs and close to the lower border of the inferior part of the duodenum. It passes downward posterior to the peritoneum, lying at first anterior to and then on the left side of the aorta. It crosses the left common iliac artery and is continued into the lesser pelvis under the name of the superior hemorrhoidal artery, which descends between the 2 layers of the sigmoid mesocolon and ends on the upper part of the rectum. Branches. Its branches are left colic, sigmoid, superior hemorrhoidal. The left colic artery, A. Colica sinistra. Runs from the left behind the peritoneum and in front of the psoas major, and after a short but variable course divides into an ascending and a descending branch. The stem of the artery or its branches cross the left ureter and left internal spermatic vessels. The ascending branch crosses in front of the left kidney and ends between the two layers of the transverse mesocolon by anastomosing with the middle colic artery. The descending branch anastomoses with the highest sigmoid artery. From the arches formed by these anastomoses, branches are distributed to the descending colon and the left part of the transverse colon. The sigmoid arteries, AA sigmoideae, 2 or 3 in number, run obliquely downward and to the left behind the peritoneum and in front of the psoas major, ureter, and internal spermatic vessels. Their branches supply the lower part of the descending colon, the iliac colon, and the sigmoid or pelvic colon, anastomosing above with the left colic and below with the superior hemorrhoidal artery. The superior hemorrhoidal artery, A. Hemorrhoidalis superior. The continuation of inferior mesenteric descends into the pelvis between the layers of the mesentery of the sigmoid colon. Crossing in its course the left common iliac vessels, it divides opposite the third sacral vertebra into 2 branches, which descend one on either side of the rectum, and about 10 or 12 centimeters from the anus break up into several small branches. These pierce the muscular coat of the bowel and run downward as straight vessels placed at regular intervals from each other in the wall of the gut between its muscular and mucous coats, to the level of the sphincter ani internus. Here they form a series of loops around the lower end of the rectum and communicate with the middle hemorrhoidal branches of the hypogastric and with the inferior hemorrhoidal branches of the internal pudendal. The middle suprarenal arteries, AA suprarenales media, middle capsular arteries, suprarenal arteries, are 2 small vessels which arise, one from either side of the aorta, opposite the superior mesenteric artery. They pass lateralward and slightly upward over the crura of the diaphragm to the suprarenal glands, where they anastomose with the suprarenal branches of the inferior phrenic and renal arteries. In the fetus, these arteries are of large size. The renal arteries, AA renalis, are 2 large trunks which arise from the side of the aorta, immediately below the superior mesenteric artery. Each is directed across the crus of the diaphragm so as to form nearly a right angle with the aorta. The right is longer than the left on account of the position of the aorta. It passes behind the inferior vena cava, the right renal vein, the head of the pancreas, and the descending part of the duodenum. The left is somewhat higher than the right. It lies behind the left renal vein, the body of the pancreas, and the lional vein, and is crossed by the inferior mesenteric vein. Before reaching the hilus of the kidney, each artery divides into 4 or 5 branches. The greater number of these lie between the renal vein and ureter, the vein being in front, the ureter behind, but 1 or more branches are usually situated behind the ureter. Each vessel gives off small inferior suprarenal branches to the suprarenal gland, the ureter, and the surrounding cellular tissue and muscle. 1 or 2 accessory renal arteries are frequently found, more especially on the left side. They usually arise from the aorta and may come off above or below the main artery, the former being the more common position, instead of entering the kidney at the hilus. They usually pierce the upper or lower part of the gland. The internal spermatic arteries, AA spermaticae internae, spermatic arteries, are distributed to the testes. They are 2 slender vessels of considerable length and arise from the front of the aorta a little below the renal arteries. Each passes obliquely downward and lateralward behind the peritoneum, resting on the psoas major. The right spermatic line in front of the inferior vena cava and behind the middle colic and ileocolic arteries and the terminal part of the ileum. The left behind the left colic and sigmoid arteries and the iliac colon. Each crosses obliquely over the ureter and the lower part of the external iliac artery to reach the abdominal inguinal ring, through which it passes, and accompanies the other constituents of the spermatic cord along the inguinal canal to the scrotum, where it becomes tortuous and divides into several branches. 2 or 3 of these accompany the ductus deferens and supply the epididymis, anastomosing with the artery of the ductus deferens. Others pierce the back of the tunica albuginea and supply the substance of the testes. The internal spermatic artery supplies 1 or 2 small branches to the ureter, and in the inguinal canal gives 1 or 2 twigs to the cremaster. The ovarian arteries, AA ovaricae, are the corresponding arteries in the female to the internal spermatic in the male. They supply the ovaries, are shorter than the internal spermatics, and do not pass out of the abdominal cavity. The origin and course of the first part of each artery are the same as those of the internal spermatic, but on arriving at the upper opening of the lesser pelvis, the ovarian artery passes inward between the 2 layers of the ovariopelvic ligament and of the broad ligament of the uterus, to be distributed to the ovary. Small branches are given to the ureter and the uterine tube, and one passes on to the side of the uterus and unites with the uterine artery. The other offsets are continued on the round ligament of the uterus through the inguinal canal to the integument of the labium majus and groin. At an early period of fetal life, when the testes or ovaries lie by the side of the vertebral column below the kidneys, the internal spermatic or ovarian arteries are short, but with the descent of these organs into the scrotum or lesser pelvis, the arteries are gradually lengthened. The inferior phrenic arteries, AA phrenicae inferiores, are 2 small vessels which supply the diaphragm, but present much variety in their origin. They may arise separately from the front of the aorta, immediately above the celiac artery, or by a common trunk which may spring either from the aorta or from the celiac artery. Sometimes one is derived from the aorta and the other from one of the renal arteries. They rarely arise as separate vessels from the aorta. They diverge from one another across the carrura of the diaphragm, and then run obliquely upward and lateralward upon its surface. The left phrenic passes behind the esophagus and runs forward on the left side of the esophageal hiatus. The right phrenic passes behind the inferior vena cava and along the right side of the foramen, which transmits that vein. Near the back part of the central tendon, each vessel divides into a medial and a lateral branch. The medial branch curves forward and anastomoses with its fellow of the opposite side, and with the musculophrenic and pericardiacophrenic arteries. The lateral branch passes toward the side of the thorax and anastomoses with the lower intercostal arteries and with the musculophrenic. The lateral branch of the right phrenic gives off a few vessels to the inferior vena cava, and the left one some branches to the esophagus. Each vessel gives off superior suprarenal branches to the suprarenal gland of its own side. The spleen and the liver also receive a few twigs from the left and the right vessels respectively. The lumbar arteries, AA lumbalis, are in series with the intercostals. They are usually 4 in number on either side, and arise from the back of the aorta, opposite the bodies of the upper 4 lumbar vertebrae. A 5th pair, small in size, is occasionally present when they arise from the middle sacral artery. They run lateralward and backward on the bodies of the lumbar vertebrae, behind the sympathetic trunk, to the intervals between the adjacent transverse processes, and are then continued into the abdominal wall. The arteries of the right side pass behind the inferior vena cava, and the upper 2 on each side run behind the corresponding crust of the diaphragm. The arteries of both sides pass beneath the tendinous arches, which give origin to the psoas major, and are then continued behind this muscle and the lumbar plexus. They now cross the quadratus lumborum, the upper 3 arteries running behind, the last usually in front of the muscle. At the lateral border of the quadratus lumborum, they pierce the posterior aponeurosis of the transversus abdominis, and are carried forward between this muscle and the obliquus internus. They anastomose with the lower intercostal, the subcostal, the iliolumbar, the the deep iliac circumflex, and the inferior epigastric arteries. Branches. In the interval between the adjacent transverse processes, each lumbar artery gives off a posterior ramus, which is continued backward between the transverse processes and is distributed to the muscles and skin of the back. It finishes a spinal branch which enters the vertebral canal and is distributed in a manner similar to the spinal branches of the posterior rami of the intercostal arteries. Muscular branches are supported from each lumbar artery and from its posterior ramus to the neighbouring muscles. The middle sacral artery, A sacralis media, is a small vessel which arises from the back of the aorta a little above its bifurcation. It descends in the middle line in front of the fourth and fifth lumbar vertebrae, the sacrum and coccyx, and ends in the glomus coccygeum, coccygeal gland. From it, minute branches are said to pass to the posterior surface of the rectum. On the last lumbar vertebrae, it anastomoses with the lumbar branch of the iliolumbar artery. In front of the sacrum, it anastomoses with the lateral sacral arteries and sends offshoots into the anterior sacral foramina. It is crossed by the left common iliac vein and is accompanied by a pair of venae committantes. These unite to form a single vessel which opens into the left common iliac vein. End of section 25. Recording by Marianne Coleman Hipkins. www.thisvoiceforyou.com.
Section 26 of Gray's Anatomy, Part 3. This is a Librivox recording. All Librivox recordings are in the public domain. For more information or to volunteer, please visit librivox.org. Anatomy of the Human Body, Part 3, by Henry Gray. The Common Iliac Arteries. A. Iliacae communes. The abdominal aorta divides, on the left side of the body of the fourth lumbar vertebra, into the two common iliac arteries. Each is about 5 centimeters in length. They diverge from the termination of the aorta, pass downward and lateralward, and divide opposite the invertebral fibrocartilage between the last lumbar vertebra and the sacrum, into 2 branches, the external iliac and hypogastric arteries. The former supplies the lower extremity, the latter the viscera and parietes of the pelvis. The right common iliac artery is somewhat longer than the left, and passes more obliquely across the body of the last lumbar vertebra. In front of it are the peritoneum, the small intestines, branches of the sympathetic nerves, and, at its point of division, the ureter. Behind, it is separated from the bodies of the fourth and fifth lumbar vertebrae, and the intervening fibrocartilage, by the terminations of the 2 common iliac veins, and the commencement of the inferior vena cava. Laterally, it is in relation above with the inferior vena cava and the right common iliac vein, and below with the sosus major. Medial to it above is the left common iliac vein. The left common iliac artery is in relation, in front, with the peritoneum, the small intestines, branches of the sympathetic nerves, and the superior hemorrhoidal artery, and it is crossed at the point of bifurcation by the ureter. It rests on the bodies of the fourth and fifth lumbar vertebrae, and the intervening fibrocartilage. The left common iliac vein lies partly medial to and partly behind the artery. Laterally, the artery is in relation with the sosus major. Branches. The common iliac arteries give off small branches to the peritoneum, psoas major, ureters, and the surrounding areolar tissue, and occasionally give origin to the iliolumbar or accessory renal arteries. Peculiarities. The point of origin varies according to the bifurcation of the aorta. In 3/4 of a large number of cases, the aorta bifurcated either upon the fourth lumbar vertebra or upon the fibrocartilage between it and the fifth, the bifurcation being, in 1 case out of 9, below, and in 1 out of 11 above this point. In about 80% of the cases, the aorta bifurcated within 1.5 centimeters above or below the level of the crest of the ilium, more frequently below than above. The point of division is subject to great variety. In 2/3 of a large number of cases it was between the last lumbar vertebra and the upper border of the sacrum, being above that point in 1 case out of 8, and below it in 1 case out of 6. The left common iliac artery divides lower down more frequently than the right. The relative lengths also of the 2 common iliac arteries vary. The right common iliac was the longer in 63 cases, the left in 52, while they were equal in 53. The length of the arteries varied, in 5/7 of the cases examined, from 3.5 to 7.5 centimeters. In about half of the remaining cases the artery was longer, and in the other half shorter, the minimum length being less than 1.5 centimeters, the maximum 11 centimeters. In rare instances the right common iliac has been found wanting, the external iliac and hypogastric arising directly from the aorta. Collateral circulation. The principal agents in carrying on the collateral circulation after the application of a ligature to the common iliac are the asthenosis of the hemorrhoidal branches of the hypogastric with the superior hemorrhoidal from the inferior mesenteric, of the uterine, ovarian, and vesical arteries of the opposite sides, of the lateral sacral with the middle sacral artery, of the inferior epigastric with the internal mammary, inferior intercostal, and lumbar arteries, of the deep iliac circumflex with the lumbar arteries, of the iliolumbar with the last lumbar artery, of the obturator artery by means of its pubic branch with the vessel of the opposite side and with the inferior epigastric. End of section 26. Recording by David Lawrence in Brampton, Ontario, July the 4th, 2009.
Section 23 of Gray's Anatomy Part 3. This is a Librivox recording. All Librivox recordings are in the public domain. For more information or to volunteer, please visit LibriVox.org. Recording by Ellie. Anatomy of the Human Body, Part 3, by Henry Gray. 1. The hypogastric artery. Arteria hypogastrica, internal iliac artery. The hypogastric artery supplies the walls and the viscera of the pelvis, the buttock, the generative organs, and the medial side of the thigh. It is a short, thick vessel, smaller than the external iliac, about 4 centimeters in length. It arises at the bifurcation of the common iliac, opposite the lumbosacral articulation, and passing downward to the upper margin of the greater sciatic foramen, divides into 2 large trunks, an anterior and a posterior. Relations. It is in relation in front with the ureter, behind with the internal iliac vein, the lumbosacral trunk, and the piriformis muscle, laterally near its origin with the external iliac vein which lies between it and the psoas major muscle, lower down with the obturator nerve. In the fetus the hypogastric artery is twice as large as the external iliac, and is the direct continuation of the common iliac. It descends along the side of the bladder and runs downward on the back of the anterior wall of the abdomen to the umbilicus, converging towards its fellow of the opposite side. Having passed through the umbilical opening, the 2 arteries, now termed umbilical, enter the umbilical cord, where they are coiled around the umbilical vein, and ultimately ramify in the placenta. At birth, when the placental circulation ceases, the pelvic portion only of the artery remains patent, and constitute the hypogastric and the first part of the superior vesical artery of the adult. The remainder of the vessel is converted into a solid fibrous cord, the lateral umbilical ligament, obliterated hypogastric artery, which extends from the pelvis to the umbilicus. Peculiarities as regards length. In 2/3 of a large number of cases, the length of the hypogastric varied between 2.25 and 3.4 centimeters. In the remaining third it was more frequently longer than shorter. the maximum length being about 7 centimeters, the minimum about 1 centimeter. The length of the common iliac, the hypogastric arteries, be in inverse proportion to each other, the hypogastric artery being long when the common iliac is short, and vice versa. As regards its place of division, the place of division of the hypogastric varies between the upper margin of the sacrum and the upper border of the greater sciatic foramen. The right and left hypogastric arteries in a series of cases often differed in length, but neither seemed constantly to exceed the other. Collateral circulation. The circulation of the ligature of the hypogastric artery is carried on by the anastomosis of the uterine and ovarian arteries, of the vesical arteries of the two sides, of the hemorrhoidal branches of the hypogastric with those from the inferior mesenteric, of the obturator artery by means of its pubic branch, with the vessel of the opposite side, and with the inferior epigastric and medial femoral circumflex, of the circumflex and perforating branches of the profunda femoris, with the inferior gluteal, of the superior gluteal with the posterior branches of the lateral sacral arteries, of the iliolumbar with the last lumbar, of the lateral sacral with the middle sacral, and of the iliac circumflex with the iliolumbar and superior gluteal. Branches. The branches of the hypogastric artery are from the anterior trunk, superior vesical, middle vesical, inferior vesical, middle hemorrhoidal, obturator, internal padental, inferior gluteal. In the female, uterine, vaginal. From the posterior trunk, iliolumbar, lateral sacral, superior gluteal. The superior vesical artery, arteria vesicalis superior, supplies numerous branches to the upper part of the bladder. From one of these slender vessels, the artery of the ductus deferens takes origin and accompanies the duct in its course to the testis, where it anastomoses with the internal spermatic artery. Other branches supply the ureter. The first part of the superior vesical artery represents the terminal section of the pervious portion of the fatal hypogastric artery. The middle vesical artery, arteria vesicalis medialis, usually a branch of the superior, is distributed to the fundus of the bladder and the vesiculae seminales. The inferior vesical artery, arteria vesicalis inferior, frequently arises in common with the middle hemorrhoidal and is distributed to the fundus of the bladder, the prostate, and the vesiculae seminales. The branches of the prostate communicate with the corresponding vessels of the opposite side. The middle hemorrhoidal artery, arteria hemorrhoidalis media, usually arises with the preceding vessel. It is distributed to the rectum, anastomosing with the inferior vesicle and with the superior and inferior hemorrhoidal arteries. It gives offsets to the vesiculae seminalis and prostate. The uterine artery, arteria uterina, springs from the anterior division of the hypogastric and runs medialward on the levata ani, and toward the cervix uteri about 2 centimeters from the cervix. It crosses above and in front of the ureter, to which it supplies a small branch. Reaching the side of the uterus, it ascends in a tortuous manner between the two layers of the broad ligament to the junction of the uterine tube and uterus. It then runs lateralward toward the hilus of the ovary, and ends by joining the ovarian artery. It supplies branches to the cervix uteri and others which descend to the vagina. The latter anastomose with branches of the vaginal arteries, and form with them two median longitudinal vessels, the ascicus arteries of the vagina, one of which runs down in front of the vagina and the other behind. It supplies numerous branches of the body of the uterus, and from its terminal portion twigs are distributed to the uterine tube and the round ligament of the uterus. The vaginal artery, arteria vaginalis, usually corresponds to the inferior vesicle in the male. It descends upon the vagina, supplying its mucous membrane, and sends branches to the bulb of the vestibule, the fundus of the bladder, and the contiguous part of the rectum. It assists in forming the ascicus arteries of the vagina, and is frequently represented by 2 or 3 branches. The obturator artery, arteria obturatoria, passes forward and downward on the lateral wall of the pelvis, and to the upper part of the obturator foramen, and, escaping from the pelvic cavity through the obturator channel, it divides into an anterior and a posterior branch. In the pelvic cavity, this vessel is in relation, laterally, with the obturator fascia, medially with the ureter, ductus deferens, and peritoneum, while a little below it is the obturator nerve. Branches. Inside the pelvis, The obturator artery gives off the iliac branches to the iliac fossa, which supply the bone and the iliacus, and anastomose with the iliolumbar artery, a vesical branch which runs backward to supply the bladder, and a pubic branch, which is given off from the vessel just before it leaves the pelvic cavity. The pubic branch ascends upon the back of the pubis, communicating with the corresponding vessel of the opposite side and with the inferior epigastric artery. Outside the pelvis, the obturator artery divides at the upper margin of the obturator foramen into an anterior and a posterior branch, which encircle the foramen under the cover of the obturator externus. The anterior branch, which runs forward on the outer surface of the obturator membrane and then curves downward along the anterior margin of the foramen, distributes branches to the obturator externus, pectineus, adductores, and gracilis, and anastomosis with the posterior branch and with the medial femoral circumflex artery. The posterior branch follows the posterior margin of the foramen and turns downward on the inferior ramus of the ischium, where it anastomosis with the anterior branch. It gives twigs to the muscles attached to the ischial tuberosity and anastomosis with the inferior gluteal. It also supplies an articular branch which enters the hip joint through the acetabular notch, ramifies in the fat of the bottom at the acetabulum, and sends a twig along the ligamentum teres to the head of the femur. Peculiarities. The obturator artery sometimes arises from the main stem or from the posterior trunk of the hypogastric, or it may spring from the superior gluteal artery. Occasionally it arises from the external iliac. In about 2 out of every 7 cases it springs from the inferior epigastric and descends almost vertically to the upper part of the obturator foramen. The artery in its course usually lies in contact with the external iliac vein and on the lateral side of the femoral ring. In such cases it would not be endangered in the operation for strangulated femoral hernia. Occasionally, however, it curves along the free margin of the lanucular ligament, and if in such circumstances a femoral hernia occurred, the vessel would almost completely encircle the neck of the hernial sac, and would be in great danger of being wounded if an operation were performed by strangulation. The internal podental artery, arteria potenta interna, internal pubic artery, is the smaller of the 2 terminal branches of the interior trunk of the hypogastric, and supplies the external organs of generation. Though the course of the artery is the same in the 2 sexes, the vessel is smaller in the female than in the male, and the distortion of its branch is somewhat different. The description of its arrangement in the male will first be given, and subsequently the difference which it presents in the female will be mentioned. The internal podental artery in the male passes downward and outward of the lower border to the greater sciatic foramen, and emerges from the pelvis between the piriformis and the coccygus. It then crosses the ischial spine and enters the perineum through the lesser sciatic foramen. The artery now crosses the obturator internus along the lateral wall of the ischiorectal fossa, being situated about 4 centimeters above the lower margin of the ischial tuberosity. It gradually approaches the margin of the inferior ramus of the ischium and passes forward between the 2 layers of the fascia of the urogenital diaphragm. It then runs forward along the medial margin of the inferior ramus of the pubis, and about 2.25 centimeters behind the pubic accurate ligament it pierces the inferior fascia of the urogenital diaphragm and divides into the dorsal and deep arteries of the penis. Relations. Within the pelvis it lies in front of the piriformis muscle, the sacral plexus of nerves, and the inferior gluteal artery. As it crosses the ischial spine, it is covered by the gluteus maximus and overlapped by the sacrotuberous ligament. Here the podental nerve lies to the medial side, and the nerve of the obturator internus to the lateral side of the vessel. In the perineum it lies on the lateral wall of the ischiorectal fossa, in a canal, Alcock's canal, formed by the splitting of the obturator fascia. It is accompanied by a pair of vena comitantes and the podental nerve. Peculiarities. The internal podental artery is sometimes smaller than usual, or fails to give off 1 or 2 of its usual branches. In such cases the deficiency is supplied by branches derived from an additional vessel, the accessory podental, which generally arises from the internal podental artery before its exit from the greater sciatic foramen. It passes downward along the lower part of the bladder and across the side of the prostate to the root of the penis, where it perforates the urogenital diaphragm and gives off the branches usually derived from the internal prudental artery. The deficiency most frequently met with is that in which the internal prudental ends as the artery of the urethral pulp, the dorsal and deep arteries of the penis being derived from the accessory prudental. The internal prudental artery may also end as the perineal, the artery of the urethral bulb being derived, with the other two branches, from the accessory vessel. Occasionally, the accessory prudental artery is derived from one of the other branches of the hypogastric artery, most frequently the inferior vesicle or the obturator. Branches. The branches of the internal prudental artery are muscular, inferior hemorrhoidal, perineal, artery of the urethral pulp, urethral, deep artery of the penis, dorsal artery of the penis. The muscular branches consist of two sets, one given off in the pelvis, the other as the vessel crosses the ischial spine. The former consists of several small offsets, which supply the levator ani, the obturator internus, the piriformis, the coccygus. The branches given off outside the pelvis are distributed to the adjacent parts of the gluteus maximus and external rotator muscles. The anastomose with branches of the inferior gluteal artery. The inferior hemorrhoidal artery, arteria hemorrhoidalis inferior, arises from the internal prudental as it passes above the ischial tuberosity. Piercing the wall of Alcock's canal, it divides into 2 or 3 branches which cross the ischiorectal fossa and are distributed to the muscles and integument of the anal region, and send offshots around the lower edge of the gluteus maximus to the skin of the buttock. The anastomose with the corresponding vessels of the opposite side, with the superior and the middle hemorrhoidal, and with the perineal artery. The perineal artery, arteria perinei, superficial perineal artery, arises from the internal prudental in front of the preceding branches and turns upward, crossing either over or under the transversus perinei superficialis, and runs downward, parallel to the pubic arch, in the interspace between the bulbocavernosus and the ischiocavernosus, both of which it supplies, and finally divides into several posterior scrotal branches, which are distributed to the skin and dartos tunic of the scrotum. As it crosses the transversus perinei superficialis, it gives off the transverse perineal artery, which runs transversely on the cutaneous surface of the muscle, and anastomosis with the corresponding vessel of the opposite side, and with the perineal and inferior hemorrhoidal arteries. It supplies the transversus perinei superficialis, the structures between the anus and the urethral bulb. The artery of the urethral bulb, arteria bulbi urethrae, is a short vessel of a large caliber which arises from the internal prudental between the 2 layers of fascia of the urogenital diaphragm. It passes medialward, pierces the inferior fascia of the urogenital diaphragm, and gives off branches which ramify in the bulb of the ureter, and in the posterior part of the corpus cavernosum urethrae. It gives off a small branch to the bulbourethral gland. The urethral artery, arteria urethralis, arises a short distance in front of the artery of the urethral bulb. It runs forward and medialward, pierces the inferior fascia of the urogenital diaphragm, and enters the corpus cavernosum urethrae, in which it is continued forward to the glans penis. The deep artery of the penis, arteria profunda penis, artery of the corpus cavernosum, one of the terminal branches of the internal prudental, arises from that vessel, while it is situated between the 2 fasciae of the urogenital diaphragm. It pierces the inferior fascia, and entering the crus penis obliquely, runs forward in the center of the corpus cavernosum penis, to which its branches are distributed. The dorsal artery of the penis, arteria dorsalis penis, ascends between the crus penis and the pubic symphysis, and piercing the inferior fascia of the urogenital diaphragm, passes between the 2 layers of the suspensory ligament of the penis, and runs forward on the dorsum of the penis to the glans, where it divides into 2 branches, which supply the glans and the prepuce. On the penis it lies between the dorsal nerve and the deep dorsal vein, the former being on its lateral side. It supplies the integument and fibrous sheath of the corpus cavernosum penis, sending branches through the sheath to anastomose with the preceding vessel. The internal prudental artery in the female is smaller than in the male. Its origin and course is similar, and there is considerable analogy in the distribution of its branches. The perineal artery supplies the labia putendi, the artery of the bulb supplies the bulbus vestibuli and the erectile tissue of the vagina, and the deep artery of the clitoris supplies the corpus cavernosum clitoris. and the dorsal artery of the clitoris supplies the dorsum of the organ, and ends in the glans and prepubis of the clitoris. The inferior internal gluteal artery, arteria glutea inferior, sciatic artery, the larger of the two terminal branches of the anterior trunk of the hypogastric, is distributed chiefly to the buttock and back of the thigh. It passes down on the sacral plexus of nerves and the piriformis, behind the internal podental artery, the lower part of the greater sciatic foramen, through which it escapes from the pelvis, between the piriformis and the coccygus. It then descends in the interval between the greater trochanter of the femur and tuberosity of the ischium, accompanied by the sciatic and posterior femoral cutaneous nerves, and covered by the gluteus maximus, and is continued down the back of the thigh, supplying the skin and anastomosing with the branches of the perforating arteries. Inside the pelvis it distributes branches to the piriformis, coccygus, and levator ani, some branches which supply the fat around the rectum and occasionally take the place of the middle hemorrhoidal artery, and vesical branches to the fundus of the bladder, vesiculae seminalis, and prostate. Outside the pelvis it gives off the following branches: muscular, coccygeal, committens nervi ischiadici, anastomotic, articular, cutaneous. The muscular branches supply the gluteus maximus, anastomosing with the superior gluteal artery in the substance of the muscle, the external rotators anastomosing with the internal podental artery, and the muscles attached to the tuberosity of the ischium anastomosing with the posterior branch of the obturator and the medial femoral circumflex arteries. The coccygeal branches run medialward, pierce the sacrotuberous ligament, and supply the gluteus maximus, the integument, and other structures on the back of the coccyx. The arteriocommittens nervi ischiadici is a long, slender vessel, which accompanies the sciatic nerve for a short distance. It then penetrates it and runs in its substance to the lower part of the thigh. The anastomotic is directed downward across the external rotators and assists in forming the so-called crucial anastomosis by joining with the first perforating and medial and lateral femoral circumflex arteries. The articular branch, generally derived from the anastomotic, is distributed to the capsule of the hip joint. The cutaneous branches are distributed to the skin of the buttock and back of the thigh. The iliolumbar artery, arteria iliolumbalis, a branch of the posterior trunk of the hypogastric, turns upward behind the obturator nerve and the external iliac vessel to the medial border of the psoas major, behind which it divides into a lumbar and an iliac branch. The lumbar branch, ramus lumbalis, supplies the psoas major and the quadratus lumborum, anastomosis with the last lumbar artery, and sends a small spinal branch through the intervertebral foramen, between the last lumbar vertebra and the sacrum, into the vertebral canal, to supply the cauda equina. The iliac branch, ramus iliacus, descends to supply the iliacus. Some offsets running between the muscle and the bone anastomose with the iliac branches of the obturator. One of these enters an oblique canal to supply the bone, which others run along the crest of the ilium, distributing branches to the gluteal and abdominal muscles, and anastomosing in their course with the superior gluteal, iliac circumflex, and lateral femoral circumflex arteries. The lateral sacral arteries, arteria sacralis lateralis, arise from the posterior division of the hypogastric. There are usually two, a superior and an inferior. The superior, of large size, passes medialward, and after anastomosing with branches from the middle sacral, enters the first or second anterior sacral foramen, supplies branches to the contents of the sacral canal, and, escaping the corresponding posterior sacral foramen, is distributed to skin and muscle of the dorsum of the sacrum, anastomosing with the superior gluteal. The inferior runs obliquely across the front of the piriformis and the sacral nerves of the medial side of the anterior sacral foramina, descends on the front of the sacrum and anastomoses over the coccyx, with the middle sacral and opposite lateral sacral artery. In its course it gives off branches, which enter the anterior sacral foramina. These, after supplying the contents of the sacral canal, escapes by the posterior sacral foramina, and are distributed to the muscles and skin of the dorsal surface of the sacrum, anastomosing with the gluteal arteries. The superior gluteal artery, arteria glutea superior, gluteal artery, is the largest branch of the hypogastric, and appears to be the continuation of the posterior division of that vessel. It is a short artery, which which runs backward between the lumbosacral trunk and the first sacral nerve, and, passing out of the pelvis above the upper border of the piriformis, immediately divides into a superficial and a deep branch. Within the pelvis it gives off a few branches to the iliacus, piriformis, and obturator internus, and just previous to quitting that cavity a nutrient artery which enters the ileum. The superficial branch enters the deep surface of the gluteus maximus, and divides into numerous branches, some of which supply the muscle and anastomose with the inferior gluteal, while others perforate the tendinous origin, and supply the integument covering the posterior surface of the sacrum, anastomosing with the posterior branches of the lateral sacral arteries. Deep branch lies under the gluteus medius, and almost immediately subdivides into two. Of these, the superior division, continuing the original course of the vessel, passes along the upper border of the gluteus minimus to the anterior superior spine of the ilium, anastomosing with the deep iliac circumflex artery and the ascending branch of the lateral femoral circumflex artery. The inferior division crosses the gluteus minimus obliquely to the greater trochanter, distributing branches to the glutei, and anastomosis with the lateral femoral circumflex artery. Some branches pierce the gluteus minimus and supply the hip joint. End of section 23. Recording by Ellie, June 2009.
Section 28 of Gray's Anatomy, Part 3. This is a Librivox recording. All Librivox recordings are in the public domain. For more information or to volunteer, please visit librivox.org. Anatomy of the Human Body, Part 3, by Henry Gray. The External Iliac Artery. A. iliaca externa. The external iliac artery is larger than the hypogastric, and passes obliquely downward and lateralward along the medial border of the soleus major, from the bifurcation of the common iliac to a point beneath the inguinal ligament, midway between the anterior superior spine of the ilium and the symphysis pubis, where it enters the thigh and becomes the femoral artery. Relations. In front and medially, the artery is in relation with the peritoneum, subperitoneal areola tissue, the termination of the ilium, and frequently the variform process on the right side and the sigmoid colon on the left, and a thin layer of fascia derived from the iliac fascia which surrounds the artery and vein. At its origin it is crossed by the ovarian vessels in the female and occasionally by the ureter. The internal spermatic vessels lie for some distance upon it, near its termination, and it is crossed in this situation by the external spermatic branch of the genitofemoral nerve and the deep iliac circumflex vein, the ductus deferens in the male, and the round ligament of the uterus in the female, curve down across its medial side. Behind, it is in relation with the medial border of the soleus major, from which it is separated by the iliac fascia. At the upper part of its course, the external iliac vein lies partly behind it, but lower down lies entirely to its medial side. Laterally it rests against the soleus major, from which it is separated by the iliac fascia. Numerous lymphatic vessels and lymph glands lie on the front and on the medial side of the vessel. Collateral circulation. The principal anastomoses in carrying on the collateral circulation after the application of a ligature to the external iliac are the iliolumbar with the iliac circumflex, the superior gluteal with the lateral femoral circumflex, the obturator with the medial femoral circumflex, the inferior gluteal with the first perforating and circumflex branches of the profunda artery, and the internal pudental with the external pudental. When the obturator arises from the inferior epigastric, it is supplied with blood by branches from either the hypogastric, the lateral sacral, or the internal pudental. The inferior epigastric receives its supply from the internal mammary and the lower intercostal arteries, and from the hypogastric by the antomoses of its branches with the obturator. Branches. Besides several small branches to the psoas major and the neighboring lymph glands, the external iliac gives off two branches of considerable size: inferior epigastric, deep iliac circumflex. The inferior epigastric artery, A. epigastrica inferior, deep epigastric artery, arises from the external iliac, immediately above the inguinal ligament. It curves forward in the subperitoneal tissue and then ascends obliquely along the medial margin of the abdominal inguinal ring. Continuing its course upward, it pierces the transversalis fascia and, passing in front of the linea semicircularis, ascends between the rectus abdominis and the posterior lamella of its sheath. It finally divides into numerous branches which anastomose above the umbilicus, with the superior epigastric branch of the internal mammary and with the lower intercostal arteries. As the inferior epigastric artery passes obliquely upward from its origin, it lies along the lower and medial margins of the abdominal inguinal ring, and behind the commencement of the spermatic cord. The ductus deferens, as it leaves the spermatic cord in the male, and the round ligament of the uterus in the female, winds around the lateral and posterior aspects of the artery. Branches. The branches of the vessel are the external spermatic artery, cremasteric artery, which accompanies the spermatic cord and supplies the cremaster and other coverings of the cord, anastomizing with the external spermatic artery. In the female it is very small and accompanies the round ligament. A pubic branch which runs along the inguinal ligament and then descends along the medial margin of the femoral ring to the back of the pubis, and there anastomoses with the pubic branch of the obturator artery. Muscular branches, some of which are distributed to the abdominal muscles and the peritoneum, astomosing with the iliac circumflex and lumbar arteries. Branches which perforate the tendon of the obliquus externus and supply the integument, astomosing with branches of the superficial epigastric. Peculiarities. The origin of the inferior epigastric may take place from any part of the external iliac between the inguinal ligament and a point 6 centimeters above it, or it may arise below this ligament from the femoral. It frequently springs from the external iliac by a common trunk with the obturator. Sometimes it arises from the obturator, the latter vessel being furnished by the hypogastric, or it may be formed of two branches, one derived from the external iliac, the other from the hypogastric. The deep iliac circumflex artery, A. circumflexa ilium profunda, arises from the lateral aspect of the external iliac, nearly opposite the inferior epigastric artery. It ascends obliquely lateralward behind the inguinal ligament, contained in a fibrous sheath formed by the junction of the transversalis fascia and iliac fascia, to the anterior superior iliac spine, where it antomoses with the ascending branch of the lateral femoral circumflex artery. It then pierces the transversalis fascia and passes along the inner lip of the crest of the ilium to about its middle, where it perforates the transversus, and runs backward between that muscle and the obliquus internus, to anastomose with the iliolumbar and superior gluteal arteries. Opposite the anterior superior spine of the ilium, it gives off a large branch, which ascends between the obliquus internus and transversus muscles, supplying them anastomosing with the lumbar and inferior epigastric arteries. Note: Sir Astley Cooper describes in Guy's Hospital Reports, Volume 1, the dissection of a limb 18 years after successful ligature of the external iliac artery. End note. End of section 28. Recording by David Lawrence. July 1st, 2009, in Brampton, Ontario.
Section 29 of Gray's Anatomy, Part 3. This is a Librivox recording. All Librivox recordings are in the public domain. For more information or to volunteer, please visit LibriVox.org. Anatomy of the Human Body, Part 3, by Henry Gray. The arteries of the lower extremity. The artery which supplies the greater part of the lower extremity is the direct continuation of the external iliac. It runs as a single trunk from the inguinal ligament to the lower border of the popliteus, where it divides into two branches, the anterior and posterior tibial. The upper part of the main trunk is named the femoral, the lower part the popliteal. The femoral artery, A femoralis. The femoral artery begins immediately behind the inguinal ligament, midway between the anterior superior spine of the ilium and the symphysis pubis, and passes down the front and medial side of the thigh. It ends at the junction of the middle with the lower third of the thigh, where it passes through an opening in the adductor magnus to become the popliteal artery. The vessel at the upper part of the thigh lies in front of the hip joint. In the lower part of its course it lies to the medial side of the body of the femur, and between these two parts, where it crosses the angle between the head and body, the vessel is some distance from the bone. The first 4 centimeters of the vessel is enclosed, together with the femoral vein, in a fibrous sheath, the femoral sheath. In the upper third of the thigh, the femoral artery is contained in the femoral triangle, Scarpa's triangle, and in the middle third of the thigh in the adductor canal, Hunter's canal. The femoral sheath, crural sheath, is formed by a prolongation downward behind the inguinal ligament of the fasciae which line the abdomen, the transversalis fascia being continued down in front of the femoral vessels and the iliac fascia behind them. The sheath assumes the form of a short funnel, the wide end of which is directed upward, while the lower narrow end fuses with the fascial investment of the vessels about 4 centimeters below the inguinal ligament. It is strengthened in front by a band termed the deep crural arch. The lateral wall of the sheath is vertical and is perforated by the lumboinguinal nerve. The medial wall is directed obliquely downward and lateralward, and is pierced by the great saphenous vein and by some lymphatic vessels. The sheath is divided by 2 vertical partitions which stretch between its anterior and posterior walls. The lateral compartment contains the femoral artery, and the intermediate the femoral vein, while the medial and smallest compartment is named the femoral canal, and contains some lymphatic vessels and a lymph gland embedded in a small amount of areolar tissue. The femoral canal is conical and measures about 1.25 centimeters in length. Its base, directed upward and named the femoral ring, is oval in form, its long diameter being directed transversely and measuring about 1.25 centimeters. The femoral ring is bounded in front by the inguinal ligament, behind by the pectineus, covered by the pectineal fascia, medially by the crescentic base of the lacunar ligament, and laterally by the fibrous septum on the medial side of the femoral vein. The spermatic cord in the male and the round ligament of the uterus in the female lie immediately above the anterior margin of the ring, while the inferior epigastric vessels are close to its upper and lateral angle. The femoral ring is closed by a somewhat condensed portion of the extraperitoneal fatty tissue named the septum femorale, crural septum, the abdominal surface of which supports a small lymph gland and is covered by the parietal layer of the peritoneum. The septum femorale is pierced by numerous lymphatic vessels passing from the deep inguinal to the external iliac lymph glands, and the parietal peritoneum immediately above it presents a slight depression named the femoral fossa. The femoral triangle, trigonum femorale, Scarpa's triangle, corresponds to the depression seen immediately below the fold of the groin. Its apex is directed downward, and the sides are formed laterally by the medial margin of the sartorius, medially by the medial margin of the adductor longus, and above by the inguinal ligament. The floor of the space is formed from its lateral to its medial side by the iliacus, psoas major, pectineus, in some cases a small part of the adductor brevis, and the adductor longus, and it is divided into 2 nearly equal parts by the femoral vessels, which extend from near the middle of its base to its apex, the artery giving off in this situation its superficial and profunda branches, the vein receiving the deep femoral and great saphenous tributaries. On the lateral side of the femoral artery is the femoral nerve dividing into its branches. Besides the vessels and nerves, this space contains some fat and lymphatics. The adductor canal, Canalis adductorius, Hunter's canal, is an aponeurotic tunnel in the middle third of the thigh, extending from the apex of the femoral triangle to the opening in the adductor magnus. It is bounded in front and laterally by the vastus medialis, behind by the adductores longus and magnus, and is covered in by a strong aponeurosis which extends from the vastus medialis across the femoral vessels to the adductores longus and magnus. Lying on the aponeurosis is the sartorius muscle. The canal contains the femoral artery and vein, the saphenous nerve, and the nerve to the vastus medialis. Relations of the femoral artery. In the femoral triangle the artery is superficial. In front of it are the skin and superficial fascia, the superficial subinguinal lymph glands, the superficial iliac circumflex vein, the superficial layer of the fascia lata, and the anterior part of the femoral sheath. The lumboinguinal nerve courses for a short distance within the lateral compartment of the femoral sheath, and lies at first in front and then lateral to the artery. Near the apex of the femoral triangle the medial branch of the anterior femoral cutaneous nerve crosses the artery from its lateral to its medial side. Behind the artery are the posterior part of the femoral sheath, the pectineal fascia, the medial part of the tendon of the psoas major, the pectineus, and the adductor longus. The artery is separated from the capsule of the hip joint by the tendon of the psoas major, from the pectineus by the femoral vein and profunda vessels, and from the adductor longus by the femoral vein. The nerve to the pectineus passes medialward behind the artery. On the lateral side of the artery, but separated from it by some fibers of the psoas major, is the femoral nerve. The femoral vein is on the medial side of the upper part of the artery, but is behind the vessel in the lower part of the femoral triangle. In the adductor canal the femoral artery is more deeply situated, being covered by the integument, the superficial and deep fasciae, the sartorius, and the fibrous roof of the canal. The saphenous nerve crosses from its lateral to its medial side. Behind the artery are the adductores longus and magnus. In front and lateral to it is the vastus medialis. The femoral vein lies posterior to the upper part and lateral to the lower part of the artery. Peculiarities. Several cases are recorded in which the femoral artery divided into two trunks below the origin of the profunda, and became reunited near the opening in the adductor magnus, so as to form a single popliteal artery. One occurred in a patient who was operated upon for popliteal aneurysm. A few cases have been recorded in which the femoral artery was absent, its place being supplied by the inferior gluteal artery which accompanied the sciatic nerve to the popliteal fossa. The external iliac in these cases was small and terminated in the profunda. The femoral vein is occasionally placed along the medial side of the artery throughout the entire extent of the femoral triangle, or it may be split so that a large vein is placed on either side of the artery for a greater or lesser distance. Collateral circulation. After ligature of the femoral artery, the main channels for carrying on the circulation are the anastomoses between 1. The superior and inferior gluteal branches of the hypogastric, with the medial and lateral femoral circumflex and first perforating branches of the profunda femoris. 2. The obturator branch of the hypogastric, with the medial femoral circumflex of the profunda. 3. The internal pudendal of the hypogastric, with the superficial and deep external pudendal of the femoral. 4. The deep iliac circumflex of the external iliac, with the lateral femoral circumflex of the profunda, and the superficial iliac circumflex of the femoral, and 5. The inferior gluteal of the hypogastric, with the perforating branches of the profunda. Branches. The branches of the femoral artery are superficial epigastric, superficial iliac circumflex, superficial external pudendal, deep external pudendal, muscular, profunda femoris, highest genicular. The superficial epigastric artery, A. epigastrica superficialis, arises from the front of the femoral artery about 1 cm below the inguinal ligament, and passing through the femoral sheath and the fascia cribrosa turns upward in front of the inguinal ligament, and ascends between the two layers of the superficial fascia of the abdominal wall nearly as far as the umbilicus. It distributes branches to the superficial subinguinal lymph glands, the superficial fascia, and the integument. It anastomoses with branches of the inferior epigastric and with its fellow of the opposite side. The superficial iliac circumflex artery, A. circumflexa ilium superficialis, the smallest of the cutaneous branches, arises close to the preceding, and piercing the fascia lata runs lateralward parallel with the inguinal ligament as far as the crest of the ilium. It divides into branches which supply the integument of the groin, the superficial fascia, and the superficial subinguinal lymph glands, anastomosing with the deep iliac circumflex, the superior gluteal and lateral femoral circumflex arteries. The superficial external pudendal artery, A. pudenda externa superficialis, superficial external pudic artery, arises from the medial side of the femoral artery, close to the preceding vessels, and after piercing the femoral sheath and fascia cribrosa courses medialward across the spermatic cord, or round ligament in the female, to be distributed to the integument on the lower part of the abdomen, the penis and scrotum in the male, and the labium majus in the female, anastomosing with branches of the internal pudendal. The deep external pudendal artery, A. pudenda externa profunda, deep external pudic artery, more deeply seated than the preceding, passes medialward across the pectineus and the adductor longus muscles. It is covered by the fascia lata, which it pierces at the medial side of the thigh, and is distributed in the male to the integument of the scrotum and perineum, in the female to the labium majus. Its branches anastomose with the scrotal or labial branches of the perineal artery. Muscular branches, rami musculares, are supplied by the femoral artery to the sartorius, vastus medialis, and adductores. The profunda femoris artery, A. profunda femoris, deep femoral artery, is a large vessel arising from the lateral and back part of the femoral artery, from 2 to 5 centimeters below the inguinal ligament. At first it lies lateral to the femoral artery, it then runs behind it and the femoral vein to the medial side of the femur, and passing downward behind the adductor longus, ends at the lower third of the thigh in a small branch, which pierces the adductor magnus and is distributed on the back of the thigh to the hamstring muscles. The terminal part of the profunda is sometimes named the fourth perforating artery. Relations. Behind it, from above downward, are the iliacus, pectineus, adductor brevis, and adductor magnus. In front it is separated from the femoral artery by the femoral and profunda veins above, and by the adductor longus below. Laterally, the origin of the vastus medialis intervenes between it and the femur. Peculiarities. This vessel sometimes arises from the medial side and more rarely from the back of the femoral artery. But a more important peculiarity from a surgical point of view is that relating to the height at which the vessel arises. In 3/4 of a large number of cases it arose from 2.25 to 5 centimeters below the inguinal ligament. In a few cases the distance was less than 2.25 centimeters, more rarely opposite the ligament, and in one case above the inguinal ligament from the external iliac. Occasionally the distance between the origin of the vessel and the inguinal ligament exceeds 5 centimeters. Branches. The profunda gives off the following branches: lateral femoral circumflex, medial femoral circumflex, perforating, muscular. The lateral femoral circumflex artery, A. circumflexa femoris lateralis, external circumflex artery, arises from the lateral side of the profunda, passes horizontally between the divisions of the femoral nerve, and behind the sartorius and rectus femoris, and divides into ascending, transverse, and descending branches. The ascending branch passes upward beneath the tensor fasciae lati to the lateral aspect of the hip, and anastomoses with the terminal branches of the superior gluteal and deep iliac circumflex arteries. The descending branch runs downward behind the rectus femoris upon the vastus lateralis to which it gives offsets. One long branch descends in the muscle as far as the knee and anastomoses with the superior lateral genicular branch of the popliteal artery. It is accompanied by the branch of the femoral nerve to the vastus lateralis. The transverse branch, the smallest, passes lateralward over the vastus intermedius, pierces the vastus lateralis, and winds around the femur just below the greater trochanter, anastomosing on the back of the thigh with the medial femoral circumflex, inferior gluteal, and first perforating arteries. The medial femoral circumflex artery, A. circumflexa femoris medialis, internal circumflex artery, arises from the medial and posterior aspect of the profunda, and winds around the medial side of the femur, passing first between the pectineus and psoas major, and then between the obturator externus and the adductor brevis. At the upper border of the adductor brevis it gives off 2 branches. One is distributed to the adductores, the gracilis and obturator externus, and anastomoses with the obturator artery. The other descends beneath the adductor brevis to supply it and the adductor magnus. The continuation of the vessel passes backward and divides into superficial, deep, and acetabular branches. The superficial branch appears between the quadratus femoris and upper border of the adductor magnus, and anastomoses with the inferior gluteal, lateral femoral circumflex, and first perforating arteries. Crucial anastomoses. The deep branch runs obliquely upward upon the tendon of the obturator externus and in front of the quadratus femoris toward the trochanteric fossa, where it anastomoses with twigs from the gluteal arteries. The acetabular branch arises opposite the acetabular notch, and enters the hip joint beneath the transverse ligament in company with an articular branch from the obturator artery. It supplies the fat in the bottom of the acetabulum and is continued along the round ligament to the head of the femur. The perforating arteries, usually 3 in number, are so named because they perforate the tendon of the adductor magnus to reach the back of the thigh. They pass backward close to the linea aspera of the femur, under cover of small tendinous arches in the muscle. The first is given off above the adductor brevis, the second in front of that muscle, and the third immediately below it. The first perforating artery, A perforans prima, passes backward between the pectineus and adductor brevis. Sometimes it perforates the latter. It then pierces the adductor magnus close to the linea aspera. It gives branches to the adductores brevis and magnus, biceps femoris, and gluteus maximus, and anastomoses with the inferior gluteal, medial, and lateral femoral circumflex, and second perforating arteries. The second perforating artery, A perforans secunda, larger than the first, pierces the tendons of the adductores brevis and magnus, and divides into ascending and descending branches which supply the posterior femoral muscles, anastomosing with the first and third perforating. The second artery frequently arises in common with the first. The nutrient artery of the femur is usually given off from the second perforating artery. When 2 nutrient arteries exist, they usually spring from the first and third perforating vessels. The third perforating artery, A perforans tertia, is given off below the adductor brevis. It pierces the adductor magnus and divides into branches which supply the posterior femoral muscles, anastomosing above with the higher perforating arteries, and below with the terminal branches of the profunda and the muscular branches of the popliteal. The nutrient artery of the femur may arise from this branch. The termination of the profunda artery, already described, is sometimes termed the fourth perforating artery. Numerous muscular branches arise from the profunda. Some of these end in the adductores, others pierce the adductor magnus, give branches to the hamstrings, and anastomose with the medial femoral circumflex artery and with the superior muscular branches of the popliteal. The highest genicular artery, A genus suprema, anastomotica magna artery, arises from the femoral just before it passes through the opening in the tendon of the adductor magnus, and immediately divides into a saphenous and a musculo-articular branch. The saphenous branch pierces the aponeurotic covering of the adductor canal and accompanies the saphenous nerve to the medial side of the knee. It passes between the sartorius and gracilis, and piercing the fascia lata, is distributed to the integument of the upper and medial part of the leg, anastomosing with the medial inferior genicular artery. The musculo-articular branch descends in the substance of the vastus medialis and in front of the tendon of the adductor magnus to the medial side of the knee, where it anastomoses with the medial superior genicular artery and anterior recurrent tibial artery. A branch from this vessel crosses above the patellar surface of the femur, forming an anastomotic arch with the lateral superior genicular artery, and supplying branches to the knee joint. End of section 29.
Section number 30 of Gray's Anatomy, Part 3. This is a Librivox recording. All Librivox recordings are in the public domain. For more information or to volunteer, please visit LibriVox.org. Recording by Marianne Coleman Hipkins. The popliteal fossa, the popliteal artery. 6b. The popliteal fossa. Boundaries. The popliteal fossa, or space, is a lozenge-shaped space at the back of the knee joint. Laterally, it is bounded by the biceps femoris above and by the plantaris and the lateral head of the gastrocnemius below. Medially, it is limited by the semitendinosus and semimembranosus above, and by the medial head of the gastrocnemius below. The floor is formed by the popliteal surface of the femur, the oblique popliteal ligament of the knee joint, the upper end of the tibia, and the fascia covering the popliteus. The fossa is covered in by the fascia lata. Contents. The popliteal fossa contains the popliteal vessels, the tibial and the common peroneal nerves, the termination of the small saphenous vein, the lower part of the posterior femoral cutaneous nerve, the articular branch from the obturator nerve, a few small lymph glands, and a considerable quantity of fat. The tibial nerve descends through the middle of the fossa, lying under the deep fascia, and crossing the vessels posteriorly from the lateral to the medial side. The common peroneal nerve descends on the lateral side of the upper part of the fossa, close to the tendon of the biceps femoris. On the floor of the fossa are the popliteal vessels, the vein being superficial to the artery and united to it by dense areolar tissue. The vein is a thick-walled vessel, and lies at first lateral to the artery, and then crosses it posteriorly to gain its medial side below. Sometimes it is double, the artery lying between the two veins, which are usually connected by short transverse branches. The articular branch from the obturator nerve descends upon the artery to the knee joint. The popliteal lymph glands, 6 or 7 in number, are embedded in the fat. One lies beneath the popliteal fascia, near the termination of the external saphenous vein, another between the popliteal artery and the back of the knee joint, while the others are placed at the sides of the popliteal vessel. Arising from the artery and passing off from it at right angles are its genicular branches. 6c. The popliteal artery. A. Poplitea. The popliteal artery is the continuation of the femoral and crosses through the popliteal fossa. It extends from the opening in the abductor magnus at the junction of the middle and lower thirds of the thigh, downward and lateralward, to the intercondyloid fossa of the femur, and then vertically downward to the lower border of the popliteus, where it divides into anterior and posterior tibial arteries. Relations. In front of the artery from above downward are the popliteal surface of the femur, which is separated from the vessel by some fat, the back of the knee joint and the fascia covering the popliteus. Behind, it is overlapped by the semimembranosus above and is covered by the gastrocnemius and plantaris below. In the middle part of its course the artery is separated from the integument and fasciae by a quantity of fat, and is crossed from the lateral to the medial side by the tibial nerve and the popliteal vein, the vein being between the nerve and the artery and closely adherent to the latter. On its lateral side above are the biceps femoris, the tibial nerve, the popliteal vein, and the lateral condyle of the femur. Below, the plantaris and the lateral head of the gastrocnemius. On its medial side above are the semimembranosus and the medial condyle of the femur. Below the tibial nerve, the popliteal vein, and the medial head of the gastrocnemius. The relations of the popliteal lymph glands to the artery are described above. Peculiarities in point of division. Occasionally the popliteal artery divides into its terminal branches, opposite the knee joint. The anterior tibial, under these circumstances, usually passes in front of the popliteus. Unusual branches. The artery sometimes divides into the anterior tibial and peroneal, the posterior tibial being wanting or very small. Occasionally it divides into 3 branches, the anterior and posterior tibial and peroneal. Branches. The branches of the popliteal artery are muscular, superior, sural; cutaneous, medial superior genicular; lateral superior genicular; middle genicular, medial inferior genicular; lateral inferior genicular. The superior muscular branches, 2 or 3 in number, arise from the upper part of the artery and are distributed to the lower parts of the adductor magnus and hamstring muscles, anastomosing with the terminal part of the profunda femoris. The sural arteries, AA suralis, inferior muscular arteries, are 2 large branches which are distributed to the gastrocnemius, soleus, and plantaris. They arise from the popliteal artery opposite the knee joint. The cutaneous branches arise either from the popliteal artery or from some of its branches. They descend between the two heads of the gastrocnemius, and piercing the deep fascia are distributed to the skin of the back of the leg. One branch usually accompanies the small saphenous vein. The superior genicular arteries, A.A. genu superioris, superior articular arteries, 2 in number, arise one on either side of the popliteal and wind round the femur immediately above its condyles to the front of the knee joint. The medial superior genicular runs in front of the semimembranosus and semitendinosus, above the medial head of the gastrocnemius, and passes beneath the tendon of the adductor magnus. It divides into 2 branches, one of which supplies the vastus medialis, anastomosing with the highest genicular and medial inferior genicular arteries. The other ramifies close to the surface of the femur, supplying it and the knee joint, and anastomosing with the lateral superior genicular artery. The medial superior genicular artery is frequently of small size, a condition which is associated with an increase in the size of the highest genicular. The lateral superior genicular passes above the lateral condyle of the femur, beneath the tendon of the biceps femoris, and divides into a superficial and a deep branch. The superficial branch supplies the vastus lateralis, and anastomoses with the descending branch of the lateral femoral circumflex and the lateral inferior genicular arteries. The deep branch supplies the lower part of the femur and knee joint, and forms an anastomotic arch across the front of the bone, with the highest genicular and the medial inferior genicular arteries. The middle genicular artery, A. genu media, azygos articular artery, is a small branch arising opposite the back of the knee joint. It pierces the oblique popliteal ligament and supplies the ligaments and synovial membrane in the interior of the articulation. The inferior genicular arteries, A.A. genu inferioris, inferior articular arteries, 2 in number, arise from the popliteal beneath the gastrocnemius. The medial inferior genicular first descends along the upper margins of the popliteus, to which it gives branches. It then passes below the medial condyle of the tibia, beneath the tibial collateral ligament, at the anterior border of which it ascends to the front and medial side of the joint, to supply the upper end of the tibia and the articulation of the knee, anastomosing with the lateral inferior and medial superior genicular arteries. The lateral inferior genicular runs lateralward along the head of the fibula to the front of the knee joint, passing in its course beneath the lateral head of the gastrocnemius, the fibular collateral ligament, and the tendon of the biceps femoris. It ends by dividing into branches which anastomose with the medial inferior and lateral superior genicular arteries, and with the anterior recurrent tibial artery. The anastomosis around the knee joint, around and above the patella, and on the contiguous ends of the femur and tibia, is an intricate network of vessels forming a superficial and a deep plexus. The superficial plexus is situated between the fascia and skin around the patella, and forms 3 well-defined arches, one above the upper border of the patella in the loose connective tissue over the quadriceps femoris. The other 2, below the level of the patella, are situated in the fat behind the ligamentum patellae. The deep plexus, which forms a close network of vessels, lies on the lower end of the femur and upper end of the tibia, around their articular surfaces, and sends numerous offsets into the interior of the joint. The arteries which form this plexus are the 2 medial and the 2 lateral genicular branches of the popliteal, the highest genicular, the descending branch of the lateral femoral circumflex, and the anterior recurrent tibial. End of section number 30. Recording by Marianne Coleman Hipkins. www.thisvoiceforyou.com.
Section 31 of Gray's Anatomy, Part 3. This is a Librivox recording. All Librivox recordings are in the public domain. For more information or to volunteer, please visit librivox.org. Anatomy of the Human Body, Part 3, by Henry Gray. The anterior tibial artery, Arteria tibialis anterior. The anterior tibial artery commences at the bifurcation of the popliteal, at the lower border of the popliteus, passes forward between the two heads of the tibialis posterior, and through the aperture above the upper border of the interosseous membrane to the deep part of the front of the leg. It here lies close to the medial side of the neck of the fibula. It then descends on the anterior surface of the interosseous membrane, gradually approaching the tibia. At the lower part of the leg it lies on this bone, and then on the front of the ankle joint, where it is more superficial, and becomes the dorsalis pedis. Relations. In the upper 2/3 of its extent, the anterior tibial artery rests upon the interosseous membrane, in the lower third upon the front of the tibia and the anterior ligament of the ankle joint. In the upper third of its course it lies between the tibialis anterior and the extensor digitorum longus. In the middle third between the tibialis anterior and extensor hallucis longus. At the ankle it is crossed from the lateral to the medial side by the tendon of the extensor hallucis longus, and lies between it and the first tendon of the extensor digitorum longus. It is covered in the upper 2/3 of its course by the muscles which lie on either side of it, and by the deep fascia; in the lower third by the integument and fascia, and the transverse and cruciate crural ligaments. The anterior tibial artery is accompanied by a pair of venae comitantes, which lie one on either side of the artery. The deep peroneal nerve, coursing through the lateral side of the neck of the fibula, comes into relation with the lateral side of the artery shortly after it has reached the front of the leg. About the middle of the leg the nerve is in front of the artery. At the lower part it is generally again on the lateral side. Peculiarities in size. This vessel may be diminished in size, may be deficient to a greater or less extent, or may be entirely wanting, its place being supplied by perforating branches from the posterior tibial, or by the perforating branch of the peroneal artery. Course. The artery occasionally deviates toward the fibular side of the leg, regaining its usual position at the front of the ankle. In rare instances, the vessel has been found to approach the surface in the middle of the leg, being covered merely by the integument and fascia below that point. Branches. The branches of the anterior tibial artery are: posterior tibial recurrent, fibular, anterior tibial recurrent, muscular, anterior medial malleolar, anterior lateral malleolar. The posterior tibial recurrent artery, arteria recurrens tibialis posterior, an inconstant branch, is given off from the anterior tibial before that vessel passes through the interosseous space. It ascends in front of the popliteus, which it supplies and anastomoses with the inferior genicular branches of the popliteal artery, giving an offset to the tibio-fibular joint. The fibular artery is sometimes derived from the anterior tibial, sometimes from the posterior tibial. It passes lateralward around the neck of the fibula through the soleus, which it supplies, and ends in the substance of the peroneus longus. The anterior tibial recurrent artery, arteria recurrens tibialis anterior, arises from the anterior tibial as soon as that vessel has passed through the interosseous space. It ascends in the tibialis anterior, ramifies on the front and sides of the knee joint, and assists in the formation of the patellar plexus by anastomosing with the genicular branches of the popliteal and with the highest genicular artery. The muscular branches, rami muscularis, are numerous. They are distributed to the muscles which lie on either side of the vessel, some piercing the deep fascia to supply the integument, others passing through the interosseous membrane, and anastomosing with branches of the posterior tibial and peroneal arteries. The anterior medial malleolar artery, Arteria malleolaris anterior medialis, internal malleolar artery, arises about 5 cm above the ankle joint and passes behind the tendons of the extensor hallucis longus and tibialis anterior to the medial side of the ankle, upon which it ramifies, anastomosing with branches of the posterior tibial and medial plantar arteries and with the medial calcaneal from the posterior tibial. The anterior lateral malleolar artery, Arteria malleolaris anterior lateralis, external malleolar artery, passes beneath the tendons of the extensor digitorum longus and peroneus tertius and supplies the lateral side of the ankle, anastomosing with the perforating branch of the peroneal artery and with ascending twigs from the lateral tarsal artery. The arteries around the ankle joint anastomose freely with one another and form networks below the corresponding malleoli. The medial malleolar network is formed by the anterior medial malleolar branch of the anterior tibial, the medial tarsal branches of the dorsalis pedis, the posterior medial malleolar and medial calcaneal branches of the posterior tibial, and branches from the medial plantar artery. The lateral malleolar network is formed by the anterior lateral malleolar branch of the anterior tibial, the lateral tarsal branch of the dorsalis pedis, the perforating and the lateral calcaneal branches of the peroneal, and twigs from the lateral plantar artery. The arteria dorsalis pedis. Dorsalis pedis artery. The arteria dorsalis pedis, the continuation of the anterior tibial, passes forward from the ankle joint along the tibial side of the dorsum of the foot to the proximal part of the first intermetatarsal space, where it divides into two branches, the first dorsal metatarsal and the deep plantar. Relations. This vessel, in its course forward, rests upon the front of the articular capsule of the ankle joint, the talus, navicular, and second cuneiform bones, and the ligaments connecting them, being covered by the integument, fascia, and cruciate ligament, and crossed near its termination by the first tendon of the extensor digitorum brevis. On its tibial side is the tendon of the extensor hallucis longus. On its fibular side, the first tendon of the extensor digitorum longus, and the termination of the deep peroneal nerve. It is accompanied by two veins. Peculiarities in size. The dorsal artery of the foot may be larger than usual to compensate for a deficient plantar artery, or its terminal branches to the toes may be absent, the toes then being supplied by the medial plantar. Or its place may be taken altogether by a large perforating branch of the peroneal artery. Position. This artery frequently curves lateralward, lying lateral to the line between the middle of the ankle and the back part of the first interosseous space. Branches. The branches of the arteria dorsalis pedis are lateral tarsal, medial tarsal, arcuate, first dorsal metatarsal, deep plantar. The lateral tarsal artery, Arteria torsea lateralis, tarsal artery, arises from the dorsalis pedis as that vessel crosses the navicular bone. It passes in an arched direction lateralward, lying upon the tarsal bones, and covered by the extensor digitorum brevis. It supplies this muscle and the articulations of the tarsus, and anastomoses with branches of the arcuate, anterior lateral malleolar, and lateral plantar arteries, and with the perforating branch of the peroneal artery. The medial tarsal arteries, Arteriae torsiae medialis, are 2 or 3 small branches which ramify on the medial border of the foot and join the medial malleolar network. The arcuate artery, Arteria arcuata, metatarsal artery, arises a little anterior to the lateral tarsal artery. It passes lateralward over the bases of the metatarsal bones, beneath the tendons of the extensor digitorum brevis, its direction being influenced by its point of origin, and it anastomoses with the lateral tarsal and lateral plantar arteries. This vessel gives off the second, third, and fourth dorsal metatarsal arteries, which run forward upon the corresponding interossi dorsalis. In the clefts between the toes, each divides into two dorsal digital branches for the adjoining toes. At the proximal parts of the interosseous spaces, these vessels receive the posterior perforating branches from the plantar arch, and at the distal parts of the spaces they are joined by the anterior perforating branches from the plantar metatarsal arteries. The fourth dorsal metatarsal artery gives off a branch which supplies the lateral side of the fifth toe. The first dorsal metatarsal artery, arteria dorsalis hallucis, runs forward on the first interosseous dorsalis, and at the cleft between the first and second toes divides into two branches, one of which passes between the tendon of the extensor hallucis longus and is distributed to the medial border of the great toe. The other bifurcates to supply the adjoining sides of the great and second toes. The deep plantar artery, ramus plantaris profundus, communicating artery, descends into the sole of the foot between the two heads of the first interosseous dorsalis, and unites with the termination of the lateral plantar artery to complete the plantar arch. It sends a branch along the medial side of the great toe, and is continued forward along the first interosseous space as the first plantar metatarsal artery, which bifurcates for the supply of the adjacent sides of the great and second toes. End of section 31. Section 32 of Gray's Anatomy Part 3. This is a Librivox recording. All Librivox recordings are in the public domain. For more information or to volunteer, please visit librivox.org. Anatomy of the Human Body, Part 3, by Henry Gray. The posterior tibial artery, Arteria tibialis posterior. The posterior tibial artery begins at the lower border of the popliteus, opposite the interval between the tibia and fibula. It extends obliquely downward, and, as it descends, it approaches the tibial side of the leg, lying behind the tibia, and in the lower part of its course is situated midway between the medial malleolus and the medial process of the calcaneal tuberosity. Here it divides beneath the origin of the adductor hallucis into the medial and lateral plantar arteries. Relations. The posterior tibial artery lies successively upon the tibialis posterior, the flexor digitorum longus, the tibia, and the back of the ankle joint. It is covered by the deep transverse fascia of the leg, which separates it above from the gastrocnemius and soleus. At its termination it is covered by the abductor hallucis. In the lower third of the leg, where it is more superficial, it is covered only by the integument and fascia, and runs parallel with the medial border of the tendo calcaneus. It is accompanied by two veins, and by the tibial nerve, which lies at first to the medial side of the artery, but soon crosses it posteriorly, and is in the greater part of its course on its lateral side. Behind the medial malleolus the tendons, blood vessels, and nerve are arranged, under cover of the lacineate ligament, in the following order from the medial to the lateral side. 1. The tendons of the tibialis posterior and flexor digitorum longus lying in the same groove behind the malleolus, the former being the more medial. Next is the posterior tibial artery, with a vein on either side of it, and lateral to the vessels is the tibial nerve. About 1.25 cm nearer the heel is the tendon of the flexor hallucis longus. Peculiarities in size. The posterior tibial is not infrequently smaller than usual, or absent, its place being supplied by a large peroneal artery, which either joins the small posterior tibial artery or continues alone to the sole of the foot. Branches. The branches of the posterior tibial artery are peroneal, nutrient, muscular, posterior medial mallular, communicating, medial calcaneal. The peroneal artery, arteria peroneia, is deeply seated on the back of the fibular side of the leg. It arises from the posterior tibial about 2.5 cm below the lower border of the popliteus, passes obliquely toward the fibula, and then descends along the medial side of that bone, contained in a fibrous canal between the tibialis posterior and the flexor hallucis longus, or in the substance of the latter muscle. It then runs behind the tibio-fibular syndesmosis and divides into lateral calcaneal branches which ramify on the lateral and posterior surfaces of the calcaneus. It is covered, in the upper part of its course, by the soleus and deep transverse fascia of the leg, below by the flexor hallucis longus. Peculiarities in origin. The peroneal artery may arise 7 to 8 cm below the popliteus, or from the posterior tibial high up, or even from the popliteal. Its size is more frequently increased than diminished, and then it either reinforces the posterior tibial by its junction with it, or altogether takes the place of the posterior tibial in the lower part of the leg and foot, the latter vessel only existing as a short muscular branch. In those rare cases where the peroneal artery is smaller than usual, a branch from the posterior tibial supplies its place, and a branch from the anterior tibial compensates for the diminished anterior peroneal artery. In one case the peroneal artery was entirely wanting. Branches. The branches of the peroneal are muscular, nutrient, perforating, communicating, lateral calcaneal. Muscular branches. The peroneal artery, in its course, gives off branches to the soleus, tibialis posterior, flexor hallucis longus, and peronae. The nutrient artery, arteria nutritia fibulae, supplies the fibula and is directed downward. The perforating branch, ramus perforans, anterior peroneal artery, pierces the interosseous membrane about 5 cm above the lateral malleolus to reach the front of the leg, where it anastomoses with the anterior lateral mallular. It then passes down in front of the tibio-fibular syndesmosis, gives branches to the tarsus, and anastomoses with the lateral tarsal. The perforating branch is sometimes enlarged, and takes the place of the dorsalis pedis artery. The communicating branch, ramus communicans, is given off from the peroneal about 2.5 cm from its lower end, and joins the communicating branch of the posterior tibial. The lateral calcaneal, ramus calcaneus lateralis, external calcaneal, are the terminal branches of the peroneal artery. They pass to the lateral side of the heel, and communicate with the lateral mallular, and, on the back of the heel, with the medial calcaneal arteries. The nutrient artery, arteria nutritia tibiae, of the tibia, arises from the posterior tibial near its origin, and after supplying a few muscular branches, enters the nutrient canal of the bone, which it traverses obliquely from above downward. This is the largest nutrient artery of bone in the body. The muscular branches of the posterior tibial are distributed to the soleus and deep muscles along the back of the leg. The posterior medial mallular artery, arteria mallularis posterior medialis, internal mallular artery, is a small branch which winds around the tibial malleolus and ends in the medial mallular network. The communicating branch, ramus communicans, runs transversely along the back of the tibia about 5 cm above its lower end beneath the flexor hallucis longus and joins the communicating branch of the peroneal. The medial calcaneal, rami calcanei medialis, internal calcaneal, are several large arteries which arise from the posterior tibial just before its division. They pierce the laciniate ligament and are distributed to the fat and integument behind the tendo calcaneus and about the heel, and to the muscles on the tibial side of the sole, anastomosing with the peroneal and medial malleolar, and on the back of the heel with the lateral calcaneal arteries. The medial plantar artery, arteria plantaris medialis, internal plantar artery, much smaller than the lateral, passes forward along the medial side of the foot. It is at first situated above the abductor hallucis, and then between it and the flexor digitorum brevis, both of which it supplies. At the base of the first metatarsal bone, where it is much diminished in size, it passes along the medial border of the first toe, anastomosing with the first dorsal metatarsal artery. Small superficial digital branches accompany the digital branches of the medial plantar nerve and join the plantar metatarsal arteries of the first 3 spaces. The lateral plantar artery, arteria plantaris lateralis, external plantar artery, much larger than the medial, passes obliquely lateralward and forward to the base of the 5th metatarsal bone. It then turns medialward to the interval between the bases of the 1st and 2nd metatarsal bones, where it unites with the deep plantar branch of the dorsalis pedis artery, thus completing the plantar arch. As this artery passes lateralward, it is first placed between the calcaneus and abductor hallucis, and then between the flexor digitorum brevis and quadratus plantae. As it runs forward to the base of the little toe, it lies more superficially between the flexor digitorum brevis and abductor digiti quinti, covered by the plantar aponeurosis and integument. The remaining portion of the vessel is deeply situated. It extends from the base of the 5th metatarsal bone to the proximal part of the first interosseous space, and forms the plantar arch. It is convex forward, lies below the bases of the 2nd, 3rd, and 4th metatarsal bones, and the corresponding interossi, and upon the oblique part of the adductor hallucis. Branches. The plantar arch, besides distributing numerous branches to the muscles, integument, and fasciae in the sole, gives off the following branches: perforating, plantar metatarsal. The perforating branches, rami perforantis, are 3 in number. They ascend through the proximal parts of the 2nd, 3rd, and 4th interosseous spaces, between the heads of the interossi dorsalis, and anastomose with the dorsal metatarsal arteries. The plantar metatarsal arteries, arteriae metatarsei plantaris, digital branches, are 4 in number, and run forward between the metatarsal bones and in contact with the interossi. Each divides into a pair of plantar digital arteries, which supply the adjacent sides of the toes. Near their points of division, each sends upward an anterior perforating branch to join the corresponding dorsal metatarsal artery. The first plantar metatarsal artery, arteria princeps hallucis, springs from the junction between the lateral plantar and deep plantar arteries, and sends the digital branch to the medial side of the first toe. The digital branch for the lateral side of the 5th toe arise from the lateral plantar artery near the base of the 5th metatarsal bone. End of section 32.
Section 33 of Gray's Anatomy Part 3. This is a Librivox recording. All Librivox recordings are in the public domain. For more information or to volunteer, please visit Librivox.org. Anatomy of the Human Body, Part 3, by Henry Gray. The Veins. Introduction. The veins convey the blood from the capillaries of the different parts of the body to the heart. They consist of two distinct sets of vessels, the pulmonary and systemic. The pulmonary veins, unlike other veins, contain arterial blood, which they return from the lungs to the left atrium of the heart. The systemic veins return the venous blood from the body generally to the right atrium of the heart. The portal vein, an appendage to the systemic venous system, is confined to the abdominal cavity and returns the venous blood from the spleen and the viscera of digestion to the liver. This vessel ramifies in the substance of the liver and there breaks up into a minute network of capillary-like vessels from which the blood is conveyed by the hepatic veins to the inferior vena cava. The veins commence by minute plexuses which receive the blood from the capillaries. The branches arising from these plexuses unite together into trunks, and these, in their passage toward the heart, constantly increase in size as they receive tributaries or join other veins. The veins are larger and altogether more numerous than the arteries, hence the entire capacity of the venous system is much greater than that of the arterial. The capacity of the pulmonary veins, however, only slightly exceeds that of the pulmonary arteries. The veins are cylindrical like the arteries. Their walls, however, are thin, and they collapse when the vessels are empty, and the uniformity of their surfaces is interrupted at intervals by slight constrictions, which indicate the existence of valves in their interior. They communicate very freely with one another, especially in certain regions of the body, and these communications exist between the larger trunks as well as between the smaller branches. Thus, between the venous sinuses of the cranium and between the veins of the neck, where obstruction would be attended with imminent danger to the cerebral venous system, large and frequent anastomoses are found. The same free communication exists between the veins throughout the whole extent of the vertebral canal, and between the veins composing the various venous plexuses in the abdomen and pelvis, for example, the spermatic, uterine, vesical, and pudental. The systemic venous channels are subdivided into three sets, namely, superficial and deep veins and venous sinuses. The superficial veins, cutaneous veins, are found between the layers of the superficial fascia, immediately beneath the skin. They return the blood from these structures and communicate with the deep veins by perforating the deep fascia. The deep veins accompany the arteries and are usually enclosed in the same sheaths with these vessels. With the smaller arteries, as the radial, ulnar, brachial, tibial, peroneal, they exist generally in pairs, one lying on each side of the vessel, and are called venae comitantes. The larger arteries, such as the axillary, subclavian, popliteal, and femoral, have usually only one accompanying vein. In certain organs of the body, however, the deep veins do not accompany the arteries. For instance, the veins in the skull and vertebral canal, the hepatic veins in the liver, and the larger veins returning blood from the bones. Venous sinuses are found only in the interior of the skull, and consist of canals formed by the separation of the two layers of the dura mater. Their outer coat consists of fibrous tissue, their inner of an endothelial layer continuous with the lining membrane of the veins. End of section 33.
Section 34 of Gray's Anatomy, Part 3. This is a Librivox recording. All Librivox recordings are in the public domain. For more information or to volunteer, please visit librivox.org. Anatomy of the Human Body, Part 3, by Henry Gray. Pulmonary veins, systemic veins, and veins of the heart. 2. The pulmonary veins, vena pulmonalis. The pulmonary veins return the arterialized blood from the lungs to the left atrium of the heart. They are 4 in number, 2 from each lung, and are destitute of valves. They commence in a capillary network upon the walls of the air sacs, where they are continuous with the capillary ramifications of the pulmonary artery, and, joining together, form one vessel for each lobule. These vessels, uniting successively, form a single trunk for each lobe, 3 for the right and 2 for the left lung. The vein from the middle lobe of the right lung generally unites with that from the upper lobe, so that ultimately 2 trunks from each lung are formed. They perforate the fibrous layer of the pericardium and open separately into the upper and back parts of the left atrium. Occasionally the 3 veins on the right side remain separate. Not infrequently the 2 left pulmonary veins end by a common opening. At the root of the lung, the superior pulmonary vein lies in front of and a little below the pulmonary artery. The inferior is situated at the lowest part of the hilus of the lung, and on a plane posterior to the upper vein. Behind the pulmonary artery is the bronchus. Within the pericardium, their anterior surfaces are invested by the serous layer of this membrane. The right pulmonary veins pass behind the right atrium and superior vena cava, the left in front of the descending thoracic aorta. 3. The systemic veins. The systemic veins may be arranged into 3 groups: the veins of the heart, the veins of the upper extremities, head, neck, and thorax, which end in the superior vena cava, the veins of the lower extremities, abdomen and pelvis, which end in the inferior vena cava. The veins of the heart: coronary sinus, sinus coronaris, VV cordis. Most of the veins of the heart open into the coronary sinus. This is a wide venous channel about 2.25 centimetres in length, situated in the posterior part of the coronary sulcus, and covered by muscular fibres from the left atrium. It ends in the right atrium between the openings of the inferior vena cava and the atrioventricular aperture, its orifice being guarded by a semilunar valve, the valve of the coronary sinus, valve of Fabricius. Tributaries. Its tributaries are the great, small, and middle cardiac veins, the posterior vein of the left ventricle, and the oblique vein of the left atrium, all of which, except the last, are provided with valves at their orifices. 1. The great cardiac vein, vein cordis magna, left coronary vein, begins at the apex of the heart and descends along the anterior longitudinal sulcus to the base of the ventricles. It then curves to the left in the coronary sulcus, opens into the left extremity of the coronary sinus, and reaching the back of the heart, opens into the left extremity of the coronary sinus. It receives tributaries from the left atrium and from both ventricles. 1. The left marginal vein is of considerable size, and ascends along the left margin of the heart. 2. The small cardiac vein, vein cordis pave, right coronary vein, runs in the coronary sulcus between the right atrium and ventricle, and opens into the right extremity of the coronary sinus. It receives blood from the back of the right atrium and ventricle. The right marginal vein ascends along the right margin of the heart and joins it in the coronary sulcus, or opens directly into the right atrium. 3. The middle cardiac vein, vein cordis media, commences at the apex of the heart, ascends to the posterior longitudinal sulcus, and ends in the coronary sinus near its right extremity. 4. The posterior vein of the left ventricle, venus posterius ventricula sinistro, runs on the diaphragmatic surface of the left ventricle to the coronary sinus, but may end in the great cardiac vein. 5. The oblique vein of the left atrium, venus obliquus anthris sinistra Marshalli, oblique vein of Marshall, is a small vessel which descends obliquely on the back of the left atrium and ends in the coronary sinus near its left extremity. It is continuous above with the ligament of the left vena cava, lig vena cava sinistra vestigial fold of Marshall, and the 2 structures from the remnant of the left Caverian duct. The following cardiac veins do not end in the coronary sinus. 1. The anterior cardiac veins, comprising 3 or 4 small vessels which collect blood from the front of the right ventricle and open into the right atrium. The right marginal vein frequently opens into the right atrium, and is therefore sometimes regarded as belonging to this group. 2. The smallest cardiac veins, veins of Fabricius, consist of a number of minute veins which arise in the muscular wall of the heart. The majority open into the atria, but a few end in the ventricles. End of section 34.
Section 35 of Gray's Anatomy, Part 3. This is a Librivox recording. All Librivox recordings are in the public domain. For more information or to volunteer, please visit Librivox.org. Recording by Bologna Times. Anatomy of the Human Body, Part 3, by Henry Gray. Veins of head and neck. Veins of exterior of head and face. 3b. The veins of the head and neck. The veins of the head and neck may be subdivided into three groups. 1. The veins of the exterior of the head and face. 2. The veins of the neck. 3. The diploic veins, the veins of the brain, and the venous sinuses of the dura mater. The veins of the exterior of the head and face are frontal, superficial temporal, supraorbital, internal maxillary, angular, posterior facial, anterior facial, posterior auricular, occipital. The front vein, venus frontalis, begins on the forehead in a venous plexus which communicates with the frontal branches of the superficial temporal vein. The veins converge to form a single trunk, which runs downward near the middle line of the forehead, parallel with the vein of the opposite side. The two veins are joined at the root of the nose by a transverse branch called the nasal arch, which receives some small veins from the dorsum of the nose. At the root of the nose the veins diverge, and each at the medial angle of the orbit joins the supraorbital vein to form the angular vein. Occasionally the frontal veins join to form a single trunk, which bifurcates at the root of the nose into the two angular veins. The supraorbital vein, venus supraorbitalis, begins on the forehead, where it communicates with the frontal branch of the superficial temporal vein. It runs downward, superficial, to the frontalis muscle, and joins the frontal vein at the medial angle of the orbit to form the angular vein. Previous to its junction with the frontal vein, it sends through the supraorbital notch into the orbit a branch which communicates with the ophthalmic vein. As this vessel passes through the notch, it receives the frontal diploic vein through a foramen at the bottom of the notch. The angular vein, venus angularis, formed by the junction of the frontal and supraorbital veins, runs obliquely downward, on the side of the root of the nose, to the level of the lower margin of the orbit, where it becomes the anterior facial vein. It receives the veins of the ala nasi and communicates with the superior ophthalmic vein through the nasofrontal vein, thus establishing an important anastomosis between the anterior facial vein and the cavernous sinus. The anterior facial vein, venus facialis anterior facial vein, commences at the side of the root of the nose and is a direct continuation of the angular vein. It lies behind the external maxillary facial artery and follows a less tortuous course. It runs obliquely downward and backward beneath the zygomaticus and zygomatic head of the quadratus labii superioris, descends along the anterior border and then on the superficial surface of the masseter, crosses over the body of the mandible and passes obliquely backward beneath the platysma and cervical fascia, superficial to the submaxillary gland, the digastricus and stylohyoideus. It unites with the posterior facial vein to form the common facial vein, which crosses the external carotid artery and enters the internal jugular vein at a variable point beyond the hyoid bone. From near its termination, a communicating branch often runs down the anterior border of the sternocleidomastoidus to join the lower part of the anterior jugular vein. The facial vein has no valves, and its walls are not so flaccid as most superficial veins. Tributaries. The anterior facial vein receives a branch of considerable size, the deep facial vein, from the pterygoid venous plexus. It is also joined by the superior and inferior palpebral, the superior and inferior labial, the buccinator and the mesenteric veins. Below the mandible it receives the submental, palatine, and submaxillary veins, and generally the vena comitans of the hypoglossal nerve. The superficial temporal vein, venus temporalis superficialis, begins on the side and vertex of the skull in a plexus which communicates with the frontal and supraorbital veins, with the corresponding vein on the opposite side, and with the posterior auricular and occipital veins. From this network, frontal and parietal branches arise and unite above the zygomatic arch to form the trunk of the vein, which is joined in the situation by the middle temporal vein from the substance of the temporalis. It then crosses the posterior root of the zygomatic arch, enters the substance of the parotid gland, and unites with the internal maxillary vein to form the posterior facial vein. Tributaries. The superficial temporal vein receives in its course some parotid veins, articular veins from the temporomandibular joint, anterior auricular veins from the auricula, and the transverse facial from the side of the face. The middle temporal vein receives the orbital vein, which is formed by some lateral palpebral branches, and passes backward between the layers of the temporal fascia to join the superficial temporal vein. The pterygoid plexus, plexus pterygoidis, is of considerable size, and is situated between the temporalis and pterygoidis externus, and partly between the two pterygoidae. It receives tributaries corresponding with the branches of the internal maxillary artery. Thus it receives the sphenopalatine, the middle meningeal, the deep temporal, the pterygoid, mesenteric, buccinator, alveolar, and some palatine veins, and a branch which communicates with the ophthalmic vein through the inferior orbital fissure. This plexus communicates freely with the anterior facial vein. It also communicates with the cavernous sinus by branches through the foramen vesali, foramen ovale, and foramen lacerum. The internal maxillary vein, venus maxillaris interna, is a short trunk which accompanies the first part of the internal maxillary artery. It is formed by a confluence of the veins of the pterygoid plexus, and passes backward between the sphenomandibular ligament and the neck of the mandible, and unites with the temporal vein to form the posterior facial vein. The posterior facial vein, venus facialis posterior, temporomaxillary vein, formed by the union of the superficial temporal and internal maxillary veins, descends in the substance of the parotid gland, superficial to the external carotid artery, but beneath the facial nerve, between the ramus of the mandible and the sternocleidomastoideus muscle. It divides into two branches, an anterior, which passes forward and unites with the anterior facial vein to form the common facial vein, and a posterior, which is joined by the posterior auricular vein and becomes the external jugular vein. The posterior auricular vein, venous auricularis posterior, begins upon the side of the head in a plexus which communicates with the tributaries of the occipital and superficial temporal veins. It descends behind the auricula and joins the posterior division of the posterior facial vein to form the external jugular. It received the stylomastoid vein and some tributaries from the cranial surface of the auricula. The occipital vein, venous occipitalis, begins in a plexus at the back of the vertex of the skull. From the plexus emerges a single vessel, which pierces the cranial attachment of the trapezius and, dipping into the suboccipital triangle, joins the deep cervical and vertebral veins. Occasionally it follows the course of the occipital artery and ends in the internal jugular. In other instances it joins the posterior auricular and through it opens into the external jugular. The parietal emissary vein connects it with the superior sagittal sinus, and as it passes across the mastoid portion of the temporal bone, it receives the mastoid emissary vein, which connects it with the transverse sinus. The occipital diploic vein sometimes joins it. End of section 35.
Section 36 of Gray's Anatomy, Part 3. This is a Librivox recording. All Librivox recordings are in the public domain. For more information or to volunteer, please visit Librivox.org. Recorded by Laurie Anne Walden. Anatomy of the Human Body, Part 3, by Henry Gray. The veins of the neck. The veins of the neck, which return the blood from the head and face, are external jugular, posterior external jugular, anterior jugular, internal jugular, vertebral. The external jugular vein, vena jugularis externa, receives the greater part of the blood from the exterior of the cranium and the deep parts of the face, being formed by the junction of the posterior division of the posterior facial with the posterior auricular vein. It commences in the substance of the parotid gland, on a level with the angle of the mandible, and runs perpendicularly down the neck, in the direction of a line drawn from the angle of the mandible to the middle of the clavicle at the posterior border of the sternocleidomastoideus. In its course it crosses the sternocleidomastoideus obliquely, and in the subclavian triangle perforates the deep fascia, and ends in the subclavian vein, lateral to, or in front of, the scalenus anterior. It is separated from the sternocleidomastoideus by the investing layer of the deep cervical fascia, and is covered by the platysma, the superficial fascia, and the integument. It crosses the cutaneous cervical nerve, and its upper half runs parallel with the great auricular nerve. The external jugular vein varies in size, bearing an inverse proportion to the other veins of the neck. It is occasionally double. It is provided with two pairs of valves, the lower pair being placed at its entrance into the subclavian vein, the upper, in most cases, about 4 cm above the clavicle. The portion of the vein between the two sets of valves is often dilated, and is termed the sinus. These valves do not prevent the regurgitation of the blood, or the passage of injection from below upward. Tributaries. This vein receives the occipital occasionally, the posterior external jugular, and, near its termination, the transverse cervical, transverse scapular, and anterior jugular veins. In the substance of the parotid, a large branch of communication from the internal jugular joins it. The posterior external jugular vein, vena jugularis posterior, begins in the occipital region, and returns the blood from the skin and superficial muscles in the upper and back part of the neck, lying between the splenius and trapezius. It runs down the back part of the neck, and opens into the external jugular vein just below the middle of its course. The anterior jugular vein, vena jugularis anterior, begins near the hyoid bone by the confluence of several superficial veins from the submaxillary region. It descends between the median line and the anterior border of the sternocleidomastoideus, and, at the lower part of the neck, passes beneath that muscle to open into the termination of the external jugular, or in some instances into the subclavian vein. It varies considerably in size, bearing usually an inverse proportion to the external jugular. Most frequently there are two anterior jugulars, a right and left, but sometimes only one. Its tributaries are some laryngeal veins, and occasionally a small thyroid vein. Just above the sternum the two anterior jugular veins communicate by a transverse trunk, the venous jugular arch, which receive tributaries from the inferior thyroid veins. Each also communicates with the internal jugular. There are no valves in this vein. The internal jugular vein, vena jugularis interna, collects the blood from the brain, from the superficial parts of the face, and from the neck. It is directly continuous with the transverse sinus, and begins in the posterior compartment of the jugular foramen at the base of the skull. At its origin it is somewhat dilated, and this dilatation is called the superior bulb. It runs down the side of the neck in a vertical direction, lying at first lateral to the internal carotid artery, and then lateral to the common carotid, and at the root of the neck unites with the subclavian vein to form the innominate vein. A little above its termination is a second dilatation, the inferior bulb. Above it lies upon the rectus capitis lateralis, behind the internal carotid artery and the nerves passing through the jugular foramen. Lower down, the vein and artery lie upon the same plane, the glossopharyngeal and hypoglossal nerves passing forward between them. The vagus descends between and behind the vein and the artery in the same sheath, and the accessory runs obliquely backward, superficial or deep to the vein. At the root of the neck, the right internal jugular vein is placed at a little distance from the common carotid artery, and crosses the first part of the subclavian artery, while the left internal jugular vein usually overlaps the common carotid artery. The left vein is generally smaller than the right, and each contains a pair of valves, which are placed about 2.5 centimeters above the termination of the vessel. Tributaries. This vein receives in its course the inferior petrosal sinus, the common facial, lingual, pharyngeal, superior and middle thyroid veins, and sometimes the occipital. The thoracic duct on the left side, and the right lymphatic duct on the right side, open into the angle of union of the internal jugular and subclavian veins. The inferior petrosal sinus, sinus petrosus inferior, leaves the skull through the anterior part of the jugular foramen, and joins the superior bulb of the internal jugular vein. The lingual veins, vena lingualis, begin on the dorsum, sides, and undersurface of the tongue, and, passing backward along the course of the lingual artery, end in the internal jugular vein. The vena comitans of the hypoglossal nerve, Ranine vein, a branch of considerable size, begins below the tip of the tongue, and may join the lingual. Generally, however, it passes backward on the hyoglossus, and joins the common facial. The pharyngeal veins, vena pharyngeae, begin in the pharyngeal plexus on the outer surface of the pharynx, and, after receiving some posterior meningeal veins and the veins of the pterygoid canal, end in the internal jugular. They occasionally open into the facial, lingual, or superior thyroid vein. The superior thyroid vein, vena thyroidea superioris, begins in the substance and on the surface of the thyroid gland, by tributaries corresponding with the branches of the superior thyroid artery, and ends in the upper part of the internal jugular vein. It receives the superior laryngeal and cricothyroid veins. The middle thyroid vein collects the blood from the lower part of the thyroid gland, and after being joined by some veins from the larynx and trachea, ends in the lower part of the internal jugular vein. The common facial and occipital veins have been described. The vertebral vein, vena vertebralis, is formed in the suboccipital triangle from numerous small tributaries which spring from the internal vertebral venous plexuses and issue from the vertebral canal above the posterior arch of the atlas. They unite with small veins from the deep muscles at the upper part of the back of the neck, and form a vessel which enters the foramen in the transverse process of the atlas, and descends, forming a dense plexus around the vertebral artery, in the canal formed by the foramina transversaria of the cervical vertebrae. This plexus ends in a single trunk, which emerges from the foramen transversarium of the sixth cervical vertebra, and opens at the root of the neck into the back part of the innominate vein near its origin, its mouth being guarded by a pair of valves. On the right side it crosses the first part of the subclavian artery. Tributaries. The vertebral vein communicates with the transverse sinus by a vein which passes through the condyloid canal, when that canal exists. It receives branches from the occipital vein and from the prevertebral muscles, from the internal and external vertebral venous plexuses, from the anterior vertebral and the deep cervical veins. Close to its termination it is sometimes joined by the first intercostal vein. The anterior vertebral vein commences in a plexus around the transverse processes of the upper cervical vertebrae, descends in company with the ascending cervical artery between the scalenus anterior and longus capitis muscles, and opens into the terminal part of the vertebral vein. The deep cervical vein, vena cervicalis profundus, posterior vertebral or posterior deep cervical vein, accompanies its artery between the semispinalis capitis and coli. It begins in the suboccipital region by communicating branches from the occipital vein and by small veins from the deep muscles at the back of the neck. It receives tributaries from the plexuses around the spinous processes of the cervical vertebrae, and terminates in the lower part of the vertebral vein. Into section 36.
Section 37 of Gray's Anatomy, Part 3. This is a Librivox recording. All Librivox recordings are in the public domain. For more information or to volunteer, please visit librivox.org. Anatomy of the Human Body, Part 3, by Henry Gray. The diploic veins. The diploic veins occupy channels in the diploe of the cranial bones. They are large and exhibit at irregular intervals pouch-like dilatations. Their walls are thin and formed of endothelium resting upon a layer of elastic tissue. So long as the cranial bones are separable from one another, these veins are confined to the particular bones. But when the sutures are obliterated, they unite with each other and increase in size. They communicate with the meningeal veins and the sinuses of the dura mater and with the veins of the pericranium. They consist of 1. The frontal, which opens into the supraorbital vein and the superior sagittal sinus. 2. The anterior temporal, which is confined chiefly to the frontal bone and opens into the sphenoparietal sinus and into one of the deep temporal veins, through an aperture in the great wing of the sphenoid. 3. The posterior temporal, which is situated in the parietal bone and ends in the transverse sinus, through an aperture at the mastoid angle of the parietal bone or through the mastoid foramen. And 4. The occipital, the largest of the four, which is confined to the occipital bone, and opens either externally into the occipital vein or internally into the transverse sinus or into the confluence of the sinuses. Torcular hierophyly. The veins of the brain. The veins of the brain possess no valves, and their walls, owing to the absence of muscular tissue, are extremely thin. They pierce the arachnoid membrane and the inner or meningeal layer of the dura mater, and open into the cranial venous sinuses. They may be divided into two sets, cerebral and cerebellar. Cerebral veins, venae cerebri, are divisible into external and internal groups, according as they drain the outer surfaces or the inner parts of the hemispheres. The external veins are the superior, inferior, and middle cerebral. The superior cerebral veins, venae cerebri superioris, 8 to 12 in number, drain the superior, lateral, and medial surfaces of the hemispheres, and are mainly lodged in the sulci between the gyri, but some run across the gyri. They open into the superior sagittal sinus. The anterior veins runs nearly at right angles to the sinus. The posterior and larger veins are directed obliquely forward and open into the sinus in a direction more or less opposed to the current of the blood contained within it. The middle cerebral vein, vena cerebri media, superficial sylvian vein, begins on the lateral surface of the hemisphere, and, running along the lateral cerebral fissure, ends in the cavernous or the sphenoparietal sinus. It is connected a. with the superior sagittal sinus by the great anastomotic vein of Trollard, which opens into one of the superior cerebral veins, b. with the transverse sinus by the posterior anastomotic vein of Labbe, which courses over the temporal lobe. The inferior cerebral veins, venae cerebri inferiores, of small size, drain the under surfaces of the hemispheres. Those on the orbital surface of the frontal lobe join the superior cerebral veins, and through these open into the superior sagittal sinus. Those of the temporal lobe anastomose with the middle cerebral and basal veins, and join the cavernous, sphenoparietal, and superior petrosal sinuses. The basal vein is formed at the anterior perforated substance by the union of a small anterior cerebral vein which accompanies the anterior cerebral artery, b. the deep middle cerebral vein, deep sylvian vein, which receives tributaries from the insula and neighboring gyri, and runs in the lower part of the lateral cerebral fissure, and c. the inferior striate veins, which leave the corpus striatum through the anterior perforated substance. The basal vein passes backward around the cerebral peduncle and ends in the internal cerebral vein, vein of Galen. It receives tributaries from the interpeduncular fossa, the inferior horn of the lateral ventricle, the hippocampal gyrus, and the midbrain. The internal cerebral veins, venae cerebri interne, veins of Galen, deep cerebral veins, drain the deep parts of the hemisphere and are 2 in number. Each is formed near the interventricular foramen by the union of the terminal and choroid veins. They run backward parallel with one another between the layers of the tela choroidea of the third ventricle and beneath the splenium of the corpus callosum, where they unite to form a short trunk, the great cerebral vein. Just before their union, each receives the corresponding basal vein. The terminal vein, vena terminalis, vena corporis striati, commences in the groove between the corporis striatum and thalamus, receives numerous veins from both of these parts, and unites behind the crus fornicis of the choroid vein to form one of the internal cerebral veins. The choroid vein runs along the whole length of the choroid plexus, and receives veins from the hippocampus, the fornix, and the corpus callosum. The great cerebral vein, vena cerebri magna, Galeni, great vein of Galen, formed by the union of the 2 internal cerebral veins, is a short median trunk which curves backward and upward through the splenium of the corpus callosum and ends in the anterior extremity of the straight sinus. The cerebellar veins are placed on the surface of the cerebellum and are disposed in 2 sets, superior and inferior. The superior cerebellar veins, venae cerebelli superiores, pass partly forward and medialward across the superior vermis to end in the straight sinus and the internal cerebral veins, partly lateralward to the transverse and superior petrosal sinuses. The inferior cerebellar veins, venae cerebelli inferiores, of large size, end in the transverse, superior petrosal, and occipital sinuses. End of section 37.
Section number 38 of Gray's Anatomy. Part 3. This is a Librivox recording. All Librivox recordings are in the public domain. For more information or to volunteer, please visit Librivox.org. Recording by Morgan Scorpion. Anatomy of the Human Body, Part 3, by Henry Gray. Sinuses of the dura mater, ophthalmic veins, emissary veins. 3b5. The sinuses of the dura mater, sinus durae matris, ophthalmic veins and emissary veins. The sinuses of the dura mater are venous channels which drain the blood from the brain. They are devoid of valves and are situated between the 2 layers of the dura mater and lined by endothelium continuous with that which lines the veins. They may be divided into two groups: 1. A postero-superior at the upper and back part of the skull, and 2. An antero-inferior at the base of the skull. The postero-superior group comprises the superior sagittal, inferior sagittal, straight, 2 transverse, occipital. The superior sagittal sinus, sinus sagittalis superior, superior longitudinal sinus, occupies the attached or convex margin of the falx cerebri. Commencing at the foramen caecum, through which it receives a vein from the nasal cavity, it runs from before backward, grooving the inner surface of the frontal, the adjacent margins of the 2 parietals, and the superior division of the cruciate eminence of the occipital. Near the internal occipital protuberance it deviates to one or other side, usually the right, and is continued as the corresponding transverse sinus. It is triangular in section, narrow in front, and gradually increases in size as it passes backward. Its inner surface presents the openings of the superior cerebral veins, which run, for the most part, obliquely forward, and open chiefly at the back part of the sinus, their orifices being concealed by fibrous folds. Numerous fibrous bands, chordae releasae, extend transversely along the inferior angle of the sinus, and lastly, small openings communicate with irregularly shaped venous spaces, venous lacunae, in the dura mater near the sinus. There are usually 3 lacunae on either side of the sinus, a small frontal, a large parietal, and an occipital, intermediate in size between the other 2. Most of the cerebral veins from the outer surface of the hemisphere open into these lacunae, and numerous arachnoid granulations, pachyonian bodies, project into them from below. The superior sagittal sinus receives the superior cerebral veins, veins from the diploe and dura mater, and near the posterior extremity of the sagittal suture, veins from the pericranium, which pass through the parietal foramina. The numerous communications exist between this sinus and the veins of the nose, scalp, and diploe. The inferior sagittal sinus, sinus sagittalis inferior, inferior longitudinal sinus, is contained in the posterior half or 2/3 in the free margin of the falx cerebri. It is of a cylindrical form, increases in size as it passes backward, and ends in the straight sinus. It receives several veins from the falx cerebri, and occasionally a few from the medial surfaces of the hemispheres. The straight sinus, sinus rectus, tentorial sinus, is situated at the line of junction of the falx cerebri with the tentorium cerebelli. It is triangular in section, increases in size as it proceeds backward, and runs downward and backward from the end of the inferior sagittal sinus to the transverse sinus of the opposite side to that into which the superior sagittal sinus is prolonged. Its terminal part communicates by a cross branch with the confluence of the sinuses. Besides the inferior sagittal sinus, it receives the great cerebral vein, great vein of Galen, and the superior cerebellar veins. A few transverse bands cross its interior. The transverse sinuses, sinus transversus, lateral sinuses, are of large size and begin at the internal occipital protuberance, one, generally on the right, being the direct continuation of the superior sagittal sinus, the other of the straight sinus. Each transverse sinus passes lateralward and forward, describing a slight curve with its convexity upward, to the base of the petrous portion of the temporal bone, and lies, in this part of its course, in the attached margin of the tentorium cerebelli. It then leaves the tentorium and curves downward and medialward to reach the jugular foramen, where it ends in the internal jugular vein. In its course it rests upon the squama of the occipital, the mastoid angle of the parietal, the mastoid part of the temporal, and, just before its termination, the jugular process of the occipital. The portion which occupies the groove on the mastoid part of the temporal is sometimes termed the sigmoid sinus. The transverse sinuses are frequently of unequal size, that formed by the superior sagittal sinus being the larger. They increase in size as they proceed from behind forward. On transverse section the horizontal portion exhibits a prismatic, the curved portion a semicylindrical form. They receive the blood from the superior petrosal sinuses at the base of the petrous portion of the temporal bone. They communicate with the veins of the pericranium by means of the mastoid and condyloid emissary veins, and they receive some of the inferior cerebral and inferior cerebellar veins, and some of the veins from the diploe. The petrosquamous sinus, when present, runs backward along the junction of the squamar and petrous portion of the temporal, and opens into the transverse sinus. The occipital sinus, sinus occipitalis, is the smallest of the cranial sinuses. It is situated in the attached margin of the falx cerebelli, and is generally single, but occasionally there are two. It commences around the margin of the foramen magnum by several small venous channels, one of which joins the terminal part of the transverse sinus. It communicates with the posterior internal vertebral venous plexuses and ends in the confluence of the sinuses. The confluence of the sinuses, confluens sinuum, torcular herophili, is the term applied to the dilated extremity of the superior sagittal sinus. It is of irregular form, and is lodged on one side, generally the right, of the internal occipital protuberance. From it the transverse sinus of the same side is derived. It receives also the blood from the occipital sinus, and is connected across the middle line with the commencement of the transverse sinus of the opposite side. The antero-inferior group of sinuses comprises the 2 cavernous, 2 intercavernous, 2 superior petrosal, 2 inferior petrosal, and the basilar plexus. The cavernous sinuses, sinus cavernosus, are so named because they present a reticulated structure, due to their being traversed by numerous interlacing filaments. They are of irregular form, larger behind than in front, and are placed one on either side of the body of the sphenoid bone, extending from the superior orbital fissure to the apex of the petrous portion of the temporal bone. Each opens behind into the petrosal sinuses. On the medial wall of each sinus is the internal carotid artery, accompanied by filaments of the carotid plexus. Near the artery is the abducent nerve. On the lateral wall are the oculomotor and trochlear nerves, and the ophthalmic and maxillary divisions of the trigeminal nerve. These structures are separated from the blood flowing along the sinus by the lining membrane of the sinus. The cavernous sinus receives the superior ophthalmic vein through the superior orbital fissure, some of the cerebral veins, and also the small sphenoparietal sinus, which courses along the under surface of the small wing of the sphenoid. It communicates with the transverse sinus by means of the superior petrosal sinus, with the internal jugular vein to the inferior petrosal sinus and a plexus of veins on the internal carotid artery, with the pterygoid venous plexus through the foramen Vessalii, foramen ovali, and foramen Lacerum, and with the angular vein through the ophthalmic vein. The two sinuses also communicate with each other by means of the anterior and posterior intercavernous sinuses. The ophthalmic veins, 2 in number, superior and inferior, are devoid of valves. The superior ophthalmic vein, venus ophthalmica superior, begins at the inner angle of the orbit in a vein named the nasofrontal, which communicates anteriorly with the angular vein. It pursues the same course as the ophthalmic artery, and receives tributaries corresponding to the branches of that vessel. Forming a short trunk, it passes between the two heads of the rectus lateralis and through the medial part of the superior orbital fissure, and ends in the cavernous sinus. The inferior ophthalmic vein, vena ophthalmica inferior, begins in a venous network at the fore part of the floor and medial wall of the orbit. It receives some veins from the rectus inferior, obliquus inferior, lacrimal sac and eyelids, runs backward in the lower part of the orbit, and divides into two branches. One of these passes through the inferior orbital fissure and joins the pterygoid venous plexus, while the other enters the cranium through the superior orbital fissure and ends in the cavernous sinus, either by a separate opening, or more frequently in common with the superior ophthalmic vein. The intercavernous sinuses, sini intercavernosi, are 2 in number, an anterior and a posterior, and connect the two cavernous sinuses across the middle line. The anterior passes in front of the hypophysis cerebri, the posterior behind it, and they form with the cavernous sinuses a venous circle, circular sinus, around the hypophysis. The anterior one is usually the larger of the two, and one or other is occasionally absent. The superior petrosal sinus, sinus petrosus superior, small and narrow, connects the cavernous with the transverse sinus. It runs lateralward and backward from the posterior end of the cavernous sinus over the trigeminal nerve, and lies in the attached margin of the tentorium cerebelli and in the superior petrosal sulcus of the temporal bone. It joins the transverse sinus where the latter curves downward on the inner surface of the mastoid part of the temporal. It receives some cerebellar and inferior cerebral veins, and veins from the tympanic cavity. The inferior petrosal sinus, sinus petrosus inferior, is situated in the inferior petrosal sulcus formed by the junction of the petrous part of the temporal with the basilar part of the occipital. It begins in the postero-inferior part of the cavernous sinus, and passing through the anterior part of the jugular foramen, ends in the superior bulb of the internal jugular vein. The inferior petrosal sinus receives the internal auditory veins and also veins from the medulla oblongata, pons, and undersurface of the cerebellum. The exact relation of the parts to one another in the jugular foramen is as follows. The inferior petrosal sinus lies medially and anteriorly with the meningeal branch of the ascending pharyngeal artery, and is directed obliquely downward and backward. The transverse sinus is situated at the lateral and back part of the foramen with a meningeal branch of the occipital artery, and between the two sinuses are the glossopharyngeal, vagus, and accessory nerves. These three sets of structures are divided from each other by two processes of fibrous tissue. The junction of the inferior petrosal sinus with the internal jugular vein takes place on the lateral aspect of the nerves. The basilar plexus, plexus basilaris, transverse or basilar sinus, consists of several interlacing venous channels between the layers of the dura mater over the basilar part of the occipital bone, and serves to connect the two inferior petrosal sinuses. It communicates with the anterior vertebral venous plexus. Emissary veins, emissaria. The emissary veins pass through the apertures in the cranial wall and establish communication between the sinuses inside the skull and the veins external to it. Some are always present, others only occasionally so. The principal emissary veins are the following. 1. A mastoid emissary vein, usually present, runs through the mastoid foramen and unites the transverse sinus with the posterior auricular or with the occipital vein. 2. A parietal emissary vein passes through the parietal foramen and connects the superior sagittal sinus with the veins of the scalp. 3. A network of minute veins, rete canalis hypoglossi, transverses the hypoglossal canal and joins the transverse sinus with the vertebral vein and deep veins of the neck. 4. An inconstant condyloid emissary vein passes through the condyloid canal and connects the transverse sinus with the deep veins of the neck. 5. A network of veins, rete foraminus ovalis, unites the cavernous sinus with the pterygoid plexus through the foramen ovale. 6. 2 or 3 small veins run through the foramen lacerum and connect the cavernous sinus with the pterygoid plexus. 7. The emissary vein of the foramen of Vesalius connects the same parts. 8. An internal carotid plexus of veins transverses the carotid canal and unites the cavernous sinus with the internal jugular vein. 9. A vein is transmitted through the foramen caecum and connects the superior sagittal sinus with the veins of the nasal cavity. End of section number 38.
Section 39 of Gray's Anatomy, Part 3. This is a Librivox recording. All Librivox recordings are in the public domain. For more information or to volunteer, please visit Librivox.org. Recording by Eric Hale, Anatomy of the Human Body, Part 3, by Henry Gray. The veins of the upper extremity and thorax. The veins of the upper extremity are divided into two sets, superficial and deep. The two sets anastomose frequently with each other. The superficial veins are placed immediately beneath the integument between the two layers of superficial fascia. The deep veins accompany the arteries and constitute the venae comitatis of those vessels. Both sets are provided with valves, which are more numerous in the deep than in the superficial veins. The superficial veins of the upper extremity. The superficial veins of the upper extremity are the digital, metacarpal, cephalic, basilic, median, Digital veins. The dorsal digital veins pass along the sides of the fingers and are joined to one another by oblique communicating branches. Those from the adjacent sides of the fingers unite to form 3 dorsal metacarpal veins, which end in a dorsal venous network opposite the middle of the metacarpus. The radial part of the network is joined by the dorsal digital vein from the radial side of the index finger and by the dorsal digital veins of the thumb, and is prolonged upward as the cephalic vein. The ulnar part of the network receives the dorsal digital vein of the ulnar side of the little finger, and is continued upward as the basilic vein. A communicating branch frequently connects the dorsal venous network with the cephalic vein about the middle of the forearm. The volar digital veins on each finger are connected to the dorsal digital veins by oblique intercapitular veins. They drain into a venous plexus which is situated over the thenar and hypothenar eminences and across the front of the wrist. The cephalic vein begins in the radial part of the dorsal venous network and winds upward around the radial border of the forearm, receiving tributaries from both surfaces. Below the front of the elbow, it gives off the vena mediana cubitae, which receives a communicating branch from the deep veins of the forearm and passes across to join the basilic vein. The cephalic vein then ascends in front of the elbow in the groove between the brachioradialis and the biceps brachii. It crosses superficial to the musculocutaneous nerve and ascends in the groove along the lateral border of the biceps brachii. In the upper third of the arm, it passes between the pectoralis major and deltoideus, where it is accompanied by the deltoid branch of the thoracoacromial artery. It pierces the coracoclavicular fascia and, crossing the axillary artery, ends in the axillary vein just below the clavicle. Sometimes it communicates with the external jugular vein by a branch which ascends in front of the clavicle. The accessory cephalic vein arises either from a small tributary plexus on the back of the forearm or from the ulnar side of the dorsal venous network. It joins the cephalic below the elbow. In some cases, the accessory cephalic springs from the cephalic above the wrist and joins it again higher up. A large oblique branch frequently connects the basilic and cephalic veins on the back of the forearm. The basilic vein begins in the ulnar part of the dorsal venous network. It runs up the posterior surface of the ulnar side of the forearm and inclines forward to the anterior surface below the elbow, where it is joined by the vena mediana cubitae. It ascends obliquely in the groove between the biceps brachii and pronate arteries, and crosses the brachial artery, from which it is separated by the lacertus fibrosus. Filaments of the medial antibrachial cutaneous nerve pass both in front of and behind this portion of the vein. It then runs upward along the medial border of the biceps brachii, perforates the deep fascia a little below the middle of the arm, and, ascending on the medial side of the brachial artery to the lower border of the teres major, is continued onward as the axillary vein. The median antibrachial vein drains the venous plexus on the volar surface of the hand. It ascends on the ulnar side of the front of the forearm, and ends in the basilic vein or in the vena mediana cubitae. In a small proportion of cases, it divides into 2 branches, one of which joins the basilic, the other the cephalic, below the elbow. The deep veins of the upper extremity. The deep veins follow the course of the arteries, forming their venae comitantes. They are generally arranged in pairs, and are situated one on either side of the corresponding artery, and connected at intervals by short transverse branches. Deep veins of the hand. The superficial and deep volar arterial arches are each accompanied by a pair of venae comitantes. The superficial and deep volar arterial arches are each accompanied by a pair of venae comitantes, which constitute respectively the superficial and deep volar venous arches, and receive the veins corresponding to the branches of the arterial arches. Thus the common volar digital veins, formed by the union of the proper volar digital veins, open into the superficial, and the volar metacarpal veins into the deep volar venous arches. The dorsal metacarpal veins receive perforating branches from the volar metacarpal veins and end in the radial veins and in the superficial veins on the dorsum of the wrist. The deep veins of the forearm are the venae comitatantes of the radial and ulnar veins, and constitute, respectively, the upward continuations of the deep and superficial volar venous arches. They unite in front of the elbow to form the brachial veins. The radial veins are smaller than the ulnar, and receive the dorsal metacarpal veins. The ulnar veins receive tributaries from the deep volar venous arches, and communicate with the superficial veins at the wrist. Near the elbow they receive the volar and dorsal interosseous veins, and send a large communicating branch, profunda vein, to the vena mediana cubiti. The brachial veins are placed one on either side of the brachial artery, receiving tributaries corresponding with the branches given off from that vessel. Near the lower margin of the subscapularis they join the axillary vein. The medial one frequently joins the basilic vein. The axillary vein begins at the lower border of the teres major, as the continuation of the basilic vein, increases in size as it ascends, and ends at the outer border of the first rib as the subclavian vein. Near the lower border of the subscapularis it receives the brachial veins, and, close to its termination, the cephalic vein. Its other tributaries correspond with the branches of the axillary artery. It lies on the medial side of the artery, which it partly overlaps. Between the two vessels are the medial cord of the brachial plexus, the median, the ulnar, and the medial anterior thoracic nerves. It is provided with a pair of valves opposite the lower border of the subscapularis. Valves are also found at the ends of the cephalic and subscapular veins. The subclavian vein, the continuation of the axillary, extends from the outer border of the first rib to the sternal end of the clavicle, where it unites with the internal jugular to form the innominate vein. It is in relation, in front, with the clavicle and subclavius, behind and above, with the subclavian artery, from which it is separated medially by the scalenus anterior and the phrenic nerve. Below, it rests in a depression on the first rib and upon the pleura. It is usually provided with a pair of valves, which are situated about 2.5 centimeters from its termination. The subclavian vein occasionally rises in the neck to a level with the third part of the subclavian artery, and occasionally passes with this vessel behind the scalenus anterior. Tributaries. This vein receives the external jugular vein, sometimes the anterior jugular vein, and occasionally a small branch, which ascends in front of the clavicle from the cephalic. At its angle of junction with the internal jugular, the left subclavian vein receives the thoracic duct, and the right subclavian vein the right lymphatic duct. The veins of the thorax. The innominate veins are two large trunks, placed one on either side of the root of the neck, and formed by the union of the internal jugular and subclavian veins of the corresponding side. They are devoid of valves. The right innominate vein is a short vessel, about 2.5 centimeters in length, which begins behind the sternal end of the clavicle and, passing almost vertically downward, joins with the left innominate vein just below the cartilage of the first rib, close to the right border of the sternum, to form the superior vena cava. It lies in front and to the right of the innominate artery. On its right side are the phrenic nerve and the pleura, which are interposed between it and the apex of the lung. This vein, at its commencement, receives the right vertebral vein, and, lower down, the right internal mammary and right inferior thyroid veins, and sometimes the vein from the first intercostal space. The left innominate vein, about 6 centimeters in length, begins behind the sternal end of the clavicle, and runs obliquely downward and to the right behind the upper half of the manubrium sterni, to the sternal end of the first right costal cartilage, where it unites with the right innominate vein to form the superior vena cava. It is separated from the manubrium sterni by the sternohyoideus and sternothyroideus, the thymus or its remains, and some loose areolar tissue. Behind it are the three large arteries, innominate, left common carotid, and left subclavian, arising from the aortic arch, together with the vagus and phrenic nerves. The left innominate vein may occupy a higher level, crossing the jugular notch and lying directly in front of the trachea. Tributaries. Its tributaries are the left vertebral, left internal mammary, left inferior thyroid, and the left highest intercostal veins, and occasionally some thymic and pericardiac veins. Peculiarities. Sometimes the innominate veins open separately into the right atrium. In such cases, the right vein takes the ordinary course of the superior vena cava. The left vein, left superior vena cava, as it is then termed, which may communicate by a small branch with the right one, passes in front of the root of the left lung, and, turning to the back of the heart, ends in the right atrium. This occasional condition in the adult is due to the persistence of the early fetal condition, and is the normal state of things in birds and some mammalia. The internal mammary veins are venae comitatus to the lower half of the internal mammary artery, and receive tributaries corresponding to the branches of the artery. They then unite to form a single trunk, which runs up on the medial side of the artery and ends in the corresponding innominate vein. The superior phrenic vein, i.e., the vein accompanying the periocardiacal phrenic artery, usually opens into the internal mammary vein. The inferior thyroid veins, 2, frequently 3 or 4 in number, arise in the venous plexus on the thyroid gland, communicating with the middle and superior thyroid veins. They form a plexus in front of the trachea, behind the sternothyroidea. From this plexus, a left vein descends and joins the left innominate trunk, and a right vein passes obliquely downward and to the right across the innominate artery to open into the right innominate vein, just at its junction with the superior vena cava. Sometimes the right and left veins open by a common trunk in the latter situation. These veins receive esophageal, tracheal, and inferior laryngeal veins, and are provided with valves at their terminations in the innominate veins. The highest intercostal vein, right and left, drain the blood from the upper 3 or 4 intercostal spaces. The right vein passes downward and opens into the vena azygos. The left vein runs across the arch of the aorta and the origins of the left subclavian and left common carotid arteries, and opens into the left innominate vein. It usually receives the left bronchial vein and sometimes the left superior phrenic vein, and communicates below with the accessory hemiazygos vein. The superior vena cava drains the blood from the upper half of the body. It measures about 7 centimeters in length and is formed by the junction of the 2 innominate veins. It begins immediately below the cartilage of the right first rib close to the sternum, and, descending vertically behind the first and second intercostal spaces, ends in the upper part of the right atrium opposite the upper border of the third right costal cartilage. The lower half of the vessel is within the pericardium. In its course, it describes a slight curve, the convexity of which is to the right side. Relations. In front are the anterior margins of the right lung and pleura with the pericardium intervening below. These separate it from the first and second intercostal spaces, and from the second and third right costal cartilages. Behind it are the root of the right lung and the right vagus nerve. On its right side are the phrenic nerve and right pleura. On its left side, the commencement of the innominate artery and the ascending aorta, the latter overlapping it. Just before it pierces the pericardium, it receives the azygos vein and several small veins from the pericardium and other contents of the mediastinal cavity. The portion contained within the pericardium is covered, in front and laterally, by the serous layer of the membrane. The superior vena cava has no valves. The azygos vein begins opposite the first or second lumbar vertebrae by a branch, the ascending lumbar vein, sometimes by a branch from the right renal vein or from the inferior vena cava. It enters the thorax through the aortic hiatus in the diaphragm, and passes along the right side of the vertebral column to the fourth thoracic vertebra, where it arches forward over the root of the right lung, and ends in the superior vena cava, just before that vessel pierces the pericardium. In the aortic hiatus, it lies with the thoracic duct on the right side of the aorta. In the thorax, it lies upon the intercostal arteries, on the right side of the aorta and thoracic duct, and is partly covered by pleura. Tributaries. It receives the right subcostal and intercostal veins, the upper 3 or 4 of these latter opening by a common stem, the highest superior intercostal vein. It receives the hemiazygos veins, several esophageal, mediastinal, and pericardial veins, and, near its termination, the right bronchial vein. A few imperfect valves are found in the azygos vein, but its tributaries are provided with complete valves. The intercostal veins on the left side, below the upper 3 intercostal spaces, usually form 2 trunks, named the hemiazygos and accessory hemiazygos veins. The hemiazygos vein begins in the left ascending lumbar or renal vein. It enters the thorax through the left crus of the diaphragm and, ascending on the left side of the vertebral column, as high as the 9th thoracic vertebra, passes across the column, behind the aorta, esophagus, and thoracic duct, to end in the azygos vein. It receives the lower 4 or 5 intercostal veins and the subcostal vein of the left side, and some esophageal and mediastinal veins. The accessory hemiazygos vein descends on the left side of the vertebral column, and varies inversely in size with the highest left intercostal vein. It receives veins from the 3 or 4 intercostal spaces between the highest left intercostal vein and highest tributary of the hemiazygos. The left bronchial vein sometimes opens into it. It either crosses the body of the 8th thoracic vertebrae to join the azygos vein, or ends in the hemiazygos. When this vein is small, or altogether wanting, the left highest intercostal vein may extend as low as the 5th or 6th intercostal space. In obstruction of the superior vena cava, the azygos and hemiazygos veins are one of the principal means by which the venous circulation is carried on, connecting as they do the superior and inferior vena cava, and communicating with the common iliac veins by the ascending lumbar veins and with many of the tributaries of the inferior vena cava. The bronchial veins return the blood from the large bronchi, and from the structures at the roots of the lungs. That of the right side opens into the azygos vein, near its termination, that of the left side into the highest left intercostal or the accessory hemiazygos vein. A considerable quantity of the blood which is carried to the lungs through the bronchial arteries is returned to the left side of the heart through the pulmonary veins. The veins of the vertebral column. The veins which drain the blood from the vertebral column, the neighboring muscles, and the meninges of the medulla spinalis form intricate plexuses extending along the entire length of the column. These plexuses may be divided into 2 groups, external and internal, according to their positions inside or outside the vertebral canal. The plexuses of the 2 groups anastomose freely with each other, and end in the intervertebral veins. The external vertebral venous plexuses, best marked in the cervical region, consist of anterior and posterior plexuses, which anastomose freely with each other. The anterior external plexuses lie in front of the bodies of the vertebrae, communicate with the basovertebral and intervertebral veins, and receive tributaries from the vertebral bodies. The posterior external plexuses are placed partly on the posterior surfaces of the vertebral arches and their processes, and partly between the deep dorsal muscles. They are best developed in the cervical region, and there anastomose with the vertebral, occipital, and deep cervical veins. The internal vertebral venous plexuses lie within the vertebral canal between the dura mater and the vertebrae, and receive tributaries from the bones and from the medulla spinalis. They form a closer network than the external plexuses, and, running mainly in a vertical direction, form 4 longitudinal veins, 2 in front and 2 behind. They therefore may be divided into anterior and posterior groups. The anterior internal plexuses consist of large veins which lie on the posterior surfaces of the vertebral bodies and intervertebral fibrocartilages on either side of the posterior longitudinal ligament. The anterior internal plexuses consist of large veins which lie on the posterior surfaces of the vertebral bodies and intervertebral fibrocartilages on either side of the posterior longitudinal ligament. Under cover of this ligament, they are connected by transverse branches into which the basovertebral veins open. The posterior internal plexuses are placed, one on either side of the middle line, in front of the vertebral arches and ligamenta flava, and anastomosed by veins passing through those ligaments with the posterior external plexuses. The anterior and posterior plexuses communicate freely with one another by a series of venous rings, one opposite each vertebra. Around the foramen magnum, they form an intricate network which opens into the vertebral veins, and is connected above with the occipital sinus, the basilar plexus, the condyloid emissary vein, and the rete canalis hypoglossi. The basovertebral veins emerge from the foramina on the posterior surfaces of the vertebral bodies. They are contained in large, tortuous channels in the substance of the bones, similar in every respect to those found in the diplo of the cranial bones. They communicate through small openings on the front and sides of the bodies of the vertebrae with the anterior external vertebral plexuses, and converge behind to the principal canal, which is sometimes double toward its posterior part, and open by valved orifices into the transverse branches, which unite the anterior internal vertebral plexuses. They become greatly enlarged in advanced age. The intervertebral veins accompany the spinal nerves through the intervertebral foramina. They receive the veins from the medulla spinalis, drain the internal and external vertebral plexuses, and end in the vertebral, intercostal, lumbar, and lateral sacral veins, their orifices being provided with valves. The veins of the medulla spinalis are situated in the pia mater and form a minute tortuous venous plexus. They emerge chiefly from the median fissures of the medulla spinalis and are largest in the lumbar region. In this plexus there are two median longitudinal veins, one in front of the anterior fissure and the other behind the posterior sulcus of the cord, and four lateral longitudinal veins which run behind the nerve roots. They end in the intervertebral veins. Near the base of the skull they unite, and form two or three small trunks, which communicate with the vertebral veins, and then end in the inferior cerebellar veins, or in the inferior petrosal sinuses. End of section 39. Recording by Eric Hale. Section 40 of Gray's Anatomy, Part 3. This is a Librivox recording. All Librivox recordings are in the public domain. For more information or to volunteer, please visit Librivox.org. Recording by Eric Hale. Anatomy of the Human Body, Part 3, by Henry Gray. The Veins of the Lower Extremity, Abdomen, and Pelvis, Part 1. The veins of the abdomen and pelvis. The veins of the lower extremity are subdivided, like those of the upper, into two sets, superficial and deep. The superficial veins are placed beneath the integument between the two layers of superficial fascia. The deep veins accompany the arteries. Both sets of veins are provided with valves, which are more numerous in the deep than in the superficial set. Valves are also more numerous in the veins of the lower than in those of the upper limb. The superficial veins of the lower extremity. The superficial veins of the lower extremity are the great and small saphenous veins and their tributaries. On the dorsum of the foot, the dorsal digital veins receive, in the clefts between the toes, the intercapitular veins from the plantar cutaneous venous arch, and join to form short common digital veins, which unite across the distal ends of the metatarsal bones in a dorsal venous arch. Proximal to this arch is an irregular venous network, which receives tributaries from the deep veins, and is joined to the sides of the foot by a medial and a lateral marginal vein, formed mainly by the union of branches from the superficial parts of the sole of the foot. On the sole of the foot, the superficial veins form a plantar cutaneous venous arch, which extends across the roots of the toes, and opens at the sides of the foot into the medial and lateral marginal veins. Proximal to this arch is a plantar cutaneous venous network, which is especially dense in the fat beneath the heel. This network communicates with the cutaneous venous arch and with the deep veins, but is chiefly drained into the medial and lateral marginal veins. The great saphenous vein, the longest vein in the body, begins in the medial marginal vein of the dorsum of the foot, and ends in the femoral vein about 3 centimeters below the inguinal ligament. It ascends in front of the tibio-malleolus and along the medial side of the leg in relation with the saphenous nerve. It runs upward behind the medial condyles of the tibia and femur and along the medial side of the thigh, and, passing through the fossa ovalis, ends in the femoral vein. Tributaries. At the ankle, it receives branches from the sole of the foot through the medial marginal vein. In the leg, it anastomoses freely with the small saphenous vein, communicates with the anterior and posterior tibial veins, and receives many cutaneous veins. In the thigh, it communicates with the femoral vein, and receives numerous tributaries. Those from the medial and posterior parts of the thigh frequently unite to form a large accessory saphenous vein, which joins the main vein at a variable level. Near the fossa ovalis, it is joined by the superficial epigastric, superficial iliac circumflex, and superficial external pudendal veins. A vein, named the thoracoepigastric, runs along the lateral aspect of the trunk between the superficial epigastric vein below and the lateral thoracic vein above, and establishes an important communication between the femoral and axillary veins. The valves in the great saphenous vein vary from 10 to 20 in number. They are more numerous in the leg than in the thigh. The small saphenous vein begins behind the lateral malleolus as a continuation of the lateral marginal vein. It first ascends along the lateral margin of the tendocalcaneus, and then crosses it to reach the middle of the back of the leg. Running directly upward, it perforates the deep fascia in the lower part of the popliteal fossa, and ends in the popliteal vein, between the heads of the gastrocnemius. It communicates with the deep veins on the dorsum of the foot, and receives numerous large tributaries from the back of the leg. Before it pierces the deep fascia, it gives off a branch, which runs upward and forward to join the great saphenous vein. The small saphenous vein possesses from 9 to 12 valves, one of which is always found near its termination in the popliteal vein. In the lower third of the leg, the small saphenous vein is in close relation with the sural nerve, in the upper 2/3 with the medial sural cutaneous nerve. The deep veins of the lower extremity. The deep veins of the lower extremity accompany the arteries and their branches. They possess numerous valves. The plantar digital veins arise from plexuses on the plantar surfaces of the digits, and, after sending intercapitular veins to join the dorsal digital veins, unite to form 4 metatarsal veins. These run backward in the metatarsal spaces, communicate by means of perforating veins with the veins on the dorsum of the foot, and unite to form the deep plantar venous arch, which lies alongside the plantar arterial arch. From the deep plantar venous arch, the medial and lateral plantar veins run backward close to the corresponding arteries, and, after communicating with the great and small saphenous veins, unite behind the medial malleolus to form the posterior tibial veins. The posterior tibial veins accompany the posterior tibial artery, and are joined by the perineal veins. The anterior tibial veins are the upward continuation of the venae comitatus of the dorsalis pedis artery. They leave the front of the leg by passing between the tibia and fibula over the interosseous membrane, and unite with the posterior tibial to form the popliteal vein. The popliteal vein is formed by the junction of the anterior and posterior tibia veins at the lower border of the popliteus. It ascends through the popliteal fossa to the aperture in the adductor magnus, where it becomes the femoral vein. In the lower part of its course it is placed medial to the artery. Between the heads of the gastrocnemius it is superficial to that vessel, but above the knee joint it is close to its lateral side. It receives tributaries corresponding to the branches of the popliteal artery, and it also receives the small saphenous vein. The valves in the popliteal vein are usually 4 in number. The femoral vein accompanies the femoral artery through the upper 2/3 of the thigh. In the lower part of its course it lies lateral to the artery. Higher up it is behind it, and at the inguinal ligament it lies on its medial side and on the same plane. It receives numerous muscular tributaries, and about 4 centimeters below the inguinal ligament is joined by the V profunda femoris. Near its termination it is joined by the great saphenous vein. The valves in the femoral vein are 3 in number. The deep femoral vein receives tributaries corresponding to the perforating branches of the profunda artery, and, through these, establishes communications with the popliteal vein below and the inferior gluteal vein above. It also receives the medial and lateral femoral circumflex veins. The external iliac vein, the upward continuation of the femoral vein, begins behind the inguinal ligament, and, passing upward along the brim of the lesser pelvis, ends opposite the sacroiliac articulation, by uniting with the hypogastric vein to form the common iliac vein. On the right side, it lies at first medial to the artery, but, as it passes upward, gradually inclines behind it. On the left side, it lies altogether on the medial side of the artery. It frequently contains 1, sometimes 2, valves. Tributaries. The external iliac vein receives the inferior epigastric, deep iliac circumflex, and pubic veins. The inferior epigastric vein is formed by the union of the venae comitatus of the inferior epigastric artery, which communicate above with the superior epigastric vein. It joins the external iliac about 1.25 centimeters above the inguinal ligament. The deep iliac circumflex vein is formed by the union of the venae comitatus of the deep iliac circumflex artery, and joins the external iliac vein about 2 centimeters above the inguinal ligament. The pubic vein communicates with the obturator vein in the obturator foramen, and ascends on the back of the pubis to the external iliac vein. The hypogastric vein begins near the upper part of the greater sciatic foramen, passes upward behind and slightly medial to the hypogastric artery, and, at the brim of the pelvis, joins with the external iliac to form the common iliac vein. Tributaries. With the exception of the fetal umbilical vein, which passes upward and backward from the umbilicus to the liver, and the iliolumbar vein, which usually joins the common iliac vein, their tributaries of the hypogastric vein correspond with the branches of the hypogastric artery. It receives A, the gluteal, internal, pudendal, and obturator veins, which have their origins outside the pelvis; B, the lateral sacral veins, which lie in front of the sacrum; and C, the middle hemorrhoidal, vesical, uterine, and vaginal veins, which originate in venous plexuses connected with the pelvis viscera. The superior gluteal veins are venae comitatus of the superior gluteal artery. They receive tributaries from the buttock corresponding with the branches of the artery, and enter the pelvis through the greater sciatic foramen, above the piriformis, and frequently unite before ending in the hypogastric vein. The inferior gluteal veins, or venae comitatus of the inferior gluteal artery, begin on the upper part of the back of the thigh, where they anastomose with the medial femoral circumflex and first perforating veins. They enter the pelvis through the lower part of the greater sciatic foramen, and join to form a single stem which opens into the lower part of the hypogastric vein. The internal pudendal veins are the venae comitatus of the internal pudendal artery. They begin in the deep veins of the penis, which issue from the corpus cavernosum penis, accompany the internal pudendal artery, and unite to form a single vessel, which ends in the hypogastric vein. They receive the veins from the urethral bulb, and the perineal and inferior hemorrhoidal veins. Deep dorsal vein of the penis communicates with the internal pudendal veins, but ends mainly in the pudendal plexus. The obturator vein begins in the upper portion of the adductor region of the thigh, and enters the pelvis through the upper part of the obturator foramen. It runs backward and upward on the lateral wall of the pelvis below the obturator artery, and then passes between the ureter and the hypogastric artery to end in the hypogastric vein. The lateral sacral veins accompany the lateral sacral arteries on the anterior surface of the sacrum and end in the hypogastric vein. The middle hemorrhoidal vein takes origin in the hemorrhoidal plexus and receives tributaries from the bladder, prostate, and seminal vesicle. It runs lateralward on the pelvic surface of the levator ani to end in the hypogastric vein. End of section 40. Recording by Eric Hale. Section 41 of Gray's Anatomy, Part 3. This is a Librivox recording. All Librivox recordings are in the public domain. For more information or to volunteer, please visit Librivox.org. Recording by Eric Hale. Anatomy of the Human Body, Part 3, by Henry Gray. The Veins of the Lower Extremity, Abdomen, and Pelvis, Part 2. The hemorrhoidal plexus surrounds the rectum and communicates in front with the vesical plexus in the male and the uterovaginal plexus in the female. It consists of two parts, an internal in the submucosa and an external outside the mascular coat. The internal plexus presents a series of dilated pouches which are arranged in a circle around the tube, immediately above the anal orifice, and are connected by transverse branches. The lower part of the external plexus is drained by the inferior hemorrhoidal veins into the internal pudendal vein, the middle part by the middle hemorrhoidal vein which joins the hypogastric vein, and the upper part by the superior hemorrhoidal vein, which forms the commencement of the inferior mesenteric vein, a tributary of the portal vein. A free communication between the portal and systemic venous systems is established through the hemorrhoidal plexus. The veins of the hemorrhoidal plexus are contained in very loose connective tissue, so that they get less support from surrounding structures than most other veins, and are less capable of resisting increased blood pressure. The pudendal plexus lies behind the arcuate pubic ligament and the lower part of the symphysis pubis, and in front of the bladder and prostate. Its chief tributary is the deep dorsal vein of the penis, but it also receives branches from the front of the bladder and prostate. It communicates with the vesical plexus and with the internal pudendal vein, and drains into the vesical and hypogastric veins. The prostatic veins form a well-marked prostatic plexus, which lies partly in the fascial sheath of the prostate, and partly between the sheath and the prostatic capsule. It communicates with the pudendal and vesical plexuses. The vesical plexus envelops the lower part of the bladder and the base of the prostate, and communicates with the pudendal and prostatic plexuses. It is drained by means of several vesical veins into the hypogastric veins. The dorsal veins of the penis are two in number, a superficial and a deep. The superficial vein drains the prepuce and skin of the penis, and, running backward in the subcutaneous tissue, inclines to the right or left, and opens into the corresponding superficial external pudendal vein, a tributary of the great saphenous vein. The deep vein lies beneath the deep fascia of the penis. It receives the blood from the glans penis and corpora cavernosa penis, and courses backward in the middle line between the dorsal arteries. Near the root of the penis, it passes between the two parts of the suspensory ligament, and then through an aperture between the arcuate pubic ligament and the transverse ligament of the pelvis, and divides into two branches, which enter the pudendal plexus. The deep vein also communicates below the symphysis pubis with the internal pudendal vein. The uterine plexuses lie along the sides and superior angles of the uterus between the two layers of the broad ligament, and communicate with the ovarian and vaginal plexuses. They are drained by a pair of uterine veins on either side. These arise from the lower part of the plexuses, opposite the external orifice of the uterus, and open into the corresponding hypogastric vein. The vaginal plexuses are placed at the sides of the vagina. They communicate with the uterine, vesical, and hemorrhoidal plexuses, and are drained by the vaginal veins, one on either side, into the hypogastric veins. The common iliac veins are formed by the union of the external iliac and hypogastric veins, in front of the sacroiliac articulation. Passing obliquely upward toward the right side, they end upon the fifth lumbar vertebra, by uniting with each other at an acute angle to form the inferior vena cava. The right common iliac is shorter than the left, nearly vertical in its direction, and ascends behind and then lateral to its corresponding artery. The left common iliac, longer than the right and more oblique in its course, is at first situated on the medial side of the corresponding artery, and then behind the right common iliac. Each common iliac receives the iliolumbar and sometimes the lateral sacral veins. The left receives, in addition, the middle sacral vein. No valves are found in these veins. The middle sacral veins accompany the corresponding artery along the front of the sacrum, and join to form a single vein, which ends in the left common iliac vein, sometimes in the angle of junction of the two iliac veins. Peculiarities. The left common iliac vein, instead of joining with the right in its usual position, occasionally ascends on the left side of the aorta, as high as the kidney, where, after receiving the left renal vein, it crosses over the aorta, and then joins with the right vein to form the vena cava. In these cases, the two common iliacs are connected by a small communicating branch at the spot where they are usually united. The inferior vena cava returns to the heart the blood from the parts below the diaphragm. It is formed by the junction of the two common iliac veins, on the right side of the fifth lumbar vertebra. It ascends along the front of the vertebral column, on the right side of the aorta, and, having reached the liver, is continued in a groove on its posterior surface. It then perforates the diaphragm between the median and right portions of its central tendon. It subsequently inclines forward and medialward for about 2.5 centimeters, and, piercing the fibrous pericardium, passes behind the serous pericardium to open into the lower and back part of the right atrium. In front of its atrial orifice is a semilunar valve, termed the valve of the inferior vena cava. This is rudimentary in the adult, but is of large size and exercises an important function in the fetus. Relations. The abdominal portion of the inferior vena cava is in relation in front, from below upward, with the right common iliac artery, the mesentery, the right internal spermatic artery, the inferior part of the duodenum, the pancreas, the common bile duct, the portal vein, and the posterior surface of the liver. The last partly overlaps and occasionally completely surrounds it. Behind, with the vertebral column, the right psoas major, the right crus of the diaphragm, the right inferior phrenic, suprarenal, renal and lumbar arteries, right sympathetic trunk and right celiac ganglion, and the medial part of the right suprarenal gland. On the right side, with the right kidney and ureter. On the left side, with the aorta, right crus of the diaphragm, and the caudate lobe of the liver. The thoracic portion is only about 2.5 centimeters in length, and is situated partly inside and partly outside the pericardial sac. The extrapericardial part is separated from the right pleura and lung by a fibrous band, named the right phrenico-pericardiac ligament. This ligament, often feebly marked, is attached below to the margin of the vena caval opening in the diaphragm, and above to the pericardium in front of and behind the root of the right lung. The intrapericardiac part is very short, and is covered anterolaterally by the serous layer of the pericardium. Peculiarities. In position. This vessel is sometimes placed on the left side of the aorta, as high as the left renal vein, and, after receiving this vein, crosses over to its usual position on the right side, or it may be placed altogether on the left side of the aorta, and in such a case the abdominal and thoracic viscera, together with the great vessels, are all transposed. Point of termination. Occasionally, the inferior vena cava joins the azygos vein, which is then of large size. In such cases, the superior vena cava receives the whole of the blood from the body before transmitting it to the right atrium, except the blood from the hepatic veins, which passes directly into the right atrium. Tributaries. The inferior vena cava receives the following veins: lumbar, renal, inferior phrenic, right spermatic or ovarian, suprarenal, hepatic. The lumbar veins, 4 in number on each side, collect the blood by dorsal tributaries from the muscles and integument of the loins, and by abdominal tributaries from the walls of the abdomen, where they communicate with the epigastric veins. At the vertebral column they receive veins from the vertebral plexuses, and then pass forward around the sides of the bodies of the vertebrae, beneath the psoas major, and end in the back part of the inferior cava. The left lumbar veins are longer than the right, and pass behind the aorta. The lumbar veins are connected together by a longitudinal vein, which passes in front of the transverse process of the lumbar vertebrae, and is called the ascending lumbar. It forms the most frequent origin of the corresponding azygos or hemiazygos vein, and serves to connect the common iliac, iliolumbar, and azygos or hemiazygos veins of its own side of the body. The spermatic veins emerge from the back of the testes and receive tributaries from the epididymis. They unite and form a convoluted plexus, called the pampiniform plexus, which constitutes the greater mass of the spermatic cord. The vessels composing this plexus are very numerous, and ascend along the cord, in front of the ductus deferens. Below the subcutaneous inguinal ring they unite to form 3 or 4 veins, which pass along the inguinal canal, and, entering the abdomen through the abdominal inguinal ring, coalesce to form 2 veins, which ascend on the psoas major, behind the peritoneum, lying one on either side of the internal spermatic artery. These unite to form a single vein, which opens on the right side into the inferior vena cava at an acute angle, on the left side into the left renal vein, at a right angle. The spermatic veins are provided with valves. The left spermatic vein passes behind the iliac colon, and is thus exposed to pressure from the contents of that part of the bowel. The ovarian veins correspond with the spermatic in the male. They form a plexus in the broad ligament near the ovary and uterine tube, and communicate with the uterine plexus. They end in the same way as the spermatic veins in the male. Valves are occasionally found in these veins. Like the uterine veins, they become much enlarged during pregnancy. The renal veins are of large size, and placed in front of the renal arteries. The left is longer than the right, and passes in front of the aorta, just below the origin of the superior mesenteric artery. It receives the left spermatic and left inferior phrenic veins, and, generally, the left suprarenal vein. It opens into the inferior vena cava at a slightly higher level than the right. The suprarenal veins are 2 in number. The right ends in the inferior vena cava, the left in the left renal or left inferior phrenic vein. The inferior phrenic veins follow the course of the inferior phrenic arteries. The right ends in the inferior vena cava. The left is often represented by two branches, one of which ends in the left renal or suprarenal vein, while the other passes in front of the esophageal hiatus in the diaphragm, and opens into the inferior vena cava. The hepatic veins commence in the substance of the liver, in the terminations of the portal vein and hepatic artery, and are arranged in two groups, upper and lower. The upper group usually consists of three large veins, which converge toward the posterior surface of the liver, and open into the inferior vena cava, while that vessel is situated in the groove on the back part of the liver. The veins of the lower group vary in number, and are of small size. They come from the right and caudate lobes. The hepatic veins run singly, and are in direct contact with the hepatic tissue. They are destitute of valves. End of section 41. Recording by Eric Hale.
Section 42 of Gray's Anatomy, Part 3. This is a Librivox recording. All Librivox recordings are in the public domain. For more information or to volunteer, please visit Librivox.org. Anatomy of the Human Body, Part 3, by Henry Gray. The portal system of veins. The portal system includes all the veins which drain the blood from the abdominal part of the digestive tube, with the exception of the lower part of the rectum, and from the spleen, pancreas, and gallbladder. From these viscera, the blood is conveyed to the liver by the portal vein. In the liver, this vein ramifies like an artery and ends in capillary-like vessels termed sinusoids, from which the blood is conveyed to the inferior vena cava by the hepatic veins. From this it will be seen that the blood of the portal system passes through two sets of minute vessels, namely, A, the capillaries of the digestive tube, spleen, pancreas, and gallbladder, and B, the sinusoids of the liver. In the adult, the portal vein and its tributaries are destitute of valves. In the fetus and for a short time after birth, valves can be demonstrated in the tributaries of the portal vein. As a rule, they soon atrophy and disappear, but in some subjects they persist in a degenerate form. The portal vein, vena porti, is about 8 centimeters in length, and is formed at the level of the second lumbar vertebra by the junction of the superior mesenteric and lienal veins, the union of these veins taking place in front of the inferior vena cava and behind the neck of the pancreas. It passes upward behind the superior part of the duodenum, and then ascends in the right border of the lesser omentum to the right extremity of the porta hepatis, where it divides into a right and a left branch, which accompany the corresponding branches of the hepatic artery into the substance of the liver. In the lesser omentum it is placed behind and between the common bile duct and the hepatic artery, the former lying to the right of the latter. It is surrounded by the hepatic plexus of nerves, and is accompanied by numerous lymphatic vessels and some lymph glands. The right branch of the portal vein enters the right lobe of the liver, but before doing so generally receives the cystic vein. The left branch, longer but of smaller caliber than the right, crosses the left sagittal fossa, gives branches to the caudate lobe, and then enters the left lobe of the liver. As it crosses the left sagittal fossa, it is joined in front by a fibrous cord, the ligamentum teres, obliterated umbilical vein, and is united to the inferior vena cava by a second fibrous cord, the ligamentum venosum, obliterated ductus venosus. Tributaries. The tributaries of the portal vein are lienal, superior mesenteric, coronary, pyloric, cystic, and paraumbilical. The lienal vein, V. lienalis, splenic vein, commences by 5 or 6 large branches which return the blood from the spleen. These unite to form a single vessel, which passes from left to right, grooving the upper and back part of the pancreas, below the lineal artery, and ends behind the neck of the pancreas by uniting at a right angle with the superior mesenteric to form the portal vein. The lienal vein is of large size, but is not tortuous like the artery. Tributaries. The lineal vein receives the short gastric veins, the left gastroepiploic vein, the pancreatic veins, and the inferior mesenteric veins. The short gastric veins, VV gastrici brevis, 4 or 5 in number, drain the fundus and left part of the greater curvature of the stomach, and pass between the 2 layers of the gastrolienal ligament to end in the lienal vein or in one of its large tributaries. the left gastroepiploic vein, V. gastroepiploica sinistra, receives branches from the anterior-superior and posterior-inferior surfaces of the stomach and from the greater omentum. It runs from right to left along the greater curvature of the stomach and ends in the commencement of the lienal vein. The pancreatic veins, VV pancreaticae, consist of several small vessels which drain the body and tail of the pancreas and open into the trunk of the lienal vein. The inferior mesenteric vein, V. mesenterica inferior, returns blood from the rectum and the sigmoid and descending parts of the colon. It begins in the rectum as the superior hemorrhoidal vein, which has its origin in the hemorrhoidal plexus, and through this plexus communicates with the middle and inferior hemorrhoidal veins. The superior hemorrhoidal vein leaves the lesser pelvis and crosses the left common iliac vessels with the superior hemorrhoidal artery, and is continued upward as the inferior mesenteric vein. This vein lies to the left of its artery and ascends behind the peritoneum and in front of the left psoas major. It then passes behind the body of the pancreas and opens into the lienal vein. Sometimes it ends in the angle of union of the lienal and supramesenteric veins. Tributaries. The inferior mesenteric vein receives the sigmoid veins from the sigmoid colon and iliac colon, and the left colic vein from the descending colon and the left colic flexure. The superior mesenteric vein, V. mesenterica superior, returns the blood from the small intestine, from the cecum, and from the ascending and transverse portions of the colon. It begins in the right iliac fossa by the union of the veins which drain the terminal part of the ileum, the cecum, and the vermiform process, and ascends between the two layers of the mesentery on the right side of the superior mesenteric artery. In its upward course, it passes in front of the right ureter, the inferior vena cava, the inferior part of the duodenum, and the lower portion of the head of the pancreas. Behind the neck of the pancreas, it unites with the lienal vein to form the portal vein. Tributaries. Besides the tributaries which correspond with the branches of the superior mesenteric artery, namely the intestinal, ileocolic, right colic, and middle colic veins, the superior mesenteric vein is joined by the right gastroepiploic and pancreatic duodenal veins. The right gastroepiploic vein, V. gastroepiploica dextra, receives branches from the greater omentum and from the lower parts of the antero-superior and posterior-inferior surfaces of the stomach. It runs from left to right along the greater curvature of the stomach between the two layers of the greater omentum. The pancreatic duodenal veins, VV pancreatico-duodenales, accompany their corresponding arteries. The lower of the two frequently joins the right gastroepiploic vein. The coronary vein, V. coronaria ventriculi, gastric vein, derives tributaries from both surfaces of the stomach. It runs from right to left along the lesser curvature of the stomach, between the two layers of the lesser omentum, to the esophageal opening of the stomach, where it receives some esophageal veins. It then turns backward and passes from left to right behind the omental bursa and ends in the portal vein. The pyloric vein is of small size and runs from left to right along the pyloric portion of the lesser curvature of the stomach, between the two layers of the lesser omentum, to end in the portal vein. The cystic vein, V. cystica, drains the blood from the gallbladder, and accompanying the cystic duct, usually ends in the right branch of the portal vein. Parumbilical veins, VV parumbilica calis. In the course of the ligamentum teres of the liver and of the middle umbilical ligament, small veins, parumbilical, are found which establish an anastomosis between the veins of the anterior abdominal wall and the portal, hypogastric, and iliac veins. The best marked of these small veins is one which commences at the umbilicus and runs backward and upward in or on the surface of the ligamentum teres between the layers of the falciform ligament to end in the left portal vein. Collateral venous circulation to relieve portal obstruction in the liver may be effected by communications between a. the gastric veins and the esophageal veins, which often project as a varicose bunch into the stomach, emptying themselves into the hemiazygos vein; b. the veins of the colon and the duodenum and the left renal vein; c. the accessory portal system of Sappi, branches of which pass in the round and falciform ligaments, particularly the latter, to unite with the epigastric and internal mammary veins, and through the diaphragmatic veins with the azygos. A single large vein, shown to be a parumbilical vein, may pass from the hilus of the liver by the round ligament of the umbilicus, producing there a bunch of prominent varicose veins known as the caput medusae. D. The veins of Retzius, which connect the intestinal veins with the inferior vena cava and its retroperitoneal branches. E. The inferior mesenteric veins and the hemorrhoidal veins that open into the hypogastrics. F. Very rarely, the ductus venosus remains patent, affording a direct connection between the portal vein and the inferior vena cava. End of section 42.
Section 43 of Gray's Anatomy, Part 3. This is a Librivox recording. All Librivox recordings are in the public domain. For more information or to volunteer, please visit Librivox.org. Recording by Shirley van Wallichem. Anatomy of the Human Body, Part 3, by Henry Gray. The Lymphatic System, Introduction. The lymphatic system consists 1. Of complex capillary networks, which collect the lymph in the various organs and tissues. 2. Of an elaborate system of collecting vessels, which conduct the lymph from the capillaries to the large veins of the neck, at the junction of the internal jugular and subclavian veins, where the lymph is poured into the blood-stream. And 3. Lymph glands or nodes, which are interspaced in the pathways of the collecting vessels, filtering the lymph as it passes through them and contributing lymphocytes to it. The lymphatic capillaries and collecting vessels are lined throughout by a continuous layer of endothelial cells, forming thus a closed system. The lymphatic vessels of the small intestine receive the special designation of lacteals or chyliferous vessels. They differ in no respect from the lymphatic vessels, generally excepting that during the process of digestion they contain a milk-white fluid, the chyle. The development of the lymphatic vessels. The lymphatic system begins as a series of sacs at the point of junction of certain of the embryonic veins. These lymph sacs are developed by the confluence of numerous venous capillaries, which at first lose their connections with the venous system, but subsequently, on the formation of the sacs, regains them. The lymphatic system is therefore developmentally an offshoot of the venous system, and the lining walls of its vessels are always endothelial. In the human embryo, the lymph sacs from which the lymphatic vessels are derived are 6 in number, 2 paired, the jugular and the posterior lymph sacs, and 2 unpaired, the retroperitoneal and the cisterna chyli. In lower mammals an additional pair, subclavian, is present, but in the human embryo these are merely extensions of the jugular sacs. The position of the sacs is as follows: 1. Jugular sac, the first to appear at the junction of the subclavian vein with the primitive jugular. 2. Posterior sac, at the junction of the iliac vein with the cardinal. 3. Retroperitoneal, and the root of the mesentery near the suprarenal glands. 4. Cisterna chyli, opposite the 3rd and 4th lumbar vertebrae. From the lymph sacs, the lymphatic vessels bud out along fixed lines corresponding more or less closely to the course of the embryonic blood vessels. Both in the body wall and in the wall of the intestine, the deeper plexuses are the first to be developed. By continued growth of these, the vessels in the superficial layers are gradually formed. The thoracic duct is probably formed from anastomosing outgrowths from the jugular sac and cisterna chyli. At its connection with the cisterna chyli, it is at first double, but the two vessels soon join. All the lymph sacs, except the cisterna chyli, are at a later stage divided up by slender connective tissue bridges and transformed into groups of lymph glands. The lower portion of the cisterna chyli is similarly converted, but its upper portion remains as the adult cisterna. Lymphatic capillaries. The complex capillary plexuses, which consist of a single layer of thin, flat endothelial cells, lie in the connective tissue spaces in the various regions of the body to which they are distributed and are bathed by the intercellular tissue fluids. Two views are at present held as to the mode in which the lymph is formed, one being by the physical processes of filtration, diffusion, and osmosis, and the other, that in addition to these physical processes, the endothelial cells have an active secretory function. The colourless liquid lymph has about the same composition as blood plasma. It contains many lymphocytes and frequently red blood corpuscles. Granules and bacteria are also taken up by the lymph from the connective tissue spaces, partly by the action of lymphocytes, which pass into the lymph between the endothelial cells, and partly by the direct passage of the granules through the endothelial cells. The lymphatic capillary plexuses vary greatly in form. The anastomoses are usually numerous. Blind ends, or cul-de-sacs, are especially common in the interstitial villi, the dermal papillae, and the filiform papillae of the tongue. The plexuses are often in two layers, a superficial and a deep, the superficial being of smaller caliber than the deep. The caliber, however, varies greatly in a given plexus from a few micromillimeters to 1 millimeter. The capillaries are without valves. Distribution. The skin. Lymphatic capillaries are abundant in the dermis, where they form superficial and deep plexuses, the former sending blind ends into the dermal papillae. The plexuses are especially rich over the palmar surface of the hands and fingers, and over the plantar surface of the feet and toes. The epidermis is without capillaries. The conjunctiva has an especially rich plexus. The subcutaneous tissue is without capillaries. The tendons of striated muscle and muscle sheaths are richly supplied. In muscle, however, their existence is still disputed. The periosteum of bone is richly supplied, and they have been described, the Havosian canals. They are absent in cartilage, and probably in bone marrow. The joint capsules are richly supplied with lymphatic capillaries. They do not, however, open into the joint cavities. Beneath the mesothelial lining of the pleural, peritoneal, and pericardial cavities are rich plexuses. They do not open into these cavities. The alimentary canal is supplied with rich plexuses beneath the epithelium, often as a superficial plexus, and the mucosa and a deeper submucosal plexus. Cul-de-sacs extend into the filiform papillae of the tongue and the villi of the small intestine. Those portions of the alimentary canal covered by peritoneum have in addition a subserous lymphatic capillary plexus beneath the mesothelium. The salivary glands are supplied with lymphatic capillaries. The liver has a rich subserous plexus in the capsule, and also extensive plexuses which accompany the hepatic artery and portal vein. The lymphatic capillaries have not been followed into the liver lobules. The lymph from the liver forms a large part of that which flows through the thoracic duct. The gallbladder and bile ducts have rich subepithelial plexuses, and the former a subserous plexus. The spleen has a rich subserous set and a capsular set of lymphatic capillaries. Their presence in the parenchyma is uncertain. The nasal cavity has extensive capillary plexuses in the mucosa and submucosa. The trachea and bronchi have plexuses in the mucosa and submucosa, but the smaller bronchi have only a single layer. The capillaries do not extend to the air cells. The plexuses around the smaller bronchi connect with the rich subserous plexus of the lungs in places where the veins reach the surface. Lymphatics have been described in the thyroid gland and in the thymus. The adrenal has a superficial plexus divided into two layers, one in the loose tissue about the gland, and the other beneath the capsule. Capillaries have also been described within the parenchyma. The kidney is supplied with a coarse subserous plexus and a deeper plexus of finer capillaries in the capsule. Lymphatics have been described within the substance of the kidney surrounding the tubules. The urinary bladder has rich plexus of lymphatic capillaries just beneath the epithelial lining, also a subserous set which anastomoses with the former through the muscle layer. The submucous plexus is continuous with the submucous plexus of the urethra. The prostate has a rich lymphatic plexus surrounding the gland and a wide meshed subcapsular plexus. The testes has a rich superficial plexus beneath the tunica albuginea. The presence of deep lymphatics is disputed. The uterus is provided with a subserous plexus. The deep lymphatics are uncertain. Subepithelial plexuses are found in the vagina. The ovary has a rich superficial plexus and a deep interstitial plexus. The heart has a rich subserous plexus beneath the epicardium. Lymphatic capillaries have also been described beneath the endocardium and throughout the muscle. Lymphatic capillaries are probably absent in the central nervous system, the meninges, the eyeball, except the conjunctiva, the orbit, the internal ear, within striated muscle, the liver lobule, the spleen pulp and kidney parenchyma. They are entirely absent in cartilage. In many places further investigation is needed. Lymphatic vessels. The lymphatic vessels are exceedingly delicate, and their coats are so transparent that the fluid they contain is readily seen through them. They are interrupted at intervals by constrictions which give them a knotted or beaded appearance. These constrictions correspond to the situations of valves in their interior. Lymphatic vessels have been found in nearly every texture and organ of the body which contains blood vessels. Such non-vascular structures as cartilage, the nails, cuticle, and hair have none, but with these exceptions it is probable that eventually all parts will be found to be permeated by these vessels. Structure of lymphatic vessels. The larger lymphatic vessels are each composed of 3 coats. The internal coat is thin, transparent, slightly elastic, and consists of a layer of elongated endothelial cells with wavy margins by which the contiguous cells are dovetailed into one another. The cells are supported on an elastic membrane. The middle coat is composed of smooth muscular and fine elastic fibers, disposed in a transverse direction. The external coat consists of connective tissue intermixed with smooth muscular fibers longitudinally or obliquely disposed. It forms a protective covering to the other coats, and serves to connect the vessel with the neighboring structures. In the smaller vessels there are no muscular or elastic fibers, and the wall consists only of a connective tissue coat, lined by endothelium. The thoracic duct has a more complex structure than the other lymphatic vessels. It presents a distinct subendothelial layer of branched corpuscles, similar to that found in the arteries. In the middle coat there is, in addition to the muscular and elastic fibers, a layer of connective tissue with its fibers arranged longitudinally. The lymphatic vessels are supplied by nutrient vessels, which are distributed to their outer and middle coats, and here also have been traced many non-modulated nerves in the form of a fine plexus of fibrils. The valves of the lymphatic vessels are formed of thin layers of fibrous tissue covered on both surfaces by endothelium, which presents the same arrangement as on the valves of veins. In form the valves are semilunar. They are attached by their convex edges to the wall of the vessel, the concave edges being free and directed along the course of the contained current. Usually 2 such valves of equal size are found opposite to one another, but occasionally exceptions occur, especially at or near the anastomosis of lymphatic vessels. Thus one valve may be of small size and the other increased in proportion. In the lymphatic vessels the valves are placed at much shorter intervals than in the veins. They are most numerous near the lymph glands, and are found more frequently in the lymphatic vessels of the neck and upper extremity than in those of the lower extremity. The wall of the lymphatic vessel immediately above the point of attachment of each segment of a valve is expanded into a pouch or sinus, which gives to these vessels, when distended, the knotted or beaded appearance already referred to. Valves are wanting in the vessels composing the plexiform network, in which the lymphatic vessels usually originate on the surface of the body. Lymph glands, lymphoglandulae. The lymph glands are small, oval, or bean-shaped bodies, situated in the course of lymphatic and lacteal vessels, so that the lymph and chyle pass through them on their way to the blood. Each generally presents on one side a slight depression, the hilus, through which the blood vessels enter and leave the interior. The efferent lymphatic vessel also emerges from the gland at this spot, while the efferent vessels enter the organ at different parts of the periphery. On section, a lymph gland displays two different structures, an external, of lighter color, the cortical, and an internal, darker, the medullary. The cortical structure does not form a complete investment, but is deficient at the hilus, where the medullary portion reaches the surface of the gland, so that the efferent vessel is derived directly from the medullary structures, while the afferent vessels empty themselves into the cortical substance. Structure of lymph glands. A lymph gland consists of one, a fibrous envelope, or capsule, from which a framework of processes, trabeculae, proceeds inward, imperfectly dividing the gland into open spaces freely communicating with each other. 2. A quantity of lymphoid tissue, occupying these spaces without completely filling them. 3. A free supply of blood vessels, which are supported in the trabeculae. And 4. The afferent and efferent vessels, communicating through the lymph paths in the substance of the gland. The nerves passing into the hilus are few in number, and are chiefly distributed to the blood vessels supplying the gland. The capsule is composed of connective tissue, with some plain muscle fibers, and from its internal surface are given off a number of membranous processes, or trabeculae, consisting, in man, of connective tissue, with a small admixture of plain muscle fibers, but in many of the lower animals composed almost entirely of involuntary muscle. They pass inward, radiating toward the centre of the gland, for a certain distance, that is to say, for about 1/3 or 1/4 of the space between the circumference and the centre of the gland. In some animals they are sufficiently well marked to divide the peripheral or cortical portion of the gland into a number of compartments, so-called follicles, but in man this arrangement is not obvious. The larger trabeculae springing from the capsule break up into finer bands, and these interlace to form a meshwork in the central or medullary portion of the gland. In these spaces formed by the interlacing trabeculae is contained the proper gland substance or lymphoid tissue. The gland pulp does not, however, completely fill the spaces, but leaves, between its outer margin and the enclosing trabeculae, a channel or space of uniform width throughout. This is termed the lymph path, or lymph sinus. Running across it are a number of finer trabeculae of retiform connective tissue, the fibres of which are, for the most part, covered by ramifying cells. On account of the peculiar arrangement of the framework of the organ, the gland pulp in the cortical portion is disposed in the form of nodules, and in the medullary part in the form of rounded cords. It consists of ordinary lymphoid tissue, being made up of a delicate network of retiform tissue, which is continuous with that in the lymph paths, but marked off from it by a closer reticulation. It is probable, moreover, that the reticular tissue of the gland pulp and the lymph paths is continuous with that of the trabeculae, and ultimately with that of the capsule of the gland. In its meshes, in the nodules and cords of lymphoid tissue, are closely packed lymph corpuscles. The gland pulp is traversed by a dense plexus of capillary blood vessels. The nodules or follicles in the cortical portion of the gland frequently show, in their centres, areas where karyokinetic figures indicate the division of the lymph corpuscles. These areas are termed germ centers. The cells composing them have more abundant protoplasm than the peripheral cells. The afferent vessels, as stated above, enter at all parts of the periphery of the gland, and after branching and forming a dense plexus in the substance of the capsule, open into the lymph sinuses of the cortical part. In doing this, they lose all their coats except their endothelial lining, which is continuous with a layer of similar cells lining the lymph paths. In like manner, the afferent vessel commences from the lymph sinuses of the medullary portion. The stream of lymph carried to the gland by the afferent vessels thus passes through the plexus in the capsule to the lymph paths of the cortical portion, where it is exposed to the action of the gland pulp. Flowing through these, it enters the paths or sinuses of the medullary portion, and finally emerges from the hilus by means of the afferent vessel. The stream of lymph in its passage through the lymph sinuses is much retarded by the presence of the reticulum, hence morphological elements, either normal or morbid, are easily arrested and deposited in the sinuses. Many lymph corpuscles pass with the afferent lymph stream to join the general blood stream. The arteries of the gland enter at the hilus, and either go at once to the gland pulp to break up into a capillary plexus, or else run along the trabeculae, partly to supply them, and partly running across the lymph paths, to assist in forming the capillary plexus of the gland pulp. This plexus traverses the lymphoid tissue, but does not enter into the lymph sinuses. From it the veins commence and emerge from the organ at the same place as that at which the arteries enter. The lymphatic vessels are arranged into a superficial and a deep set. On the surface of the body, the superficial lymphatic vessels are placed immediately beneath the integument. Accompanying the superficial veins, they join the deep lymphatic vessels in certain situations by perforating the deep fascia. In the interior of the body, they lie in the submucous areolar tissue throughout the whole length of the digestive, respiratory, and genitourinary tracts, and in the subserous tissue of the thoracic and abdominal walls. Plexiform networks of minute lymphatic vessels are found interspersed among the proper elements and blood vessels of the several tissues. The vessels composing the network, as well as the meshes between them, are much larger than those of the capillary plexus. From these networks, small vessels emerge, which pass either to a neighboring gland or to join some larger lymphatic trunk. The deep lymphatic vessels, fewer in number but larger than the superficial, accompany the deep blood vessels. Their mode of origin is probably similar to that of the superficial vessels. The lymphatic vessels of any part or organ exceed the veins in number, but in size they are much smaller. Their anastomosis also, especially those of the large trunks, are more frequent, and are effected by vessels equal in diameter to those which they connect, the continuous trunks retaining the same diameter. Hemolymph nodes or glands and hemonodes, which are so abundant in some mammals, are probably not present in man. Lymph. Lymph found only in the closed lymphatic vessels is a transparent, colorless, or slightly yellow watery fluid of specific gravity about 1.015. It closely resembles the blood plasma, but is more dilute. When it is examined under the microscope, leukocytes of the lymphocyte class are found floating in the transparent fluid. They are always increased in number after the passage of the lymph through lymphoid tissue, as in lymph glands. Lymph should be distinguished from tissue fluid, which is found outside the lymphatic vessels in the tissue spaces. End of section 43.
Section 44 of Gray's Anatomy, Part 3. This is a Librivox recording. All Librivox recordings are in the public domain. For more information or to volunteer, please visit Librivox.org. Anatomy of the Human Body, Part 3, by Henry Gray. The thoracic duct. The thoracic duct, ductus thoracicus, conveys the greater part of the lymph and chyle into the blood. It is the common trunk of all the lymphatic vessels of the body, excepting those on the right side of the head, neck, and thorax, and right upper extremity, the right lung, right side of the heart, and the convex surface of the liver. In the adult it varies in length from 38 to 45 centimeters, and extends from the second lumbar vertebra to the root of the neck. It begins in the abdomen by a triangular dilatation, the cisterna chyli, which is situated on the front of the body of the second lumbar vertebra to the right side of and behind the aorta, by the side of the right crus of the diaphragm. It enters the thorax through the aortic hiatus of the diaphragm, and ascends through the posterior mediastinal cavity between the aorta and the azygous vein. Behind it in this region are the vertebral column, the right intercostal arteries, and the hemiazygous veins, as they cross to open into the azygous vein. In front of it are the diaphragm, esophagus, and pericardium, the last being separated from it by a recess of the right pleural cavity. Opposite the fifth thoracic vertebra, it inclines toward the left side, enters the superior mediastinal cavity, and ascends behind the aortic arch and the thoracic part of the left subclavian artery, and between the left side of the esophagus and the left pleura to the upper orifice of the thorax. Passing into the neck, it forms an arch which rises about 3 or 4 centimeters above the clavicle, and crosses anterior to the subclavian artery, the vertebral artery and vein, and the thyrocervical trunk or its branches. It also passes in front of the phrenic nerve and the medial border of the scalenus anterior, but is separated from these two structures by the prevertebral fascia. In front of it are the left common carotid artery, vagus nerve, and internal jugular vein. It ends by opening into the angle of junction of the left subclavian vein with the left internal jugular vein. The thoracic duct at its commencement is about equal in diameter to a goose quill, but it diminishes considerably in caliber in the middle of the thorax, and is again dilated just before its termination. It is generally flexuous, and constricted at intervals so as to present a varicose appearance. Not infrequently it divides in the middle of its course into two vessels of unequal size, which soon reunite, or into several branches which form a plexiform interlacement. It occasionally divides at its upper part into two branches, right and left, the left ending in the usual manner, while the right opens into the right subclavian vein, in connection with the right lymphatic duct. The thoracic duct has several valves. At its termination it is provided with a pair, the free borders of which are turned toward the vein, so as to prevent the passage of venous blood into the duct. The cisterna chyli, receptaculum chyli, receives the two lumbar lymphatic trunks, right and left, and the intestinal lymphatic trunk. The lumbar trunks are formed by the union of the efferent vessels from the lateral aortic lymph glands. They receive the lymph from the lower limbs, from the walls and viscera of the pelvis, from the kidneys and suprarenal glands, and the deep lymphatics of the greater part of the abdominal wall. The intestinal trunk receives the lymph from the stomach and intestine, from the pancreas and spleen, and from the lower and front part of the liver. Tributaries. Opening into the commencement of the thoracic duct on either side is a descending trunk from the posterior intercostal lymph glands of the lower 6 or 7 intercostal spaces. In the thorax, the duct is joined, on either side, by a trunk which drains the upper lumbar lymph glands and pierces the crus of the diaphragm. It also receives the efferents from the posterior mediastinal lymph glands and from the posterior intercostal lymph glands of the upper 6 left spaces. In the neck it is joined by the left jugular and left subclavian trunks, and sometimes by the left bronchomediastinal trunk. The last named, however, usually opens independently into the junction of the left subclavian and internal jugular veins. The right lymphatic duct, ductus lymphaticus dexter, about 1.25 cm in length, courses along the medial border of the scalenus anterior at the root of the neck and ends in the right subclavian vein, at its angle of junction with the right internal jugular vein. Its orifice is guarded by two semilunar valves, which prevents the passage of venous blood into the duct. Tributaries. The right lymphatic duct receives the lymph from the right side of the head and neck through the right jugular trunk, from the right upper extremity through the right subclavian trunk, from the right side of the thorax, right lung, right side of the heart, and part of the convex surface of the liver, through the right bronchomediastinal trunk. These three collecting trunks frequently open separately in the angle of union of the two veins. End of section 44.
Section 45 of Gray's Anatomy, Part 3. This is a Librivox recording. All Librivox recordings are in the public domain. For more information or to volunteer, please visit librivox.org. Anatomy of the Human Body, Part 3, by Henry Gray. The lymphatics of the head, face, and neck. The lymph glands of the head. The lymph glands of the head are arranged in the following groups: occipital, posterior auricular, anterior auricular, parotid, facial, deep facial, lingual, retropharyngeal. The occipital glands, lymphoglandulae occipitales, 1 to 3 in number, are placed on the back of the head close to the margin of the trapezius and resting on the insertion of the semispinalis capitis. Their afferent vessels drain the occipital region of the scalp, while their efferents pass to the superior deep cervical glands. The posterior auricular glands, lymphoglandulae auriculares, mastoid glands, usually 2 in number, are situated on the mastoid insertion of the sternocleidomastoideus, beneath the auricularis posterior. Their afferent vessels drain the posterior part of the temporoparietal region, the upper part of the cranial surface of the auricula or pinna, and the back of the external acoustic meatus. Their efferents pass to the superior deep cervical glands. The anterior auricular glands, lymphoglandulae auriculares anteriores, superficial parotid or preauricular glands, from 1 to 3 in number, lie immediately in front of the tragus. Their afferents drain the lateral surface of the auricula and the skin of the adjacent part of the temporal region. Their efferents pass to the superior deep cervical glands. The parotid glands, lymphoglandulae parotidae, form 2 groups in relation with the parotid salivary gland. These are a group embedded in the substance of the gland, and a group of subparotid glands lying on the lateral wall of the pharynx. Occasionally small glands are found in the subcutaneous tissue over the parotid gland. Their afferent vessels drain the root of the nose, the eyelids, the frontotemporal region, the external acoustic meatus, and the tympanic cavity, possibly also the posterior parts of the palate and the floor of the nasal cavity. The efferents of these glands pass to the superior deep cervical glands. The afferents of the subparotid glands drain the nasal part of the pharynx and the posterior parts of the nasal cavities. Their efferents pass to the superior deep cervical glands. The facial glands comprise 3 groups: A. Infraorbital or maxillary, scattered over the infraorbital region from the groove between the nose and cheek to the zygomatic arch. B. Buccinator, 1 or more placed on the buccinator opposite the angle of the mouth. C. Supramandibular, on the outer surface of the mandible, in front of the masseter and in contact with the external maxillary artery and anterior facial vein. Their efferent vessels drain the eyelids, the conjunctiva, and the skin and mucous membrane of the nose and cheek. Their efferents pass to the submaxillary glands. The deep facial glands, lymphoglandulae faciales profunda, internal maxillary glands, are placed beneath the ramus of the mandible, on the outer surface of the pterygoideus externus, in relation to the internal maxillary artery. Their afferent vessels drain the temporal and infratemporal fossae and the nasal part of the pharynx. Their efferents pass to the superior deep cervical glands. The lingual glands, lymphoglandulae linguales, are 2 or 3 small nodules lying on the hyoglossus and under the genioglossus. They form merely glandular substations in the course of the lymphatic vessels of the tongue. The retropharyngeal glands, from 1 to 3 in number, lie in the buccopharyngeal fascia, behind the upper part of the pharynx and in front of the arch of the atlas, being separated, however, from the latter by the longus capitis. Their afferents drain the nasal cavities, the nasal part of the pharynx, and the auditory tubes. Their efferents pass to the superior deep cervical glands. The lymphatic vessels of the scalp are divisible into a. Those of the frontal region, which terminate in the anterior auricular and parotid glands. b. Those of the temporoparietal region, which end in the parotid and posterior auricular glands. And c. Those of the occipital region, which terminate partly in the occipital glands, and partly in a trunk which runs down along the posterior border of the sternocleidomastoideus to end in the inferior deep cervical glands. The lymphatic vessels of the auricula and external acoustic meatus are also divisible into 3 groups. a. An anterior, from the lateral surface of the auricula and anterior wall of the meatus to the anterior auricular glands. b. A posterior, from the margin of the auricula, the upper part of its cranial surface, the internal surface and posterior wall of the meatus to the posterior auricular and superficial deep cervical glands. c. An inferior, from the floor of the meatus and from the lobule of the auricula to the superficial and superior deep cervical glands. The lymphatic vessels of the face are more numerous than those of the scalp. Those from the eyelids and conjunctiva terminate partly in the submaxillary, but mainly in the parotid glands. The vessels from the posterior part of the cheek also pass to the parotid glands, while those from the anterior portion of the cheek, the side of the nose, the upper lip, and the lateral portions of the lower lip end in the submaxillary glands. The deeper vessels from the temporal and infratemporal fossae pass to the deep facial and superior deep cervical glands. The deeper vessels of the cheek and lips end, like the superficial, in the submaxillary glands. Both superficial and deep vessels of the central part of the lower lip run to the submental glands. Lymphatic vessels of the nasal cavities. Those from the anterior parts of the nasal cavities communicate with the vessels of the integument of the nose, and end in the submaxillary glands. Those from the posterior 2/3 of the nasal cavities, and from the accessory air sinuses, pass partly to the retropharyngeal, and partly to the superior deep cervical glands. Lymphatic vessels of the mouth. The vessels of the gums pass to the submaxillary glands. Those of the hard palate are continuous in front with those of the upper gum, but pass backward to pierce the constrictor pharyngeus superior, and end in the superior deep cervical and subparotid glands. Those of the soft palate pass backward and lateralward, and end partly in the retropharyngeal and subparotid, and partly in the superior deep cervical glands. The vessels of the anterior part of the floor of the mouth pass either directly to the inferior glands of the superior deep cervical group, or indirectly through the submental glands. From the rest of the floor of the mouth, the vessels pass to the submaxillary and superior deep cervical glands. The lymphatic vessels of the palatine tonsil, usually 3 to 5 in number, pierce the buccopharyngeal fascia and constrictor pharyngeus superior, and pass between the stylohyoideus and internal jugular vein to the uppermost of the superior deep cervical glands. They end in a gland which lies at the side of the posterior belly of the digastricus on the internal jugular vein. Occasionally 1 or 2 additional vessels run to small glands on the lateral side of the vein under cover of the sternocleidomastoideus. The lymphatic vessels of the tongue are drained chiefly into the deep cervical glands lying between the posterior belly of the digastricus and the superior belly of the omohyoideus. One gland situated at the bifurcation of the common carotid artery is so intimately associated with these vessels that it is known as the principal gland of the tongue. The lymphatic vessels of the tongue may be divided into 4 groups. 1. Apical, from the tip of the tongue to the suprahyoid glands and principal gland of the tongue. 2. Lateral, from the margin of the tongue. Some of these pierce the mylohyoideus to end in the submaxillary glands. Others pass down on the hyoglossus to the superior deep cervical glands. 3. Basal, from the region of the valate papillae to the superior deep cervical glands. And 4. Median, a few of which perforate the mylohyoideus to reach the submaxillary glands, while the majority turn around the posterior border of the muscle to enter the superior deep cervical glands. The lymph glands of the neck. The lymph glands of the neck include the following groups: submaxillary, submental, superficial cervical, anterior cervical, deep cervical. The submaxillary glands, lymphoglandulae submaxillares, 3 to 6 in number, are placed beneath the body of the mandible in the submaxillary triangle, and rest on the superficial surface of the submaxillary salivary gland. One gland, the middle gland of Starr, which lies on the external maxillary artery as it turns over the mandible, is the most constant of the series. Small lymph glands are sometimes found on the deep surface of the submaxillary salivary glands. The afferents of the submaxillary glands drain the medial palpebral commissure, the cheek, the side of the nose, the upper lip, the lateral part of the lower lip, the gums, and the anterior part of the margin of the tongue. Efferent vessels from the facial and submental glands also enter the submaxillary glands. Their efferent vessels pass to the superior deep cervical glands. The submental or suprahyoid glands are situated between the anterior bellies of the digastrici. Their afferents drain the central portions of the lower lip and floor of the mouth, and the apex of the tongue. Their efferents pass partly to the submaxillary glands, and partly to a gland of the deep cervical group situated on the internal jugular vein at the level of the cricoid cartilage. The superficial cervical glands, lymphoglandulae cervicalis superficialis, lie in close relationship with the external jugular vein as it emerges from the parotid gland, and, therefore, superficial to the sternocleidomastoideus. Their afferents drain the lower parts of the auricula and parotid region, while their efferents pass around the anterior margin of the sternocleidomastoideus to join the superior deep cervical glands. The anterior cervical glands form an irregular and inconstant group on the front of the larynx and trachea. They may be divided into a superficial set, placed on the anterior jugular vein; b. a deeper set, which is further subdivided into prelaryngeal, on the middle cricothyroid ligament, and pretracheal, on the front of the trachea. This deeper set drains the lower part of the larynx, the thyroid gland, and the upper part of the trachea. Its efferents pass to the lowest of the superior deep cervical glands. The deep cervical glands, lymphoglandulae cervicalis profundi, are numerous and of large size. They form a chain along the carotid sheath lying by the side of the pharynx, esophagus, and trachea, and extending from the base of the skull to the root of the neck. They are usually described in 2 groups. 1. The superior deep cervical glands lying under the sternocleidomastoideus, in close relation with the accessory nerve and the internal jugular vein, some of the glands lying in front of, and others behind the vessel. 2. The inferior deep cervical glands extending beyond the posterior margin of the sternocleidomastoideus into the supraclavicular triangle, where they are closely related to the brachial plexus and subclavian vein. A few minute paratracheal glands are situated alongside the recurrent nerves on the lateral aspects of the trachea and esophagus. The superior deep cervical glands drain the occipital portion of the scalp, the auricula, the back of the neck, a considerable part of the tongue, the larynx, thyroid gland, trachea, nasal part of the pharynx, nasal cavities, palate, and esophagus. They receive also the efferent vessels from all the other glands of the head and neck, except those from the inferior deep cervical glands. The inferior deep cervical glands drain the back of the scalp and neck, the superficial pectoral region, part of the arm, and occasionally part of the superior surface of the liver. In addition, they receive vessels from the superior deep cervical glands. The efferents of the superior deep cervical glands pass partly to the inferior deep cervical glands, and partly to a trunk which unites with the efferent vessel of the inferior deep cervical glands and forms the jugular trunk. On the right side this trunk ends in the junction of the internal jugular and subclavian veins. On the left side it joins the thoracic duct. The lymphatic vessels of the skin and muscles of the neck pass to the deep cervical glands. From the upper part of the pharynx the lymphatic vessels pass to the retropharyngeal, from the lower part to the deep cervical glands. From the larynx two sets of vessels arise, an upper and a lower. The vessels of the upper set pierce the hyothyroid membrane and join the superior deep cervical glands. Of the lower set, some pierce the conus elasticus and join the pretracheal and prelaryngeal glands. Others run between the cricoid and first tracheal ring and enter the inferior deep cervical glands. The lymphatic vessels of the thyroid gland consist of two sets, an upper, which accompanies the superior thyroid artery and enters the superior deep cervical glands, and a lower, which runs partly to the pretracheal glands and partly to the small paratracheal glands which accompany the recurrent nerves. These latter glands receive also the lymphatic vessels from the cervical portion of the trachea. End of section 45.
Section 46 of Gray's Anatomy, Part 3. This is a Librivox recording. All Librivox recordings are in the public domain. For more information or to volunteer, please visit Librivox.org. Anatomy of the Human Body, Part 3, by Henry Gray. Section 46. The Lymphatics of the Upper Extremity. The lymph glands of the upper extremity. The lymph glands of the upper extremity are divided into two sets, superficial and deep. The superficial lymph glands are few and of small size. One or two supratrochlear glands are placed above the medial epicondyle of the humerus, medial to the basilic vein. Their afferents drain the middle, ring, and little fingers, the medial portion of the hand, and the superficial area over the ulnar side of the forearm. These vessels are, however, in free communication with the other lymphatic vessels of the forearm. Their efferents accompany the basilic vein and join the deeper vessels. One or two deltoidiopectoral glands are found beside the cephalic vein, between the pectoralis major and deltoideus, immediately below the clavicle. They are situated in the course of the external collecting trunks of the arm. The deep lymph glands are chiefly grouped in the axilla, although a few may be found in the forearm, in the course of the radial, ulnar, and interosseous vessels, and in the arm along the medial side of the brachial artery. The axillary glands, lymphoglandulae axillaries, are of large size, vary from 20 to 30 in number, and may be arranged in the following groups. 1. A lateral group of from 4 to 6 glands lies in relation to the medial and posterior aspects of the axillary vein. The afferents of these glands drain the whole arm with the exception of that portion whose vessels accompany the cephalic vein. The efferent vessels pass partly to the central and subclavicular groups of axillary glands and partly to the inferior deep cervical glands. 2. An anterior or pectoral group consists of 4 or 5 glands along the lower border of the pectoralis minor, in relation with the lateral thoracic artery. Their afferents drain the skin and muscles of the anterior and lateral thoracic walls, and the central and lateral parts of the nama. Their efferents pass partly to the central and partly to the subclavicular groups of axillary glands. 3. A posterior or subscapular group of 6 or 7 glands is placed along the lower margin of the posterior wall of the axilla in the course of the subscapular artery. The afferents of this group drain the skin and muscles of the lower part of the back of the neck and of the posterior thoracic wall. Their efferents pass to the central group of axillary glands. 4. A central or intermediate group of 3 or 4 large glands is embedded in the adipose tissue near the base of the axilla. Its afferents are the efferent vessels of all the preceding groups of axillary glands. Its efferents pass to the subclavicular group. 5. A medial or subclavicular group of 6 to 12 glands is situated partly posterior to the upper portion of the pectoralis minor, and partly above the upper border of this muscle. Its only direct territorial afferents are those which accompany the cephalic vein, and one which drains the upper peripheral part of the mamma, but it receives the efferents of all the other axillary glands. The efferent vessels of the subclavicular group unite to form the subclavian trunk, which opens either directly into the junction of the internal jugular and subclavian veins, or into the jugular lymphatic trunk. On the left side it may end in the thoracic duct. A few efferents from the subclavicular glands usually pass to the inferior deep cervical glands. The lymphatic vessels of the upper extremity. The lymphatic vessels of the upper extremity are divided into 2 sets, superficial and deep. The superficial lymphatic vessels commence in the lymphatic plexus, which everywhere pervades the skin. The meshes of the plexus are much finer in the palm and on the flexor aspect of the digits than elsewhere. The digital plexuses are drained by a pair of vessels which run on the sides of each digit and incline backward to reach the dorsum of the hand. From the dense plexus of the palm, vessels pass in different directions, viz., upward toward the wrist, downward to join the digital vessels, medialward to join the vessels on the ulnar border of the hand, and lateralward to those on the thumb. Several vessels from the central part of the plexus unite to form a trunk, which passes around the metacarpal bone of the index finger to join the vessels on the back of that digit and on the back of the thumb. Running upward in front of and behind the wrist, the lymphatic vessels are collected into radial, median, and ulnar groups, which accompany respectively the cephalic, median, and basilic veins in the forearm. A few of the ulnar lymphatics end in the supratrochlear glands, but the majority pass directly to the lateral group of axillary glands. Some of the radial vessels are collected into a trunk, which ascends with the cephalic vein to the deltoidiopectoral glands. The efferents from this group pass either to the subclavicular axillary glands or to the inferior cervical glands. The deep lymphatic vessels accompany the deep blood vessels. In the forearm they consist of 4 sets, corresponding with the radial, ulnar, volar, and dorsal interosseous arteries. They communicate at intervals with the superficial lymphatics, and some of them end in the glands which are occasionally found beside the arteries. In their course upward, a few end in the glands which lie upon the brachial artery, but most of them pass to the lateral group of axillary glands. End of section 46. Recording by Selena Arter. Section 47 of Gray's Anatomy, Part 3. This is a Librivox recording. All Librivox recordings are in the public domain. For more information or to volunteer, please visit Librivox.org. Anatomy of the Human Body, Part 3, by Henry Gray. The lymphatics of the lower extremity. The lymph glands of the lower extremity. The lymph glands of the lower extremity consist of the anterior tibial gland and the popliteal and inguinal glands. The anterior tibial gland, lymphoglandula tibialis anterior, is small and inconstant. It lies on the interosseous membrane in relation to the upper part of the anterior tibial vessels, and constitutes a substation in the course of the anterior tibial lymphatic trunks. The popliteal glands, lymphoglandulae popliteae, small in size and some 6 or 7 in number, are embedded in the fat contained in the popliteal fossa. One lies immediately beneath the popliteal fascia, near the terminal part of the small saphenous vein, and drains the region from which this vein derives its tributaries. Another is placed between the popliteal artery and the posterior surface of the knee joint. It receives the lymphatic vessels from the knee joint together with those which accompany the genicular arteries. The others lie at the sides of the popliteal vessels and receive as efferents the trunks which accompany the anterior and posterior tibial vessels. The efferents of the popliteal glands pass almost entirely alongside the femoral vessels to the deep inguinal glands, but a few may accompany the great saphenous vein and end in the glands of the superficial subinguinal group. The inguinal glands, lymphoglandulae inguinales, from 12 to 20 in number, are situated at the upper part of the femoral triangle. They may be divided into 2 groups by a horizontal line at the level of the termination of the great saphenous vein. Those lying above this line are termed the superficial inguinal glands, and those below it the subinguinal glands, the latter group consisting of a superficial and a deep set. The superficial inguinal glands form a chain immediately below the inguinal ligament. They receive as afferents lymphatic vessels from the integument of the penis, scrotum, perineum, buttock, and abdominal wall below the level of the umbilicus. The superficial subinguinal glands, lymphoglandulae subinguinales superficiales, are placed on either side of the upper part of the great saphenous vein. Their efferents consist chiefly of the superficial lymphatic vessels of the lower extremity, but they also receive some of the vessels which drain the integument of the penis, scrotum, perineum, and buttock. The deep subinguinal glands, lymphoglandulae subinguinales profundae, vary from 1 to 3 in number, and are placed under the fascia lata, on the medial side of the femoral vein. When 3 are present, the lowest is situated just below the junction of the great saphenous and femoral veins, the middle in the femoral canal, and the highest in the lateral part of the femoral ring. The middle one is the most inconstant of the 3, but the highest, the gland of Cloquet or Rosenmuller, is also frequently absent. They receive as afferents the deep lymphatic trunks, which accompany the femoral vessels. The lymphatics from the glands penis, vel, clitoridis, and also some of the efferents from the superficial subinguinal glands. The lymphatic vessels of the lower extremity. The lymphatic vessels of the lower extremity consist of 2 sets, superficial and deep, and in their distribution correspond closely with the veins. The superficial lymphatic vessels lie in the superficial fascia, and are divisible into 2 groups, a medial, which follows the course of the great saphenous vein, and a lateral, which accompanies the small saphenous vein. The vessels of the medial group are larger and more numerous than those of the lateral group, and commence on the tibial side and dorsum of the foot. They ascend both in front of and behind the medial malleolus, run up the leg with the great saphenous vein, pass with it behind the medial condyle of the femur and accompany it to the groin, where they end in the subinguinal group of superficial glands. The vessels of the lateral group arise from the fibular side of the foot. Some ascend in front of the leg and just below the knee, cross the tibia to join the lymphatics on the medial side of the thigh. Others pass behind the lateral malleolus and, accompanying the small saphenous vein, enter the popliteal glands. The deep lymphatic vessels are few in number, and accompany the deep blood vessels. In the leg, they consist of 3 sets, the anterior tibial, posterior tibial, and peroneal, which accompany the corresponding blood vessels, 2 or 3 with each artery. They enter the popliteal lymph glands. The deep lymphatic vessels of the gluteal and ischial regions follow the course of the corresponding blood vessels. Those accompanying the superior gluteal vessels end in a gland which lies on the intrapelvic portion of the superior gluteal artery near the upper border of the greater sciatic foramen. Those following the inferior gluteal vessels traverse 1 or 2 small glands, which lie below the piriformis muscle and end in the hypogastric glands. End of section 47. Recording by Selena Arter.
Section 48 of Gray's Anatomy, Part 3. This is a Librivox recording. All Librivox recordings are in the public domain. For more information or to volunteer, please visit Librivox.org. Recording by Jennifer Stearns. Anatomy of the Human Body, Part 3, by Henry Gray. Lymphatics of the Abdomen and Pelvis, Part 1. The lymph glands of the abdomen and pelvis may be divided from the situations into a. Parietal, lying behind the peritoneum and in close association with the larger blood vessels, and b. Visceral, which are found in relation to the visceral arteries. The parietal glands include the following groups: external iliac, common iliac, epigastric, iliac circumflex, hypogastric, sacral, lumbar, lateral aortic, pre-aortic, retroaortic. The external iliac glands, from 8 to 10 in number, lie along the external iliac vessels. They are arranged in 3 groups, one on the lateral, another on the medial, and a third on the anterior aspect of the vessels. The third group is, however, sometimes absent. Their principal afferents are derived from the inguinal and subinguinal glands. The deep lymphatics of the abdominal wall below the umbilicus and of the adductor region of the thigh, and the lymphatics from the glands penis vel clitoridis, the membranous urethra, the prostate, the fundus of the bladder, the cervix uteri, and upper part of the vagina. The common iliac glands, 4 to 6 in number, are grouped behind and on the sides of the common iliac artery, 1 or 2 being placed below the bifurcation of the aorta, in front of the 5th lumbar vertebra. They drain chiefly the hypogastric and external iliac glands, and their efferents pass to the lateral aortic glands. The epigastric glands, lymphoglandulo epigastrico, 3 or 4 in number, are placed alongside the lower portion of the inferior epigastric vessels. The iliac circumflex glands, 2 to 4 in number, are situated along the course of the deep iliac circumflex vessels. They are sometimes absent. The hypogastric glands, lymphoglandulo hypogastrico, internal iliac gland, surround the hypogastric vessels and receive the lymphatics corresponding to the distribution of the branches of the hypogastric artery. That is, they receive lymphatics from the pelvic viscera, from the deeper parts of the perineum, including the membranous and cavernous portions of the urethra, and from the buttock and back of the thigh. An obturator gland is sometimes seen in the upper part of the obturator foramen. The sacral glands are placed in the concavity of the sacrum, in relation to the middle and lateral sacral arteries. They receive lymphatics from the rectum and posterior wall of the pelvis. The efferents of the hypogastric group end in the common iliac glands. The lumbar glands, lymphoglandulo lumbales, are very numerous, and consist of right and left lateral aortic, pre-aortic, and retroaortic groups. The right lateral aortic glands are situated partly in front of the inferior vena cava, near the termination of the renal vein, and partly behind it on the origin of the psoas major, and on the right crus of the diaphragm. The left lateral aortic glands form a chain on the left side of the abdominal aorta in front of the origin of the psoas major and left crus of the diaphragm. The glands on either side receive a. the efferents of the common iliac glands, b. the lymphatics from the testes in the male and from the ovary, uterine tube, and body of the uterus in the female, c. the lymphatics from the kidney and suprarenal gland, and d. the lymphatics draining the lateral abdominal muscles and accompanying the lumbar veins. Most of the efferent vessels of the lateral aortic glands converge to form the right and left lumbar trunks, which join the cisterna chyli, but some enter the pre and retroaortic glands, and others pierce the crura of the diaphragm to join the lower end of the thoracic duct. The pre-aortic glands lie in front of the aorta and may be divided into celiac, superior mesenteric, and inferior mesenteric groups, arranged around the origins of the corresponding arteries. They receive a few vessels from the lateral aortic glands, but their principal efferents are derived from the viscera supplied by the three arteries with which they are associated. Some of the efferents pass to the retroaortic glands, but the majority unite to form the intestinal trunk, which enters the cisterna chyli. The retroaortic glands are passed below the cisterna chyli on the bodies of the third and fourth lumbar vertebrae. They receive lymphatic trunks from the lateral and pre-aortic glands, while their efferents end in the cisterna chyli. The lymphatic vessels of the abdomen and pelvis. The lymphatic vessels of the walls of the abdomen and pelvis may be divided into two sets, superficial and deep. The superficial vessels follow the course of the superficial blood vessels and converge to the superficial inguinal glands. Those derived from the integument of the front of the abdomen below the umbilicus follow the course of the superficial epigastric vessels, and those from the sides of the lumbar part of the abdominal wall pass along the crest of the ilium, with the superficial iliac circumflex vessels. The superficial lymphatic vessels of the gluteal region turn horizontally around the buttock and join the superficial inguinal and subinguinal glands. The deep vessels run alongside the principal blood vessels. Those are the parietes of the pelvis, which accompany the superior and inferior gluteal and obturator vessels, follow the course of the hypogastric artery, and ultimately join the lateral aortic glands. Lymphatic vessels of the perineum and external genitals. The lymphatic vessels of the perineum, of the integument of the penis, and of the scrotum or vulva follow the course of the external pudendal vessels and end in the superficial inguinal and subinguinal glands. Those of the glans penis vel clitoridis terminate partly in the subinguinal glands and partly in the external iliac glands. The visceral glands are associated with the branches of the celiac, superior and inferior mesenteric arteries. Those related to the branches of the celiac artery form three sets, gastric, hepatic, and pancreaticorenal. The gastric glands consist of two sets, superior and inferior. The superior gastric glands, lymphoglandulo-gastrico-superiores, accompany the left gastric artery and are divisible into three groups, namely, a. upper, on the stem of the artery, b. lower, accompanying the descending branches of the artery along the cardiac half of the lesser curvature of the stomach, between the two layers of the lesser omentum, and c. pericardial, outlying members of the gastric glands, disposed in a manner comparable to a chain of beads around the neck of the stomach. Jameson and Dobson. They receive their efferents from the stomach. Their afferents pass to the celiac group of the pre-aortic glands. The inferior gastric glands. Lymphoglandulo-gastrico-inferiores, right gastric epilogic gland, 4 to 7 in number, lie between the two layers of the greater omentum along the pyloric half of the greater curvature of the stomach. The hepatic glands, lymphoglandulo-hepatico, consist of the following groups: a. hepatic, on the stem of the hepatic artery, and extending upward along the common bile duct, between the two layers of the lesser omentum, as far as the porta hepatis. The cystic gland, a member of this group, is placed near the neck of the gall bladder. b. subpyloric, 4 or 5 in number, in close relation to the bifurcation of the gastro-duodenal artery. In the angle between the superior and descending parts of the duodenum, an outlying member of this group is sometimes found above the duodenum on the right gastric pyloric artery. The glands of the hepatic chain receive efferents from the stomach, duodenum, liver, gallbladder, and pancreas. Their efferents join the celiac group of pre-aortic glands. The pancreaticorenal glands, lymphoglandular pancreaticorenalis, splenic glands, accompany the renal splenic artery and are situated in relation to the posterior surface of the upper border of the pancreas. One or two members of this group are found in the gastrorenal ligament (Jamieson and Dobson) in the work that was cited. Their afferents are derived from the stomach, spleen, and pancreas. Their efferents join the celiac group of the pre-aortic glands. The superior mesenteric glands may be divided into three principal groups: mesenteric, ileocholic, and mesocolic. The mesenteric glands, lymphoglandulo-mesenterico, lie between the layers of the mesentery. They vary from 100 to 150 in number, and may be grouped into three sets, namely, one lying close to the wall of the small intestine, among the terminal twigs of the superior mesenteric artery; a second, in relation to the loops and primary branches of the vessels; and a third along the trunk of the artery. The ileocholic glands, from 10 to 20 in number, form a chain around the ileocholic artery, but show a tendency to subdivision into two groups, one near the duodenum, and another on the lower part of the trunk of the artery. Where the vessel divides into its terminal branches, the chain is broken up into several groups, namely, a. Ileal, the region in the ileal branch of the artery; b. Anterior ileocholic, usually of 3 glands, in the ileocholic fold, near the wall of the cecum; c. Posterior ileocholic, mostly placed in the angle between the ileum and the colon, but partly lying behind the cecum at its junction with the ascending colon; d. A single gland between the layers of the mesenteriole of the vermiform process; e. Right colic, along the medial side of the ascending colon. The mesocolic glands, lymphoglandulo-mesocolico, are numerous, and lie between the layers of the transverse mesocolon, in close relation to the transverse colon. They are best developed in the neighbourhood of the right and left colic flexures. One or two small glands are occasionally seen along the trunk of the right colic artery, and others are found in relation to the trunk and branches of the middle colic artery. The superior mesenteric glands receive afferents from the jejunum, ileum, cecum, vermiform process, and the ascending and transverse parts of the colon. Their efferents pass to the pre-aortic glands. The inferior mesenteric glands consist of a. Small glands on the branches of the left colic and sigmoid arteries; b. A group in the sigmoid mesocolon around the superior hemorrhoidal artery; and c. A pararectal group in contact with the muscular coat of the rectum. They drain the descending iliac and sigmoid parts of the colon and the upper part of the rectum. Their efferents pass to the pre-aortic glands. End of Section 48. Recording by Jennifer Stearns, Concord, New Hampshire. Section 49 of Gray's Anatomy, Part 3. This is a Librivox recording. All Librivox recordings are in the public domain. For more information, or to volunteer, please visit Librivox.org. Recording by Jennifer Stearns. Anatomy of the Human Body, Part 3, by Henry Gray. Lymphatics of the Abdomen and Pelvis, Part 2. The lymphatic vessels of the abdomen and pelvic viscera. The lymphatic vessels of the abdominal and pelvic visceral consist of 1. Those of the subdiaphragmatic portion of the digestive tube and its associated glands, the liver and pancreas; 2. Those of the spleen and suprarenal glands; 3. Those of the urinary organs; 4. Those of the reproductive organs. 1. The lymphatic vessels of the subdiaphragmatic portion of the digestive tube are situated partly in the mucous membrane and partly in the seromuscular coats, but as the former system drains into the latter, the two may be considered as one. The lymphatic vessels of the stomach are continuous at the cardiac orifice with those of the esophagus, and at the pylorus with those of the duodenum. They mainly follow the blood vessels, and may be arranged in 4 sets. Those of the first set accompany the branches of the left gastric artery, receiving tributaries from a large area on either surface of the stomach, and terminate in the superior gastric glands. Those of the second set drain the fundus and body of the stomach on the left of a line drawn vertically from the esophagus. They accompany, more or less closely, the short gastric and left gastroepiglottic arteries, and end in the pancreaticolenal glands. The vessels of the third set drain the right portion of the greater curvature as far as the pyloric portion, and end in the inferior gastric glands, the efferents of which pass to the subpyloric group. Those of the fourth set drain the pyloric portion and pass to the hepatic and subpyloric glands and to the superior gastric glands. The lymphatic vessels of the duodenum consist of an anterior and posterior set, which open into a series of small pancreatical duodenal glands on the anterior and posterior aspects of the groove behind the head of the pancreas and the duodenum. The efferents of these glands run in 2 directions, upward to the hepatic glands and downward to the preaortic glands around the origin of the superior mesenteric artery. The lymphatic vessels of the jejunum and ileum are termed lacteals, from the milk-white fluid they contain during intestinal digestion. They run between the layers of the mesentery and enter the mesenteric glands, the efferents of which end in the preaortic glands. The lymphatic vessels of the vermiform process and cecum are numerous, since in the wall of this process there is a large amount of adenoid tissue. From the body and tail of the vermiform process, 8 to 15 vessels ascend between the layers of the mesenteriole, 1 or 2 being interrupted in the gland which lies between the layers of this peritoneal fold. They unite to form 3 or 4 vessels, which end partly in the lower and partly in the upper glands of the ileocolic chain. The vessels from the root of the vermiform process and from the cecum consist of an anterior and posterior group. The anterior vessels pass in front of the cecum and end in the anterior ileocolic glands and in the upper and lower glands of the ileocolic chain. The posterior vessels ascend over the back of the cecum and terminate in the posterior ileocolic glands and in the lower glands of the ileocolic chain. Lymphatic vessels of the colon. The lymphatic vessels of the ascending and transverse parts of the colon finally end in the mesenteric glands, after traversing the right colic and mesocolic glands. Those of the descending and iliac sigmoid parts of the colon are interrupted by the small glands on the branches of the left colic and sigmoid arteries, and ultimately end in the preaortic glands around the origin of the inferior mesenteric artery. Lymphatic vessels of the anus, anal canal, and rectum. The lymphatics from the anus pass forward and end with those of the integument of the perineum and scrotum in the superficial inguinal glands. Those from the anal cavity accompany the middle and inferior hemorrhoidal arteries and end in the hypogastric glands, while the vessels from the rectum traverse the pararectal glands and pass to those in the sigmoid mesocolon. The efferents of the latter terminate in the preaortic glands around the origin of the inferior mesenteric artery. The lymphatic vessels of the liver are divisible into 2 sets, superficial and deep. The former arise in the subperitoneal areolar tissue over the entire surface of the organ, and may be grouped into A, those on the convex surface, B, those on the inferior surface. A. On the convex surface. The vessels from the back part of this surface reach their terminal glands by 3 different routes. The vessels of the middle set, 5 or 6 in number, pass through the vena cava foramen in the diaphragm and end in 1 or 2 glands which are situated around the terminal part of the inferior vena cava. A few vessels on the left side pass backward toward the esophageal hiatus and terminate in the pericardial group of superior gastric glands. The vessels from the right side, 1 or 2 in number, run on the abdominal surface of the diaphragm, and after crossing its right crus, end in the preaortic glands which surround the origin of the celiac artery. From the portions of the right and left lobes adjacent to the falciform ligament, the lymphatic vessels converge to form 2 trunks, one of which accompanies the inferior vena cava through the diaphragm, and ends in the glands around the terminal part of this vessel. The other runs downward and forward, and turning around the anterior sharp margin of the liver, accompanies the upper part of the ligamentum teres, and ends in the upper hepatic glands. From the anterior surface, a few additional vessels turn around the anterior sharp margin to reach the upper hepatic glands. B. On the inferior surface, the vessels from this surface mostly converge to the porta hepatis and accompany the deep lymphatics emerging from the porta to the hepatic glands. One or two from the posterior parts of the right and caudate lobes accompany the inferior vena cava through the diaphragm, and end in the glands around the terminal part of this vein. The deep lymphatics converge to ascending and descending trunks. The ascending trunks accompany the hepatic veins and pass through the diaphragm to end in the glands around the terminal part of the inferior vena cava. The descending trunks emerge from the porta hepatis and end in the hepatic glands. The lymphatic vessels of the gallbladder pass to the hepatic glands in the porta hepatis. Those of the common bile duct to the hepatic glands alongside the duct and to the upper pancreato-duodenal glands. The lymphatic vessels of the pancreas follow the course of its blood vessels. Most of them enter the pancreatorenal glands, but some end in the pancreato-duodenal glands, and others in the pre-aortic glands near the origin of the superior mesenteric artery. 2. The lymphatic vessels of the spleen and suprarenal glands. The lymphatic vessels of the spleen, both superficial and deep, pass to the pancreato-renal glands. The lymphatic vessels of the suprarenal glands usually accompany the suprarenal veins and end in the lateral aortic glands. Occasionally, some of them pierce the crura of the diaphragm and end in the glands of the posterior mediastinum. 3. The lymphatic vessels of the urinary organs. The lymphatic vessels of the kidney form three plexuses, one in the substance of the kidney, a second beneath its fibrous capsule, and a third in the perinephric fat. The second and third communicate freely with each other. The vessels from the plexus in the kidney substance converge to form 4 or 5 trunks which issue at the hilum. Here they are joined by vessels from the plexus under the capsule, and, following the course of the renal vein, end in the lateral aortic glands. The perinephric plexus is drained directly into the upper lateral aortic glands. The lymphatic vessels of the ureter run in different directions. Those from its upper portion end partly in the efferent vessels of the kidney and partly in the lateral aortic glands. Those from the portion immediately above the brim of the lesser pelvis are drained into the common iliac glands, while the vessels from the interpelvic portion of the tube either join the efferents from the bladder or end in the hypogastric glands. The lymphatic vessels of the bladder originate in 2 plexuses, an intra and extramuscular, it being generally admitted the mucous membrane is devoid of lymphatics. Footnote: some authorities maintain that a plexus of lymphatic vessels does exist in the mucous membrane of the bladder. Consult Médecine opératoire des voies urinaires par J. Eberin, Paris, 1909. End footnotes. The efferent vessels are arranged in 2 groups, one from the anterior and another from the posterior surface of the bladder. The vessels from the anterior surface pass to the external iliac glands, but in their course minute glands are situated. These minute glands are arranged in 2 groups, an anterior vesicle in front of the bladder, and a lateral vesicle in relation to the lateral umbilical ligament. The vessels from the posterior surface pass to the hypogastric, external, and common iliac glands. Those draining the upper part of this surface traverse the lateral vesical glands. The lymphatic vessels of the prostate terminate chiefly in the hypogastric and sacral glands. But one trunk from the posterior surface ends in the external iliac glands, and another from the anterior surface joins the vessels which drain the membranous part of the urethra. Lymphatic vessels of the urethra. The lymphatics of the cavernous portion of the urethra accompany those of the glans penis, and terminate with them in the deep subinguinal and external iliac glands. Those of the membranous and prostatic portions, and those of the whole urethra in the female, pass to the hypogastric glands. 4. The lymphatic vessels of the reproductive organs. The lymphatic vessels of the testes consist of 2 sets, superficial and deep, the former commencing on the surface of the tunica vaginalis, the latter in the epididymis and body of the testes. They form from 4 to 8 collecting trunks which ascend with the spermatic veins in the spermatic cord and along the front of the psoas major to the level where the spermatic vessels cross the ureter and end in the lateral and pre-aortic groups of lumbar glands. The lymphatic vessels of the ductus deferens pass to the external iliac glands, those of the vesiculae seminalis partly to the hypogastric and partly to the external glands. The lymphatic vessels of the ovary are similar to those of the testes, and descend with the ovarian artery to the lateral and pre-aortic glands. The lymphatic vessels of the uterine tube pass partly with those of the ovary and partly with those of the uterus. The lymphatic vessels of the uterus consist of two sets, superficial and deep, the former being placed beneath the peritoneum, the latter in the substance of the organ. The lymphatics of the cervix uteri run in three directions, transversely to the external iliac glands, posterolaterally to the hypogastric glands, and posteriorly to the common iliac glands. The majority of the vessels of the body and fundus of the uterus pass lateralward in the broad ligaments, and are continued up with the ovarian vessels to the lateral and pre-aortic glands. A few, however, run to the external iliac glands, and one or two to the superficial inguinal glands. In the unimpregnated uterus, the lymphatic vessels are very small, but during gestation they are greatly enlarged. The lymphatic vessels of the vagina are carried in three directions, those of the upper part of the vagina to the external iliac glands, those of the middle part to the hypogastric glands, and those of the lower part to the common iliac glands. On the course of the vessels from the middle and lower parts small glands are situated. Some lymphatic vessels from the lower part of the vagina join those of the vulva and pass to the superficial inguinal glands. The lymphatics of the vagina anastomose with those of the cervix uteri, vulva, and rectum, but not with those of the bladder. End of section 49. Recording by Jennifer Stearns, Concord, New Hampshire.
Section 50 of Gray's Anatomy, Part 3. This is a Librivox recording. All Librivox recordings are in the public domain. For more information or to volunteer, please visit librivox.org. Read by Laurie Anne Walden. Anatomy of the Human Body, Part 3, by Henry Gray. The lymphatic vessels of the thorax. The lymph glands of the thorax may be divided into parietal and visceral, the former being situated in the thoracic wall, the latter in relation to the viscera. The parietal lymph glands include the sternal, intercostal, and diaphragmatic glands. 1. The sternal glands, lymphoglanduli sternales, internal mammary glands, are placed at the anterior ends of the intercostal spaces, by the side of the internal mammary artery. They derive afferents from the mamma, from the deeper structures of the anterior abdominal wall above the level of the umbilicus, from the upper surface of the liver through a small group of glands which lie behind the xiphoid process, and from the deeper parts of the anterior portion of the thoracic wall. Their efferents usually unite to form a single trunk on either side. This may open directly into the junction of the internal jugular and subclavian veins, or that of the right side may join the right subclavian trunk, and that of the left the thoracic duct. 2. The intercostal glands, lymphoglanduli intercostales, occupy the posterior parts of the intercostal spaces, in relation to the intercostal vessels. They receive the deep lymphatics from the posterolateral aspect of the chest. Some of these vessels are interrupted by small lateral intercostal glands. The efferents of the glands in the lower 4 or 5 spaces unite to form a trunk, which descends and opens either into the cisterna chyli or into the commencement of the thoracic duct. The efferents of the glands in the upper spaces of the left side end in the thoracic duct, those of the corresponding right spaces in the right lymphatic duct. 3. The diaphragmatic glands lie on the thoracic aspect of the diaphragm and consist of 3 sets, anterior, middle, and posterior. The anterior set comprises a. 2 or 3 small glands behind the base of the xiphoid process, which receive afferents from the convex surface of the liver, and b. 1 or 2 glands on either side near the junction of the 7th rib with its cartilage, which receive lymphatic vessels from the front part of the diaphragm. The efferent vessels of the anterior set pass to the sternal glands. The middle set consists of 2 or 3 glands on either side close to where the phrenic nerves enter the diaphragm. On the right side some of the glands of this group lie within the fibrous sac of the pericardium, on the front of the termination of the inferior vena cava. The afferents of this set are derived from the middle part of the diaphragm, those on the right side also receiving afferents from the convex surface of the liver. Their efferents pass to the posterior mediastinal glands. The posterior set consists of a few glands situated on the back of the crura of the diaphragm, and connected, on the one hand, with the lumbar glands, and, on the other, with the posterior mediastinal glands. The superficial lymphatic vessels of the thoracic wall ramify beneath the skin, and converge to the axillary glands. Those over the trapezius and latissimus dorsi run forward, and unite to form about 10 or 12 trunks which end in the subscapular group. Those over the pectoral region, including the vessels from the skin covering the peripheral part of the mamma, run backward, and those over the serratus anterior upward to the pectoral group. Others near the lateral margin of the sternum pass inward between the rib cartilages, and end in the sternal glands, while the vessels of opposite sides anastomose across the front of the sternum. A few vessels from the upper part of the pectoral region ascend over the clavicle to the supraclavicular group of cervical glands. The lymphatic vessels of the mamma originate in a plexus in the interlobular spaces and on the walls of the galactophorus ducts. Those from the central part of the gland pass to an intricate plexus situated beneath the areola, a plexus which receives also the lymphatics from the skin over the central part of the gland, and those from the areola and nipple. Its efferents are collected into 2 trunks which pass to the pectoral group of axillary glands. The vessels which drain the medial part of the mamma pierce the thoracic wall and end in the sternal glands, while a vessel has occasionally been seen to emerge from the upper part of the mamma and, piercing the pectoralis major, terminate in the subclavicular glands. The deep lymphatic vessels of the thoracic wall consist of 1. The lymphatics of the muscles which lie on the ribs. Most of these end in the axillary glands, but some from the pectoralis major pass to the sternal glands. 2. The intercostal vessels which drain the intercostales and parietal pleura. Those draining the intercostales externi run backward, and, after receiving the vessels which accompany the posterior branches of the intercostal arteries, end in the intercostal glands. Those of the intercostales interni and parietal pleura consist of a single trunk in each space. These trunks run forward in the subpleural tissue, and the upper 6 open separately into the sternal glands, or into the vessels which unite them. Those of the lower spaces unite to form a single trunk which terminates in the lowest of the sternal glands. 3. The lymphatic vessels of the diaphragm which form 2 plexuses, one on its thoracic and another on its abdominal surface. These plexuses anastomose freely with each other, and are best marked on the parts covered respectively by the pleurae and peritoneum. That on the thoracic surface communicates with the lymphatics of the costal and mediastinal parts of the pleura, and its efferents consist of 3 groups: A. Anterior, passing to the gland which lies near the junction of the 7th rib with its cartilage. B. Middle, to the glands on the esophagus and to those around the termination of the inferior vena cava. And C. Posterior, to the glands which surround the aorta at the point where this vessel leaves the thoracic cavity. The plexus on the abdominal surface is composed of fine vessels, and anastomoses with the lymphatics of the liver, and, at the periphery of the diaphragm, with those of the subperitoneal tissue. The efferents from the right half of this plexus terminate partly in a group of glands on the trunk of the corresponding inferior phrenic artery, while others end in the right lateral aortic glands. Those from the left half of the plexus pass to the pre- and lateral aortic glands, and to the glands on the terminal portion of the esophagus. The visceral lymph glands consist of 3 groups, that is, anterior mediastinal, posterior mediastinal, and tracheobronchial. The anterior mediastinal glands, lymphoglandulae mediastinales anteriores, are placed in the anterior part of the superior mediastinal cavity, in front of the aortic arch, and in relation to the innominate veins and the large arterial trunks which arise from the aortic arch. They receive afferents from the thymus and pericardium, and from the sternal glands. Their efferents unite with those of the tracheobronchial glands to form the right and left bronchomediastinal trunks. The posterior mediastinal glands, lymphoglandulae mediastinales posteriores, lie behind the pericardium in relation to the esophagus and descending thoracic aorta. Their afferents are derived from the esophagus, the posterior part of the pericardium, the diaphragm, and the convex surface of the liver. Their efferents mostly end in the thoracic duct, but some join the tracheobronchial glands. The tracheobronchial glands form four main groups: A. Tracheal, on either side of the trachea; B. Bronchial, in the angles between the lower part of the trachea and bronchi, and in the angle between the two bronchi; C. Bronchopulmonary, in the hilus of each lung; and D. Pulmonary, in the lung substance, on the larger branches of the bronchi. The afferents of the tracheobronchial glands drain the lungs and bronchi, the thoracic part of the trachea, and the heart. Some of the efferents of the posterior mediastinal glands also end in this group. Their efferent vessels ascend upon the trachea and unite with efferents of the internal mammary and anterior mediastinal glands to form the right and left bronchomediastinal trunks. The right bronchomediastinal trunk may join the right lymphatic duct, and the left the thoracic duct, but more frequently they open independently of these ducts into the junction of the internal jugular and subclavian veins of their own side. In all town dwellers there are continually being swept into these glands from the bronchi and alveoli large quantities of the dust and black carbonaceous pigment that are so freely inhaled in cities. At first the glands are moderately enlarged, firm, inky black, and gritty on section. Later they enlarge still further, often becoming fibrous from the irritation set up by the minute foreign bodies with which they are crammed, and may break down into a soft slimy mass, or may calcify. The lymphatic vessels of the thoracic viscera comprise those of the heart and pericardium, lungs and pleura, thymus, and esophagus. The lymphatic vessels of the heart consist of two plexuses: A. Deep, immediately under the endocardium, and B. Superficial, subjacent to the visceral pericardium. The deep plexus opens into the superficial, the efferents of which form right and left collecting trunks. The left trunks, 2 or 3 in number, ascend in the anterior longitudinal sulcus, receiving, in their course, vessels from both ventricles. On reaching the coronary sulcus they are joined by a large trunk from the diaphragmatic surface of the heart, and then unite to form a single vessel which ascends between the pulmonary artery and the left atrium, and ends in one of the tracheobronchial glands. The right trunk receives its afferents from the right atrium and from the right border and diaphragmatic surface of the right ventricle. It ascends in the posterior longitudinal sulcus, and then runs forward in the coronary sulcus, and passes up behind the pulmonary artery to end in one of the tracheobronchial glands. The lymphatic vessels of the lungs originate in two plexuses, a superficial and a deep. The superficial plexus is placed beneath the pulmonary pleura. The deep accompanies the branches of the pulmonary vessels and the ramifications of the bronchi. In the case of the larger bronchi the deep plexus consists of two networks, one submucous between the mucous membrane, and another peribronchial outside the walls of the bronchi. In the smaller bronchi there is but a single plexus, which extends as far as the bronchioles, but fails to reach the alveoli, in the walls of which there are no traces of lymphatic vessels. The superficial efferents turn around the borders of the lungs and the margins of their fissures, and converge to end in some glands situated at the hilus. The deep efferents are conducted to the hilus along the pulmonary vessels and bronchi, and end in the tracheobronchial glands. Little or no anastomosis occurs between the superficial and deep lymphatics of the lungs, except in the region of the hilus. The lymphatic vessels of the pleura consist of two sets, one in the visceral and another in the parietal part of the membrane. Those of the visceral pleura drain into the superficial efferents of the lung, while the lymphatics of the parietal pleura have three modes of ending. These are: A. Those of the costal portion join the lymphatics of the intercostales interni, and so reach the sternal glands. B. Those of the diaphragmatic part are drained by the efferents of the diaphragm, while c, those of the mediastinal portion terminate in the posterior mediastinal glands. The lymphatic vessels of the thymus end in the anterior mediastinal, tracheobronchial, and sternal glands. The lymphatic vessels of the esophagus form a plexus around that tube, and the collecting vessels from the plexus drain into the posterior mediastinal glands. End of Section 50. End of Anatomy of the Human Body, Part 3, by Henry Gray.